Good morning. सुनाई दे रहा आपको? हाँ, सुनाई दे रहा है. Good morning. आप थोड़ा पर बहुत धीमे हैं क्या? Hello. एक बार सर सुनाई दे रहा है ठीक से? आवाज गूंज रही है क्या? गुड मॉर्निंग एवरीवन. ऐसे क्यों हो रहा है?
Nidhi, please ensure that your YouTube link is closed, or maybe it's muted.
Hello.
Yeah, Prashanshu.
Hello. Nidhi, you are coordinating this meeting.
I'm doing that, Dr. Sharma.
Hello, I'm Dr. Sharma here. Can you hear me?
Yes, sir. Yes, sir.
But I think there is some problem in the sound.
Yes, sir. We are working on that. Good morning, everyone. Now is it phone? Oh, it's not visible. Good morning, everyone.
Good morning. Good morning.
Good morning, sir. Yes, sir. Yes, sir. So
I think now it is better.
it is better. Yes. Okay.
Yeah. Now
Good morning, Dr. Sharma. Manivannan.
Good morning. Good morning. Oh, how are you, Dr. Manivannan?
Fine, sir.
Great. Listening to you after a long time, your voice.
Yeah, sir.
Everybody is fine at home?
Yeah, all well, sir.
Thank you. Thank you. So nice of you.
Logia sir is present.
Currently, I'm not able to see him.
Yes, sir, present.
Sir is there?
Yes, sir, sir is here.
Logia sir
Okay. Good morning,
Logia
sir.
is there.
Good morning,
Sir,
sir.
good morning, sir.
Morning, very good morning.
Very good morning.
Yeah.
Sir, happy anniversary, sir. Happy anniversary, sir.
Today is
So much.
very anniversary. Happy anniversary, sir. I didn't know. Great.
Happy anniversary, Pops.
Thank you so much.
Happy anniversary, sir.
Thank you so much. Thank you so much. ये अभी इसका वो करेक्ट है क्या?
Sir, I think Namrata also today only, sir.
No.
आगे बोल रहे हैं।
Hi, everyone. I'm really excited to be joining you all on Pop's anniversary and this wonderful conference.
Thank you so much.
Is it Namrata, sir?
Namrata also today, no, sir.
Namrata joining.
Okay.
I have sent a link to Namrata, and I'm happy that despite the odd hours for her, she is on. She could log in and she is available. Namrata, could you switch on your video, Namrata? Everybody would like to see you.
Yeah. I'm not
Sure. Just 1 second.
Okay. If not convenient, no problem. But I'm very happy to see that you could join.
Yes, give me just 1 second.
Hello, Dr. Ansari. Good morning.
Good morning, sir.
How are you?
Shabana has also joined.
Yeah. So, hello, Dr. Namita, how are you?
Hello, Uncle. I'm very well. Thank you. How are you doing?
Yeah, I'm very
Hi.
fine. Very nice. Nice to see you
Hi,
after a long
hi,
time.
Namrata. Manivannan here.
Hello. Namaste, Uncle.
Namaste. How are you? Happy anniversary.
Oh,
Great,
it's Pop's anniversary. Yes.
Yeah.
Such an it's going to be a memorable day for both of them.
Yeah.
No doubt. That's true.
How's everything? How is everything in India? It looks like you guys are taking precautions. You guys are in masks.
Yeah. Still we are using the mask. Yeah.
Sir, I am alone in my room, so I am not using mask.
But we use it because I'm in the office, but we have to use
Yes.
because people used to come and go. And so sometimes it's always safe to use the mask, which is very important.
Yes, sir.
Very important. How are the COVID numbers there in India now? How is the situation?
Now it is slowly going down. It's around ranging between 65 to 70,000 per day.
Okay.
But yes, from almost 3 lakhs per day, it has come down. So the government is now relaxing all these facilities now slowly.
Okay. That's awesome that you are at least able to do the conference virtually.
Yeah. This is a
Yeah,
basically, I think.
almost since 2 years it is going virtual conference.
Yeah. 21, since last, this is the second time then we are having the virtual conference. Yeah. Good morning, Dr.
Morning,
Charu.
Dr. Saloya. Good morning, Dr. Sharma.
Very good morning. How are you?
All fine, thank you. Good morning to everybody.
Yeah, good morning, ma'am.
Good morning, Dr. Bijayan. How are you?
Oh, so nice to see. Dr. Vijay, so nice to see.
Yeah, this is Dr. Sharma here.
But everything set. Which one you use?
Good morning, sir. Good morning.
Morning, Dr. Subrat. How are you?
Good morning, sir. Good morning, Professor Loya,
Subrat,
sir.
good morning, Dr. Subrat.
Good morning, ma'am. Good morning. How are you?
Morning.
Fine. I'm fine. Good.
Classmates are explaining.
Sir, namaste.
Namaste,
Subrat
sir.
Subrat.
Good morning, Dr. Subrat. How are you?
Fine, sir. Fine.
Great, great.
But where are you now?
Good morning. Good morning, sir. Good morning.
Dr. Subrat, we have put on a weight. I think I'm seeing you.
Hello, good morning. Hello. Do you hear me, sir?
Oh, Subrat ne apna.
Subrat, nice.
No, but Subrat has definitely put on a lot of weight. He's very happy in Rajasthan.
Yeah, because I saw him after a long time, so I couldn't.
Yeah, he's happy at Rajasthan. Professor Loya's blessing.
No doubt. Nowadays, I'm in Kota.
Oh, you are.
Bilkul.
Yeah, nice picture, Subrat.
और नैप टाइम की फोटो है मेरी.
Yeah.
Good morning, Taru.
Dr. Deepa.
Hello.
Morning.
This is Dr. Mati.
Good morning.
Good morning, Dr. Haldar. Everybody. You are mute.
Channa ji, you are mute.
Yeah.
Professor Channa, could you unmute? Okay. अच्छा ठीक है. हाँ ठीक है. अपन will go by schedule, but तो कम करना पड़ता है. इको क्यों होता है? अच्छा. यूजर तो ऑफ रखते. निधि आपका ऑन है क्या? इको क्यों आ रही है? बीच में इको आ रही है नहीं आ रही.
Good morning, Loya.
Good morning, Professor Vijayan.
Good morning. Nice to see you.
My pleasure, my pleasure. After a long time.
Morning, Professor Chandana. Good morning.
Good morning, Taru.
How are you?
Good morning, Chandana. How are you, Chandana?
Good morning, Dr. Vijayan. Great pleasure to see you.
वहाँ उनके कनेक्शन में कुछ है, वहाँ से इको आ रहा है.
Hi, Loya.
अभी ये टाइम में जो सेट कर लो जो भी. हाँ तो तुम अभी वो प्रॉब्लम नहीं होगी. अब अभी अपने पास टाइम है. हेलो. अगर कोई भी इश्यू हो तो सेटल कर लो. इनको तो जरा बीच में नहीं आना.
Hello. Hello.
आप नीति बोल के बताओ, इधर हम ऑफ करते हैं अभी। हाँ, ट्राई है, कोई दिक्कत नहीं है।
हेलो सर।
हेलो।
आवाज सुनाई देता है?
Good morning, everyone.
We just checking. Yeah, that's fine. Yeah.
Dr. Sharma, good morning.
Dr. Sharma.
Yes.
Dr.
Sharma,
R. Sharma.
good to see you.
Yeah. Yes, sir.
हर जगह
You have to unmute, Dr. Sharma. How do you do?
Good morning, Dr. Suranty Sharma.
We are just trying to find out if there is any issue.
Yeah.
Could you speak out and you would like to see that everything goes well?
See you.
If there is issue in all.
Good morning, Dr. Suranty Sharma. How are you?
जैसे हम करते हैं
Good morning. How are you, sir?
नमस्कार, बहुत बढ़िया।
नमस्कार.
Namaste,
Dr. Sharma.
Good morning.
Good morning, Professor
बोलिए।
Loya.
Oh, so nice to see.
Namaskar.
So nice to see. Welcome.
Namaskar.
Your microphone is muted.
No, I'm fine. I'm unmuted myself.
Yeah, okay.
Namaskar all.
Oh, namaskar.
Namaskar.
Hope all are fine.
Yeah. Everything is good. Nice to see you.
Good morning, everybody. Professor Loya. Good morning.
Good morning, Dr. Nakvi.
Hello, Taru. How are you?
Good, fine.
Very pleased to see you again.
Same here. Same here.
Namaskar, Sharmaji. Namaskar.
Namaskar. How are you?
I am fine, sir. I am fine.
Great.
Pleased to see you. Very good.
Good morning,
Dr.
sir.
Samta Yadav. Good morning.
Yeah,
Good morning, Nagvi sir.
Namaskar.
How are you?
Namaskar.
Nagvi sir, good morning.
Fine. Thank you.
Dr. Yadav, namaskar.
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 Savita ji, namaskar.
Namaskar.
Savita ji, namaskar.
Namaskar, everyone. Namaskar.
Congratulations, Dr. Surinda Sharma, for lifetime.
It's my honor.
Yeah, congrats, Dr. Surinder.
Congratulations, sir. Congratulations. Yeah, great.
Om Prakash is not logging.
Yeah. He's not logging?
Yeah, not so far.
So far, yeah.
So far, but maybe in the process.
Maybe in the process. Still time is there, so I think.
Yeah, maybe in another few minutes he will be joining. And Professor Akbar Shah is also not seen.
No, sir,
Yeah.
not seen, sir.
Somebody can telephone him, I think.
Dr. Deepa, could you hear me? Dr. Deepa? Join किया क्या? ये विवेक के नाम से है? Oh.
Dr. Vijayan was Kasturi's Viva examiner, sir.
Yeah. And I'm so pleased to see Professor Vijayan. And sir, nice to see you.
I first met you in Dr. Vijayan conference in Pondicherry, sir.
That's right.
Good, yeah.
Myself too. I was also there.
Sharma.
Yeah. Namaskar.
It was a wonderful conference.
Wonderful conference.
Yes.
That opened so many avenues for me, Dr. Vijayan.
Yeah, it was 88 or 99 somewhere, sir.
I think quite early, yeah. It's very good.
88.
Yeah,
See, I have t-shirt weather here. So you
Vijayan sir, good morning. Vijayan sir, good morning. You are Viva examiner for
Nice
my
to
wife
see you all
in 95, sir. Karnataka University.
Yeah, that's right. It's long back.
Morning, sir.
No, I was mentioning I have a T-shirt weather here.
Oh,
Yeah.
great.
India, yeah. We can see your T-shirt.
That's right.
प्रमोद ने भी ज्वाइन किया हुआ टोटल कितना हो गए ज्वाइन किए 56
including us.
अभी अपन
Sir, Dr. Curtoch is coming personally here, or he will be joining?
In fact, we have spoken with him, and he said there is no problem. He was most willing to join and come personally here. But then he said that he will be using the mask, and without mask he would not would like to deliver the. So then we prefer that he can do it from his residence. And I would meanwhile share with you that what we have done. Maybe later on it will be done, but this is the newsletter being edited by Dr. Deepa and Dr. Vikas, and this will be one who will be released by the chief guest and the dignitaries. Then there is another newsletter. This newsletter deals with the stem cell, and there was a lot of motivation from Dr. Taru also, and her initiative, and then taken over by Dr. Deepa and Dr. Vikas, that came up very well. This document is beautifully compiled. And then the another newsletter is about the AIDS. The title is HIV/AIDS Beginning to Current Scenario. This is first time that our society is going to release 2 newsletters in one event. Generally, we do it one newsletter.
One.
And it is biannual. So one is released in September during midterm activity, and another during the annual meeting in February. So
great.
This is another newsletter which will be released today. And then simultaneously, this is what you say is the abstract book.
Abstract book, yeah.
And you will be very pleased to know that there are about 91 posters being presented, and it was very difficult for the experts to evaluate them and go for marking. So very good quality of what I want to say is very good quality of the posters being presented. And of course, all the abstracts and everything along with the program is there in this document.
Great. Congratulations to all of you.
So this, I would say, a lot of exercise being done by Professor Ansari and Dr. Barkha, and they worked there, and you will be pleased to know that all this thing is being done by our own group. I would say that there is an input of everybody to starting from communication, coordination, compilation, and to bring it out, and a lot of coordination with the editors. And I must say that editors did a wonderful job in ensuring the quality of the manuscripts, articles, and timely editing them, passing on them to our ISR secretariat, and then, of course, later on, it was taken care by Barkha and Dr. Ansari. So this is how things have come up. And when
great job, editors. Great.
And
when it's a team, sir, your team, our team.
Yeah, congratulations.
Good, sir.
Great teamwork.
Shipra is here. Dr. Shipra.
Yes, sir.
Have you wished, sir?
Hello.
How are you? Hello.
My voice is breaking.
Oh, good morning, OP. How are you? Can you hear us?
Hi, Dr. Sharma. Congratulations, Dr. Lohia, Dr. Sharma. Congratulations for that.
Thank you. Thank you.
Good morning, Chaturvedi.
Good morning, sir.
Doctor.
How are you, Dr. OP Om Prakash?
Yeah, unmute.
Yeah, you have to unmute. You are
Unmute Om Prakash.
Okay, now can you hear me?
Yes, we can hear you. Congratulations, Om.
Thank you, everyone. Thank you. Good to see you.
Congratulations, Dr.
Good morning,
Prakash.
sir. Congratulations, sir.
Thank you very much. Thank you.
Nice
Namaste,
to see you,
Doctor.
Dr. Nice to see
Namaskar.
you.
Namaskar.
Sharma, congratulations. Dr. Ansari, congratulations, boy. बहुत बढ़िया.
Thank you. Thank you. Thank you so much, sir.
Om Prakash, Om
Haji,
Prakash
Haji.
chair is being occupied by our Dr. Barkha. You used to sit here. Hello, Dr. Das.
Professor Kamboj.
Good morning.
Good morning, Dr. Kamboj.
Good morning, sir.
How are you, sir?
Good morning, Dr. Kamboj.
Good morning.
Curtoch sir has also joined. Namaste, sir. Good morning. आपका म्यूट है सर. Curtoch sir, आपका म्यूट है माइक.
Hello.
Namaste. Good morning, sir. How do you do?
Good morning.
Good morning, Dr. Kamboj.
Morning, sir.
Vijayan from Cochin.
Good morning.
Good morning, sir. Good morning, Dr. Curtoch sir.
Good morning,
everybody.
Good morning.
Good morning, Professor.
Good morning.
Good morning, everyone.
Hi, Savita. Good morning.
Good morning, sir. Good morning. Good morning, Dr. Chaturvedi. Good morning.
Good morning to everybody. Good morning.
Hello, good morning, everybody.
Morning.
Good morning, Kabir.
Kabir here.
Good morning.
Good morning, Professor Kamboj. Kabir here.
Yes,
Oh, Dr. Kabir, how are you?
Oh, I'm otherwise I'm okay, but
Okay.
literally I'm not fit.
Okay. Namaskar, sir. Namaskar. Good morning, sir.
शर्मा जी कहाँ से बोल रहे हैं?
शर्मा जी गुड
मैं
मॉर्निंग.
ऑफिस से बोल रहा हूँ, सर.
नमस्कार, कटोच जी.
अच्छा.
मैं ऑफिस में ही हूँ.
नमस्कार, सर.
ओके. नमस्कार कम्बोज साहब. नमस्कार प्रोफेसर लोहिया. नमस्कार. यस.
नमस्कार सर.
गुड मॉर्निंग.
नमस्कार सर. नमस्कार.
साहब गुड मॉर्निंग.
कम्बोज साहब को नमस्कार.
नमस्कार लोहिया जी.
कम्बोज साहब को नमस्कार.
नमस्कार.
डॉक्टर शर्मा कह रहे हैं मैं ऑफिस में हूँ, तो मेरा क्वेश्चन था कि आप घर कब जाते हैं?
उन्होंने घर ही ऑफिस बना रखा.
नहीं सर, जब उनको टाइफाइड होता है बस तभी जाते हैं, नहीं तो ऑफिस में ही रहते हैं.
अच्छा.
He was really, yeah.
Dr. Sharma, are you listening? Comments?
Yes, sir. I'm listening. Yes, I am listening, sir. January I had a problem because I had a typhoid admitted. Now I'm perfect. Correct.
Okay, that is why they say that you are at home only during illness.
Namaskar, Dr. Lohia.
Oh, namaskar, sir. We are waiting for you.
Namaskar, sir. Namaskar.
Good morning, sir. Namaste, Professor Madan.
Congratulations, sir.
Congratulations, sir.
Namaskar, sir.
Thank you.
Congratulations, sir. Congratulations, sir.
What a happy morning to see you, sir.
Pleasure to see you all.
नमस्कार सर. नमस्कार.
नमस्कार नकवी साहब.
सर बहुत बहुत मुबारक हो सर. बहुत बहुत मुबारक हो. ये हमारा भी सीना चौड़ा कर देता है सर. इनाम आपको मिलता है, अवार्ड आपको मिलता है, लेकिन खुशी हमें सबसे ज्यादा होती है.
आप उसके हिस्सेदार हैं.
थैंक यू सर.
नहीं, ये डॉक्टर नकवी, इसको खाली इनाम कहना नाइंसाफी है. खाली इनाम नहीं है.
नहीं.
Something else, but so much as you said, I'll not say anything now.
You are right. You are right.
There has to be a separate thing in the honor. इनाम खाली चलेगा नहीं.
नहीं, you are right.
Check your connections. नीचे बॉटम में राइट में उसका वो है ना, स्क्रीन को बड़ा कर सकते नहीं, वहाँ नहीं, उस पे स्क्रीन का जो स्लाइड है उसका आपकी जो कौन सी बात
उसको थोड़ी करना है।
कंबोज साहब थे, कहाँ गए? नमस्कार।
नमस्कार जी, नमस्कार। नमस्कार मदान साहब,
नमस्कार जी।
मुबारक हो, सभी लोग मुबारक कह रहे हैं, खाली मुबारक हो रहा।
क्या बात है। ये मुबारक नमस्कार। नमस्कार कटोच साहब, नमस्कार।
मुबारक
सर। काम्बो सर, हमें कुछ करनाल में करना पड़ेगा इस इवेंट्स को और वाइब्रेंट करने के लिए। कटोच I would agree with me, and there has to be different things.
I must share with you, I must share with everybody that Professor Madan has agreed to have a grand celebrations, and we have to decide the date, time, and we look forward that some occasion where all of us could be there, and we felicitate him and as well we enjoy the celebrations.
Lord, maybe next year only.
That's great.
Very good. Great.
Good morning, Dr. Lohiya. Good morning, Dr. Kamboj.
Morning,
good morning. Good morning, everybody. Good morning. Good morning. Good. Yeah. Very nice to see all of you. Yes, as you very rightly said, let's soon have an occasion to have a physical meeting.
Yes.
We hope that in next year we will be able to organize the physical.
Let's hope. I'm thinking it will end, pandemic will end this year, but now today WHO said be prepared for another virus.
Oh. Another virus or another variant?
Another variant of this, another more severe variant of COVID.
Variant of this, huh?
That is sister of Omicron. Stealth variant.
Scaring everybody.
Good morning.
Kambo sir, in Hyderabad, people say brother of Omicron.
They say sister of Omicron.
Hello, Dr. D. Singh.
Good morning, sir. Professor.
Good morning, Dr.
Nice to see you.
Good morning, Doctor.
Good morning, Dr. Kabir.
Good morning, sir. Good morning.
Good morning, sir. Kambo sir, good morning, sir. Dr. Namaskar.
Good morning, sir. Good morning.
Hi, Dheer.
Kato sir, good morning, sir. Good morning, Namaskar.
How are you, Dr.
Good morning, sir.
Congratulations, Dr. Professor Madan, sir.
Thank you very much. आप इसके हिस्सेदार हैं, आपको पता है।
नहीं सर, ये आपकी जहाँ हम बैठे हैं फर्स्ट आप ही थे यहाँ पे।
इधर भी देख लेना, इधर भी पूरा सही कर देना।
ये ज्वाइंट शेयर है because joint director research of India is the first actually. Dr. Madan was the first.
You are as a partner. No choice. And all this is possible because of you.
Thank you very much, sir. Professor Surandar, good morning, sir. Good
morning, sir. Good morning. How are you, sir?
I'm fine. How are you doing, sir?
Doing fine.
sir.
Dr. Suranty Sharma, hi, congratulations.
Thank you, sir. Thank you. How are you, sir?
Hello, Dr. Sharma. Dr. Sharma, how are you?
Fine, sir. How are you?
Oh, I am fine.
Good to hear that.
Good morning, everyone. We'll be starting in 3 minutes. So we'll be muting everyone. Thank you.
Yes.
Dr. Surinder, namaskar.
Yes, sir. Namaskar, sir. Namaskar. What is the occasion? Everybody is congratulating you. What happened?
He's honored with Padam Shri. President of India.
I have got to see the face of Dr. Sharma today. And that's what is a pleasure.
We used to call you Sri Madan, Professor Madan ji, now Padam Sri now we have to add that.
You can go without that also. It's enough to call it Madan. I think that does suffice.
Okay, sir. Whatever you say, you are Padam Sri to us now. It doesn't matter.
Suffixes and prefixes are just to fix. The name stands. That's what is important.
Sir, hearty congratulations to you.
No matter what, but everybody is asking you to do some grand program there in Karnal. So that's what we are all looking
No,
forward
it is not in Karnal. It has to be in Jaipur. That's what the Dr. Lohiya was saying about. Dr. Lohiya has suggested it to me, and I'm saying that it has to be in Jaipur.
Well, Dr. Lohiya is president. He can suggest anything, and we all have to agree to that.
Yes. That's true.
Yeah.
How have you been equipping? Everything fine, Dr. Sharma?
Yes, sir.
Well, glad to know that.
Yeah, we are all doing fine here, trying to stay away from COVID. So far, so good.
Yeah, well.
So we just have to worry because I work in NICU.
It is playing foul. It's coming back over and over again, and that's what is disturbing.
Yeah.
Good morning, everyone. We are right on time, so we are going to mute everyone. I hope that is okay with you all.
आपका भी आवाज नहीं आ रही है आपके आवाज yourself is muted. You are muted yourself also.
Is it audible, sir?
Now it is.
Now it is okay.
Okay.
Yes, now it's fine.
Yeah, now we can. Yeah.
Okay. Okay. So a very good morning to all of you. I'm Nidhi Shukla, working as DST Women Scientist Fellow at BISR Jaipur. It is my great pleasure to welcome all of you to this 3-day international conference on reproductive healthcare and 32nd annual meeting of the Indian Society for the Study of Reproduction and Fertility that is being organized by ISSRF at University of Rajasthan, Jaipur. So just to give a brief background about ISSRF, this society was established in 1988. And it has more than 1,700 members, comprising of different scientists all over the world, public health executives, and clinicians from the field of reproductive sciences. The main aim of our society is to bring a collaboration so that we can bring a fruitful and better collaboration. Our society has been organizing annual meeting as well as midterm activities regularly. Other activities of society is to publish newsletters. ISSRF also has an online peer-reviewed journal, that is Journal of Reproductive Healthcare and Medicine, JRHM. With great honor, I invite Professor N.K. Lohiya, who is president of ISSRF, and he's NACI senior scientist. And Dr. Lohiya needs no mention. He has so many awards and accolades in his name, which won't be possible to summarize in few minutes for me. So I would request Professor Lohiya, sir, to kindly say a few words about this conference and the inaugural function. Over to you, sir.
Thank you, Nidhi. And I think the all dignitaries and guests participating in the event are very much familiar with the ISSRF, and I would like to go back in the year 1988, and it was the idea of Dr. T.C. Anand Kumar, who happened to be the then director of IRR, Institute for Research and Reproduction, Mumbai, and he founded the society. And subsequently, we see that it was nurtured by the Dr. Puri, who happened to be the president and also director of that institution, which later named as an
No audio.
over the
We have lost the sound.
Audio lost. Audio lost.
No audio.
Appreciate your patience, sirs. We'll just get back.
Nidhi, you have to check the audio.
Yes, sir. Yes, sir.
see that the society has become very strong, and you would be very pleased to see that this 1 year we had almost 67 life members joining the society, and overall there is hardly any institution, any university, any medical college, medical university, veterinary institutions and universities which is not represented, and that is a matter of contentment for all of us. Generally, we say that this is a unique society. The reason being that when we talk of reproductive health, we see that it covers human, it covers animals, it covers livestock. And a society which is providing such a platform of interaction with clinicians, molecular biologists, basic scientists, and veterinarians, and they interact with each other. And we saw that the sharing of the information in terms of whatever latest scientific developments are taking place is contributing a lot towards reproductive health issues. While formulating this year's scientific program, I'm very happy to share that a care is being taken to see that all members of the society could have the platform for presentation of their latest research findings. And from that point of view, almost all aspects of reproductive health being covered in the theme of the conference. And we see that it covers almost 3 keynote speakers, almost 6 orators, around 29 invited speakers, and oral presentations 18, and poster presentations 91. I must share with you that the platform is being provided to see that the young scientists get opportunity to share their research findings. And in order to encourage young scientists, there are a number of provisions with the society in terms of awards. We have almost 10 Young Scientist Awards. We have several opportunities for young scientists to participate in the form of the Best Poster Presentation Awards. And the society also takes care to acknowledge the contributions being made by the senior scientists in the form of various orations. So with this, I would say that society is ensuring to meet mandate, which was initially fixed by the senior colleagues of the society. And as on today, we see the society ensuring this is 32nd annual meeting, meaning by that despite very unfavorable conditions due to COVID-19 pandemic and now the omics, we ensure that the regularity is being maintained by the society, and we are organizing, of course, in a virtual mode, but the 32nd annual meeting of the society. In addition, as being mentioned, we ensure regularity of publication of the newsletter, and now today, after some time, our dignitaries will release the newsletter of 28th issue and 29th issue. In addition, society has a journal, and that is Journal of Reproductive Health Care and Medicine, and being published by scientific scholar from US. And I would use this opportunity to request all of you to kindly support this journal in terms of contribution of your important research findings. And this year, Society has taken a decision that those who are awardees and those who will be delivering orations are requested to submit one manuscript after the conference, maybe in a month or two, so that the journal is strengthened. I'm very pleased to say that the society is also ensuring midterm activity in the form of awareness programs. So for last several years, without any fail, we have midterm activity in the month of September. And that again is a good visibility of the society. And the young minds, specifically senior school students and early college students, they become part of it. And it has been very useful in terms of identifying important health issues for them. And senior colleagues are requested to deliver lectures which would serve the purpose of awareness amongst the young minds. With this brief, I have a great pleasure in welcoming our dignitaries, which were being introduced a little later. We have with us the chief guest of the event, Professor Dr. Curtoch sir. Then we have very distinguished scientists with us, Dr. Kamboj from CDRI, earlier CDRI director. Then we have recently, everybody is complimenting Dr. Madan, who has been felicitated with the Padma Award, Padma Shri, and all of them will be joining in the inaugural function a little later. And with this, I have a pleasure extending very warm welcome to one and all, and we hope that your participation in the event would make the conference ISSRF-2022 a memorable event. And once again, a big thank you to everyone for joining the event. Thank you so much.
Thank you so much, sir. That was so enlightening, and it was so kind of you to tell us about the ISSRF and its activities. Now, with utmost pride, I invite our guest of honor, Padma Shri Dr. M.K. Madan. Dr. Madan has done extensive work in the field of reproductive endocrinology, in vitro fertilization, and cloning. And Dr. Madan is former Vice Chancellor of DU Vasu, Mathura. And as everyone is congratulating Dr. Madan, so we all know that he has been recently conferred with prestigious Padma Shri, the fourth highest civilian award in the Republic of India. Many congratulations, sir. We are really lucky and grateful to have you with us today for this inaugural session. And I would really, it's my really like a pleasure to invite you, and I would request you to say a few words for this inaugural session. Over to you, sir. Thank you.
Thank you very much. And at the outset, firstly, greetings from me to you all. And at the same time, thanks to the organizers, Dr. Lohia and his team, for an opportunity which we are having through this webinar to be speaking to each other, even under the circumstances which have kept us away from meeting each other for the last 2 years in a more close and intimate fashion, though not from science itself. I would like to thank you for the kind good wishes you had for me regarding the recent recognitions which has come from the Government of India to the work which a scientist has done in his laboratory. I attribute this award as not an award to me, but I attribute it as an award to science and to all those people who have been associated with me, particularly those who have been in my laboratory, my institution, my profession, and in the larger interests, the gathering which we are having today, who are very much associated with reproductive biology and have been part of what I have been trying to do. Friends, being associated with an exalted group like you is always a privilege. And to be addressing them at the start of the conference gives me an extra feel that we have got a long way to go, and we have come a long way so far. When I talk of reproduction and reproductive biology, I understand that we have been handicapped during the last 2 years for some reasons which are not unknown to each one of you. This has been the worst of times, and in some aspects it has been the best of the times. Best of the times means it gave us more time to be speaking in the digital fashion on the webinars, but you know how we suffered. It has been a period of foolishness and loss of wisdom, and also that it was a period of intellectual feedback through which that we worked in an era of darkness without light. But at the same time, that this was a period of great hope, and the despair which had gone in is now edging out, and we are looking for that the coming months and coming years will put us in gear. I know that as far as the reproductive sciences are concerned, that with respect to the functioning of the biology, we have had a large number of advances which have taken place in the scientific field over these years. As has been mentioned earlier, that we started with reproductive biology in the earlier years, that when we did not know what is the circulatory condition of hormones in the body which are determining the reproductive systems and reproductive functioning. And it was at that time that we started with radioimmunoassays and got hold of almost as many as 14 of them in the biological systems for animal models, for livestock, because that is the one which was not being investigated. I remember that at that time, as Dr. Lohia said, that we had just started with our endocrinology work at Bombay, and Dr. Sheth was the one who was doing the radioimmunoassay, but in an heterologous system. And so we also started in a heterologous system till we had to bring in hormones from bovine system, purify them, and use the purified hormones, produce the antibodies, do titers, and work out the circulating levels of the hormones. That, in fact, has laid the foundations in the country as far as the understanding of the reproductive function is concerned. And then reproductive health becomes a very important part of it. Reproductive health, whether it is the human system or it is the animal system. In fact, that what we first tried to talk about in the livestock was the issues of fertility. We had an acute problem of repeat breeding. We had an acute problem of animals not settling to the process of insemination, artificial insemination, and technique, and which had been introduced in the livestock much, much earlier. And the difficulties which at that time were with respect to getting an optimum fertilization rate. Interestingly, as we all know, that in the area of reproductive biology, we have got the same aim. For example, we are talking of augmenting fertility, whether it is animals or in human, but our approaches become different, because in process of fertility, we are ensuring that the rate at which the population increases among the humans is reduced, but the couples, but the people who are suffering with reproductive infirmities are being helped out. And on the other side, that we are saying that the augmentation of the reproduction should take place. We should have more number of births. And in that direction, we took up the work with working on the ovaries, causing them to superovulate, getting out a larger number of ova from there, fertilizing them, and this fertilization was taking place in vivo, and then taking up these embryos at 3 to 4 days of age, between 4 to 7 days of age, and transplanting them into the female reproductive tract of other animals. I think that way we started with the process of what subsequently came out to be the star technology at present, which we call the in vitro fertilization technology. And in that process, we had to understand that what are those events which are taking place, the events of what we say the sperm maturation and the ovum maturation, the event of fertilization, and the earlier gastrulation and blastulus formation. And these events had to be picked up onto the laboratory bench, and the progress took place. At this stage, I remember what Dr. Edwards said a decade and a half back, the one who had started with the human in vitro fertilization. He had a very interesting observation to make. And he had said that, I quote, I wanted to find out exactly who was in charge, whether it was the God himself or whether it was the scientist in the laboratory. And to that he answered himself that it was us. And I think that when we are talking of the present day technologies, and we say that we are going ahead, seemingly supernatural ability to unite sperm and eggs outside the body and implant them directly into the womb, which has been heralded as the remarkable achievement for fertility. And over that, now different issues have crept up. Events have taken place and considerable success has been made. And I will just enumerate about 3 or 4 of them. For example, we talk of now egg freezing. It is not considered an experiment. It is done normally. We have got frozen embryos put in the can, and these can be revived. We have already revival of these frozen embryos and young ones produced, not only young ones produced from frozen embryos, but clones produced from frozen embryos. Then we can have more reliability in selecting the best embryos for IVF. Today, where we are now is that we can get over to the health of individual embryos. I think that's a great advance. We have got now laboratories, some laboratories in the Western world, which are now written and described as the embryo health laboratories. And they have got the capability of judging an embryo just post fertilization and as it develops, and then keeping track of it, that in the first 4 days, that how is it developing, whether it is fit for continuing as in the gestation, or we need not to get it transferred and work with other embryos which we have got from the same parentage from which we are working for the process in the human populations. We have also had an advance, what we call as the better viewing of the infertility in the male, which used to be a big problem. And we have now a lot of work going on in the area of sperm understanding. Also that picturing particularly the men who produce little or no sperm and who had previously been rendered as sterile. Now through a technique which is called as the TESE, T-E-S-E as it is spoken, which has been developed and now improved considerably. Similarly, that we have now more options for preventing births through IUD, and also more options to have a single female, in the case of bovines, producing more number of young ones. We can have through this ovum pickup techniques as many as sometimes 40 oocytes picked up and then fertilized in vitro, and then the young ones produced out of it 8, 10s, 12s. And there is now this type of work which is being pursued over into the field for this reproductive augmentation. One is that the other aspect of it is in the case of the disease. We have now a greater understanding of the problems of ovarian diseases, and in that connection, the ovarian freezing, better freezing techniques for the embryos, and also understanding of the lifestyle affecting the fertility. In the case of bovines, now we have got means by which we are talking of judging the fertility in case of in the case of buffaloes, in case of cattle, in case of sheep and goat, and now doing technologies by which we can have the greater fertility, augmenting by through putting in certain genes in the case of animals that which have got capacity for better fecundity, better ovulations, and using that particular gene through interrogation, about which you will be hearing during the conference, more about that during the conference. Where are we, therefore, on this in the area of reproductive biology? We're having, on one hand, a great amount of advance taking place, and on the other hand, that we are seeing that in terms of getting them translated from, for example, from an animal model to a human model, that we need more work to be done, and there is a great need for greater translational activity and world. I'm sure that during this conference, as we are progressing, we'll have a lot more to be talked about, and we are having our colleagues will be talking something very excitingly about the advances in which that we in the country have been taking lead, as in previously in reproductive biology, and that's in the area of cloning. We have got presentations on cloning, and we have got as many as 10 clones out of the same animal. And just as a point of caution with which I would like to end this introductory point of this function is that it is presenting great challenges for reproduction as such. The gametes have lost their concepts that gametes are the ones which are most essential, and the union of the gametes is the one which produces a young one. Today in our laboratory, for example, in the laboratories at Karnal, we have been able to produce a young one from the air cell and produce a young one from the cell in the semen, which has remained stored for over 10 years. And using this cloning technologies, extending it through the stem cell technologies, and going over into the area of regenerative medicine, that we are opening a great field for research and investigation. And in the direction, I would like the young people to commit themselves and find out that how they can be used for our better purposes. With these words, I have great pleasure, again, number one, to thank you all, thank our society organizers, our society leadership, and I have to acknowledge here that I am speaking it here under the august chair of Professor Curtoch, who has been a great help to us earlier in our research, and who is commanding light to us in the human health and animal health, and also Professor Curtoch, Professor
Kamboj.
Dr. Kamboj.
Yeah, and who is with us, and a brand of other young people who have been coming and who have now taken up responsibilities in various institutions in the country, including our young scientists in the livestock. For example, we have got Dr. Tharo, Dr. Chauhan, Dr. Naqvi, who have been with us over years, and they are now here. Thank you very much again.
A big round of applause for Sir. That was such an stimulating talk, and it was so enlightening for all of us. So So now I request all our esteemed dignitaries to join us in the release of the abstract book, as well as 28th and 29th issues of ISSRF newsletter. As Lohia sir mentioned that usually we release only one newsletter per year, but this year we are releasing 2 newsletters at the same time. Sir, please.
May I request the chief guest of the function, Professor Curtoch, and guest of honor, Professor Madan, and the Professor Kamboj, who is presiding over the event, to kindly release first the abstract book. And as being mentioned, abstract book is being compiled by Professor Ansari and Dr. Barkha. They worked very hard to see that it comes to the expectations of all of you. And when we will mail it to all members of the society, all guests, I'm sure that you would appreciate the efforts put in compilation of this, and the quality of abstracts being contributed by our esteemed senior scientists, young scientists, for awardees and so on and so forth. So may I request to first release this abstract book, come program come abstract. Could we see Professor Curtoch on the screen, please?
Yes, sir, we can.
Yeah. Very nice. And Professor Madan and all that, we can see it on the screen. And then second document I would request to be released is the 28th issue of the newsletter, and it deals with HIV and AIDS, beginning to current scenario. And I must add that society takes care of identifying the current most relevant reproductive health issues, and the newsletter will cover almost all scientists dealing in this field, and there are about 19 articles being contributed for this, and it is being edited by Professor Martin Ahmed, along with contributed by Dr. Ansari and Dr. Barkha. And I would request Dr. Martin later to say a few words, maybe a minute or two, if he is live and if it is login. Dr. Martin, please.
Yes, sir. Yes, sir. Can you hear me, sir? Hello.
Yes. Yeah, please speak on.
So, yes, first of all, I'll thank our organization, ISSRF, and our president, Professor Lohia sir, for giving me an opportunity to edit a magazine for this international conference. And secondly, actually, it has not been, I have not been able to come up with this newsletter without the help and the constant support and inputs from Dr. Lohia sir. It contains, as Dr. Lohia sir told, that it contains around about 19 topics, and it has been well covered. I had tried to do the justice with the theme of the newsletter, that is HIV/AIDS beginning to current scenario, and I am thankful that some of my teachers and mentors have helped me to shape this
newsletter.
Yes, shape this newsletter, and some of my friends and colleagues globally, they have contributed to the articles on HIV/AIDS, for which I am very thankful. And I really wish this international conference success. to add that I'm the person Curtoch at Kolkata during one of our AIDS Society of AIDS Society of India conference. And there, I got permission from him. He was then DG ISMR. I guess it is 2018, and I guess I'm talking from a remote village in Bihar.
No problem. I think.
So
Thank you so much.
activity might not be.
Martin, thank you so much. We could listen.
Yes.
We could appreciate
Thank you,
your
sir. Thank you very much. Thank you very much.
Yeah. Okay.
Yes,
thank
sir.
you
Thank
so
you very
much.
much, sir. Thank you.
Yeah. And then we have another newsletter. This is 29th issue of the newsletter. And this talks about recent advances in stem cell biology and therapeutics. And the initiative was taken by Dr. Taru Sharma. And because Taru Sharma was to join as a director of a institution, National Institute on Animal Biotechnology. So obviously, a lot of other commitments were there. And Dr. Deepa Bhartiya took up the responsibility. She was the one who initially communicated to ISSRF about her willingness to edit this newsletter, and along with Dr. Vikas, Dr. Vikas Chandra from IVRI institution. So I'll request Deepa, if available, to please say a few words about the newsletter.
Yeah. Hello, everybody. Can you hear me?
Yes.
Okay. It was a wonderful pleasure to come up with this newsletter. And I really thank Dr. Lohia, all my seniors for the encouragement, and all my colleagues who have contributed very interesting articles in this newsletter. I hope all of you will enjoy reading it and get updated on the recent advances on stem cells. Thank you so much.
Thank you so much, Dr. Deepa and Dr. Vikas, for your valuable contribution to the society, bringing out the 29th issue of the newsletter. Thank you so much.
Thank you to our dignitaries for release of abstract book and newsletters. Now, with great pleasure, I invite Dr. R.S. Sharma, Secretary ISSRF. Dr. R.S. Sharma is ICMR Emeritus Scientist who has published several research articles in international journals, and he has received many awards such as Lifetime Achievement Award and Lapteshwar Award in the area of population control, to just name few. I would request Dr. Sharma to please introduce the Lifetime Achievement Awardees and their accomplishment to all of us. Please, sir.
Thank you, Dr. Nidhi Shukla. It is as a Secretary of ISSRF, it is my great pleasure and honor to introduce the Lifetime Achievement Awardees of the ISSRF for the year 2022. Now, as our standard policy based on the outstanding contribution made by these scientists, the Search Cum Selection Committee of the ISSRF nominated 3 Lifetime Achievement Awardee for the year 2020. And I'm going to introduce one by one these Lifetime Achievement Awardee. The first one is Dr. Suranty Sharma. Dr. Suranty Sharma is a research scientist and professor in the Department of Pediatrics at Women and Infant Hospital of Rhode Island, USA, and the Warren Alpert Medical School of Brown University. He is the principal investigator and director of NIH-funded Center for Biomedical Research Excellence of Reproductive Health. Dr. Sharma was the recipient of the 2007 Black Bell Munksgaard Award for Outstanding Research in Reproductive Immunology. He recently received the 2014 Distinguished Service Award from the American Society for the Reproductive Immunology. Dr. Sharma's career has spanned 3 decades in reproductive biology and reproductive immunology, with long-term research interest in placental biology, reproductive immunology, and pathogenesis of adverse pregnancy outcome. Now, as the secretary of the conference. It is, and also this is my great pleasure and honor to confer the Lifetime Achievement Award of the ISSRF to Dr. Suranty Sharma for the year 2022. Now I would request Dr. Suranty Sharma to say a few words. Dr. Sharma.
Thank you very much, Dr. Sharma, for those kind words. First, greetings from the US to everyone there. It's midnight here, so there it's morning, so early good morning to all of you. It's a Lifetime Achievement Award. And I think indeed sometimes lifetime dreams come true, and this is one of them. And I have been awarded so many prestigious awards in my lifetime, and this is right up there. I mean, it's so important, so thrilling for me to receive this award from my Indian peers. And here in the US, we do research, or I'm a physician scientist, so I also attend to the clinic. And we get these awards, but getting Lifetime Achievement Award from India and from my Indian peers is a great thrill personally. And I thank Professor Lohia and his organizing committee for bestowing this coveted award on me. And so I'll just keep my comments brief. And I would be giving a talk this morning, 7 o'clock here, US time. So I'll be talking about what we are doing in the lab in terms of science. Professor Madan has already touched upon so many themes in reproductive biology, reproductive medicine. So I'm not going to really say anything more. I will just piggyback on his comments, saying that reproductive biology, reproductive medicine field is at the cutting edge stage at this point, and so many wonderful things are coming up, and we are learning a lot. And so I'll just stop right here and thank everyone again there, and good luck for the meeting. Thank you.
Thank you, Dr. Sharma, and the congratulations for this award. Now,
thank you.
I have a great pleasure of introducing my fellow colleague, Dr. Om Prakash Sharma. Dr. Om Prakash Sharma is the Executive Director and Clinical Development Oncology at Hungary Pharmaceutical, Princeton, New Jersey, USA. He led successful development and global regulatory approval of multiple drugs in different therapeutic areas. His research interests are in clinical development of drug conjugates and novel androgen receptors inhibitors for multiple tumor types, including prostate cancer, breast cancer, and other tumor types. He is the recipient of Alexander von Humboldt Foundation Fellowship to advance his research work at Max Planck Institute, Germany. He is a board member, the chairperson, and the president of several international social organizations. Now, on behalf of the society, it is my great pleasure to confer the ISSRF Lifetime Achievement Award to Dr. Om Prakash Sharma of the year 2022. Now, I request Dr. Sharma to say a few words.
Can you hear me?
Yes, yeah. Yes, Dr. Sharma.
Yeah, thank you very much, Dr. R.S. Sharma. This is really truly an honor. I'm humbled with this recognition, particularly by ISSRF, which I have been member for a long time. Plus, I originated from the same family. So from Professor Lohia and Dr. R.S. Sharma has been my colleague. And so thank you very much for that. I would like to take this opportunity to first of all, to thank my family, particularly my wife and my son, who both of them are surgeons. My son is a neurosurgeon. My wife is a vascular surgeon. So it has been really tremendous support from my family to continue my research career. which I originated from the reproductive biology or reproductive endocrinology, and then really brought lab science to patients through the drug development, and which has been a really interesting journey, because I went from lab research to obstetrics and gynecology, urology, and currently in oncology. For last almost a decade, I have been in oncology, where also I have been using those skills and trainings which I received earlier on in the prostate cancer, breast cancer, and it is very fulfilling to continue my own interest, research interest, and at the same time bringing that research to fruition to the patients. And it every day gives me the meaning of life. It gives me an objective for that. So I'm truly thankful for my entire this journey, and I have been enjoying this. And with that, thank you very much. And I'm glad to be part of this conference, and being able to participate in this one. And again, as Dr. Sharma said, I'll be speaking around 5 o'clock our time here in the US for my talk. Thank you.
Thank you, Dr. Sharma, and congratulations for this award. Now I
thank you.
The next lifetime achievement awardee, Dr. M.A. Akbar Shah. Dr. Akbar Shah is a senior research National College Tiruchirappalli. He is the former professor of animal science and biomedical science, Bharatidas Dasan University, Tiruchirappalli. He is the founder and general secretary, Society of Alternatives to Animal Experimentation in India. He is a scientist and a teacher for last 38 years. His area of research includes the endocrinology, the reproductive biology, the phytotherapy, the cancer biology, obesity, in vivo and in vitro toxicology, the alternatives to animal experimentation. He has published more than 250 research papers. He has operated several sponsored projects, both from various funding agencies like UGC, DST, CSIR, ICMR, and has been awarded grants for international collaborative studies, including from NIH USA and Kumar Breast Foundation USA. This is in addition to more than 50 treble awards. He has received Paranaidu Govinduraju Gilt Medal Oration Awards in 2016 from the Society of Reproductive Biology and Comparative Endocrinology, and also recently has been awarded the Lomas Oration Award from Modern Institute of Medical Sciences. Thank you. And now I, on behalf of the society, it's my great pleasure to confer the Lifetime Achievement Award of 2022 to Dr. A.M. Akbar Shah. Dr. Akbar Shah. Now I would request you to kindly say a few words.
You have to unmute, Dr. Shah.
You have to unmute Dr. Shah.
I'm sorry. Yeah. So it is really very a pleasant privilege to be addressing the members of the society, the Indian Society for the Study of Reproduction and Fertility. Most respected Professor Lohia, the senior scientist associated with the society, Dr. Kamboj, Dr. Curtoch, Dr. Madan, and my friends, Dr. Iss Sharma, Dr. Ansari. It has been very pleasant. I've been so happy to have been conferred this Lifetime Achievement Award. I have been associated with this society, Indian Society for the Study of Reproduction and Fertility, since inception, 1988, since when Dr. Professor Anand Kumar was in the Alma Papayas, then Dr. Anil Sheth and Dr. Chandarpuri, and then now we have Dr. Lohia doing a wonderful job, supported by Professor Iss Sharma. I have been associated with this reproductive biology research since 1980s, and it is really nearly 40 years, a long journey of very highly enjoyable research conducted in the reproductive biology with special emphasis on reproductive health. Dr. Madan made a special mention about this society is concerned also about men who produce less sperm and who do not produce sperm. My research has been essentially centering around this particular concept of male infertility and subfertility, looking at human samples and animal experimentation. It has been a really enjoyable long journey in reproductive biology and male reproductive health. And I am really very grateful to this Indian Society for the Study of Reproduction and Fertility for giving this great recognition on me by conferring the Lifetime Achievement Award. I thank all those who are associated with nominating me to this award. And I thank everyone who is associated with this conference. I wish this conference all the very best and success. Thank you very much.
Thank you, Dr. Akbar Shah. On behalf of the society, I congratulate all the Lifetime Achievement Awardees. Thank you. Over to Dr. Shukla.
Thank you, Dr. Sharma, for introducing us to all the Lifetime Achievement Awardees. And we congratulate all of them. Many congratulations, sir. Now with great excitement and pleasure, I would invite our chief guest, Dr. William Curtoch, who does not need any introduction. As a stalwart in the field, we all know him very well. He is NASI ICMR chair, and we are so lucky that he is associated with RUHS Jaipur. He is former secretary, DHR, Government of India, and DG ICMR, Delhi. Dr. Curtoch has developed the molecular methods of rapid diagnosis of tuberculosis, leprosis, and DNA chips, and many DNA fingerprinting methods. He has been recipient of various prestigious awards. So I request Dr. Sir to please say a few words for our inaugural session today. Sir, to you, please.
Thank you very much. Respected Padam Shri, Dr. Madanji, Dr. Kamboj, Dr. Lohia, Dr. Ansari, and of course, Dr. Barkha, the people who really took a keen part in organizing this entire show. You know, there's a very mixed feeling because, you know, I'm very much a part of this family led by Dr. Lohia because since the time I've been here and associated with the ISSRF, it has been very close association with Professor Lohia. Dr. R.S. Sharma, of course, was my colleague in ICMR, so that's the entire regulatory affairs which were evolving. I think I was a part of that scene. So when I look back, you know, start with I think the part which today's morning address by Professor Madan, you know, what the scientific temperament is, scientific temper, scientific temperament, scientific, you know, the path, which can lead you anywhere actually, whether you trust in the Almighty or you don't trust. But anyway, if you have that novelty in your thinking and the perseverance, it leads to the outcome. So I think these 34 years of journey since Dr. Anand Kumar played the critical role in establishing this ISSRF, I think it has moved so well actually. I think very unique blend of the biologists, human and the veterinary medicine, you know, where the clinicians, the public health figure, the experimental people, everybody has been part of the story actually. And it's moving, it's moving so well actually. My main association, you know, started with the Dr. Chandrapuri's, you know, that inviting me to initial, I think 2008, I moved to Delhi, and that's the time in the very beginning, I think it was some of the meetings there. And then I started getting association with everything actually. Then I saw this strength actually, the strength, I think one of the very unique associations, societies, which integrates everybody. You know, people have excellence in science everywhere, actually, many places. But the tendency is to segregate, you know, dissect more and more, segregate, more divisions, more gaps. And this society, I think, still continues to be an integrated one. You know, there's a synergy there. You try to bring everybody together. Even in the organization part, you look at, you know, with the Professor Lohia now at the helm for quite some time. And with even his look at this, the co-chairperson, you know, Dr. Mittal, Dr. Suri, I think that when you look at Dr. R.S. Sharma, he's both the secretary and that's the other side. These all three represent the three blends. Same with the 8 I was looking at the 8 vice chairs actually. From north to south, east to west, they also represent the same actually. So a society which takes care of everybody, that's the clinicians, on the both sides, medical and the veterinary, looks at the public health people, looked at the biology, and looks at the application, and looks at the basic science. So I think this has a future actually. So this 34 years of journey has not gone in waste. I don't think anybody can say that the feeling of despair. I don't think it has not gone that way. It is moved much beyond the personalities. Personalities are important. They will continue to be the leaders, but it has gone beyond that actually. When you look today, I think the basic core of the controlling the fertilization, that to the best we can do for the human side or the veterinary side. You want that where you want to produce, you should. Where you don't want to produce, you shouldn't actually. So that has been the goal actually. So that science towards that in an ethical way, where there is best ethics practices for animals, best ethical practices for the humans, that has been the cornerstone. In vitro fertilization, artificial, in vitro fertilization, then producing the children, then access of the services, that continues there actually. The regulatory part also is moving well. Dr. Iss Sharma keeps on briefing me on that also. But no, the areas, I think that the gaps still remain quite huge actually. Polycystic ovarian syndrome has caught up the attention of almost every type of the stakeholder. But has it improved the therapy? Has it improved the outcome? Has it led to the better protection? It is moving towards that. So the progress cannot be negated. It cannot be said is insignificant. But huge gaps everywhere. I think there you need a bigger coalitions. People working on the environmental health, people working on the environment, people working on the biology, people who are working on the clinical side. Because suddenly the things have changed. In our lifetime, we have seen that such a huge, that number has increased tremendously actually. So I think those reasons, they pose a question actually. What we don't understand, why it is happening. And so there are speculative science, you can really have some you're looking at some part of the elephant, somebody looking at the tail, somebody looking at the other side. So that needs a better integration. So I think when you move towards maybe a year from now, next year from now, there's a time to build, I think, very stronger groups on environmental health and reproductive health focused on these issues. Certainly, I think the cell biology, certainly the in vitro fertilization, certainly that making it more and more perfect. They continue to be on the top agenda. Actually, they will always be there. But I think the other parts of the reproductive health, I think that you have to address. Then we're looking at the challenges like the COVID pandemic, which everybody has been in the grip of it. The people who perish, okay, that we really mourn them till we are here. But the people who are still on the ground, what is happening to them? What is happening to their reproductive health? What is happening to their mental health, which is complicating the reproductive health, and vice versa. So I think these all stories, though may not be that dominant a problem when they are competing priorities, but they have to be addressed by somebody. So I think as we move on, stay focused, as always this society has the privilege of being there, these leaders and the people who have led group of people, the top leaders, everybody has tried to make it on a focused platform. So I think I pray to Almighty because I trust in God, and that wisdom stays in our head, that we continue to investigate the science for the benefit in a very focused way, but then start looking at some things which are not seen by others. Some areas I'm not thinking of everything. I don't belong to reproductive health, so the things don't come to me automatically. Though I was a resident in gynecology long back in 75, 76, but that's an old story. So I don't. I'm not talking of that. But there, again, you find a problem. For example, a genital tuberculosis. The same story. Nothing. No new technology has made a difference. Molecular methods have come, but they have not made a difference. So why when their technologies are there, they don't reach the people who are where they should reach. So I think that applied aspects of certain areas which are ignored, they continue to be ignored because there are not many people. The sufferers can't push it. So I think that the introspection of the gap areas which are left untouched due to the fashionable science taking a dominance. So I think there should be groups to look at them. Reproductive health is a very broader area. So I think I take this opportunity this morning first to say not only thanks to Pastor Lohia, but because he has been giving me the opportunity, me, my wife, we have been going to the schools everywhere during the midterm conferences also to connect to the people. So with that connection, many of us can make a better effect. But I think that will be a cornerstone of, reaching out to the people, the knowledge reaching out to the people. But then generating more knowledge in a more focused way, I think that will always remain important. So greetings to all the people who are attending this morning inaugural session, all the people who have made a difference to organizing not only this conference, but the entire society. Then, the lifetime awardees, which are all eminent people today, meeting again legendary people like Professor Madan, Dr. Kamboj, again this morning, right in this morning session. And, then everybody who's contributing to the growth. We are not talking about many of them because there could be a huge number of names, at least out of the, I say 120 people who are logged in, much more than 50 are the prominent people in their area. So I think I greet each of them, greet all the people who are really delivering orations, who are making presentations. But then I think the responsibility continues to filter out, keep the people on the path so that the outcomes are visible in a very, I think, measurable way. Again, we keep that temperament, what Dr. Madan said, his remarks. They touched me this morning, that's the scientific temperament is the way to move for everybody who wants to practice it. Best wishes, greetings, and Jai Hind.
Thank you so much, sir. That was such a great talk and such a motivational talk. Thank you so much, sir, for being here and for your time. Thank you again. Now, with absolute pleasure, I would invite another eminent personality who is our guest of honor, Dr. VP Kamboj. Dr. VP Kamboj is Chairman, BCIL, Delhi. He is former Director of CSIR, CDRI, Central Drug Research Institute, Lucknow. This place has a very special connection with me as I started my research journey at CDRI being as a trainee. So yeah, I am really excited to see sir. Dr. Kamboj has many awards and distinctions in his name, such as Ranbaxy Award and Vigyan Gaurav Samman. I would request Dr. Kamboj to deliver the presidential address. Sir, to you, please.
Thank you, Barkha. Professor William Curtoch, former Director General ICMR and Secretary to Government of India, Department of Health Research, and currently NASI Chair in Public Health Research at the Rajasthan University of Health Sciences. Professor N.K. Lohia, the life behind the society. Professor M.L. Madan, I'll first congratulate him for getting the Padma Shri Award and for his eye-opening address in this morning. और मैं जो डॉक्टर सुरेंद्र शर्मा यूएसए, डॉक्टर ओम प्रकाश शर्मा भी न्यू जर्सी यूएसए एंड डॉक्टर एम.ए. अकबर शाह सभी को बहुत बहुत बधाई दूंगा फॉर लाइफ टाइम अचीवमेंट अवार्ड पाने के लिए।
धन्यवाद सर, धन्यवाद।
पार्डन, धन्यवाद। ओके, थैंक यू। बाकी भी जो लोग बोलने वाले हैं, मैं सब लोगों को ही बहुत बहुत आशा से देख रहा हूं कि सोसाइटी का जो ट्रेंड लोहिया साहब ने सेट किया हुआ है सभी लोग उसको फॉलो करेंगे। प्रोफेसर लोहिया, डॉक्टर एस अंसारी और बरखा खिलानी जो इन्होंने किताब निकाली है हमारे एब्स्ट्रैक्ट्स की और न्यूज़ लेटर्स की, वो बहुत ही अच्छी नज़र आ रही हैं और सभी लोग उसे पढ़ के एंजॉय करेंगे। ये वर्ष जो है हमारे देश के लिए बहुत ही खास वर्ष है क्योंकि ये 75th ईयर हमारा इंडिपेंडेंस का है और इसको आजादी का अमृत महा उत्सव के तौर पर मनाया जा रहा है। तो इसलिए और एक और भी खास बात इस वर्ष हुई कि जो हिपोक्रेटिस ओथ हमारे जो मेडिकल साथी हैं जब एमबीबीएस की डिग्री लेके कन्वोकेशन में जाते थे उसको लेते थे, तो इस वर्ष से जो है अब महर्षि चरक शपथ जो कि 2500 साल पहले थी और एसेंस वही है जो हिपोक्रेटिस ओथ ने बाद में कैप्चर किया। लेकिन इसका जो एक खास फीचर महर्षि चरक शब्द का है कि कोई भी डॉक्टर जो है वो मरीज को जब ट्रीट करेगा तो पैसे के भाव से नहीं करेगा, और दूसरा जब लेडीज को ट्रीट करेगा तो प्राइवेसी मेंटेन करेगा, और हर डॉक्टर जो भी ट्रीटमेंट देता है जिस मरीज का उसके बारे में प्राइवेसी मेंटेन करेगा। तो ये जो है दो इवेंट बहुत ही खास है जिस साल में हम मना रहे हैं। तीसरा, आप सब लोग को ये पता होगा कि नेशनल फैमिली हेल्थ सर्वे का जो है रिपोर्ट दिसंबर लास्ट ईयर में आई, और उसमें भी बहुत ही अच्छी बात हुई है कि हमारा जो फैमिली प्लानिंग हम लोगों ने 1952 में रिकॉग्नाइज किया था, इंडिया वाज द फर्स्ट कंट्री, और उसमें भी जो हमारा अचीवमेंट हुआ है, अब जो हमारा रिप्रोडक्शन रेट है 2 हो गया है, जो रिप्लेसमेंट लेवल फर्टिलिटी 2.1 था उससे भी नीचे है, और ये कह रहे हैं कि 30 और 31 to 35 के बीच में ये 1.73 हो जाएगा, कि हमारी चिंता फिर बढ़ जाएगी कि बच्चे ज़्यादा बढ़ाने के लिए ना कि कम करने के लिए। जो गोल देश ने सेट किया था वो आज़ादी के अमृत महोत्सव में आके वो हमने अचीव कर लिया कि उससे भी नीचे आ गए। अब चिंता होनी चाहिए कि इसे कैसे बढ़ाया जाए। और दूसरा, हमारे देश में ड्रग रिसर्च में इतनी ज़्यादा उन्नति हुई है, यदि आप देखें कि आजकल जो है इंडिया को डिस्पेंसरी ऑफ द वर्ल्ड कहते हैं, यानी 20% ड्रग्स जो है वो इंडिया कंट्रीब्यूट करता है वर्ल्ड में। लेकिन आपको जान के हैरानी होगी, इसमें से 45% जो है वो अमेरिका अलोन कंज्यूम करता है, और टोटल जो ड्रग प्रोडक्शन है वर्ल्ड में, और 35% जो है वो यूरोप कंज्यूम करता है। डेवलपिंग कंट्रीज जो है उनके लिए सिर्फ इंडिया के लिए 1% है, बाकी सारी कंट्रीज के लिए 9% की रहती है। हम लोग देश में आयुष को बहुत बढ़ोतरी दे रहे हैं, लेकिन टोटल अगर आयुष की आउटपुट देखी जाए तो 1000 करोड़ के बराबर है, और जबकि हर्बल मेडिसिन की इंटरनेशनल जो है ग्रोथ आज के कल जो है 100 करोड़ है, और 14% के लेवल से बढ़ रही है। हमें ये नहीं पता कि इंडिया आयुष मेडिसिन का ओरिजिन और हर्बल ड्रग्स का ओरिजिन होने के बावजूद भी जो 1000 करोड़ की सेल है वो सिर्फ इंडिया में है, उससे बाहर नहीं है। और इसका जो कॉन्सिक्वेंस हुआ है ड्रग्स का, ये हुआ कि जो हमारी लाइफ एक्सपेक्टेंसी अगर आप 47 में जब देश आज़ाद हुआ था उसमें जाए तो 32 ईयर्स थी, जिसमें मेन की 29 थी और लेडीज की 33 थी। तो 32 ईयर्स जो होता है लाइफ एक्सपेक्टेंसी थी, और जब ये अब जो है 70 हो गई है। और बाकी जो देश, हालांकि हमारे से 10 साल आयु में सारा, और जापान 15 साल ज़्यादा 85 ईयर्स है, और बाकी जो यूरोप के और अमेरिका है वो 75 ईयर्स 75 to 78 ईयर्स है। और जो हमारी इन्फेंटेड मोर्टेलिटी थी जो जब हम आज़ाद हुए थे 270 थी पर 1000 लाइव बर्थ पे, और अब वो कम होके 113 हो गई है, और जबकि कुछ स्टेट्स में 43 लोएस्ट है। और ये जो ज़्यादा है ये जो हमारे देश में जिसे बीमारू स्टेट्स कहते हैं, बिहार, मध्य प्रदेश, उत्तर प्रदेश और राजस्थान, इनकी वजह से ये 113 है, वरना तो ये और नीचे आ जानी चाहिए थी। और इसमें यूपी की सबसे ज़्यादा है, वो 197 है। क्यों? ये इसलिए है कि अभी तक हम जो है इन्फेक्शस डिजीज पर कंट्रोल नहीं कर पाए जितना हमने, हालांकि देश ने बहुत अचीवमेंट किया है। इसके साथ मैटरनल मोर्टेलिटी जो है हमारी, वो भी जो पार्टीशन के टाइम पे थी 270 थी, और अब कम होके वो 20 के करीब आ गई है, जबकि वर्ल्ड ओवर बाकी जो डेवलप्ड कंट्रीज है वहाँ पे 8 है। तो उस पर भी हमें अभी बहुत आगे करना है। और देश ने कई डिजीजेस जो नई वैक्सीन वगैरह आने से एलिमिनेट हो गई है, स्मॉल पॉक्स 78 में एलिमिनेट, पोलियो 2015 में हो गई, डिप्थीरिया, रुबेला ये वर्चुअली कंटेड है, प्लेग भी वर्चुअली कंटेड है, मैटरनल मॉर्टेलिटी जो है मिनिमाइज है। तो अब ये देखिए कि देश ने इन सबको, और अभी तो आप सब ने देखा है कि जब कोविड-19 की वैक्सीन आई तो इंडिया सबसे फोर फ्रंट पे था। हमारी 7 वैक्सीन जो अंडर डेवलपमेंट थी, जिनमें से 2 तो देश में इंट्रोड्यूस हो गई हैं। एक तो कोवैक्सीन है जो कि इनएक्टिवेटेड वायरस है, और दूसरी जो है वो डीएनए, दैट इज द फर्स्ट वर्ल्ड वैक्सीन इन द वर्ल्ड, डीएनए कोविड-19 की वैक्सीन है। और अभी जो मैसेंजर आरएनए की वैक्सीन है वो भी फेज 3 क्लिनिकल ट्रायल में चली गई है। और कल के न्यूज़पेपर ने आपने पढ़ा होगा कि ग्लैंड फार्मा ने नीजल स्प्रे डेवलप किया है, जो कि जब वायरस नाक में होता था उसे एक दफ़ा स्प्रे करने से तो वायरस, वो नाइट्रिक ऑक्साइड का है, उसकी डिस्कवरी हालांकि कनाडा में हुई थी, लेकिन पहली जो वैक्सीन बनी, ड्रग बनी है नेजल स्प्रे, वो इंडिया में है। और वो कहते हैं 99% तक 24 आवर्स में वायरस को एलिमिनेट कर देती है एक सिंगल डोज लेने की वजह से। तो अगर ये सारी जो चीज़ें अचीवमेंट हुई है, वो ड्रग जो रिसर्च जो है, मैंने आपको कहा बताया कि इंडिया जो है 20th डिस्पेंसरी ऑफ द वर्ल्ड है, 20% ड्रग्स इंडिया कंट्रीब्यूट करता है, और चीपेस्ट से, जिनकी जो 3rd लार्जेस्ट कंट्रीब्यूटर है ड्रग्स के लिए और 13th बाई प्राइस वाइज है। यानी कि गरीबों के लिए जो इंडिया दवाई बनती है वो बहुत ही अच्छी है। और वैक्सीन में इंडिया इज द लार्जेस्ट प्रोड्यूसर ऑफ वैक्सीन इन द वर्ल्ड। दुनिया में 3 बच्चों में से 2 जो है वो भारत में बनी हुई वैक्सीन उनको मिलती है। और यूएन जो है 70% यूएन की सप्लाई इंडिया मीट करता है, और 130 देशों में इंडिया में बनी हुई वैक्सीन जाती है। आपने देखा होगा जब ये कोविड-19 की वैक्सीन आई तो हमारे देश में ही कई साइंटिस्ट ने और कई नेताओं ने ये डाउट किया कि इंडिया की वैक्सीन जो है वो उतनी अच्छी क्वालिटी की होगी कि नहीं होगी। लोगों को ये नहीं पता कि दुनिया की 130 देशों में भारत में बनी हुई वैक्सीन जाती है, और यूएसए अप्रूव्ड जो एफडीए अप्रूव्ड प्लांट्स हैं इंडिया में, नेक्स्ट टू यूएसए है, इतने ओवर 100 प्लांट्स है। तो बेस्ट क्वालिटी मेडिसिन जो है इंडिया में बनती है इस एरिया में। तो इन्हीं चंद शब्दों के बाद मैं इसके साथ में डॉक्टर लोहिया साहब का बहुत धन्यवाद करूँगा, जो सोसाइटी को बहुत मेहनत से आगे बढ़ा रहे हैं, और उन्होंने मुझे इस में बोलने का मौका दिया। धन्यवाद लोहिया साहब, और धन्यवाद एवरीबॉडी।
Thank you so much, sir, for such a great talk. It was so informative. Thank you so much, sir. Now I would invite Professor A.S. Ansari, convener ISSRF 2022. Dr. A.S. Ansari is a professor at Center for Advanced Studies, Department of Zoology, University of Rajasthan. Dr. Ansari has outstanding research publications and many awards and accolades. I would request Dr. Ansari to give vote of thanks to all our dignitaries. Over to you, sir, please.
Thank you, Ms. Nidhi. Honorable Dr. William Curtoch, Nasi ICMR Chair, RUHS Jaipur, former Secretary, THR, Minister of Health and Family Welfare, and ICMR New Delhi. Dr. Padma Shri Professor ML Madan, guest of honor, and Dr. Sri Kamboj, our presenting address of the inaugural function, and Professor Lohiya, Dr. Jb Sarma, former Senior Director General ICMR and medical scientist, and Dr. Bakha, Organizing Secretary and District guests and delegates. On behalf of the organizing committee, it is my proud privilege to extend a hearty vote of thanks on the occasion of the inaugural function of the virtual international conference on reproductive health and 32nd annual meeting of the Indian Society for the Study of Reproduction and Fertility. First of all, we are utmost grateful to Dr. William Curtoch, who, in spite of various commitments of immense importance and busy schedule, very kindly spared valuable time to address the inaugural function. His words have always been a source of inspiration to the scientific community of the country. Thank you, sir. We are also grateful to Dr. V.P. Kamboj for his discourse on current reproductive health challenges and preside over the inaugural function. We are equally grateful to Padma Shri Professor M.L. Batan for sharing livestock biotechnological advances to cope with the national needs. We are grateful to Dr. Iss Sharma for introduction of lifetime achievement awardees and their accomplishments. We are extremely grateful to keynote speakers, orators, invited speakers, young scientist awardees for sharing their latest scientific findings on different aspects of reproductive health care research. I would take this opportunity to express my deep regards to Professor N.K. Lohia for being a constant source of motivation and guidance force in our endeavor and efforts to excel this virtual conference. We are heartily thankful to dedicated entire organizing team, especially Dr. Barkha Khilwani as an organizing secretary and Ms. Nidhi for anchoring the whole proceeding. Colleagues, PhD scholars and students who worked very hard in making this virtual conference for a grand success. In the last but not the least, we are grateful to all distinguished guests and delegates. Without team, this conference would not have taken place. Once again, I thank one and all who contributed in the successful organization of the conference. Thank you so much. Jai Hind.
Thank you. Thank you for acknowledging all of us. So before we move ahead to our oration session, before we move ahead to our oration session, I would request all our dignitaries and all our participants to please switch on camera for a minute so that we can click a screenshot. Thank you in advance. Thank you, everyone. So now we will move ahead with our first oration, which is Founder President Dr. T.C. Anand Kumar Memorial Oration. And for this oration, our chairpersons are Dr. Sunita Mittal, who is Director and HOD, Gynecology Department, Fortis Institute, Gurugram. Dr. Mittal is considered as one of the best gynecologists in Delhi and Gurgaon. And the other chairperson is Dr. Anil Suri, who is Professor of Eminence, National Institute of Immunology, Delhi. Dr. Suri is an eminent cancer researcher working in the field of translational cancer research. I think Dr. Suri couldn't join us because he has some other commitments. So I request Dr. Mittal to please chair this session. Over to you, ma'am, please. Ma'am.
Please unmute yourself.
Dr. Sunita, ma'am, you have to unmute yourself, please. I think there is some technical issue. I think she is joining again. Sorry for this disturbance. I think Dr. Mitchell is unable to join, so I would request Loya sir to please do this oration.
It's a great pleasure to invite Professor Jb Sarma for this very important, prestigious oration. And we all know Professor Sarma is currently based at All India Institute of Medical Sciences and Professor of Upstream Gynecology. And earlier he was at Maulana Azad Medical College. And he has been awarded with Dr. B.C. Roy Award by Honorable President of India in 2015. And around 350 publications in index journals, edited 2 books, and editor-in-chief of Upstream Gynecology today, and chief editor of Indian Upstream Gynecology Journal of Pediatrics and Obstetrics and Gynecology, on editorial board of 6 journals, and around 200 papers being presented and chaired number of national and international conferences. And with this brief introduction, I have great pleasure inviting Professor Jb Sarma to deliver the oration.
Yeah. Thank you very much, Sir Loya, sir. And I'm at the outset, I thank the organizer, that is Dr. Loya, Dr. R.S. Sharma, to award me this prestigious oration. It was my dream, and it has been made true. And dignitaries on the dais, I am really privileged, and especially Dr. T.C. Anand Kumar, who was such a famous person, first president of ISSRF, and he was the second, I think, person to do IVF in India, and is a great luminary in reproductive biology. So I'm really, really honored to have his oration. And my topic, and thanks Dr. Loya sir for your kind introduction. So, and Dr. R.S. Sharma, and I also congratulate the lifetime awardees. So my topic will be role of female genital tuberculosis in female infertility, Indian perspective. Next slide, please. So that I'm on time. Yeah. So we know that tuberculosis is a major public health problem, and next slide, please. And we'll be dealing mainly with the female genital tuberculosis. Okay. And so the first case was described by Morgagnin as early as 1744, and ever since we have been seen different types of tuberculosis. And Robert Koch discovered the bacillus in 1884. Next, please. Yeah. So epidemiology wise, basically, it is many patients of female genital tuberculosis are asymptomatic cases. So that's why sometimes diagnosis is underdiagnosed, basically. And it affects almost 50% population in third world countries. And in India, we see almost 10 to 16%. At AIMS, we see almost 16% to 18% cases of infertility are caused by tuberculosis. Next, please. So yeah, so that's why, female genital tuberculosis is a type of extrapulmonary tuberculosis, and almost 10% cases of extrapulmonary tuberculosis are because of it. And as I said, 10 to 16% cases at in AIMS slightly higher, that FGTB causes infertility. Next, please. Yeah. So basically, the prevalence of infertility in case of infertility clinic of TB is only 1% in USA and Scandinavian countries, but like in South Africa, 4 to 8%. But in India, it varies, and usually the average is 10 to 19%, higher in India, in AIMS, etc., where we get patient referred from Bihar and Jharkhand, etc., where the prevalence is much higher. Next, please. Yeah. Next slide, please. Okay. So that's why I like tuberculosis FGTB is a very, very important cause of infertility, and we need to get better diagnosis. Dr. Curtoch had done a lot of work. Next slide, please. on the molecular test for this thing, but still as Dr. Curtoch also said in his inaugural address, that diagnosis may remain difficult. Next slide, please. Yeah, can someone keep on changing slides? Slide is not moving. Yeah, thank you. So basically, like, can my slides be shown? So basically, tuberculosis, female genital tuberculosis remains an important cause of infertility in India, almost 10 to 18% cases. And for that, early diagnosis and treatment is mandatory so that we can prevent the patients from going into permanent sterility. So can I have the slides, please? I can't see. Dr. Barkha, can my slides be shown, please?
Yes, sir. We are very sorry. We had some internet issues.
It's all right. So yeah, now I can see. Yes, definitely. So we know that most of the cases are caused by Mycobacterium tuberculosis, the human form, but some cases Mycobacterium bovis, etc., can also cause, and rarely the others rare causes. And bovis is important in rural areas where people may drinking milk directly without boiling, and there can be udder tuberculosis in the udders of the animals. So that may be the cause in the village areas. Next, please. Yeah. Next slide, please. If someone can keep moving the slide so that I can finish on time. No? Yeah. Yeah. So this is again the natural history of tuberculosis. All of us knows. I won't go into detail of that. Next, please. Yeah. Next slide, please. So basically, yeah, again, next slide, please. We have already known it. So basically, like, we know the primary genital tuberculosis and secondary genital tuberculosis. That means from the very beginning, patient had got genital tuberculosis. Usually, genital tuberculosis tends to be secondary. Like the patient, so that's why detailed history is very important. Past history of tuberculosis in the family or in the patient. So patient had, that may have been asymptomatic also or subsymptomatic. Like patient had pulmonary tuberculosis, patient had lymph node tuberculosis, and now she develops the genital tuberculosis. That's the most common cause. But very rarely from the infected male partner also the tuberculosis can be there, and that tends to be the vulval tuberculosis, cervical tuberculosis, etc. But most cases, almost 90 to 95% of the FGTB are secondary TB, secondary usually to the pulmonary TB or sometimes extrapulmonary TB. Next, please. Next, please. Yeah, next, please. We know this slide. Yeah. Okay, so the mode of spread most common is the hematogenous spread, basically, but it can be sometimes lymphatic spread and direct contagious spread from the GIT. And the sites involved, this I want to highlight, the fallopian tube are involved in almost 95 to 100% cases, uterus in about 70%, ovaries about 20 to 30%, cervix 5 to 15%, and myometrium 2 to 5%, vagina vulva rare 1 to 2% only. Next, please. So this is important because fallopian tubes are usually blocked by them. Yeah, and this is basically the various pathogenesis, just like tuberculosis, poverty, malnutrition, overcrowding, poor ventilation, diabetes, illicit drug use, kidney ailment, HIV infection, especially in African countries. HIV is the co-infection, and now the COVID also is important for us. So patients of COVID can also develop tuberculosis. Next, please. Yeah. Next slide, please. So then the female genital tuberculosis basically it always secondary to pulmonary tuberculosis, but rarely because of extrapulmonary TB also, like lymph node TB or abdominal tuberculosis. Next, please. Yeah. Okay. The symptoms of genital TB. So this is what I want to highlight. Like in case of pulmonary TB, patient will mostly will be symptomatic, like cough, sputum, etc., and hematemesis, but hemopsis. But in case of genital TB, almost 10 to 12% patient may be asymptomatic. Then they may have mild symptoms like slight rise of temperature, slight malaise and all. But then they can have menstrual symptoms. Like initially in the early stage when there are ulcerative lesion, patient will have menorrhagia, but later on oligomenorrhea, hypomenorrhea, or amenorrhea can also occur. And sometimes patient may have abdominal mass also. And then infertility is the most common symptom or sign of that. Next, please. Yeah. So next, yeah, same way, the patient 10 to 12% patient there may not be any signs, but the signs can be like the patient has associated pulmonary TB, so you can have rales or rhonchi in the lungs. Some patient have lymph node tuberculosis, then you can feel the lymphadenopathy or some other features same way. And when we do abdominal examination of these patients, there can be lump in the abdomen, lower abdomen, sometimes like feeling, maybe doughy feeling can be there. And when we do vaginal examination, patient may have abdominal vaginal discharge, patient may have sometimes ulcerative lesions or lesion in the cervix if there is a cervical tuberculosis. tuberculosis, which may masquerade like carcinoma also. And but usually they may be enlarged uterus, there may be pyometra, there may be Ovarian masses on pelvic examination, but some patient may be asymptomatic. Next, please. Yeah. Next slide. Yeah. So the diagnosis, I mean, so what is important is that TB mindedness is very important. So index TB guideline, I was part of the chair from, in this in EPTB, Dr. Jb Sarma was one of the leading person, and Dr. R.S. Sharma also contributed. So in this case, basically what we teach, the past history of tuberculosis is very important. And same way, like, past history of ATT intake, and then the, is important. Next, please. So we have to be TB minded, then only we can detect. Next, yeah. So who should be investigated? Basically, any patients, especially in India, all infertility patients, we always investigate them to rule out or rule in the tuberculosis. Same way, any past history of tuberculosis, family history of tuberculosis, and even in the past, many of my patients may not disclose the history, but in the childhood or adolescent, when the girl was adolescent, she might have looked after dying grandparents who died of pulmonary TB. And in India, it's a custom that adolescent girls look after their, grandfather, grandmothers, and that may make them actually infertile. I have seen many such girls in my practice. Next, please. So that history you have to take in, patient may not disclose that history in front of the husband and of the tuberculosis. So we have to take in confidence, and that is very important. And now Dr. Curtoch already said this. So the most important single test we do is endometrial aspirate or biopsy, especially because most patients are infertility. But menstrual blood can be taken, but doesn't give very good results. But in case of unmarried girls who are sexually non-active and you are suspecting, then you can do endometrial blood, like menstrual blood also, but the results are poorer. So we should always try to do endometrial aspirate or biopsy with the Carmen's cannula number 3 to 4, and single most important investigation. And we usually perform it in the luteal phase, that means premenstrual phase. So patients should have abstinence in that cycle, otherwise we can interrupt the pregnancy. And we take the sample, we put it into the saline, and then we send the sample for AFB microscopy, AFB culture, and then we take the white part of the specimen that we do the gene expert on that, and you can do LEMP also. Then we do PCR on that, and then LP can be done, and then we take a formalin sample, especially the white tissue, and then we look for any epithelioid granuloma. Having said that, epithelioid granuloma, because the menstrual there is a menstruation and shedding of the endometrium, so there may not be enough time for the proper granuloma to be formed. So local lymphocytosis can also be shown in that. So this is very important. Next, please. Yeah. So endometrial biopsy, or sometimes peritoneal biopsy can also be taken. And then we do this NET, that means CB NET or gene expert, is a very, very important. PCR, we don't though PCR alone, we don't take as a method of diagnosis, but that can show that patient is screen positive. Then we can do the further testing on the patient on laparoscopy or histoscopy. So like if the patient is AFB microscopy positive, AFB culture positive, gene expert positive, or histopath epithelial granuloma positive, then we take it positive TB and we start treatment. But if only PCR positive, then we do further test on the patient, either ultrasound, CT scan for any Ovarian masses, or we do histoscopy and laparoscopy to see any changes of the TB changes. And then if the patient is PCR positive infertility with some change on laparoscopy, we take it as a probable TB and we treat the patient. Next, please. I'll be showing the slides. Next, please. Yeah. So same again, we have already done that. Yeah. So AFB smear and culture. Next, please. Culture may take 6 to 8 weeks. And the results are actually, we don't pick up many cases. Then backtech culture you can do, MGIT culture you can do, and MB back. Next, please. Yeah. Next slide, please. Yeah. So if the slide, yeah. So the advantage is that the rapid culture method, we can get better pickup rate and all. This is the PCR basically can be false positive. So alone we don't take it as a method to start the treatment. Okay. Next, please. Yeah. Okay. Yeah, we have done it. Next, please. Okay. Yeah. So microbial confirmation, we should try to do it, but not always possible. And because if you depend only on AFB microscopy culture, and we pick up only 20 to 30% cases, we miss 60, 70 to 80% cases. But if we do PCR, depend on that, we pick up extra 200% cases. So that's why we have to keep a balance. Next, please. Yeah. Okay. So these are the again tests. I mean, we have already done that. Yeah. Next, please. So okay. So we do either G. Nielsen staining on that. Yeah. And drug sensitivity testing should be tried if possible, but unfortunately, being possibly disease, I mean, sometimes it's very difficult to do positive culture. So drug sensitivity testing will be difficult, but should be tried if possible. And I have seen actually about 4 cases of MDR tuberculosis by drug sensitivity testing in patients. So we are getting MDR TB also in genital TB. Next, please. Okay, yeah. So the same thing we have already done that. So ultrasound, hysterosalpingography, CCT, MRI, PET, not to be done in all cases, but where you are suspecting tuberous masses. And endoscopy, laparoscopy, histoscopy, I said the patient needs infertility diagnosis and the patient who is PCR positive. So the patient is like AFP microscopy culture, gene expert or histopath positive, then we don't have to do any more tests, we straight away start treatment. But if only PCR positive, then we can do either ultrasound, CT or etc. depending on TO masses present, or we do laparoscopy, histoscopy, which pick up many percentage of cases. Next, please. So this is, we could use the term composite reference standard for that. Next, please. Yeah. Next slide, please. Yeah. So gene expert is a very reliable test. Okay. And then CBNet, same thing basically. So next, please. It can pick up, give the results within few hours, and it also tells us about rifampicin resistance. So that's why CBNet is a very, very important test approved by the WHO and Government of India also NTEP program. Next, please. Yeah. Next slide, please. So we do the gene expert for all of our patients. Next slide, please. Yeah. Those. Yeah. Same thing. Next slide, please. So these are the LPA and LEMP tests we did in our lab. We have a studies ongoing with the Ministry of Health funding. Next, please. So these are some of the patients who were LEMP positive in our study. Yeah. Next, please. LMA LAM is done on the urine of the patient. Again, we have to doing a study on it. So this is the basically. Yeah. Yeah. Next, please. So LEMP testing. That's also test still being subjected to the test. Yeah. So these are the very rapid diagnostic tests. Basically, I have already highlighted them. Next, please. So mainly we do basically culture and the MGIT culture or bacterial culture, and then we do gene expert. These are the main tests which we do. And this is the study which we published in the European Journal of Gynae, and we found about 30% sensitivity of this thing, almost 100% specificity of gene expert. Next, please. Yeah. Next slide, please. Okay. Yeah. So as far as radiology is concerned, it is useful mainly their tuberous mass. Like in this patient, we can see a tuberous mass, and that. But again, unfortunately, the ultrasound or CT scan, MRI, they can't tell you whether it is definitely. They can't tell you it is tuberculosis or malignancy or this thing. But they can highlight if the patient is like infertility, young patient, so less likely to be malignancy, and the patient has got like infertility, then PCR is positive. So then this TO mass, I'll label it as tuberculosis. So that's why that 2-3 things we combine, we call this composite reference standard. But alone, like any tuberous mass on ultrasound, CT, MRI, cannot definitely say it is tuberculosis. So in addition to other tests, we make the diagnosis. Next, please. Yeah. Next slide, please. So I'll just, yeah, this is the CT scan again. We can see that TO masses on CT scan, MRI, or we can see the lymph node. And next, please. So along with PCR positive infertility, this finding will suggest tuberculosis. So this is the study which we just published in, in the Journal Tuberculosis of CT scan. Next, please. Next slide, please. So we have done studies on MRI, we have done study and this PET CT published in European Journal last year. And the beauty of PET CT is that, even after the giving ATT, the mass sometimes may persist. So if on PET CT, it shows positive uptake, that means we are dealing with the MDR or patient is still persistent disease. If it's just a like mass, but without any increased uptake, then it is just a like enlarged lymph node or something like that. Then laparoscopy and hysterosalpingography or this thing, can be combined to each other. Next, please. And next slide, please. Yeah. So HSG usually we avoid in doing these patients, but like many times we do HSG without diagnosis of TB, and then we can get the findings. I'll just show you some slide. Next, please. Yeah. Next slide, please. So so they can be like beating of the tube, they can be blocked tube, they can be dry tree branch sign. You can see the whole uterus is just shrunken and tubes are also shrunken. Next, please. So these are the various advanced findings. Unfortunately, these will be the very advanced cases where infertility treatment will be poor. Then laparoscopy is a very good modality and it can pick up many more cases because we are directly visualizing the tubes, ovaries, and peritoneal disease. So someone can quickly show me the slides next time so we can quickly do the next, please. Yeah. Next slide, please. Okay. So it's a abdominoperineal tuberculosis may masquerade like malignancy also. So the laparoscopy can help in that. Next slide, please. So it's an important cause of that. And yeah, so these are the various findings. I will be just showing you the slide, like dilated tubes, hydrosalpinx, pyosalpinx, BD tubes, and various adhesions. You can see the, like this tube is distended, tube is hyperemic. Alone, this is not like, this hyperemia of the tube can be seen in chlamydia also, but positive is they have the cages nodules on the peritoneum. This is very specific of tuberculosis. Next, please, just keep showing slides, please. I just want to show, yeah. So this is the adhesions. I mean, various perihepatic adhesions, abdominal adhesions. They are not specific of tuberculosis, but with positive PCR infertility, young patients, so they usually show tuberculosis. Next, please. And the adhesions are thick actually. So this is the perihepatic adhesions. I published study on it, and these are seen in case of tuberculosis, almost 45% case of genital TB and abdominal TB. You can see the perihepatic adhesion. Next, please. And along with that, you can see the cages tubes are very much congested. Tubes are just like calcified. Next, please. This is a very advanced case of tuberculosis, and hydrosalpinx can be there. Next slide, please. Yeah, again, cages nodules on the peritoneum. Next slide, please. The abdominal TB. Again, here you can see the, I have seen 22 patients have published that also, that it can coexist with endometriosis. Normally, we make the types of endometriosis, we rule out tuberculosis and vice versa. But in India, the two things are so common. So this patient had both proven tuberculosis and endometriosis. So they can coexist also. Next, please. Next slide, please. Yeah. Okay. Yeah, again. So hydrosalpinx, next slide, please. Again, this you can see distended tube, and yeah, in this case, you can see the cages material being like as a white carpet has been laid on the uterus and fallopian tubes. So this is very specific of tuberculosis, tubercles and cages nodules. Next, please. So quickly, some of the slide can be shown. Yeah, and this is the sign I discovered, that hanging gallbladder sign. So gallbladder looks like, because liver gets adherent to the anti-abdominal one diaphragm, so gallbladder looks as if it's hanging up like this. So it's a sign I have described in tuberculosis. Next slide, please. So hanging gallbladder sign can be seen in tuberculosis. Next slide, please. Yeah. Next slide, please. So this is because of perihepatic adhesion. Same way, other abdominal adhesion can be there. So if the slide can be shown, please. Next slide. So same way, I have described other signs on abdominal tuberculosis, like compartment sign, and a blue python sign. Can some of the slide be moved, please? The same slide is showing. Dr. Barkha. Okay. So, I mean, so laparoscopy is a very, very good modality. The slide can be shown next page. So, in which we can see, like, perihepatic adhesions, abdominal adhesions, compartment sign, like, the compartment sign basically is in that case, though, like the omental adhesions are formed. So, that tries to basically accommodate the infection in one compartment, so that there is not generalized peritonitis. So that is basically body's protective mechanism. So I have observed this sign in case of the abdominopelvic tuberculosis. Same way, I have seen sign in which the both fallopian tubes are distended, and they look like, when you put the dye, they look like a blue python. So it's a blue python sign. And I also described that sigmoid colonic adhesion band and ascending colonic adhesion, like the lower 2/3 and upper 1/3 junction, there is a band on the tube. So these are the various signs which I observed in case of abdominopelvic tuberculosis, and I published them in PubMed Index Journal. So so that's what I feel like Dr. Curtoch has also said, that doing work on tuberculosis, where the problem is Indian, so we can get more findings, because Americans and research will not work on these findings. So can I see the slides, please? I can't see the slides. So I mean, so doing a like laparoscopy and histoscopy on the patients of genital tuberculosis is a very, very useful method, and it helps the patient as well as the clinician to pick up early signs of tuberculosis, so that patient can be diagnosed and treated on time. Dr. Barkha, can I have my slides, please? I can't see. Yeah, okay.
Yes, sir, we are sharing.
Okay, yeah, that's all. So sorry for that. Yeah. So this is another sign I described. So you can see the both fallopian tubes are fused to each other, and that is called Sarma's kissing fallopian tube sign. Then this is the adhesive band, which is seen on the lower 2/3 upper 1/3 of the and ascending colon. Next, please. Next slide, please. If they can be quickly moved. So I want to show the more slide. Yeah. And this is the, that it's important to look the whole of abdominal cavity. This is the, basically, you can see the fallopian tube look like a dried tree branch sign. So this is the on laparoscopy as well as on histoscopy. So this I described, there's a tree standing near IIT Delhi. So it resembled that. So I gave this sign, dried tree branch sign, because of the calcified tubes. So these are very advanced patients. So this, again, we have published studies on, like, next slide, please. I want to show the pictures. So that the so this is called parachute sign. If you see the ascending colon is adherent to the anti-abdominal wall like a parachute, and so this is also seen in tuberculosis. And this is important because while doing laparoscopy, you can see the cages nodules on this, so it's a ascending colon. And this is important because we can injure this also while doing laparoscopy. So you have to be very careful in these patients because bowel injury is more common. Next slide, please. So this is the, Sarma's parachute sign I given, published in the journal. And here the adhesive band on the basically sigmoid colon. There is an adhesive band. That's also I described. Next, please. Next slide, please. You can quickly move the slides so that we can finish on time. I don't want to encroach on the time. Next slide, please. Okay. So these are the various, the compartment sign I have already said. And then this is the TB algorithm basically we can develop. Next slide, please. I published lot of studies on this. So this is the histoscopic finding. Again, on histoscopy you can see the pale looking cavity and you can see the adhesions here in this thing. So they are not specific of tuberculosis, but with positive PCR with infertility and these pale endometrium tubercles there, or cages nodules, or this adhesion, so they are they are suggestive of tuberculosis and we should start the treatment. Next slide, please. So usually laparoscopy, histoscopy we do together under general anesthesia. Again, you can see that bands there, adhesive bands in the endometrial cavity. Next, please, along with PCR. So same thing, and then on ultrasound finding, this is the paper recently published in Human Reproductive Sciences PubMed Index journal. So you can see along with Dr. Ashok Khurana. So you can see here the various findings will be there. Same way that this is the Gene Expert study which we published in European Journal of Gynae, funded by Government of India. So Gene Expert had 30% sensitivity and about 100% specificity. So this is the basically, so we should do more laparoscopy on these patients and then we can pick up more cases by laparohisto. Next, please. So this is what I want to highlight, that no single test, like if you do alone microscopy culture, you can pick up only 20 to 30% cases. But if you combine microscopy, culture, Gene Expert, LEMP, and then PCR, laparoscopy, histoscopy, ultrasound or CT. So by the combination, we can pick up almost 80 to 90% case of tuberculosis. So this is called composite reference standard, which we have then. And laparoscopy, histoscopy, but should be done by experienced person because there are more chances of injury to the various organs as we have observed in these patients because of adhesion. So it should not be left to the junior resident or senior residents. Next, please. And the laparoscopy, histoscopy can also suggest us and these are the various basically new tests. And next, please, the basically National Tuberculosis Elimination Program. Next slide, please, quickly, if you can move. Okay, next, please, again. So, NTP, next slide, please. So, National Tuberculosis Elimination Program. Next, please. Okay. Next, please. Okay, so this I want to highlight that now the name is the National Tuberculosis Elimination Program, not RNTCP anymore. And this is basically along with WHO, we use the treatment. Next slide, please. I just want to show the slide of the treatment. Next slide, please. So next slide, please. So basically, what I want to highlight that we give very simple, there been changes. We give RHZE, rifampicin, isoniazid, ethylbutol, and paraginamide for 2 months daily, not thrice weekly, daily under DOTS regimen. That means directly observed treatment strategy is free of cost by Government of India. Patient gets money also. So please, they should get the treatment from Government of India, pre-hospital, rather than buying from the market. The drugs are quality assured drugs and free of cost. Plus patient gets 500 rupees a month for food also. Okay, and then so first 2 months is the basically RHZD for 2 months, then RHE for next 4 months. So we stop Z like that. So next slide, please. So this I want to highlight. Total treatment is for 6 months, 2 months RHZD, and then 4 months RHE. Daily treatment under DOTS, free of cost from Government of India, plus 500 rupees free of cost from for the food, etc. for the government. And we should give it should be basically communicated to the government through NICSA program. So it is a duty of the doctor to inform the government. Next, please. And next slide, please. Monitoring should be done all the patient because surgical treatment very rarely is required, because even if we do laparoscopy, this is the patient I did laparotomy. You can see the cages material coming out, but everything was so jumbled up. So I just took biopsy, came out. Don't try to do surgery because there is a chance of injuring the bowel, and in any case we have to give AT to this patient. But just I want to highlight this patient. I opened her thinking to malignancy, but turned out to tuberculosis. So I closed the abdomen and gave her treatment for tuberculosis. Next slide, please. And then these patients are really helped by assisted reproduction. So this is important. After treating the patient with tuberculosis, anti-tuberculosis therapy, then the tubes are usually blocked. So I repeat the laparoscopy. If the tubes are open, they can try conception. If tubes are closed but endometrium is fine, they go for IVF ET. And if the tubes are also blocked and endometrium also gone, then they go for surrogacy or adoption. So this is the index TB guidelines are there to guide us for that. Next, please. Next slide, please. So I'm almost over. Yeah. So this is my last slide. So take home message that genital tuberculosis is an important cause of infertility in India, and we should basically think about it. And early diagnosis and treatment can help the patient from developing permanent sterility. So we should be TB minded, and we should use the composite reference standard, not a single test. And so we have to be TB minded, and the timely diagnosis, early treatment can save these patients from going into permanent sterility. So thank you very much, and I again thank Dr. Lohia sir for giving me this opportunity. Thank you.
Thank you so much, sir, for a great talk. It was so informative. Thank you, sir. Thank you.
Thank you. Thank you. So much obliged, sir.
Slide.
Very good. Wonderful.
No, thank you, madam. We were missing you, ma'am. Thank you.
Okay, first of all, let me apologize that I lost my connection at the crucial moment. I want to pay my tribute to Dr. Anand Kumar, and of course, compliments to Dr. Jb Sarma for a wonderful oration, for a very, very enigmatic condition, where the woman doesn't know she's suffering from tuberculosis quite often unless she starts suffering from sequelae like infertility or PID. And even provide, firstly, of course, he has already highlighted the diagnostic dilemmas. Once you have diagnosed, once you have adequately treated also, of course, it may not be possible to completely circumvent her infertility. So it still continues to be. But striking thing is, even National Tuberculosis Elimination Program puts genital tuberculosis in one corner, others, because we know the major killer is pulmonary tuberculosis, and genital is always secondary to primary somewhere else. And of course, we do have a lot of genital tuberculosis in men too. Here we are talking about women, but even men have a lot of problems. So this remains to be a very, very important condition. And I'm glad conference organizers have started the first oration on such an enigmatic condition. What I was saying in the beginning, even in most physical conferences, I have not seen such good attendance in the hall, even in the inaugural session, not only first scientific session. So for that, compliments go to Professor Lohia, Professor Ansari, and their team members. I won't name all because we're running a little late, but it has been an excellent, excellent organization.
Thank you so much.
You spoke very well.
Thank you so much,
Thank you, madam. And I want to thank Dr. Sunita Mittal, madam. I started my TB research with you, madam. I'm personally obliged to you for that, in my early phase. Thank you, madam.
आप करें हो गया next हो गया done.
So with this great oration, we are going to move our first session, which is on female reproductive health. And the chairperson for this sessions are Dr. Malini Laloraya. She's a scientist G, Division of Molecular Reproduction, Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram. Dr. Malini has pioneered the concept of positive role of free radicals in female reproduction as early as 1989. And the other chairperson for this session is Dr. Ajit Kumar Saxena, who is Professor and Head, Department of Pathology, All India Institute of Medical Sciences, Patna. Dr. Saxena has worked on genetics and epigenetics of birth defect, cancer, infertility, and stem cell research. So I would request our chairpersons to please chair this session. I just have a small request to make. We are running a little bit late on time. So I would request all our invited speakers and oral presenters to please finish on time. Thank you. Over to you, ma'am.
Hello, everyone. I hope I'm audible. Hello, am I audible?
Yes, you are audible, Dr.
Okay, so first of all, welcome everybody to this session on female reproductive health. And I have the honor of introducing the first speaker, Dr. Savita Yadav, and I'll be a little quick as we are running out of time. Dr. Savita Yadav is a professor at the Department of Biophysics, AIIMS, And her research interests are on protein signature discoveries in male infertility as well as reproductive cancer. She has got several awards like Sir Power Fellowship, Professor Goraya Memorial Award, Eames Excellence Prize, Hariom Trust Award, and second prize in Diagnostic Drug Discovery, Dr. G.P. Dalwar Award, and she's a fellow of the National Academy of Medical Sciences as well as fellow of Academy of Biomedical Sciences. She has more than 100 publications. So not taking a lot of time, I would invite Dr. Savita Yadav to deliver her talk.
Thank you, Dr. Malini, for kind introduction. शेयर करना है, हाँ शेयर करना है, कैसे शेयर करूँ? कहाँ कहाँ है स्लाइड्स, कहाँ है स्लाइड्स, कहाँ है हम प्रेजेंट करने के लिए ये Can you see my slides?
Yes.
Yes, okay. Thank you, Dr. Malini, for kind introduction. And first of all, I would like to thank Dr. Lohia, Dr. R.S. Sharma, and other team members of ISSRF for giving me this opportunity to present my work on this platform. And I would like to congratulate all the Lifetime Achievement Awardees and Dr. Madan for Padma Shri and Dr. Jb Sarma, who is my colleague as well as collaborator for this work. Heartiest congratulations to them. I'm going to speak about differential expression of salivary signature proteins in ovarian cancer. As you all know, ovarian cancer is the third most commonly detected cancer among Indian women, and this is 7th most common cancer in the world. And the 5-year survival rate is 49% worldwide, and in India it is only 45%, and it is often termed as silent killer due to asymptomatic nature of the disease. So there are more than 80% of new cases of ovarian cancer are diagnosed at advanced stage, and it leads to over 70% deaths. So this shows that diagnosis at early stage is required to have significant impact on the mortality. This is the statistics. You can see that mortality rate is higher in India, and this is the third most common cancer in India. If you look at incidences, it is estimated that by year 2035, incidences of ovarian cancer are going to rise by 55% and death rate will increase by 67%. This is the schematic diagram of ovarian cancer stages. And this study is we have conducted on epithelial ovarian cancer, which is the most common ovarian cancer. And BRCA1, BRCA2 are mutation are risk factor for ovarian cancer. Similarly, family history. If person has endometriosis, then they have also have a chances of having cancer 2-3-fold more. And reproductive history and infertility are also risk factor. If one person is obese, that also is a risk factor for ovarian cancer. Now, survival rate is directly correlated with the diagnosis. So what are the screening tests which are available to detect ovarian cancer? They are transvaginal ultrasound. Transvaginal ultrasound can only tell about the mass present over there or growth present over there. It cannot tell whether it is cancerous or benign. And most, like all primary health center, may not have this facility. Another marker is tumor marker protein CA125. This is most widely used method for screening, and it is elevated in 90% of advanced epithelial ovarian cancer. But it is also normal in 50% cases of early stage ovarian cancer. And it is also elevated in 2 to 3% of postmenopausal women without ovarian cancer. So this is not specific for ovarian cancer tumor only. Another marker is HG4, which is also widely used in West, and India also it is used limitedly. So many people are not able to get this transvaginal ultrasound done because they are in rural background. They are not able to reach to the specialty centers, and then when they reach there, they are already at the advanced stage. So their survival rate goes down. So this is the apt picture for any cancer patient. When person get cancer, he has to move backward and forward. He has to go for chemo, radiotherapy, surgery, and life is not very good. So how can we increase their survival rate? By having some non-invasive diagnostic test or screening method. See, saliva can be used as a diagnostic fluid, and if it is used as a diagnostic fluid, no skilled person is required for saliva test. Then it has good protein concentration, around 2 milligram per ml. It is not as complex as blood, and then it could be cost effective also. So this has a probability of using saliva as a diagnostic fluid for early detection of diseases. We have done this study to see whether saliva can be used as a diagnostic tool. So what we have done in this, we have this is the overview of study. We have recruited the patient, we have collected their saliva, and then we have done 2D Dij method, and then we got differentially expressed protein which were validated by different methods, and then later on these differentially expressed proteins were studied in cell culture to see that what kind of effect they must be having on the cell line. So this is the overview. And these are the patient recruitment which we have done from Department of Gynecology, and Dr. Jb Sarma helped us in this. And then we have recruited patient, and we have excluded patient who are pregnant or oral contraceptives were there, and patient who were assigned for radiotherapy. Then we have done 2D Dij. 2D Dij method, we have labeled our disease patient with disease saliva with Cy5 and control saliva with Cy3, and then we have done dye swapping so that dye bias can be minimized. We have done 4 sets of experiments here and got 12 gels. And these differentially labeled spots were scanned by Typhoon scanner, and then these spots were analyzed by same spot progenesis software. So once we know that which were the proteins which were differentially expressed, preparative gel was run, spots were cut, and then they were sent for their identification. We got 44 differentially expressed proteins. Out of this, 25 proteins were overexpressed and 19 proteins were underexpressed. See, we cannot go with each and every protein ahead, so we have chosen 4 proteins from this list to go ahead with, depending on their biological functions in cancer and their fold chains. So proteins in blue were chosen for further studies. Then see, I have done work on all these proteins. We are working on these proteins, and in today's lecture, I'll be speaking only about multimarin 1, which was differentially expressed in saliva. Multimarin 1 is a massive protein. It is a disulfide-linked homopolymer glycoprotein from the amylin family. And it has a RGD motif present there, which makes it behave like an adhesive protein in extracellular matrix. We have many authors have observed altered expression of MMRN1 in cancers like hepatocellular carcinoma, cervical cancer. They've used this thing and they got the altered expression. So they could only say that altered expression was there, but nobody has studied whether it is acting as a repressor or activator. If it is acting as a repressor or activator, it will make it as a potential therapeutic target. So since none of the studies were there who has explored the role of MMRN1 in ovarian cancer, so we decided to see that how this protein works in ovarian cancer. So see, we got differentially expressed protein in saliva. This we have done it by Western blot analyzer. You can see that in advanced stage, it is definitely present at high concentration in Western blot. And then when we have done ELISA, in ELISA also its level are overexpressed in case of saliva of ovarian cancer patient in case of advanced stage. So since we got this protein overexpressed in saliva, our next question was whether this protein is present in saliva, is it present in tissue also, and what is the expression level of MMRN1 in tissue. So we have done immunohistochemistry, and then you can see that at low grade cancer this is overexpressed, and at high grade cancer also it is overexpressed. The overexpression of this protein increases with the high grade cancer. It is less in low grade cancer and more in high grade cancer. So after seeing this, we wanted to see that, we have already done ELISA with high grade cancer patient in saliva, and there it was increased. So we wanted to see that whether it is increasing in low grade cancer also. Then we have again run ELISA with different set of samples, and then we were able to see, yes, it is differentially expressed even in low grade cancer, and it is more, its level are more in case of high grade cancer. Then we wanted to see whether, at protein level we saw that it is differentially expressed in saliva. Then we wanted to see whether okay, at immunohistochemistry level as a protein in tissue it is present, what is happening at mRNA level. So at mRNA level also we have done RT-PCR, and then we were able to see that at advanced stage it is overexpressed. Then again we have done it with the low grade and high grade, and this is with progression of cancer, this elevation of MMRN1 is increasing. After doing this experiment, we wanted to see, we know that it is getting overexpressed. What function it may be having in the ovarian cancer, we didn't know. So we wanted to see its function in vitro. So we have done knockdown experiment with ovarian cancer cell line. We have used COV3 cell line. We have knocked down MMRN1, and then that we were able to achieve at 40 picomole, and then it was nearly 1.47 times knockdown was there in case of MMRN1. So after knockdown, we wanted to study that when we have knocked down MMRN1, what is it doing to the viability of the cell, whether it is causing apoptosis, what is happening to wound healing, cell adhesion, cell migration, and cell invasion, because all these phenomena play important role in any cancer. So we wanted to see that what happened to these properties of the cell when MMRN1 is knocked down. We have done MTT assay, and then you can see that viability of the cell in MMRN1 knockdown cells was down. It was reduced by 55%. Since it is a matrix protein, it may be playing important role in cell adhesion. So we did cell adhesion assay, and then in this also you can see that in MMRN1 knockdown cells, the cell adhesion property was reduced by 46%. Then we have studied cell apoptosis by flow cytometry. Q1 quadrant show the apoptotic cell. This shows late apoptotic cells and live cells, and this shows necrosis. So in quadrant 1, we were able to see that number of cells, apoptotic cells, were increased. There was increase in cell apoptosis by 5.9% in case of MMRN1 knockdown cells. So it is playing an important role in apoptosis also. Then what we have done, we wanted to see whether these cells are proliferating if we create a wound. So we have created a wound by pipette tip in control as well as MMRN knockdown cells, and then you can see that in and then we have measured whether these cells are proliferating towards wound. Then in case of MMRN knockdown cell, we could see that wound was not completely healed. it was taking time, but in case of control at 72 hours, it was completely, cells have migrated towards the wound. We don't know what role it may be playing in wound healing, but definitely these cells are proliferating towards, migrating towards towards the wound. So that was also one of the important observation we could get by cell scratch assay. Then we wanted to see whether these cells are migrating, knockdown cells are migrating, and how they are migrating. Then we have done cell migration assay. In cell migration assay, we have put one membrane, and then we have changed FBS concentration in the media, and this, if FBS concentration is more, it acts as a chemoattractant, and cells which migrated below the membrane were measured. And you can see that in mmRNA, knockdown cells, cell migration was less compared to control. And we have we have stained it with crystal violet, and you can see that cell migration is less in case of mmRN1 knockdown cells. Then we have studied invasion. In invasion assay, what we have done, same experiment was performed, but matrigel was kept in between. Matrigel is a it mimics extracellular matrix. So we wanted to see that whether these cells are invading through extracellular matrix, and then we could see that invasion of cell was less in case of mmRN1 knockdown cells. And this was seen by staining also, that it was less in case of mmRN siRNA transfected cell. It means it may be playing role in cell invasion and migration also. So by these simple experiments, we were able to conclude that mmRN1 is playing an important role in cell proliferation, migration, adhesion, and invasion. It is also preventing cell death. Orientation and arrangement of ECM have a specific role in tumor progression. So ECM undergoes modification with biophysical and biochemical changes to manipulate cell adhesion and migration. Our protein is an mmRN1 is an ECM protein, and its elevated level can be directly correlated to ovarian cancer cell invasion and proliferation. And increased expression of mmRN1 with tumor grade supports this hypothesis. So at we didn't know whether it is playing and how it is playing a role. So by this these experiments, we could say that overexpression of mmRN1 is helping in ovarian cancer progression. So we observed elevated level of multimarin-1 in saliva as well as tissue of the epithelial cell ovarian cancer patient with progression of disease. So this support its potential use in non-invasive detection of ovarian cancer in future. We are working on multimarin-1, we are working on lipocalin-1, and we are getting promising result. And in future, because of COVID, we are not able to collect saliva. In future, we collect more number of samples, we validate in large cohort, then maybe these proteins have a have a tendency or have a potential to be used as a biomarker for early detection of ovarian cancer non-invasively. So in the end, I would like to add that silent killers are not silent as we think. They start shouting at the early stage, at the molecular level. So if we work towards developing new techniques and ideas for detection and screening, then we can definitely hear their voice, and then what survival rate can be increased, mortality rate can be decreased. Thank you so much for patient listening. I would like to thank ICMR and Serve India for funding these studies. And I would like to gratefully acknowledge all the project collaborators, Dr. Alpana Sharma, Dr. Sunish Kumar, Dr. Jb Sarma, who has recruited sample, given us sample, Dr. Sandeep Mathur, who has helped us in immunohistochemistry, and the people who have done experiment is Abhinav cell line experiment are done by him, Benita, who has collected samples, and then done work on saliva. Swati, who is working on another protein, FGF8 of saliva. Vikrant, who is working on lipocalin. And I thank them all because without them, this work was not possible. Thank you so much for patient listening.
Thank you, Professor Savita, for the wonderful talk. And I don't see any questions on the chat box as of now. So since we are running late on the session, I think people can write to you in the chat box and then have questions.
Yeah, they can email me. I'll reply to them. Yeah.
So thank you so much, Professor Savita. And I now invite Dr. Roya Rosanti for the next talk. Oh no, I'm sorry. Just a minute. Okay, it's Professor Ashutosh Haldar, I believe. Professor Ashutosh Haldar is there?
No, I have probably after lunch. Royal
But,
rate is
okay.
right.
There is
It's right.
I was right on the okay.
Yeah, ma'am. Dr. Roya Rosati is the next one.
Okay, all right. So I would invite Dr. Roya Rosati for the next talk, and very briefly I will introduce her that her main research interests are in endometriosis, obstetric stem cell biology, and PCOS in India. And she is the art chief specialist and research director and founder of MHRT Hyderabad. She was professor and head of the Department of OBGYN, Shadan Institute of Medical Sciences, and also served as professor earlier at Ovasi Hospital and Research Center of the Deccan College of Medical Sciences. She has participated in conducting several projects of ICMR and has received several awards and accolades, namely the prestigious Rajiv Gandhi Shiromani Award, the Rashtriya Ekta Award, the Obstetrics and Gynecology Award by Third World Organization for Women Scientists, Honorary Award by National Academy of Medical Sciences, Best Consultant Award by Mahavir Hospital and Research Center. It's my pleasure to welcome Dr. Roya Rosati to deliver her talk.
Thank you, Dr. Malini, for of ISSRF-2022. My talk is on the stimulation protocol in severe endometriosis. Is there any difference? We all are very aware that approximately 15% of the patients with the infertility, they are having some kind of different stages of endometriosis. Endometriosis is believed that this lesion in the pelvis, it creates a very hostile microenvironment of the fertilization of oocyte and early development of embryo in the fallopian tubes in vivo. Endometriosis may also affect the ovarian reserve and responsiveness to ovarian stimulation in IVF program. These effects lead to the lower fertilization and pregnancy rate in patients with endometriosis going for the IVF treatment compared to other group. The other group like male factor or tubal factor, they perhaps have a better result with ART compared to patients with endometriosis because of this various inflammatory and the immunological problem which happens in vivo in patients with endometriosis. Therefore, lots of work and research has been done to improve the result and the success rate in ART in patients, especially with severe endometriosis who are going for ART treatment. One of these molecules which is widely being researched is a GnRH agonist in patients with endometriosis treated by IVF. Several studies exist comparing the reproductive outcome in patients with endometriosis undergoing IVF treated with or without GnRH agonist. GnRH agonist is being started on the day 21 of menstruation, and the stimulation with the gonadotropin will be started on the day 2. And the tendency for a higher pregnancy per cycle is being observed and reported if the GnRH agonist is being used compared to the cycle, which is no down regulation has been done. GnRH analog is being initiated at the mid-luteal phase to achieve down regulation as it continues till the day of the HCG injection and stimulation. How about the use of prolonged down regulation with GnRH agonist prior to the ovarian stimulation? Prolonged down regulation refers to the extension of 2 to 3 months before the actual analog administration, which is used in so-called long protocol. Prolonged use of the GnRH agonist in patients with endometriosis aims at inactivating the endometriotic tissue, and it is also known to interfere with the paracrine and endocrine factors which are involved in the pathogenesis of endometriosis. Women with endometriosis following the prolonged down regulation with GnRH agonists are probably starting IVF cycle with a disease which is under control and perhaps with the increased chance of pregnancy rate. Studies which is examine the value of prolonged down regulation in patients with endometriosis undergoing the IVF. It shows the higher clinical pregnancy rate per transfer in 21 patients compare the patient who received GnRH analog for 60 days or more before the ovarian stimulation, in compared to 11 patients with endometriosis who were treated with the standard mid-luteal GnRH agonist down regulation. So they had a 67% pregnancy rate versus 27%. Here the table shows there is a two group where one group had a long protocol, 3 months before stimulation had a long GnRH analog therapy, and then the other group, the 11 patient, had no, only the GnRH agonist was used during the stimulation protocol, and we can see the success rate, clinical pregnancy rate, which is 67%, versus very low level, which is lesser level, 11 to 12%, compared to the one which is long protocol not used. The majority of the retrospective studies conclude that the reproductive outcome in the woman with endometriosis undergoing ART is improved after prolonged down regulation with GnRH agonist before starting ovarian stimulation. All the literature indicate in a study which showed 32 patients underwent ovarian stimulation for oocyte retrieval without any preparation, that is protocol A, whereas 35 women underwent 6 months period of the hormonal suppression therapy with the long GnRH agonist therapy protocol B before IVF procedure. Patients with severe endometriosis undergoing IVF reported that use of the GnRH agonist for 6 months prior to the stimulation. They are the 35 patients, 48 cycle were compared to the stimulation where they had only 32 patients, 51 cycle, only gonadotropin was used with the GnRH agonist at the time of stimulation. Therefore, the 25% versus only 3.9% clinical pregnancy rate was reported. It's concluded that combining GnRH agonist therapy before IVF ET provided an improvement in conception rate in patients with a severe endometriosis. Similarly, a prospective study reported that a significantly higher pregnancy rate per transfer in 15 patients, 23 IVF cycle, who received GnRH agonist for 2 months as compared to 69 patients who were treated by short or ultra-short GnRH agonist protocol. Therefore, the 34.7% pregnancy rate versus 10.7%. So it indicates that higher rates of pregnancy rate are achieved in vitro fertilization and ART following a long-term down regulation in patients with extensive endometriosis. The result of the IVF cycle, it shows that basically no differences found in control where it is given short or ultra-short protocol with the GnRH agonist in mild cases. but pregnancy rate is improved in patients with a severe endometriosis, that is a stage 3 or a stage 4. Randomized patients with the various stages of endometriosis, again in two groups, and the first group received a 3 months agonist regulation before initiation of ovarian stimulation for IVF, and in second group, again, long-term agonist protocol microdose Valera use, and significantly higher ongoing pregnancy rate was present in the first group who had received the down regulation for 3 months compared to the second group, that is 80% versus 53.85%. Comparing prolonged down regulation to agonist, the protocol suggests that the prolonged administration of GnRH agonist is beneficial for reproductive outcome. However, the two groups compare the different severity of the endometriosis and pregnancy rate achieved is unduly high. Overall, it appears that the use of prolonged GnRH agonist down regulation prior to the initiation of the ovarian stimulation is beneficial in patients with endometriosis undergoing IVF as compared with the use of the GnRH long protocol. Here again, we have another two groups where 25 patients will receive a deeper preparation of GnRH Lupride 3.75, and the first injection was administered during the early follicular phase. Control ovarian stimulation described within the 45 days of the last Lupride injection. And group 2 consisted of the 26 control with the endometriosis who did not receive long-acting GnRH agonists, but instead they received control ovarian hyperstimulation regime. And in this, it is very clearly showing that implantation rate is much more if the pre-treatment LHRH long protocol has been used. Comparison of the clinical pregnancy rate amongst the endometriosis with the stage 1 and 2 compared to stage 3 and 4. In subgroup analysis of the randomized control trial, ultra-long protocol group compared with the long protocol group was not associated with any significant differences in clinical pregnancy rate in mild stage, that is in stage 1 and 2. But the ultra-long protocol had a significantly higher clinical pregnancy rate in patients with the stage 3 and 4 compared to the long protocol group. It is indicating that compared with the GnRH long protocol, the GnRH ultra-long protocol improved the clinical pregnancy rate or implantation rate of infertile patients, which may improve the clinical outcome in patients with the severe endometriosis. GnRH analog pre-treatment can improve the microenvironment of the ovary and quality of oocyte. Pre-treatment with GnRH analog could also reduce the concentration of interleukin-1, tumor necrosis factor, nitric oxide, which are toxic to the embryo in the peritoneal fluid. It could also make the pituitary cell unresponsive to the endogenous GnRH analog, and to achieve the desensitization, reduce the secretion of the FSH and luteinizing hormone, and inhibiting the ovarian activity, reducing the level of estradiol, promoting the atrophy and apoptosis of the ectopic foci, and favoring the embryo implantation. GnRH or ultra-long protocol significantly improved the clinical pregnancy rate in patients with the endometriosis of stage 3 and 4 compared with the long GnRH protocol, but had no effect in patients with the stage 1 and 2. This might be because compared to the patient in the stage 1 and 2, changes in the pelvic anatomy and the level of the inflammatory molecules and the response in pelvic cavity and the degree of the local microenvironment balance, and there is an imbalance in the endometrium, were all heavier and severe in patients with severe endometriosis. That is why the longer duration of the LHRH analog, it helped this patient so that the severity and this microenvironment become better and get normalized. Therefore, the success rate is more in these patients with the severe endometriosis who used long suppression for almost 3 to 6 months. What about other indicators? What are indicators? But the most important indicator for us as a clinician who are treating these difficult cases of endometriosis is implantation rate, where the other factors are fertilization rate, clinical pregnancy rate, basal, that is FSH, duration of ovarian stimulation, dose of the gonadotropins, and number of the retrieved oocyte. Supplement indicators are age, BMI, antral follicle count, basal estradiol, and CA125. What about the role of the removal of the ovarian endometrioma before IVF? Endometrioma resection prior to the IVF may compromise or destroy the adjacent normal ovarian tissue. Moreover, the aspiration of the endometrioma is associated with the increased risk of the infection. On the other hand, endometrioma may interfere with the stimulation or may impose difficulty in oocyte aspiration and retrieval, and is being considered to be responsible for producing toxic product, which is affecting the mature oocyte and affecting the cleavage and implantation. Therefore, the treatment of the endometriomas before ART can be performed either by local resection or cauterization or laser treatment. How about the use of GnRH antagonists? Till now, I was explaining to you the LHRH protocol, then long protocol, very long protocol, and now the role of the antagonist protocol. There are been trials which is based on the putative estrogen threshold theory. The rationale for the treatment protocol in different mode of the action of GnRH antagonist compared to GnRH agonist is the immediate receptor blockage of the gonadotropin cell, immediate subsequent decrease of the LH and FSH estradiol secretion down to the postmenopausal value, and there later on a slow increase in the estrogen production within the following 3 days. All the patients, they were free of symptoms during the period of the GnRH antagonist treatment. LH and FSH concentration, they display a very consistent value that was in a low level. Endometriotic lesion appear clinically devascularized and atropic. Signs of the hypoestrogenism and glandular and stromal atrophy were noted in biopsy taking after the treatment during the second laparoscopy. This could raise through the direct endometrial effect of the GnRH antagonist, which is improving the theory of the therapeutic window. GnRH antagonists also got that the result of there has been a pilot study which confirmed the feasibility of using a GnRH antagonist in the treatment of the patient with the severe endometriosis, and the weekly administration of 3 mg cetrorelix offer a very safe and effective treatment option, especially with regards to the disease-free symptoms. However, the effects on the endometriotic lesions doesn't seem to be superior to the effect which is GnRH agonist has. Most important is the almost lack of any side effects. To conclude with, sequential administration of GnRH antagonists and cetrorelix, it creates a very new opportunity for medical treatment of symptomatic endometriosis. Sequential treatment with cetrorelix may have a limited side effects with the immediate and effective suppression of endogenous gonadotropin while preserving basic estrogen production during the course of the COS and the treatment. The weekly administration of GnRH antagonists in women with endometriosis appear to be simple, effective, convenient approach to reach to the therapeutic window without any signs of the major side effects. No further direct treatment is necessary. No metabolic changes are to be expected, although further studies required to confirm this. The use of the GnRH antagonist in women patients with the endometriosis could be a reasonable, low cost-effective therapeutic option. It's the treatment much more cheaper than the one when we are using GnRH agonist. In that GnRH agonist or long protocol is more expensive and more number of the HMG ampoule need to be used. Unlike when we are using antagonists, it is cheaper, less ampoules are used. Let us see what is Cochrane review. Cochrane review tells this review raises important questions regarding the merits of the long-term GnRH agonist therapy compared to the pre-treatment prior to the IVF and ICSI in women with endometriosis. Contrary to the previous finding, we are very uncertain to use whether the long-term GnRH agonist therapy impact on the live birth rate or indeed the complication rate compared to the standard IVF and ICSI. Further, we are uncertain whether the intervention impact on the clinical pregnancy rate, multiple pregnancy, miscarriage, mean number of the oocytes, number of the embryos, and in the light of the paucity and very low quality of existing data, therefore, for the primary outcome which is being examined, further high quality trials are need to be done where to determine the impact of the long-term GnRH agonist therapy on ICSI and IVF outcome, and not compared to the pre-treatment, but compared to the other proposed alternative. Now we are having the GnRH agonist, we are having the non-protocol of the GnRH agonist compared to the antagonist which we have. Really speaking, long-term trial and long study, multi-center study has to be done to really tell us where we are landing and what is the result exactly. There is a new concept which is very important for all of us to know is segmentation of IVF in severe endometriosis. In order to limit the risk of the disease flaring and optimizing the result, some authors propose GnRH antagonist protocol, agonist trigger, and freeze all the embryos in patients with endometriosis. GnRH antagonist protocol freeze all and then frozen transfer embryo after a prolonged down regulation has shown an excellent outcome in endometriosis. Our result also, it is in favor of the segmented IVF. To conclude with, so the GnRH ultra-long protocol can improve the clinical pregnancy rate of infertile patients with severe endometriosis, especially 3 and 4 stage. On the basis of retrospective studies, the reproductive outcome of patients with endometriosis, they showed enhanced if down-regulation with GnRH agonist is used before ovarian stimulation for IVF. There is evidence which is not robust.
Sir Roya, could you wind up? We are already little.
Just few lines.
Yeah.
Suggest that use of the prolonged GnRH agonist, though beneficial in patients with endometriosis. Whether only a very specific subset it is to be used, we don't know. No benefits from the medical suppression. We should remember gestrinone, danazole prior to IVF, it doesn't help us really. GnRH antagonist protocol, freeze all, frozen transfer after prolonged down regulation, it is shown an excellent outcome. A very prospective randomized trial in patients with the endometriomas undergoing surgical treatment or not to evaluate the usefulness of the removal prior to the ovarian stimulation, the result to be yet to become. To conclude with, administration of GnRH agonist for 3 months prior to the IVF in infertile patients with endometriosis had no side effects on the ovarian response to the exogenous gonadotropin. GnRH ultra-long protocol can improve clinical pregnancy rate of patients with the stage 3 and 4 endometriosis in randomized trial, and a larger sample needed and future studies could ideally be performed in conjunction with the evaluation of the role of the endometrial receptivity markers, and larger trials should be conducted by Iss Rf or ISSRF or IFS or ISSR to confirm or reconfirm all these data. Thank you very much.
Thank you, Dr. Roya, for your wonderful talk. I'm sure there will be a lot of questions. Even I have a question, but I will refrain myself and message you the question because we are running short of time. And so thank you so much for your wonderful talk. And I would like to invite the next speaker, Dr. Rupesh Srivastava. Is he there? Yes.
Yes, ma'am, I'm there.
Yeah. So a brief introduction on Dr. Rupesh Srivastava. He is just a minute, please. Associate Professor at Department of Biotechnology, All India Institute of Medical Sciences, New Delhi, and his group works actively on osteoimmunology. I'm sure it's a very interesting field, and especially focusing on menopausal osteoporosis. He has several awards, namely Dr. Jb Sarma Young Scientist Award twice, one from the Indian Immunology Society and one from ISSRF. He is also recipient of the Science Communicator Award of Indian Science Congress. He is editorial board and review member for several journals, is a gold medalist during his master's, and has published in various international journals. He has 26 peer-reviewed research papers, 8 reviews, and 5 book chapters. A request to you, Dr. Rupesh, if you keep time and give us some time to ask questions, it will be nice. And I invite Dr. Rupesh for the talk.
Yeah, thank you, Dr. Malini, for talking over to me so nicely. First of all, I would like to thank all the organizers, especially Professor Lohia and Dr. Sharma for giving me this opportunity so that I can present my work here. So, are my slides visible?
Yes.
But this bar is making a problem. I don't know how to remove this bar. Any idea how can I disable this? Or it will be there only.
Which bar are you talking about?
Can't you see anything? Any bar on the bottom left-hand side? Nothing?
No.
Okay, then it's fine. So maybe it is from my end only.
Yeah.
Okay, so first of all, so I will just start this. So today I will be talking about something related to regulatory B-cells. So before that, why I have chosen bone health is that because we just have only one skeleton and with which we are basically born and which supports us throughout our lives. And it is going to be there with us until we die. And regardless of this fact, the age, gender, race, nationality, or belief set, we are all going to have just only one skeleton. And yet this essential organ is so often taken for granted. So with this, I will move ahead now. Not able to move my slides. Okay, it's fine. So in bone health, again, I will be talking about osteoporosis. Why osteoporosis? Because osteoporosis is a very chronic bone disease. It is characterized by low bone mass and deterioration of bone tissues, and which leads to increased risk of broken bones and which leads to fragility fractures. So if you see the scenario globally, you can see that there are more than 200 people who are suffering from osteoporosis till date. And this is the data which is not including our data, the Indian data, because we really don't have that data. And from the global average, you can see one fracture occurs every 3 seconds, and 1 in 5 men and 1 in 3 women are going to be affected with this. And if we talk about Asia, by 2050, 50% of the fractures are going to occur in Asia, making it a world capital for your osteoporosis related fractures. The Indian scenario is also very grim because we are one of the most populous country in the world. And in India too, more than between 25 to 60% of the Indian postmenopausal women are going to suffer on osteoporosis related fracture in their lifetime. And the aging population we are having, because we are also have most populous countries, so definitely we are having a lot of people who are in crossing 50 and 60s. So they are at high risk, apart from also the women's also in the postmenopause who are there. So these are the people at high risk. So if you see the hospitalization burden of the disease, so this is the hospitalization burden, which is a data which shows clearly that more than 43% of the hospitalizations which are occurring are basically due to fractures alone. And if you see the heart one is only 25%, and stroke 26%, breast cancer 6%, and osteoporotic related fractures are basically occupying 43% of the hospitalizations. This is the US data, but I'm sure the data for the Indian India will also be more or less near to this, or maybe it can be even more than that. So moving ahead, so this is also having a huge economic burden of more than 130 billion USD per year. So before dealing with this, I would like to draw attention of all the viewers here that the role of immunology in osteoporosis, because earlier it was thought that immune system is not that very much important in playing a substantial role in the pathophysiology of osteoporosis. But our group from the last 8 to 10 years have been actively working in discerning the role of immune system, both the adaptive immune system and the innate immune system, in how they are basically affecting osteoporosis. So if you see here, we coined this term immunoporosis basically in 2018 itself to exclusively define the role of each and every subset of these both innate and adaptive immune system in osteoporosis. And from that time onwards, and earlier from that, from the last 8 years, we have been doing a lot of extensive work here. And this is one of the most cited articles in frontiers immunology and the bone itself, which we have been doing so. And due to these contributions, we have also been on the cover page of Immunity in 2019, that 25th anniversary issue of the Immunity itself 2 years back. So coming now, so we have already defined what is the role of Treg and TS17 cell access in osteoporosis with because what happens in the normal conditions, the homeostatic conditions, the balance is more toward the Tregs, which is basically taking care of your inflammatory responses. And within, when an inflammation is there, for example, in case of your inflammatory bone loss, this balance is basically lost. So this is how we define the role of Treg and TS17 balance in a lot of these cases in the case of postmenopausal osteoporosis. And we published a lot of probiotics with the help of 6 to 7 papers we published on that, can defining this role of Treg and TS17 cell balance in osteoporosis. But later on, I basically, in the last 2 years, I was just wondering, it can't be only Treg and TS17 cells, which should be the master regulator for pathophysiology of osteoporosis from an immunological point of view. So there should be something else also. There should be some other cells also, not even a single, maybe some other cells. So in search of that, in quest for that, basically, I thought maybe there should be some other adaptive or an innate immune cells, which is basically regulating this axis or is being regulated at all. So the first cell which came to my mind were the B cells. Why B cells? Because B cells have been defined to have various non-humoral functions. If you see here, B cells have been defined to have various roles in tissue development. They also regulate various other T cells, DCs, and other responses also. And apart from the B cells itself, we also have your regulatory B cells. So regulatory B cells have been a subset of Tregs, which are defined in the last decade itself. And they induce, they basically have an anti-inflammatory role by secretion of 10, 35 and TJ beta cytokine, and they promote Tregs differentiation, they inhibit TS17 differentiation, and they also regulate NRG and CD8 T cells, which are discovered my group, my labs group when I was doing PhD. Dr. GC Mishra was there. They discovered this role, how Tregs basically they control this. So they have an established role in basically in various other immune responses also, which we just talked about. So quickly moving ahead, I don't know whether my slides are basically getting hanged or what. Okay, so Tregs have various role in inflammation also. And the last slide was very quick, I think so. So they have a very established role in inflammation and diseases, various inflammation diseases, including your COVID-19 and the recently we described in osteoporosis, which I'm going to talk about today, apart from other autoimmunity, asthma and allergy diseases also. So before going to that, I will talk about what are the different subsets of these regulatory B cells. So if you see, so we have various subsets of Tregs, and the most common one of them, the most defined one of them is basically your beaten Tregs. So this is the one which we discovered 10 years back, and this is the one which I'm going to talk about today. So we have various other subsets in case of humans also and mice also, because this study is mostly done in mice and we are still working on human data. So I will be talking about these beaten Tregs today. So the question which came was, can regulatory B cells also inhibit inflammatory bone loss in postmenopausal osteoporosis? So before going to that, the first challenge was how to generate Tregs in vitro, because we have various literatures, various unestablished literatures that Tregs can be induced in a number of ways. So what we did was that we took a model, well-established model that we used LPS for inducing Tregs in vitro. We took the mice, the spleens, and we isolated the B cells negatively. We activated them with LPS 10 microgram per ml. And after those Tregs were ultimately used for inducing or doing the further studies. You can see here clearly that in the control, there is 0.12% of these Tregs are there. But in case of LPS, it is 6.45%. So there is 53 times increase in the induction of Tregs within just 24 hours. So this is how this is the population which is CD19 IL-10 positive Tregs, and this is the population which is basically the beaten Tregs, CD19 positive, 1D high, CD5 positive, IL-10 positive. These are the one which I'm going to talk about in my future slides. So the next question was, so we were able to induce or generate Tregs in vitro. So the next question was, do they really have any anti-osteoclastogenic potential? Because nobody till now has ever done that. So what we did was that we defined a strategy. What we did, we took out the spleen, we isolated the B cells, and we induced the B cells to become Brex, just I talked about in the previous slide. On parallelly, what we did, we flushed out the bone marrow, and we cultured that for 24 hours in the presence of MCSF. And then I co-cultured this Brex induced with the LPS along with these bone marrow cells in the presence of MCSF and rankle, which are known cytokines for inducing osteoclastogenesis in vitro. And after 4 days, I performed TRAP staining. TRAP is basically a marker for defining our osteoclast. So what I found was that when I cultured these bone marrow cells with regulatory B cells at different ratios, so these are the positive control M plus R. So you can see wonderful osteoclasts, which multinucleated osteoclasts. They can have up to 200 or 300 nuclei also in a single cell. And when I co-cultured them with Brex in a dose-dependent manner or in a ratio manner, you can see here that basically it was significantly inhibiting the generation of osteoclasts. So because you can see from the big osteoclasts are there at 10:1 ratio, and they are significantly going down. And these are just for the positive controls, only the B cells were used, not the Brex. So B cells are already known to actually induce osteoclastogenesis through the secretion of rankle. But in the case of Brex, it was actually inhibiting them significantly. And these are the other quantitative data which we took, which we analyzed from here. Then the next question was, are the Brex only inhibiting, or they're also affecting the functional activity of these osteoclasts? For that, with an F-acting assay, the similar set of experiments were performed. Only thing was that we cultured them in 24-well plates, because we used a glass cover slips for doing the confocal, the fluorescence microscopy. And you can see here, these are the F-acting rings. F-acting is very important for the function of osteoclast, because it is due to this F-acting that the osteoclasts bind to the surface of bone very tightly and create an acidic environment, which is basically dissolving the hydroxyapatite crystals there and leading to your bone resorption. So you can see here in the positive control, you can see number of multinucleated osteoclasts in this merged image, along with a very intact F-acting ring. But as we moving with the increasing dose of these Brex, co-culturing them with BMC, the bone marrow cells, this is significantly or totally lost. So this assay further confirmed our results that, okay, Brex are not only inhibiting osteoclasts, but they're also inhibiting the functional activity which is required. So these are basically, Brex are very potent in that. Now, how are they inhibiting that osteoclastogenesis? So this again was a question, because without this, we cannot do anything. So for that, firstly, we thought whether it is a contact dependent manner or it is a contact independent manner, because B cells, they secrete mostly IL-10. And also Brex have also been shown to express TJ-beta on the surface membrane. So whether it is doing in a contact dependent manner or a contact independent manner, for elucidating this, what we did was that we used a transwell insert. We took the LPS activated B cells in the upper chamber and the bone marrow cells in the lower chamber. And these are the membrane, semi-pub membrane, all of you know that. So when we did the same set of experiments, we found that even though we are keeping this LPS activated Brex in the upper transwell insert, then also they were able to inhibit significantly osteoclastogenesis, proving the point that there are certain soluble factors which must be involved in inhibiting osteoclastogenesis through these regulatory B cells. So then our first shot was that it should be IL-10 or IL-35. So first we took the IL-10, because IL-10 is mostly secreted by Brex. It is a signature cytokine of regulatory B cells. So what we did to confirm this, we took a positive control M plus R, and then we cultured these osteoclast precursors with regulatory Brex in the presence of anti-IL-10 neutralization antibodies, monoclonal antibodies at 10 microgram per ml. The moment we added these anti-IL-10 antibody, all the inhibitory effect of Brex was gone, as you can see in the isotype. So this confirmed that this is basically the cytokine or the mediator of this anti-osteoclastogenic activities, IL-10. So this was basically a eureka moment for us, that defining the regulatory B cells and then also defining how it is inhibiting that. And they are also not only inhibiting osteoclastogenesis, they were also basically inhibiting this F-acting ring. At the moment we are adding anti-IL-10, all these inhibitory properties of the Brex are gone with respect to the isotype. So basically now we have basically proven Brex have a very huge potential of anti-osteoclastogenic potential. But how huge? What is the magnitude of that? We did calculation and we found that one Brex was able to inhibit 30 osteoclast precursors if we took 1:5 ratio. So this is also, it is known that one Brex induces threefold induction of Tregs from the literature itself. So technically speaking, also Tregs are known to actually inhibit 100 osteoclast precursors. And we found 30 osteoclast precursors from the Bregs. And if Bregs can induce threefold Tregs, then this will go up to 330 osteoclast. One Breg can basically affect the potential. It is having the potential of inhibiting osteoclast of one Breg to 300, more than 300 osteoclasts. This is just 1:5 ratio if you're taking. If you are taking 1:1, this will be even more, 1,000, 3,000, it will go to that. So this is the huge, or such is the magnitude of the anti-osteoclastic potential of regulatory Biases. So now what? Now we have done everything in vitro. So now we took a model for in vivo. In case of your osteoporotic mice, ovarectomy-induced bilateral ovarectomy was performed, and then we thought, what is the immunoporotic role of these Bregs in vivo? So when we induced or when we developed these osteoporotic mice, and then we did the bone analysis, serum, bone marrow, and the spleen for flow cytometry, and we found, yes, when we what we did is that we performed what is the percentage of these regulatory Biases in bone marrow, which is in the site where the osteoclastogenesis is basically taking place, the bone loss, and also the spleen, because spleen is basically the lymphoid organ where the induction of regulatory Biases takes place in case of a normal host physiology. So if you can see here, in the sham, this was 4.58, and ovarectomy, it was significantly reduced. So both this CD19 and IL-10 positive regulatory Biases, and also these your CD19, CD5 and 1D. So both these were significantly reduced in case of your ovarectomy. Then again, this proves that, okay, in case of ovarectomy, the Bregs are compromised. Both their number and also the function must also be compromised. This was not the only thing. We published this in 6 months back in Frontiers in Immunology, the same which I'm talking about today. And also, apart from these two subsets, we also looked into other subsets which we still haven't published, the CD19 11B positive Bregs and CD19 FOXP3 positive Bregs. These are other subtypes of these regulatory B6, and these are also basically dwindled. This population also going down in case of rectomy with respect to the sham mice. Now, why? So what is the importance of this? So basically, we already defined the Treg and TS17 cell axis. Now we have added the Bregs here. Why Bregs we have added? Because Bregs have the property of regulating both Tregs and TS17. Bregs have been known to inhibit Tregs and induce Tregs and inhibit TS17 cells in various inflammatory diseases, including rheumatoid arthritis, MS, diabetes, and everywhere. So this has never been done in case of your postmenopausal or osteoporosis. So this is for the first time we have created that, and we are having a lot other data also, which is proving that we have a Breg, Treg, TS17 cell axis. That paper is already in the communication, and which I can talk about later. So why I'm talking about this axis is very important, because if you see here, these are the Bregs, and they control both the TS17 and the regulatory pieces. And these Treg and TS17s, along with our Bregs, they also control these osteoclasts. And this axis, all the known drugs which are presently used for treating osteoclastogenesis in case of your osteoporosis are basically targeting this axis. Either they are bisphosphonates or teriparatide or denosumab. They all are basically targeting this immunoporotic axis. So this is how, if you can target this Breg, Treg, TS17 cell axis, we would be basically able to create a lot of positive impact on amelioration of the disease, because all the drugs, these drugs are having various side effects, depression, heart attack. So if you somehow can manage to basically develop certain molecules or certain strategies that will be not basically having any side effects and which are much useful, that is basically the whole purpose of doing these kind of things. So this is what the fact is, that if we can just increase 10% of the BMD, then we can delay the development of osteoporosis by almost 13 years. So how can we do that? So basically, we have a number of strategies, and osteomicrobiology is one of the key there, which we are working on that. And we have already shown this with the probiotics, that there is a gut immune and bone axis. And if you can modulate this gut microbiota with healthy probiotics or prebiotics or symbiotics or AIP diet, the SCFAs and other molecules which are secreted by these probiotics can basically make the balance more towards a regulatory phenotype, with having more of Tregs and Bregs and less of TS17 cells, which can ultimately have a balance of your osteoclasts and osteoblasts. So these are some of the things which we are actively doing in a lab. So we have various trust areas of immunology of osteoimmunology of osteoporosis. Along with that, we have therapeutics, fertility. Also, we are looking how bone health and fertility is being affected, and how COVID-19 is also affecting bone health in the long term. So these are some of the areas which we are working on, and these are some of the recent contributions of our lab that we have established the gut immune bone axis in case of osteoporosis, and we have harnessed the role of probiotics for osteoporosis as a therapeutic strategy for management and treatment of osteoporosis. We have discovered and pioneered in Treg TS17 cell axis, and also recently this BREGs also. So now we say this is the BREG and Treg TS17 cell axis, which is very important in case of your osteoporosis. So I thank all of my PhD students and my lab members who have basically been involved in these studies, especially Lena. She's my PhD student, and I thank again all the funding agencies and collaborators in the study. And once again, I urge all of you to kindly love your bones and protect your future. With this, I finish up my talk, and the floor is now open for questions.
Wonderful talk, Dr. Rupesh. And since you have completed in time, we have 1 minute. So I think we can at least take one question. Are there any questions? So since there are, I think, no questions, maybe I will ask you a question.
Yeah, sure, ma'am.
So how do you think BREGs are regulated by the steroid hormones?
BREGs and steroid hormones. The steroid means which steroid basically you're talking about? The
well, osteoporosis occurs when,
glucocorticoid-induced osteoporosis. Yes. So we are actually now working on that. We have some preliminary data that they are able to inhibit the osteoclastogenesis in the case of glucocorticoid-induced osteoporosis also. And I would be able to share that in the future slides maybe, but they are having an impact. They can basically regulate that also. Because estrogen, in the deficiency of estrogen also, we are studying that what is the role of if there is no estrogen, so how that is affecting the regulatory B cells. And this is also, we are now the similar studies we are also doing in humans also. So we are doing such kind of things. So we have some data, but still that's not complete. So with time, I think we will be able to have more validated data regarding this. So that's a very good question, I would say, because that is very important. Because in the case of COVID-19 also, which we are working right now, because substantial use of steroids have already basically targeted these osteoclastogenesis and osteoblast also. And we are looking how basically COVID-19 is basically also affecting your bone loss in various other inflammatory conditions apart from your not only osteoporosis, rheumatoid arthritis, osteoarthritis. So they must have been aggravated. So we are looking into that angle also.
So thank you for this wonderful talk, Dr. Rupesh. And
ma'am, sorry, excuse me, we just have an announcement to make.
Okay, yes.
Yeah. So Dr. Rupesh Srivastava, along with Dr. Pradyumna Mishra, will be editing the next issue of ISSRF newsletter. And their topic is a kaleidoscopic view of advances in reproductive health research as India turns 75. So we look forward to your contribution. We request all the members to send their research articles for this exciting issue. So this is what we wanted to say, ma'am. Yeah. Thank you.
Yeah, thank you. Thank you, all of us, all of you.
So thank you, Dr. Rupesh. And I invite the next speaker, Dr. Vidhu. Could you connect, Dr. Vidhu?
Good afternoon, everybody. Yes, Dr. Malini, I'm able to connect finally. There was a lot of internet outage at my end. I was running both pillars to get connected.
Okay, anyway, that's great because at least we will get to hear you. So
yeah,
I'll give
a part of my work.
Yeah, I'll give a quick introduction. Dr. Vidhu is an assistant professor at Department of Anatomy, All India Institute of Medical Sciences. Her research interests are involved in unraveling the non-genomic sperm contribution, genomic integrity, and telomere dynamics in infertility and early pregnancy loss, along with impact of lifestyle intervention on sperm biology. She has received awards, several awards, Dr. Satyanarayan Memorial Young Achiever Award, the Corporate Medical Council in Training Award by American Society of Reproductive Medicine, the Lalore Foundation Award of American Society of Andrology, international travel awards, both by CSIR and ICMR. She has published 8 papers, 3 reviews, commentaries, book chapters, as well as abstracts. So, Dr. Vidhu, I invite you to deliver your talk, and we would be interested, obviously, in listening to yoga in early pregnancy loss, especially on the male reproductive angle. Over to you, Dr. Vidhu.
Thank you so much, ma'am. It's a pleasure to be presenting my work at this platform. So I'll just share my screen. Sorry, just a minute. Yeah.
So there'll be a buzzer at around 13 minutes. Okay.
All right, ma'am. Perfect. Thank you so much.
Yeah.
So good afternoon, everybody. And thank you for having my paper selection here. So I would be talking on yoga in early pregnancy loss, potential impact on the sperm gene expression profile. Early pregnancy loss is the loss of non-viable intrauterine pregnancy before 10 weeks of gestation. It is devastating when it goes into recurrence, that is in the recurrent early pregnancy loss, and also in the recurrent implantation failure in the IVF cycles. Various etiologies have been underlying which have been cited to predispose to the early pregnancy loss. There are a host of maternal factors, anatomical, hormonal infections, thrombophilias, endometrial factors, and all. We've always focused on the female factors for the same. But the contribution of the male factors cannot be negated. The contributions of the sperm to embryogenesis are more than just a mere vector for providing a haploid gene. The sperm transmits the genomic DNA, oocyte activation factor, the centriole, various transcription factors, but it also has the non-genomic paternal contributions in terms of the various mRNAs and the other non-coding RNAs being transferred to the oocyte at fertilization. The spermatozoa per se itself is a cell in crisis. Why? Because the sperm DNA is condensed into a nucleoprotamine structure. It is highly condensed. It is 6 times condensed than the somatic cells. It occupies a central region, the nucleoprotamine, whereas still in the periphery, there is a nucleohistamine structure, which is the one which is more prone to be attacked by, prone to oxidative attack. And the increasing oxidative stress in the spermatozoa predisposes to a DNA damage in the germinal. After fertilization, either assisted or spontaneous conceptions, and if this repair is not corrected prior to fertilization, this increases the mutational load carried in the embryo, leading to the pregnancy loss. Just got hung. Yeah, the non-genomic paternal contributions are the various mRNAs which are transferred. Basically, the spermatozoa is genome is transcriptionally inactivated, and the embryonic genome is only activated. Embryonic genome takes activated after the first cleavage division. These cells, the dysregulation in the sperm transcripts, the oxidative stress, and the disrupted genome integrity, they impair the embryonic development. Various studies, previous studies have shown that yoga and meditation has the potential to decrease oxidative stress, DNA damage, as well as a normalization of this gene expression. With this aim, the current study was planned to assess the impact of yoga in optimizing the free radical levels, reducing the oxidative DNA damage, as well as the normalizing of levels. Normalizing the level of the
DC members.
As well as normalizing the levels of the dysregulated sperm transcript. This was achieved by the estimation of effect of yoga-based lifestyle on assessing the oxidative stress, DNA integrity. We also assessed the sperm telomere length as well as the gene expression profile. This prospective single-arm exploratory study included 30 male partners of the couples who previously had recurrent pregnancy loss, as well as 30 male partners who have had experienced recurrent implantation failures in the IVF cycles. The parameters, the semen samples were assessed at the baseline pre-yoga levels, as well as 45 days after the adoption of a yoga lifestyle. And we assess our ROS levels by chemiluminescence, the DNA damage by sperm chromatin structure assay. The sperm RNA was isolated for the qPCR analysis of this panel of genes, as well as the telomere length was evaluated by with the sperm DNA. This is the details of, these are the various practices which were adopted in a session of yoga. As the controls, as the subjects were aged matched, there was not significant difference among the participants. These are the baseline parameters. We had also included 30 fertile controls also for the comparison. Sorry. At the baseline, the levels of the progressive motility, the sperm concentration, the ROS levels, as well as the DFI and the telomere length, they were found to be significantly different in the patients as well as with respect to the controls. Now we saw that from the baseline levels and after the adoption of the yoga, from the baseline to the post-yoga levels, we found a significant increase in the progressive motility in both the recurrent pregnancy loss and implantation failure. We also found an increase in the sperm concentration. There was a significant decrease in the level of reactive oxygen species, but it was surprising that the ROS levels were seen to decline as early as 10 days of adoption of yoga. We had actually taken the samples at earlier levels, also at 10 and 21, but this study I'm showing the expression at the almost 6 weeks, 45 days. And the results I'm showing are of the 6 weeks. Sorry. The DFI levels, the DNA damage, we did not find, it did decrease, but it was not significant, and it was not found to be significant in RPL patients, but significant in implantation failure patients. These are the dot plot cytograms of the done by the DNA damage with the flow cytometry and analysis of FACT scan offset, wherein the x-axis represents the fragmented DNA, and on the y-axis we can see the native the sperm cells with the native DNA. We see that it is of the same patient, which were the DNA and DFI was analyzed pre-yoga and the post-yoga levels. We can see the shift of the sperm cells from the x-axis to the y-axis, the improvement in the damage. The sperm telomere length, we also found an increase in the telomere length, significant increase in the length in both the patient category, and it was interesting to find that the sperm telomere length, it actually correlated negatively with both ROS and the DFI levels at the baseline. And it correlated both with oxidative stress and DNA damage, which actually predisposes to a decrease in the telomere length. For the sperm gene expression analysis, we selected a panel of these genes based on previous studies on the literature review. These genes were analyzed, and the housekeeping genes were beta-actin and GAPTH, and the samples were run in duplicate. Sorry, it's a duplicate slide. This slide shows the relative gene expression in the RPL patients. If I have to compare the pre-yoga levels with the control, I found a significant difference in the expression of a couple of genes from the control levels. But the gene expression in our study, we found the pre to post-yoga changes. There were pre to post-yoga changes, and the post-yoga levels were found to normalize towards the level of those controls. See, if most of cases, they were found to normalize towards the levels of the control values. But a significant difference was found, pre to post-yoga difference was found in WENT5A, TOM7, as well as PAP1 in RPL. Now, another thing which I found was, which we found in the study was that the initial axis, this is the axis fold chain by, and the axis fold change in the gene expression at the baseline. And this is a green one representing the axis fold chain post-yoga with respect to the pre-yoga levels. Like all the genes which were found to be upregulated were found to be downregulated. I'll be discussing further about the significance of how this mattered. They found the expression was changed. This showing difference in the RPL patient. We found a significant difference in OG1 and PAP1 genes, pre and post-yoga levels in the OG1 and PAP1 genes in the implantation failure. And all the genes which were downregulated showed upregulation, where the upregulated genes were found to show downregulation. And the levels, relative expression was also found to normalize towards the level of the controls. If I have to just go towards that, because the oxidative stress is the one, there are varying degrees of the oxidative stress which predisposes to the DNA damage. In our study, we found that the oxidative stress was increased. And I did not quote, sorry, I missed. There was a positive correlation which we found between the ROS levels and the DNA damage levels. While both ROS and the DNA damage, they correlated negatively with the progressive motility as well as the sperm concentration and also with the sperm telomere length. This is just to show that there are increases spacing of the DNA damage, like impending oxidative attack is in a graded manner. It causes oxidation of the bases and the mutagenic bases, the 8-hydroxydeoxyguanosine, to production of the A basic sites, protein cross-linking, and as well as the DNA strand breaks, both double-stranded and the single-stranded DNA breaks, which predispose to implantation failure, the pregnancy losses. Yeah. So in my study, now I'll just have a gist with what was the relevance of the genes that we studied. Now the FOXG1 gene is the one which is responsible for the early embryo patterning. It is a key regulator of neurogenesis. Actually, this gene was found to be upregulated in my patient population. It continues to be expressed in the neuroenteric zone, and the apparent upregulation of this gene is the one which causes a defective embryonic development in the neurogenesis part. This gene was found to be down-regulated post-yoga in my study. The SOX3, that is a sex-determining region of Y, BOX3 gene, it is caused for the regulation of embryonic development and the determination of cell fate. And also formation of hypothalamic pituitary axis. It is the one which is responsible for initiating the male sex determination. SOX3 was interestingly found to be down-regulated in both RPL and RIF cases. Both FOXG1 and FOXG1 was upregulated, while post-yoga I found a down-regulation. SOX3, which was significantly down-regulated in both RPL and RIF, showed an upregulation post-adoption of yoga. Now that these are ribosomal, the RPS6 gene, which is a major substrate of protein kinases. It is actually the primitive, causes overexpression of RPS6 gene, leads to hyperphosphorylation, which has been linked. It is actually the first substrate of the protein kinases which has been shown. It has been shown to the causes of many developmental disorders. The RBM9, it is a key regulator of the alternative exon splicing in the nervous system. It has been seen for the development of the central nervous system. And the RPL10A, was previously known as a NED6, that is a neural precursor cell express developmentally down-regulated, and it is actually down-regulated in neural precursor cells during the development. All three were found to be upregulated. And yes, this is important that initially what happened was in very primitive previous studies, because some transcripts were actually discovered and quoted to be transmitted to oocytes in 2005 by Ostermeyer et al. In their evolutionary study. It was a detailed study by Bonash et al. And Fernell et al. Who said that these three are the ones which are associated with increased incidence of miscarriages. In our study, we found all three genes were upregulated in recurrent pregnancy loss, while they were found to be downregulated in implantation failure. I found that these upregulated were found to be downregulated in post-yoga. Wnt5A responsible for intracellular growth and differentiation, whereas cellular regulation. Heat shock proteins, they're basically just cytoplasmic chaperones. And increased oxidative stress is the one which causes upregulation of HSP90 expression. In our study, we found an upregulation of both Wnt and HSP90 in our both patient groups, while they were down-regulated post-yoga. And this was found to be significant. There was a significant it was found to be significant in both the groups. TOM7, it is a translocase of outer membrane to mitochondrial membrane. And eukaryotic initiation, the translation initiated factor, the EIF5A. Both the genes are actually upregulated in my population. The TOM7 gene is the one which is also, it is a mitochondrial, it is associated with the mitochondria, and its expression is related to the increasing oxidative stress. We found an upregulation of these genes in both patient groups, and they were found to be downregulated post-yoga. Now, coming to the DNA damage repair and the detection and the repair genes, the OG1, it is actually the first enzyme in the base excision repair pathway. It is for the DNA damage repair gene. And the PARP1, it is for the DNA damage detection. That high PARP expression is actually high PAP expression is actually found in the mature spermatozoa. In our how OG1 damages the repair is that it excises the mutagenic base, that is the 8-hydroxydeoxyguanosine outside.
Dr. Vidhu, can you try to wrap up now?
Just 1 minute more, ma'am. So all these genes, what I found was that OG1 was upregulated and downregulated in RFF, and it was downregulated. It was on the contrary, the regulation was there. While the PAP1, which should be upregulated for the detection, was initially downregulated, and we found an upregulation of this gene post-yoga. So I'll just try to conclude that the derangement in the expression, genomic integrity, they carried over to the next generation. And yoga may alter the gene transcription, causing the modification at the level of the DNA methylation. And the integration of yoga may help to decrease stress, oxidative stress, DNA damage, and germline mutations. So in our study, we finally, I would like to just part a take-home message that adoption of yoga and meditation would lead to decrease in oxidative stress. And study, we already found that all these parameters were normalized. Thank you so much.
So thank you, Dr. Vidhu, for your talk. We will have 2 questions if time is there. And I see Dr. Pradeep has raised a hand. So Dr. Pradeep.
Hi, Vidhu.
Yes, sir.
So all those gene expression changes, you say that you monitor in the sperm from the patient, your subjects, right?
Yes, sir.
So but these RNAs, mRNA, so that would mean that all these messenger RNAs were there in the sperm, but there is no guarantee that they are translated by the sperm.
Yes, sir.
So yeah.
So, but there is a pool of retained mRNAs which are actually transferred, and they are the one which actually cater to the embryonic development. There is a suite of novel mRNAs in the spermatozoa which are still retained, and they are the ones which are responsible for the embryonic development.
Do you mean to say that these mRNAs delivered by the sperm will be translated in the oocyte?
Yes, sir.
Is there any evidence to say that any of these sperm-derived RNAs is translated by the oocyte, early oocyte?
Sir, it has actually been shown in the previous studies by that they are transmitted to the oocyte, and there have been evidence-based studies also supported by previous authors.
Okay, all right.
Any other question?
Hi, Dr. Dhawan. So it was very interesting talk. So what I wanted to know is whether do you think that other kind of physical activities like Pilates, something like that, can reduce the metabolic activity in the systematic way and may be beneficial as well as your study?
Sir, what kind of physical activities as such?
Like that is.
So your voice is cracking.
Is it clear now? So I have actually sent my question. So is it available?
I'm sorry,
Yeah,
I'm not able to.
I can just
hear you
tell
properly.
what he has written on the chat. He has asked, do you think other type of exercise or physical activity like Pilates may change metabolic activity in a systemic way, may be beneficial?
Sir, there are studies like, when I was reading about the yoga, there are other modalities also like Tai Chi and Qigong and all. I read about those things which have actually, they have shown to decrease oxidative stress. But I have not gone into the depth of whether they has been seen to show the decrease and affect the gene expression, and if there have been any epigenetic modifications have been, cited about the same.
Okay, so thank you, Dr. Vidhu. I think we will close your question answer, and people can ask you questions over the chat. Okay, so I invite the next speaker, Dr. Sanjeev Yadav. Is Dr. Sanjeev Yadav there? I think. Hello? Is Dr. Sanjeev? Yeah, he's there.
Is I'm audible? Okay.
Yes, you're audible. Yeah, so Dr. Sanjeev Yadav is working at BHU, and his research interest is nanoscience and technology. Although your title of the talk is very different from that. And he has been awarded the PAG Young Investigator Award by Photobiology. And he has got fellowships for attending meetings, and he has 15 publications in national and international journals. So, Dr. Sanjeev Yadav, over to you.
Okay. Thank you, Madam, for the introduction. Honorable chairpersons, colleagues, dear students, good afternoon to all of you.
You need to share your screen.
Yes, yes, Madam.
So you'll have 12 minutes if you want people to ask questions. And if you don't want any questions, you can speak.
No, I will try to wind up in time, chairperson, ma'am.
Yeah, okay.
Yeah.
And you might hear a beep at 12 minutes.
Yeah, yes, okay. And I'm also thankful to the organizing secretaries also for giving me chance to present my lab work. And I must say that this is my first presentation in this society. So, and some of my lab findings I am presenting here, madam. I will speak on a topic: continuous artificial lights disrupts central and peripheral reproductive clocks, leading to altered uterine physiology and reduced pregnancy success in albino mice.
Can you put it in presentation mode?
Okay. Is it fine, madam? Is it?
I can see that you have clicked that, but it is not fine. Is it fine for others?
Is slides?
No, ma'am.
Slides, it's moving or not, madam? I have shared complete slides.
Slide is moving, but it is not in presentation mode. So we can see the sidebar and.
If you can just do it in the slideshow mode, full.
Yeah.
Share entire screen. I think share entire screen.
Okay, okay. Then again, I'm.
Yeah, entire screen. Share entire screen.
More. Share.
Ma'am, maybe he's unable to do that. Maybe we can go ahead as we are running out of time. Yeah.
Share. Now it's actually, madam, I'm missing that.
Okay, it's okay. You can present with this. I think we can see, everybody can see the things. In between, if you can do it, it's fine. Go ahead.
Okay, okay, ma'am. So this is the image from the NASA Earth Observatory in 2020, and the scenario of the light pollution is increasing day by day. And in 2010, the widespread use of light was not much, and in 2020 the scenario has increased, and the widespread use of 24-hour light has become an alarming factor for coronal disruption related human reproductive disorders. And as you all know that life on Earth is adapted to the rotation of our planet, and for many years we have known that living organisms, including humans, have an internal biological clock that helps them to anticipate and adapt to the regular rhythm of the day. But question arises, how does this clock generally actually works? And for students, I want to emphasize that in 2017, for the Nobel Prize in Physiology was given for the discoveries of the molecular mechanism in controlling the circadian rhythms. But question arises how it works. It's the circadian rhythms, it's embedded deep within the brain, a master clock that is suprachiasmatic nucleus that inside the hypothalamus regulates the timing of many biological clocks, hormonal and behavioral processes such as sleep, metabolism, aging, and overall health. The molecular clockwork is composed of group of a core clock genes per cry, clock, and BML organized in a
Sanjeev, I think
transcription.
at least you have to move these slides also, the presentation, because you're just stuck up at once. That's what is visible, I think, to everybody. Okay, yeah. It's not in presentation mode, but you can at least move those slides. Yeah, now it is.
Is now, madam, my slides is moving or not not in presentation mode also? If I'm like in that
Yeah, it is
fashion,
moving now. Yeah, it's fine. So move like that, okay?
Okay, fine, ma'am. So the role of circadian rhythm in pregnancy was also recently being discovered. In the process of the evolution, also the timing of follicular ruptures and the steatogenesis may both be regulated by the circadian clock in ovary and act in the timing of the ovulations. It has been also known that the circadian rhythms clocks, they have been found in the process of fertilization also, and the report suggests that loss of the circadian clock function decreases the reproductive fitness in males. And in the process of implantation also, clock genes and their proteins rhythmicity expression within the luminal epithelium, stroma, myometrium region of the uterus have been reported. The process of placentation was also closely regulated by the clock genes, and circadian clock that regulates the process of trophoblast, proliferation, migration also have been tightly regulated by the clock genes. The process of the parturations is also in red report suggests is modified by the photoperiods, suggesting the role of these clock genes in the important rhythm in the pregnancy. But the question arises, what happens if we disturb the internal clocks? For the short time risks, it was reported that the behavior and physiology of the animals get disturbed, their sleep, alertness, metabolism, and the depression behaviors have been reported. And physiology, long-term risks like the pregnancy disorders, preterm birth, hormonal imbalance, and cancer, it was reported. But our main lab's focus was on pregnancy disorders. So despite knowing the importance of coordinated and synchronized timing of central and peripheral reproductive clocks in female reproductive disorders and infertility, the mechanism between clock coordination is poorly understood. Here we examine the effects of continuous light, prolonged light on clock genes, expression in the hypothalamic regions of the suprachiasmatic nucleus, ovary, uterus, along with the important downstream markers of uterine physiology such as estrogen receptors, hepatocyte growth factors, progesterone receptor, and HOXA10 that are important in the maintenance of uterine tissue integrity during pre and post pregnancy. And this was the experimental design. We have exposed the albino mice for 30 days in a constant light condition and the control light conditions. And we have divided the studies into two groups. First, the non-pregnancy studies, and the second one is the pregnancy studies. In a non-pregnancy studies, at this the 6 Z-Zever points, we have sacrificed the animals and noted down the various parameters for the non-pregnancy studies. And for the pregnancy studies, we have again divided our studies into prenatal studies, the peri-implantation studies, and the post-implantation studies of day 4, and the postnatal studies up to the offspring number. And these are the methods that we have analyzed for the work that we have performed in our lab. These methods we have followed. And what we found that the effect of the continuous lights on the estrous cycles. The continuous lights, they have, after exposing the animals for 30 days on the continuous lights, we have found that the average period length of the estrous cycle was not significantly affected, because the average period length of estrous cycles of this female was, in normal condition, it was 4, but not up to 5 to 6 days. The changes we have found, but that was not significant, or the effect of the continuous light on the estrous cycles of this female. Again, we have checked the effects of the continuous light on the histomorphology of the uterine tissues at different Z-zebra points in these animals. We have found that the constant light, the continuous light, have increased the luminal thickness of the uterine tissues, as you can see there in this histogram. We have checked the effect of these continuous lights on the clock gene expression in the hypothalamus also. We have found that the rhythms, the continuous light, they have altered the measure amplitudes and acrophase rhythms of the clock genes in the SCN regions. Similarly, we have checked the effects of the continuous lights on the clock genes in the ovary also. And as you can see there in the cosine analysis, the measure amplitudes and acrophase of these all clock genes was also altered there in the ovary after the effects of the continuous lights. We have also checked the effects of continuous light on the clock genes in uterus also. And in uterus also, the effect on the clock genes was reported after the analyzing with the cosine analysis, but not significant difference was observed there in an amplitude and acrophase of these clock genes. We have checked the effects of the continuous lights on the uterine, the hepatocyte growth factors and the serum estradiol levels also, and it was found that the level of the HGF was disturbed by the constant light, and the serum estradiol level was also changed as compared to the control group. We have checked the expression of the estrogen receptors after exposing the animals on the continuous lights, and we have found that the expression was upregulated as compared to that of the control mice, and the complementary results of the more immunopositive ER cells we have found there at the different Z-zebra points in the uterus. And for the pregnancy studies, we have checked the effects of the continuous lights on the serum progesterone level and uterine progesterone receptors and the Hox-Turner expression level. And we have found that the continuous lights, they have upregulated the expression of the progesterone receptors and Hox-Turner genes. We have checked the effects of the lights on the gestation times also, implantation frequency and the offspring number. And we have found that the continuous lights have decreased the implantation site number in these albino mice. They have increased the gestation time continuous lights. They have decreased the offspring number, and they have also decreased the total weight of the offsprings in the continuously exposed light conditions. So from these results, we may conclude that the artificial continuous light prolonged exposures for 30 days has disturbed the clock genes, master clock genes that was situated there in the suprachiasmatic nucleus, which alter the peripheral reproductive clocks that are located there in ovarian and uterine clocks. This disturbed or desynchronized rhythms in a non-pregnancy phase has disturbed the level of the daily estradiol rhythms, the daily uterine clock rhythms, and the rhythms of HGF, and the uterine pathomorphical changes was observed. And during the pregnancy, this altered rhythm of the reproductive biological clocks have upregulated the progesterone level, the progesterone receptors, and HOXA1 genes, resulting in a poor destabilization and overlap implantation and the postnatal. So this led us to propose that duration of light exposure at workplace or home for females is important in prevention of the pregnancy anomalies. So I would also like to acknowledge my PhD students and the financial assistance from the ISSRF, DST Serve, BHU, the Institute of Eminence grant, and the work that I am presenting here is one of the student work of my the PhD student Miss Megha Das, and I am also thankful to my the supervisor, Professor Chandana Haldar, for introducing me to this society, and first time I am presenting my work in front of this prestigious society. Thank you all for your kind attention.
Ma'am, you have to unmute yourself.
Thank you for the wonderful talk, Dr. Sanjeev. And I would open the talk for questions, and will encourage the younger generation, the students also, to ask some questions. Yeah.
Can I ask a question?
Yes,
yes. Yes, sir. Yes, sir. Yes, sir.
Sanjeev, interesting. So these rats perceive light through the retina or through the pineal gland, or both?
Through the sir, in this albino mice, the effect of the pineal gland was not much was reported, sir. So from retina we are expecting.
So in the night, even if there is continuous light, if the animal sleeps, there should be no problem, right?
Sorry, sir.
What I'm saying is, suppose you are exposing the animal to continuous light, but if the animal is closing its eyes, then it will not perceive this continuous light.
One thing I want also, sir, here mention that these animals, they are nocturnal animals, the albino mice.
Okay.
So,
so they won't sleep in the night. All right. Okay.
Yes.
All right.
And they are very good, the model for study of photoperiodic studies, sir.
All right.
Are there any other questions?
I had some basic thought to exchange.
Yeah.
I just wanted to know.
Yes, Professor Madan.
That we have a lot of information about this effect of light on reproductive functioning. And I was wondering what light is the direction in which the research is being pursued, and they are going to shed us to, lead us to. Why does light affect? If you are saying that it is the light, how and why does it affect? Because there's very rightly pointed out that the functioning of the pineal has long been defined. And there are two hemote. One is the hemote enzyme, and another is the pineal gland secretions. And on top of it, that we are talking of this light inhibiting about the cycle state is an indirect activity, which is because of the prolactin which is coming in here. We find that any stress situation, the light, and it results into prolactin increase, and prolactin inhibits the gonadotrophic activity from the pituitary. And that is the cycle which it has long been established. I don't know if there is some further information on this with the Honorable Speaker.
Thank you, sir. It was a very nice question, sir. In reference to the pineal gland, melatonin, and the gonadal axis, lot of work already being done. But in connection of the clock genes, which is recently being discovered, and after the regulations by the clock genes, which are located there in the central clock, that is suprachiasmatic nucleus, and the in reproductive peripheral clocks also, as we know that most of the female reproduction is very directly balanced by the timing of all the events, whether it's the paturation, the event of the implantation. If any signal, if any information is not rightly being sent to this, will definitely alter the, and lot of reports now have been have already in this direction are there, sir.
Are you talking of a thought, or you are talking of a process? You are aware of KISS proteins, the activity which the KISS proteins are having, and they have the
Yes.
role
In photoprodic modulation. Yes, sir, it is having.
So,
but in that connection, sir, we have not done any work. We are more focusing on the central and the peripheral reproductive clocks, sir.
Okay. Okay.
Thank you, Sanjeev, for your talk. And maybe I'll drop you a line about, I had some questions and some points where you said that the day of post-implantation in mice which was selected. So we'll have a discussion separately on that. Okay.
Yes, ma'am. Yes.
So thank you so
happy
much. Yeah. And I would like to thank all the speakers of this session for their wonderful talks, Dr. Yadav, Dr. Rosati, Dr. Rupesh, Dr. Vidhu, and Sanjeev. And so I hope all of you enjoyed these talks and are waiting for lunch for this to stop. So I formally close this session. Over to the organizers.
Thank you, ma'am, for wonderfully chairing the session. And what a great session we had. Since we are running late on time, so we are skipping the lunch and we are moving to the next session. I'm so sorry, ma'am, because I know it's going to be your first talk in that session.
All right.
Yeah, so we are going to our next session, and the chairpersons for our, so the next, sorry. Dr. Sanjeev, can you stop sharing your screen, please?
Okay.
Just stop sharing.
Okay. Okay.
Yeah.
I'm doing it.
Yeah, sure.
Okay, stop sharing option.
Okay. So our next session is on PCOS, and the chairperson for that session is Dr. Iss Sharma. And
okay, is it okay? Fine.
So Dr. Iss Sharma, yes, Dr. Iss Sharma is the secretary of ISSRF, and since we have already introduced him and we are running late on time, so the next chairperson is Professor PJ John. He's head Department of Zoology, University of Rajasthan, Jaipur, and Dr. John's research interest is on reproductive toxicology. So I would request our chairpersons to please chair this session. Over to you, sirs, please.
Thank you, Nidhi. Thank you very much.
Thank you, ma'am. Thank you for inviting us.
So this second session, which is on PCOS, is a very important session because the PCOS is one of the very important endocrine and heterogeneous disorder with the association of comorbidities. And the scientists, which I have the speakers, they are really going to give the very good talks on this topic. So with the permission of my co-chair, I would invite, without wasting the time, and introduce the first speaker, Dr. Malini Laloria. Dr. Laloria is a scientist G at the Division of Molecular Reproduction at Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram. And her research interest, I think so many people know, but most importantly, she is working on the PCOS, and I think that is very, very important. Embryo implantation program of the successful pregnancy, and she is also interested in understanding the pathogenesis of PCOS and type 2 diabetes. She has got a number of awards like Professor Govinda Rajlu Gold Medal Oration 2020, Dr. Jb Sarma Gold Medal of ISSRF, Lapteskar Award of ISSRF, Rain Visiting Professorship at University of Western Australia, Visiting Fellowships at Healthy Babies Program of NIH, International Postdoctoral Fellowships, MP Young Scientist Award, and so many others. I think she has a number of publications, around 107 publications to her credit. So with these words, I invite Dr. Malini Laloria to deliver her talk.
Thank you so much, Professor Sharma. And just let me share the screen so that. So at the outset, I would like to thank the organizers, especially Professor Lohia, Professor Sharma, for inviting me for this talk. Can you all see my screen and hear me?
Yes, we can.
Okay, so I will this time try to give a brief talk because hope I can give the 15-minute talk. Let's see. So I have been working on embryo implantation and PCOS. So this time I thought instead of showing a lot of data to you, I would talk about a molecule which is very important in immune regulation. And one of our very recent works, which is not yet published, it is under consideration. So this autoimmune regulator is a very interesting molecule, and it is dubbed the key molecule to regulate our autoimmunity and tolerance. This was identified actually as a locus underlying susceptibility to a polyendocrine autoimmune disease known as autoimmune polyendocrinopathic candidiasis ectodermal dystrophy, called as APECD in short. It is also called as autoimmune polyglandular syndrome type 1, which is lethal. It's a multi-organ lethality, and also an interesting thing is it's a monogenic disorder. So what I'm finding on that is it is easy to work on it because a single gene mutation in the single gene causes this disorder. It was first identified by Nagamine and the Finnish-German APCD consortium because this is more common in the Finnish population. And it is because of mutation in this autoimmune regulator gene. And around 1998, similar time, mutations were characterized in this gene in APS patients by She Lab. And I will come to it why I'm talking about the She Lab also. So, and then Dr. She cloned AIR from mouse homologue. And so they were one of the first group of people who identified AIR gene mutations in this Finnish population. He was the director of Center for Biotechnology, but has currently left and is founder of a company. Bijors et al. Showed that this molecule might be going to the nucleus, and they found it present in thymus, spleen, and lymph node, all the immune cells. Now, this molecule was known to be present in the nucleus, but it was not known whether it's a transcription factor. So it was found to be a transcription factor, and this work was done by Dr. Pradeep, and I was also part of that work, where we showed that AIR can bind to DNA and is probably a transcription factor. So the first paper showing that this autoimmune regulator is a DNA binding protein was a work of Dr. Jin Xiong's lab, where both I and Pradeep worked in 2000. So after this seminal paper, a lot of work came out, and now Deanne Mathis is one of the main person who talks about its role in thymocyte tolerization and its regulation of autoimmunity and clonal deletion. For student purpose, I will come to clonal deletion, what it is, to explain to you, and also to our people who are listening to me. And along with that, so Dr. Pat Peterson in Finland, in Tartu University, had knockouts of AIR as well as She Lab, and they found that there is a fertility problem in these mice along with the immune problem. Now, what AIR gene does is actually it stimulates promiscuous expression of all our peripheral tissue proteins in the thymus. This makes the thymus recognize that these are our own genes, and it causes clonal deletion of these. Let me just change the point. So it causes clonal deletion of all our known self genes, which should not be, so those cells should not be targeted. So if AIR is defective, you will have autoimmunity. And when I said promiscuous expression, all the genes which are required, this is just an example, it is important in that. Extrathymic expression of AIR also was noticed by all those people who did, but it was more in T cell and B cells. And then later, even in She Lab in the mouse, they found it's present in the spleen, heart, lungs, skeletal muscle, intestine, brain, eye, kidney, thyroid, adrenal, ovarian, testes. And while I was there, I always used to tell, why you ignored uterus? So after I came back to India, we did identify the air isoform in uterus, and the gene bank submission also is in our name. So air's role in reproduction was known because infertility was found to be common in these APCD patients. And there was primary ovarian insufficiency in these subjects, and male subjects had problems in testicular atrophy, hypogonadotrophy, azoospermia, and anti-sperm antibodies, as well as Leydig cells. Air knockout mice develop all features of APCD, including infertility, which is amounting to 85%, both in male and female. So air's role in testes has been studied, and major is Halonen et al. And Schaller et al. And Dr. Pradeep's lab, where they have shown that it is important in degradation of cyclin B2 in the testes, while air knockout females were shown to have immune defects, which is shown by Jasti et al. Now, as well as biphasic air expression is seen in early embryos, but till date, what was known is targeting on its immune role only, that it is causing an immune attack on either the sperm or on the ovaries, except for Dr. Pradeep's lab, where this was not an immune role. So air has both activating and repressive functions, and importance of air and embryo implantation was shown by us, and this has actually been very widely reviewed. So what we had shown is that air actually can bind to HOXA10 as well as BMP2 INS on their promoter, and thereby regulate these genes and cause gestulization. And if you silence air, you knock off pregnancy. So this we had published in 2018, and I just want to show from the student perspective, and also from Dr. Sharma is also there. So this is also of interest for clinicians, many clinicians, because many works which we do in our labs, we probably cannot translate it, but some clinician can go look at it and translate it. And I feel actually very, happy that our work was seen by clinician in Canada, and they have actually quoted in this entire review that based on our work, they changed the treatment regimen of APCD patient, and the first ever child from an APCD patient is born. So this gives immense satisfaction that you have been able to help in a different way. The science you did was used by someone, and a kid is born from that. And this was very much reviewed and centered in Frontiers thing. And so this is all our work talking about reduced embryonic development, implantation failure. And this is Jasti's work for air. Now, hormones as well as sex-dependent variation of air expression is known. And actually, we are working currently on that aspect, that air has 4 LXLL domains that are NR boxes, which will allow estrogen receptor interaction with air. Also, air can bind, ERE has air binding motif. So now what I said, that air has a very important role in tolerance. So in tolerance, one was promiscuous gene expression. The other is it has a role in Treg induction, as well as role of DC cells. I would like you to focus on this Treg induction part. And what happens is, if in the normal thymus air is there, as you will have tolerance, because the effector T cells negative selection is happening of them because of the promiscuous gene expression. If air is not there, negative selection doesn't happen, and you have autoimmunity and tumor rejection. Sorry. So both fetus-specific and paternally inherited Treg development, P-Treg and T-Treg development also is under control of air. So air is a regulator of FOXP3 also. So reports exist that air promotes Treg induction, and FOXP3 being the master regulator, and defects in FOXP3 also called a syndrome called as IPEX. I am sure many people have heard my talk on the low levels of T regulatory cells in PCOS. T regulatory cells are critical for embryo implantation and the sustenance of the embryo, because embryo has the paternal genes, so it's a semi-allograft. The mother's body has to tolerate the embryo to take it to full term. So this is regulated by FOXP3 and an expansion of the Treg population, which give tolerance. So we showed in around 2015 that in PCOS subjects, the Treg numbers are low. That would mean that could explain the reason why they have threefold risk of abortions. So PCOS patients, others also, after our work, there are other works also showing the same thing. Now, another interesting molecule is HIF, which is important in adapting to hypoxic conditions and is another candidate now considered to be important in autoimmunity. It is expressed in all immune cells, and the relevance of this molecule in our study comes from the fact that HIFs can regulate FOXP3. But how air, HIF and FOXP3 connect is not known. So what HIF does actually is, in a hypoxic condition, it increases Tregs. But in a normoxic condition, what HIF does is it promotes Th17 pathway and inhibits Treg development. So based on all these available literature, we base this kind of hypothesis that there is a link between air, HIF and FOXP3. And so we wanted to look at this expression of air gene in PCOS. Now, before I go to this, let me tell you an interesting thing is I looked for at least 2 years, I looked for mutations in air in PCOS subjects, and we could not get any mutation in PCOS subjects. But still, we continued our thought now and thought we look at the gene expression level. So as you all know, this is the first session, so I'll very briefly introduce and leave it to Dr. Ashutosh and Shravani to do the job of introducing PCOS in detail. But PCOS is becoming a global epidemic, and 2 in every 5 adolescent and 1 in every 5 adult women suffer from PCOS, and they have problems in conception, weight gain, cysts, low sex drive, irregular periods, male pattern baldness, high testosterone level, insulin resistance, fatigue, acne, mood changes. So it is classified as a metabolic syndrome also. And as we said, the major issue is that there is a LH-FSH dysregulation, which causes increased testosterone. And you might have heard of our works where we say that how circadian rhythm also can regulate PCOS. So now moving on into some new and interesting pathways. So we had blood. So this is on blood samples because we are looking at T regulatory cells. And we had our Rotterdam consensus where oligo or anovulation, clinical and biochemical hyperandrogenemia and PCOS by ultrasound was the inclusion criteria, while women with poor ovarian reserve, other concomitant drug therapy, any abnormality in liver function and androgen disorders or autoimmune disorders were excluded. And these are all the parameters which we have access. So interestingly, we did not find any air gene mutation in PCOS, but we could find that. So we were looking at the coding sequence. That is another thing. Maybe one should look at the promoter region, because we found that air expression was less in these PCOS subjects. So going after this, so when we looked at HIF expression, we saw inverse correlation. So in PCOS subjects, when air is low, what we see is you have an increased HIF. And we also analyzed the promoter of HIF and found a typical air binding consensus, this consensus which was given by us only in SHE-Lab. So air can bind to HIF promoter. So we think that air is probably a repressor of HIF. And we did chip assays to show that air is there. You can see that we did chip with air and you can see HIF presence. Now, when we analyzed it phenotypically, because you have now these four phenotypes A, B, C, and D in PCOS, and we see that actually air is downregulated in all the phenotypes while HIF is upregulated here. And so what we did is we thought, okay, we will silence air in PBMC, and which took some time because these are again primary cells. So we had to standardize the dosage of silencing concentration. And we could show that if you silence air, here, this is air is silenced here, and you can see that HIF goes up and FOXP3 comes down, suggesting that in the normal conditions, when air is there, it keeps HIF repressed so that FOXP3 is there and it is promoting TREG development. But in the PCOS condition where there is no air to hinder, you have a lot of HIF, and HIF goes to ubiquitinate, cause ubiquitination and degradation of FOXP3, leading to low TREGs. So what we therefore say that when low air is there, you have increased HIF causing FOXP3 degradation, reduced TREG, leading to autoimmune etiology. So we talk now about a defective air HIF1 FOXP3 axis in peripheral blood of PCOS. And this entire work was done by my two fellows, Dr. Betsy, who's a graduate student working on PCOS, and Dr. Ranjini, who is working on air in implantation, and now we are looking at its role in PCOS. I would like to acknowledge all the clinical collaborators, Dr. Sati Pillai, Dr. K. Jayakrishnan, without whom this work would not have been possible, and the support staff of both these hospitals. Obviously, funding from my institute was the entire, what we could do with this work, and all the facilities of our institute helped us do this work. And CSR and DST inspired for support of both the fellows. Dr. Ranjini is a pool scientist in my unit. This is our current group, and I think I will end a little early maybe, but.
Thank you, Dr. Malini, for really delivering your talk within the stipulated rather before that. Question from the audience?
Yeah, Dr. Sharma, good afternoon. Thank you so much, Malini. Wonderful talk. I enjoyed. Besides your air stuff, when you spoke about HIF, this hypoxia inducible factor 1, I was always looking at this factor from the climate change view, and all the cattle, buffalo, as well as goat. When you take them to a high mountain, how this factor varies. So it was very interesting when I really heard you corroborating it with the implantation and reproductive biology. Thank you so much for that. I enjoyed your lecture as ever. Thank you, Dr. Sharma, for giving me a chance to say 2 words. Thank you.
Thank you, Dr. Taru.
Thank you for kind words, Dr. Taru. Sorry, I think I cannot speak much because morning I had the session and I had some issues with the vaccine. So still I have some facial, I get tired on the
Yeah, but you look so gorgeous. We cannot find out that you have some issues.
Yeah, that's the thing. Looks can be deceptive.
Sorry for interruption, but is there any question? So that if no, then I would request Dr. John, my co-chair, to introduce the next speaker, please. Dr. John.
Yeah. Yes. Yeah. Thank you very much, Dr. Sharma, for giving me this opportunity to introduce the next speaker of the session is Dr. Ashutosh Halder from All India Reproductive Genetics, Molecular Cytogenetics, and the Clinical embryology and teratology. And he has been honored with many awards like Professor N.R. Muthukal Memorial Oration Award in 2021. Former, he was working as an assistant professor in SGPGI in Lucknow. He is an awardee of Dr. Subhash Mukherjee Oration Award of Endocrine Society of India in 2019, and he is a member of several national and national academic societies, scientific societies, and he has presented his work in several national and international conferences. So he has published over 90 papers of national and international repute, and I have an honor to invite him to give his top presentation on the polycystic ovary syndrome, associations with environmental, genetics, and epigenetic factors. So, Dr. Ashutosh Halder.
Thank you, Dr. John. Good afternoon, everyone.
Good afternoon.
I hope my slide is visible. Is it visible now?
Yes, it is visible.
So I must thank the organizer for allowing me to talk. And it's a very difficult post-lunch session. All are sleepy. Anyway, the research presentation here is on polycystic ovary syndrome, and I will be speaking on our work on association with environmental, epigenetic, and genetic factors with PCOS. So currently there is no other society, only everybody consensus, all the three societies accepted NIH phenotypic 2012 classifications of PCOS. And phenotypic classifications, there are three criteria. One is hyperandrogenism, it may be clinical or biochemical, an ovulatory dysfunction, and polycystic ovarian morphology. Phenotype A means all three are present. Phenotype B means first two are present, no polycystic ovary. Phenotype C is first and last is present, no ovulatory dysfunction, which is very, very rare. Phenotype D is last two present, no hyperandrogenism here. So hyperandrogenism definition, clinical hyperandrogenism, Freeman-Galloway score or more or equal to 9. But we have not considered by any society of acne, hair fall, acanthosis, voice changes, those things. Biochemical, there is no consensus, no guidelines from any of the societies. High testosterone, arbitrarily. But most of the publications, they found that cutoff of 0.6 nanogram per ml should be the criteria. FAI, free androgen index. Most of there is no guideline by any of the society. It's 4.5. Most of the authors recommended this. Ovulatory dysfunction, there is a consensus. Amenorrhea or oligoovulation. Menstrual cycle is more than 35 days and amenorrhea more than 182 days. Polycystic ovary morphology, this is also again there is a lot of fallacies because of strength, capacity of transducer of ultrasound machine and subjects of experience of the ultrasoundologist, and it should be targeted to include or exclude the polycystic ovary, otherwise it will be misleading. And lowest ultrasound machine that's used is 5 megahertz, and with 5 megahertz the number of follicles should be more or equal to 12 in one or both the ovaries, and of size 2 to 9 mm, and ovarian volume more than 10. We are working on PCOS for last 5-6 years, and we are working on biomarkers of PCOS. Then to find out the reason for discrepancy between hyperandrogenism clinical versus biochemical, then etiological factors associated with PCOS, mechanism of PCOS in association with endocrine disrupting chemicals, bisphenol A and advanced glycation end products. It's related to epigenetics, and role of genes of steroid biosynthesis pathway in the patients with mildly high basal 17 hydroxycholesterol. So this work is just started. So androgens, Regarding androgens, we have evaluated all level androgens, but we found that dihydrotestosterone is the best marker in Indian context, North Indian context. And we have published recently that paper, dihydrotestosterone, a potential biomarker of hyperandrogenemia in polycystic ovary syndrome, a case control study from North India. Here we got mean value of DST in cases 584 versus 257 picogram per ml in control. And this is highly significant. Then we also do ROC curve analysis, and we found 0.89 total PCOS cases, but 0.95, which is a diagnostic range of value of AUC in phenotype A cases. Then DST, so we concluded DST can be considered as the best available biomarker of hyperandrogenemia in PCOS patient. Then we also worked on AMH and anti-Mullerian hormone, and we found median value of AMH was 8.5 nanogram per ml in PCOS versus 2.5 in control, and it's also highly significant. High value, this is from mean plus minus SD. We got more than 85% cases with high value, and more than 90% cases in phenotype A and phenotype D. Maximum rather we got with phenotype D, which is without hyperandrogenemia. And best thing is that AMH is not affected by age, hormone intake, or BMI, or hyperandrogenemia clinical or biochemical. These are assessed by Spearman correlation analysis. Then AUC of ROC was 0.93, which is a diagnostic range. We can take as a diagnostic marker, and prediction model with using multivariate binary logistic regression, also maximum regression coefficient and weightage we found with AMH. It's much ahead of any other marker. So we conclude AMH can be considered as a diagnostic biomarker of PCOS, and in particular where ultrasound diagnosis or focused ultrasound was not carried out. And this paper is under division in IJM paper, a journal. So regarding environmental factors, we are working on bisphenol A, advanced glycation end product, and recently we included DDT phthalate also. It's ongoing. So this is bisphenol A. Bisphenol A, there was 50 controls and 118 cases. And if you see, the mean in control was 138.64, whereas in total cases is 1176. It's a very high value, nearly 8 times. And mean plus 2 SD, which is a cutoff for high value, is 441. And these differences of everywhere, it's highly significant. We also analyzed bisphenol A in relation to BMI, and we found that high BMI has a much higher value. It's at least 2 times than the normal BMI value in PCOS cases. And these are the AUC of ROC curve, and we found best with phenotype D, that is 0.86. Summary: ROC cutoff value 244 nanogram per ml. High level, that is 244. This is not the mean plus 2 SD, it's ROC cutoff. Higher was in 64%. AUC of ROC 0.84 in both total and phenotype A. Specificity was 90%, sensitivity was 64% total and 65% phenotype A. Spearman's analysis, significant correlation found between BMI and androgens. And logistic analysis, we found that patients with high BPA level have 8 times more PCOS in comparison to control. Then age, advanced glycation end product. Here we control number was very low, only 10. We have purchased another kit, that work is going on. We found that it's very high in phenotype A, around 77% than control. These are the phenotype A and phenotype D. ROC is 0.87 with phenotype A. It's a very good it's a very strong correlation. Regarding epigenetics and genetics, these are the genes and epigenetic mechanisms reported, published repeatedly, like epigenetics, methylation, hypomethylation of LH-HCG receptor, YAP1, FOXO3, and hypermethylation in CYP19A, prior GC1A, then non-coding RNA, miRNA 93, GLUT4, MIR. These are repeatedly long non-coding RNA. These are reported in the literature. Like miRNA, also miRNA A21, miRNA A93 and 320. These are repeatedly published. Regarding genetics, AMS genes, CYP21, PI3K, interleukin, saturnin, adiponectin, insulin receptors, CYP17A, then D1A, already reported several times. Phenotype A, specific C9orf3, DEND1A, ERBB3, RAB5, phenotype B, TOX3, steroid 5-alpha reductase A2, A1, HMG A2, phenotype C, THADA, SORT2, phenotype D, ERB4, YAP1, GATA4, NIL2, Jb TB16. These are already FSH receptor beta and FTO. These are reported. So what we did, now I'll come epigenetic factors in our analysis. We did global DNA methylation ELISA in 50 control and 122 cases. And we found that control versus cases, there was a difference and p-value was significant, 0.004, highly significant. So significant difference in global methylation we obtained. Most PCOS patients were hypermethylated. Phenotype A cases almost all hypermethylated, and no hypermethylation in phenotype D. And phenotype D cases mostly normal or some hypermethylated. So if you see the phenotype-wise epigenetics findings are different. So probably all the types are not homogeneous. There are differences there, in particular probably etiology-wise. Then we did 850K DNA methylation array through Illumina platform. And the findings here is, we have compared our data. There was 12 cases, control versus hypomethylation, that is phenotype D, control versus hypermethylation, that is phenotype A, then the hypermethylation versus hypomethylation cases, and control versus all PCOS cases. What we found in comparison one, that is control versus hypomethylation, one gene differential methylation. And whereas second comparison, there was no gene or no promoter, two promoter region was all differential methylation. And phenotype A versus phenotype D, one promoter was differentially methylated, and control versus all cases, there was no gene or promoter was differentially methylated. Comparison one detects CCL4L1, that is a cytokine chemokine regulatory gene, differentially methylated, it's hypermethylated. Comparison two detects two promoter for gene ENSG 0000271778, which is a long non-coding RNA that is hypomethylated, and another pseudogene which is hypermethylated. Then comparison 3 detects one promoter, that is the same as with comparison 2, that is differentially methylated, hypomethylated. Comparison 4, there was no gene or promoter. So all differentially methylated sites linked with over 100 functional genes, and it's because of this few number of cases, we are increasing the number of cases more. Then final analysis and pathway will be analyzing later on. Genetic factors. Here we did whole exome sequencing of phenotype A PCOS, 51 cases. And analysis we did in the following way. Pathogenic, likely pathogenic rare variant identified. Then we also looked for insulin and obesity related genes in this 51 cases, and steroid biosynthetic pathway genes in this phenotype A 51 cases. So first genes variants identified with PCOS. One gene is a GATA4, which is reported already in literature multiple times, but that is not a mutation genetic, but epigenetic differential methylation of GATA4. We found mutations most likely either likely pathogenic or uncertain origin in 32 cases. So this is not a less finding, it's a very important finding. We have to find out how it causes PCOS with these different mutations. So further analysis is required. Just to briefing this initial data. Then we got 2 cases with THADA mutation, and these are mostly deleterious mutation. Then TCF7L2, fibrin3, then FDFT1 in 3 cases, FEM1B 1 case, UCP2 in 1 case, SGTA in 1 case, NIL2 in 2 cases, mostly deleterious or non-deleterious few. Then we analyzed this data. There are also, we got rare variants, mutations, which is very, very rare in the allele frequency in the general population. And these are the genes, THADA, TCF, this is a different mutation, ABN3, HSD3 beta 1, DEND-A1A, 3 cases, SUVX, AMH also we got, FDFT1, FSH receptor, ADPOQ, Inhibin A, KRR1, SGTA, YAP1. See, many of these genes already reported in the literature mutation with the PCOS. We got these rare mutations, but pathological or likely pathological, that has been reported yet with these mutations. So we compared this variant. Next analysis was insulin and obesity related genes. And here we found 8 cases had this gene mutation, pathogenic or likely pathogenic variants. These are ghrelin in 5 cases, UCP1 in 1 case, UCP2 in 1 case, and insulin receptor substrate 1 in 1 case. And if we compared PCOS with these variants and PCOS without this any variant, we found that age-wise is no difference, but VMI there was a difference, and it is significant. Means with variant have overweight, whereas control without this was within normal. And testosterone level, MS level, there was no difference, but fasting insulin was very, very high with these mutations in comparison to without this variant in PCOS cases. So these are the initial findings. This work also going on. More cases we have submitted for whole exome sequencing, and data will come soon. So once 100 plus cases, we will publish this. Then these are the insulin and obesity related genes variants, the ghrelin mutations. See, single mutation C214C2A, altering protein leucine to 72 methionine in 5 cases, single mutation in 5 cases. So this cannot be a just joke, it's a real problem probably. So this ghrelin gene in PCOS is very, very important, especially with obesity. And this is also satiety center for intake of food also is related. UCP1 in 1 case, this is the mutation and this is the protein change. UCP2, this is C262C to T, and protein change is arginine to cysteine, and 1 case, and insulin receptor substrate 1, this is also 1 case, serine to glycine change. Next is steroid biosynthetic pathway genes. We got 10 cases with this biosynthetic pathway gene mutation, either pathogenic or likely pathogenic or uncertain. And here we got CYP21A2, cytochrome P450 family 21 subfamily A member 2, 5 cases. And STAR in 1 case, POR in 2 cases, and permanent cytochrome P450 oxidoreductase, HSD 17 beta 6, hydroxysteroid 17 beta dehydrogenase 6 in one case, ACRA 1CU3 in one case. And when we compared this mutation cases with PCOS and without this mutation PCOS cases, we found age, BMI, and testosterone. There is no difference between with or without mutation, but we found AMH much lower in this variant, with this variant than the without variant, and fasting insulin is not high. It's much less than the without mutation. So this steroid biosynthetic probably indicates a totally different type of disorder, and this is all phenotype A cases. I was not expecting this kind of results with whole exome sequencing. I thought I anticipated that phenotype D should be having with more AMH and this kind of phenotype B with the steroid biosynthetic pathway gene problem. But even in phenotype A, there are many cases we are getting already 10, and probably another 50 lot comes. It may be 20-25 cases. It's very, very high incidence probably. This is all atypical CAH condition, and this cannot be diagnosed by biochemical analysis. You require mutational analysis, rather whole exome sequencing. And this, if it is if you get 20 or 25 in out of 100 cases, then it's a very substantial number of cases related to this disorder, and this is treatable because steroid biosynthetic gene affecting this kind of problem, you can treat with supplement, and it will be totally curable if it is not very like overt CAH. And probably if it is a 20% or 25%, then it should be a routine test in future to screen with steroid biosynthetic pathway gene. And now we are working on this pathway genes with another rider. Those cases of PCOS with 17 hydroxyprogesterone level is higher. So probably we are expecting much more higher percentage in that. And these are the genes and mutations and protein changes with steroid biosynthetic pathway we obtained in 51 cases, CYP21A2 in 5, and C1174 G2A change leading to alanine to threonine at 392 position, and 3 number of cases. And another 2 C995 1 case, then star 1 case, POT 2 cases, but both are different mutation. HSD 17 beta 6 B6 1 case, and 1C3 1 case. So I think genetics also play a very high percentage of cases we are getting with pathogenic or likely pathogenic mutations, and probably we need to screen for genetics as well as epigenetics in future for PCOS. Thank you for your attention. I think I have finished in 15 minutes time. Okay. Sir, I'm over.
Dr. John, you are there? Hello, Dr. John.
Dr. Sharma. Dr. Sharma, can I ask one question?
Sure. Please, that's what I'm thinking to ask. Yes, if any question, please go ahead.
Very good study, Ashutosh.
Thank you, sir.
There's a study published in the medical news which has come in PLOS. Exactly the same what you have done. They have covered over 250,000 cases. Effect of genetics, epigenetic factors, and obesity on PCOS. These are the major disturbances in the reproductive disorders, particularly menstrual cyclicity. They have done a retrospective study in 250,000 cases. PLS one that has come day before yesterday, medical news quoted this.
Thank you, sir.
Are you aware of this? I don't know what they have studied in that, but they have definitely associated this thing, obesity, with PCOS, and reproductive disorders, primarily menstrual disorders.
Oh.
So I particularly look for literature when I write a paper. Otherwise, so many work to analyze that it takes a long time.
No, this came in medical news, so I get the medical news. That is why I could find this thing.
Thank you, sir. I'll go through that paper.
Okay. Any other question from the audience?
Very nice lecture, Dr. Haldar. Can I ask one question?
One question, please go ahead, Dr. Singh.
Very nice talk, Dr. Haldar, and very informative also. And I like the work which you have done on BPA, but I was not able to, capture that. What were the levels of BPA and how did you measure them? I mean, the units I could not understand. Is it in that table? So if you can elaborate, what is the level which is normal there, and what is the PCOS level of?
Anyway, I switched off that slide. Just I'm narrating. We had 50 control and 118 PCOS cases of all types. And we did BPA level by ELISA method. And there is very high sensitive ELISA kits commercially available. So what we found in control is around 100 something. With control 50 mean value. So we did control mean plus 2 SD and found out that cutoff of high value is 441. And in cases in total, we got probably 1,000 something, 1,700 approximately in total PCOS cases. So it's a nearly 4 times difference between control and total cases. So it's there is no doubt that PCOS cases are very extremely high value, 4 times than the control mean plus 2 SD cutoff. Whereas we also compared with high BMI and normal BMI. High BMI means anything 25 or more. So there also we found the difference is 2 times. 1,000 something with normal BMI and 2,000 something value with high BMI. So it's a significant difference and it's a clear cut like a textbook picture. We never expected that kind of, but it's a more than textbook picture. It's a 4 times than the control mean deviation. So it's a very obviously.
Thank you. Hello.
Just one correlation with the androgens. Did you find that most of these patients had high androgens?
We did correlation study. Yes, BPA we got correlation, significant correlation with androgen and BMI.
So
BMI wise already I have shown, but androgen wise I have not seen. But androgen also linked correlation.
Sorry for interruption now, Dr. Singh and Dr. Haldar, because of shortage of time. Now I would have to request the next speaker. Thank you, Dr. Haldar, for your excellent talk.
Thank you, sir.
Dr. Sribani, you are around?
Yes, sir.
Okay. So I would request organizer to upload the biodata of Dr. Sribani Mukherjee. And yeah, so Dr. Sribani Mukherjee, the scientist F at the ICMR, National Institute for Research in Reproductive and Child Health, Mumbai. And her research areas are reproductive endocrinology, female infertility, special research interest, especially for understanding molecular mechanism of ovulatory dysfunction with the male focus on polycystic ovarian syndrome, identification of biomarkers to predict oocytes quality and pregnancy outcome. She has a number of awards to her credit like ICMR Sodkanta Dingley Award, Baptistker Award, GP Talwar Gold Medal Award for ISSRF, DHR International Fellowship, Fellow of Frontiers in Reproduction USA, recipient of several grants from various funding agencies like DBT, DHT, DHR, and she has publication to her around more than 50 publication in very eminent and reputed journal. So with this word, I request Dr. Srivani Mukherjee to deliver her talk. Dr. Srivani.
Thank you very much, sir, for your kind introduction. And thanks the organizing committee for giving me the opportunity to share our work over here. So I will share my slides. Dr. Haldar and Dr. Laloria has introduced the polycystic ovary syndrome very nicely. So today I will talk about insight into the follicular defect in women with PCOS with special emphasis on angiogenesis. So it is a complex heterogeneous disorder where ovary is the main organ which is implicated in the PCOS, and the follicular growth is arrested at the pre-antral or the early antral phase, and the corpus luteum is also defective. So our lab is trying to understand what are the molecular pathology behind this follicular disturbance in the PCOS. For that, we carried out some high throughput study. We worked on the follicular fluid because it is not possible to obtain ovarian sample for research from PCOS women. So when these women undergo IVF for the infertility treatment, we can collect the follicular fluid and granulosa cells. And most of the study related to ovary we have done with these samples. So we carried out first the proteome analysis of the follicular fluid from the PCOS woman compared to the controls.
Sorry to interrupt. Your slides are not moving and it's not in the presentation mode. If you can do that.
From my side, it is in the presentation mode. Just a minute. There is some, my side, it is in the presentation mode. And it is moving here. I will try. I won't put in the presentation mode. Now you see whether that is moving.
No, it's not moving.
Yeah, it's still in the first slide.
Yeah, now it's second slide. Now it is second slide.
In the presentation mode. If I put in the presentation mode, I will try once more. Is it moving?
No.
Okay, so in the presentation mode, there is some problem. It is not moving. I will keep the slide like this. Is it okay?
Yes, ma'am. Please go ahead.
Right,
okay.
I think, yeah.
Okay.
Now it is moving.
Okay, sir. Thank you. So we carried out the quantitative proteome analysis of the follicular fluid, and we could identify 188 differentially expressed protein, which belongs to many processes like composition of basal lamina, COC matrix, involved in oxidative stress metabolism. And interestingly, we found many proteins to be downregulated, which are related to the angiogenesis. We carried out another study where we analyzed the methylome profiling of the cumulus granulosa cells of the women with PCOS. There also we found many proteins, many genes which are differentially methylated. They belongs to angiogenesis, platelet-derived growth factor signaling, VGF signaling, fibroblast growth factor. So both of our high throughput study indicate that there may be dysregulation of the angiogenesis in this woman. Angiogenesis is very vital for the follicular growth. The primary and the primordial follicle depends on the stromal blood vessels for the nutrient and hormonal supply. But once they develop into the secondary follicle, they develop their own blood vessels around the thecal layer. But the granulosa compartment are avascular. After ovulation, the follicle, once the ovum is released, the basal membrane break down, the thecal cell and the endothelial cells, they merge with the granulosa cell transform into granulosa lutein cells, and they form the corpus luteum, which is the most intense vascular organ in the female body. So if there is any defect in the vascularization or angiogenesis process in this woman, the growth of the follicle will be affected and also the function of the corpus luteum. Let me introduce you the players in the angiogenesis. Hypoxia is the main inducer of angiogenesis, which induces the transcription of the HIF1 alpha, which induce transcription of many pro-angiogenic factors like VEGF, PIGF, PDGF. They signals the endothelial cells to proliferate, then breakdown of the vessel lamina occurs, and then endothelial cell migrate, form the tube-like structure, and which is matured by the angiopoietin and PGF. Apart from these factors, pro and anti-angiogenic factors, there are anti-angiogenic factors which inhibit the formation of the blood vessels, and extracellular matrix, which also plays a very important role, which sequester all these growth factors. And when they are cleaved by the MMPs or plasmin, etc., they release these growth factors, which then bind to the respected receptors. When we start our work, when we look at the literature, there are hardly any reports on the angiogenesis in the PCOS. Doppler study showed that there is increase in the twofold increase in the blood vessels density in the cortical stromal region of the PCOS woman. However, the vascularization around the antral follicle and the perivascular blood flow was significantly lower in the PCOS woman. A very elegant study by Ma et al. In 2018 using a DHT treated mouse model showed that around the cystic follicle, the density of the vasculature is very low. So all this indicate that though there is an increased blood flow, ovary is pumping blood, but still the follicles are not receiving the blood, which help in the follicular growth. And supporting this, our proteomic study also indicates there is downregulation of the many important angiogenic factors in the follicular fluid of the PCOS woman. For that, we thought, let us look about this aspect. We recruited women from the IVF clinic who are undergoing the same stimulation protocol. Controls are the normal menstruating women, though are undergoing IVF due to the male factor or oocyte donor, and PCOS were diagnosed with the Rotterdam criteria. We classified our study participants. This is the baseline characteristic before starting the IVF protocol. Here you can see increased LH, LH, FSH ratio, increased AMH, etc. We also do the hormonal measurement on the day of ovum pickup, both in the follicular fluid and serum. The progesterone level are significantly lower in the PCOS woman, both in serum, in the follicular fluid and in the follicular fluid and serum of the PCOS woman. All the androgen-related parameters were significantly increased. This IVF parameters we get from the IVF clinic. Though the total number of oocytes and M2 oocytes are almost similar, though it's higher in PCOS cases. However, the rate of fertilization of the PCOS woman was significantly low, and also the grade 1 embryos. So as we got many of the angiogenic molecule present in the follicular fluid, we thought maybe the follicular fluid also taking part in the angiogenesis process. So we thought, let us check that using the Hubeck cell. We cultured the Hubeck cell in presence of the follicular fluid obtained from the control woman and for the PCOS woman. When we use control follicular fluid, you can see they form nice tube-like structure, but in PCOS, the tube-like structure is disrupted. We carried out scratch assay. When we use the control follicular fluid, the wound closure was significantly higher than when we use the PCOS. So it indicates that yes, follicular fluid can induce the angiogenesis, but it is this angiogenic potential is significantly low in women with PCOS. There was a report that the granulosa lutein cell or granulosa cell behave like the endothelial cells. So we also check that. We use the primary granulosa cell. We culture them in the matrigel, and here you can see the control granulosa cell. They form very nice tube-like structure like the endothelial cells. However, PCOS they form, but they are not that much good. So this was a very challenging experiment using the primary granulosa cell to do this experiment, but my student, she did quite nicely. And the other important phenomena of the endothelial cell, they can uptake or internalize the acetylated LDL. So we use fluorescently labeled acetylated LDL. Here you can see in the control, the uptake was significantly higher than the granulosa cell obtained from the PCOS woman. So this indicates that yes, endothelial cell has capacity to behave like the granulosa cell has a capacity to behave like an endothelial cell. So next we look at the expression of various factors in the granulosa cell follicular fluid. The VEGF is the key angiogenic factor. In the follicular ferritin serum, the levels were significantly high, and it was reported by other people also. But in the granulosa cell here, you can see the gene expression of the VEGF were downregulated. Interesting, HIF-F1 were also significantly downregulated, and hypoxia is a main inducing factor for the angiogenesis, and hypoxia induces HIF-1A. And by IF, we also shown that VEGF-A downregulated. The main receptor for VEGF-A, that is VEGF-R2, was significantly downregulated. This indicates that VEGF signaling is significantly low in the granulosa cell. The FGF2 is another important pro-angiogenic factors. Its level was significantly low in the follicular fluid as well as in serum. In the granulosa cells, its expression was significantly downregulated, and the receptor FGF1R2 were also downregulated. The angiopotens are important for stabilization and maturation of the blood vessels. The angiopoietin 1 is the main factor, and angiopoietin 2 acts as antagonistic of angiopoietin 1. Angiopoietin 1 is significantly downregulated, and the main receptor for that is Tie2 are also significantly downregulated. So the signaling, both the signaling of FGF2 and angiopoietin 1 were downregulated in the granulosa cells. Then we look at the expression of the endothelial cell marker. CD31 is an endothelial cell marker, which is expressed by the granulosa cell, but is significantly low in the PCOs compared to the control. And PROC1 is also another endocrine-derived vascular endothelial growth factor, which is known to be expressed in the ovary. We found, though it is expressed in the granulosa cell, but is downregulated in the PCOS1 compared to the controls. Now coming to the anti-angiogenic factors, SERPIN-A1 were significantly upregulated in the granulosa cells, but the other like thrombospondin and SPERP, which are the anti-angiogenic factor, they were comparable between the control and PCOS. The ECM proteins, which are also important for the angiogenesis, like FN1, HSPG2, ADAMTS, all were downregulated. Some of them we measured in the follicular fluid also. We found a similar trend like in the granulosa cells of SERPIN-A1, FN1, and HSPG2 in the follicular fluid too. Our interests were to see whether the expression of any of these angiogenic genes can influence the oocyte-related parameter, and we found that there is a significant positive correlation between the oocyte fertilization rate and the transcript gene expression of the VEGF, ADAMTS, then FGF2 and angiopoietin 2. In the women with PCOS, it is known that the oocyte quality is comparatively quite poor compared to the controls. So maybe these factors also partly contributing to the poor oocyte quality in this woman. So based on this, we propose a model. In PCOS, there is a defect in angiogenesis, which inhibit the growth of the follicle beyond the prenatal stage. And the second time point when the angiogenesis is very important is the formation of the corpus luteum. And if there is defective angiogenesis, it can lead to the compromised corpus luteum formation and functioning, low progesterone level, luteal phase insufficiency, and frequently miscarriages. And all these features are very common in the PCOS. So the follicular angiogenesis is dysregulated in the PCOS women. So we published this in the Human Reproduction last year. So however, as we work with the primary granulosa cell, it was not possible to look for many of the angiogenic factors because angiogenesis is a multi-step process and many factors involved. We could hardly see the main factors, but we were interested to know what are the other factors related to angiogenesis is differentially expressed in the PCOS ovary. For that, we have carried out the in silico analysis, and we also looked for their regulating microRNAs. We carried out the literature search using the PubMed, where we look for the angiogenic genes and the polycystic ovarian syndrome. There was 112 studies. Of that, we could include only 12. We could We excluded the reviews, annual study, cell line study, non-English study, in silico, and non-ovarian studies. And we identified there are 18 genes or protein reported to be dysregulated in PCOS ovary. Parallelly, we carried out a reanalysis of the microarray gene expression dataset of the 7 dataset which is related to the ovary of the PCOS woman, and we reanalyzed to identify the differentially regulated gene, which was 7572. Of that, after doing the pathway analysis, we could get total 87 genes which are related to the angiogenesis. So now we got total 99 genes. This is a partial list of the 99 genes. Then we wanted to know of this 99 genes, what are the microRNA regulating these genes. For that, we do a data analysis of TARBIS and TARBIS and identified there was 428 genes. But we don't know whether these genes are expressed in the ovary of the PCOS woman or not. For that, we again did a literature search and we enlisted all the microRNAs reported to be expressed in the ovary of the PCOS woman. Again, 202 study we got. We could include 56 studies based on the exclusion criteria what we have used earlier, and we got a total 874 microRNA reported to be expressed, not differentially expressed, but expressed in the ovary. We overlapped this 2 data set and we found there was 188 common microRNA. Then we carried out the pathway analysis and then validated few of the genes, and of this 188, 22 were already reported to be differentially expressed in the PCOS woman. So this is the list of the microRNA mRNA interaction in the PCOS. Then we carried out functional analysis of the dysregulated angiogenesis genes, and we found out that PI3 kinase signaling pathway was the most enriched pathway. Even when we analyze the microRNA, again PI3 kinase were the most enriched pathway. We have shortlisted 4 genes from this pathways, and then we fetched the microRNA mRNA interaction. This is the interaction network from this pathway, and of that we selected 1 microRNAs for the validation. And we carried out ddPCR for validation of this microRNA, and all these microRNAs, these 4 microRNAs were upregulated in the PCOS, and we have already shown that the target genes of the microRNA, the VEGF-A1, FGFR1 and FGFR1 were downregulated in the PCOS, and the other one, PTGF1, were shown by the other people, that is downregulated in the PCOS. The other 22 microRNAs which were already reported to be dysregulated in the PCOS but not related to angiogenesis, we fetched the microRNA mRNA interaction, and by in silico analysis we obtained 50 such pairs of microRNA mRNA interaction, and of that 33 pairs shows opposite trend, like when microRNAs are up, the gene are downregulated. So to conclude that, we have shown that the gene and protein expression of the angiogenic related genes in the granulosa cells and in the follicular fluid were significantly different in the PCOS and the control, and the angiogenic potential of the follicular fluid and the granulosa cells are compromised in the PCOS woman, indicating that the angiogenesis is defective in the ovarian follicular compartment, and that because of that, there is a functioning of the corpus luteum is defective, which leads to the corpus luteum insufficiency, low progesterone level, and the frequent miscarriages in this woman. We have prepared a data set of the angiogenic genes which are dysregulated in the PCOS and their related microRNAs, and we have shown that microRNAs plays an important role in altered angiogenesis in the PCOS woman. And if by the therapeutic means, if we can rectify or improve the angiogenic process in the follicles of the PCOS woman, it may help in the follicle maturation, its proper growth, then corpus luteum sustenance, oocyte fertilization, and also reduce the frequency of the miscarriages which faced by the PCOS woman. I would like to acknowledge that this is my team, my staff and student who are working all very hardly to give meaningful, produce meaningful data. And the work today I presented is work done by Kritika, who has recently submitted her synopsis and soon submit her thesis. And all my clinical collaborators, my funding agency, and all the participants in my study. Thank you very much for your patient listening. I'm open for taking any question.
Thank you, Dr. Sarbani. Now, is there any question from the audience to Dr. Mukherjee? Any question, please? If there is no any questions, then I must thank Dr. Sarbani Mukherjee for your excellent talk on this very important topic. Thank you, Dr. Mukherjee. Dr. Are around?
Yes, sir, I'm around.
Yeah. So please introduce the next speaker.
Yeah. Okay. Yeah. So may I request your organizers to put the CV of the next speaker?
Yes.
Yeah.
Did there.
Okay. Yeah. So, indeed, it is an honor for me to invite the next speaker, Seema Ram, and who has been doing his research on melatonin and its therapeutic potential in management of polycystic ovarian syndrome, diabetes, and in mammalian model, clock gene disruption during such metabolic and reproductive endocrinopathy, natural therapeutics for stress-induced immunologic, metabolic, and endocrine disorders. And Dr. Seema Rai, he's from Guru Ghazidas Vishwavidyalaya, Chhattisgarh. And he's been honored with many fellowships. His fellowship award 2020 is the Bharat Ratna Dr. Radhakrishnan Gold Medal Award in 2018 by the Global Economic Progress and Research Association of New Delhi. And he has been awarded with the S.L. Mishra Medal in 2018, Briards, Allahabad during 20th Indian Agricultural Scientist and Farmers Congress. He is an ISSA visiting scientist 2012 under bilateral program, University of Warsaw, Poland. And he has been awarded with many more awards, and he is a member of many scientific societies as well. He has published more than 42 papers in peer review at national and international journals and written few book chapters as well. Now, I invite Dr. Seema Rai to give his talk.
Thank you, sir. Thank you for your kind words as my introduction. First of all, I would like to thank to the all members of the organizing committee, and as well as the Lohia sir and secretary of this ISSRF for providing me this platform to share a few of my research findings. Today, I'm going to just let me share the screen.
जो हमारा टिप्स का कैट आता है ना वो 576 टिप्स का आता है 96 के हिसाब से और आपने क्वांटिटी ऑर्डर की है 1000 तो या तो आपको 576 लेनी होगी या फिर आपको 1152 लेनी होगी अगर 576
I hope my screen is visible.
Yes.
तो क्वांटिटी वो 1152 टिप्स से दी जाएगी
अब ये वहां तो पता नहीं चलता ना
हाँ जन
Okay.
तो या तो
So today the topic of my
नहीं वो 2 पैकेट दे दो बाकी का जो बिल आपका
interaction of melatonin and thyroid hormone receptor with estrogen receptor alpha and in letrozole induced rat polycystic
नहीं वहां पे तो
ovarian
आपको
syndrome.
उतना ही करो जो ये सेपरेट होगा ना
ऊपर जाएगा तो उसमें हो जाएगा
ये सेपरेट होगा ना
Dr. Seema, you are not audible.
She's not audible.
I think the organizers muted her. Actually, the disturbance, there was Dr. Rita's voice also was coming that time.
Dr. Rita's and others' voice were there, but now we can't listen her.
Yeah, so organizers have to unmute her. Probably she now doesn't have the privilege to unmute herself.
Please help her out because we can see
yes,
her
ma'am. Yes,
presentation.
ma'am. We are working on that. Please give us a minute. Dr. Seema, ma'am, if you are listening to us, please unmute yourself.
Yeah,
Yes,
yeah.
yes.
Thank you. So may I proceed after the first slide?
Yes, ma'am.
Yes, you can proceed.
So first of all, I would like to summarize my previous talk because previous work because that is related to the today's work also. Previously, as I am working with the PCOS model, and that has been induced by the letrozole injection, oral supplementation of the letrozole. And previously, my findings suggest that melatonin has restored and recovered the altered level of the biochemical parameter in terms of the lipid peroxidation and antioxidative enzyme, as well as the other related hormones and FSH, LH, and leptin. It was also noted during that time that the function of the melatonin as well as the plant extract has been mediated by the melatonin receptor MT1 and MT2 via few cytokines like IL-2 and IL-6, as well as ER alpha and IL-6 TN alpha. And these all have a cross talk with each other, and this orchestra of the receptor, they have restored and maintained the reproductive homeostasis. Now, the second part of my work is related to the two axes, that is the ovarian hypothalamo pituitary thyroid and ovarian axis. Why? Because thyroid disorder is very much prevalent among the females, and it can also interfere with the reproduction and fertility. And there are available evidences which suggest that change in the number of the Graafian follicle and primordial as well as the antral follicles are maximum most of the time associated with the thyroid disorder. However, the fallopian tube and uterine structure were noted unchanged. The relationship between the thyroid and the melatonin has also been reported by myself along with my supervisor, Professor C. Halder, when I was doing my PhD under her supervision. And further, a special group, that is the C cells of the thyroid, were noted to synthesize the thyroidal melatonin under the influence of the TSH. And this regulates the synthesis of thyroxine via paracrine mode of the action. It is also well established fact. The melatonin receptor subtype MT1 is also been identified on the thyroid follicle. Moreover, melatonin may have the direct effect on the hypothalamic pituitary thyroid axis that was reported in 2003 and 2002 and 2003 by a group of workers, that is Carbonic and Levinsky. Now, many things has been already discussed by Professor Malini, Professor Halder, and Sarbani madam during their talk. This is how we can presume about the normal and PCOS ovaries. They can lead a lot of alterations in the histological, biochemical, and other parameters. They can also alter the testosterone level, androgen level, which has been recognized as the hyperandrogenism. And they also lead to the fluctuation in the LH and FSH ratio. So that is, these are the markers which are being used to characterize the PCOS from the normal ovary. Now, what is the hypothalamo-pituitary, ovarian, and thyroid axis? How we can correlate these two things? The thyroid and ovary has direct effect upon each other. There could be three general options or types which can establish the interrelationship between these two. First, a peripheral sensitization of the tissue by thyroid hormone to the ovarian hormone. Second, an effect of thyroid on pituitary ovarian axis and or the vice versa. And third, an indirect effect upon the other glands, such as the adrenal cortex, which can affect the sexual activity. Therefore, it has been suggested that estrogen have their effect on thyroid by virtue of the shotgun effect upon the pituitary, thereby suppressing the release of thyrotropic hormone as well as to that of gonadotropins. This is the general principle of the working mechanism of thyroid hormone and melatonin via their receptor, because thyroid hormone acts via the cytoplasmic receptor and melatonin acts via GPCRS pathway using the MT1 and MT2 receptors via the second messenger and phospholipase C. And thyroid receptors, they use if they are free, they can bind to the heat shock protein to maintain its integrity, and then they can bind to the DNA sites to initiate the transcription, and therefore and then finally to the synthesis of the hormone. This is the next part of the slide, that a relation between the estrogen and the melatonin. Melatonin could influence the estrogenic action in three different ways. It could downregulate the gonadal steroid synthesis and consequently decrease their circulating level, or it may interact with the ER alpha, thus behaving as a selective estrogen receptor modulator. And third, it can downregulate the activity of some enzymes, such as the androgen, and involved in the synthesis of the estrogen from androgen, and therefore behaving as selective estrogen enzyme modulator. So this is how they have the relation. Now the next, I would like to, because the work is related overall with the receptor, that is why I am focusing all the things, first of all, with the receptors. The next part is the relation between DIO2 and TR-alpha. Deiodinase, that is DIO2, is a key regulator enzyme for the thyroid hormone action, and it converts T4 to T3. So I'm sorry. And this triiodothyronine binds to TR-alpha, then mediates the transcriptional pathway. The mechanism of thyroid hormone action in ovary mostly depends on the DIO2 and TR-alpha. So looking into this much of the information, the next step, that is objective, was to note the effect of exogenous melatonin during a combined thyroid ovarian disorder. Therefore, this study has been undertaken to elucidate the receptor-mediated neuroendocrine regulation of metabolic and reproductive function in the letrozole-induced rat model treated with a combination of melatonin and thyroxine. So this is my animal model, and this is the experimental design. Total 36 animals were taken, and the 6 animals were given the vehicle, that is the 1% carboxymethyl cellulose, CMC. The 18 animals, after 18 animals were kept in the with the treatment of the oral dose of the letrozole with a rate of 1 milligram per kg body weight. After the successful induction of the letrozole, they have been further divided into 3 groups: PCOS with the 6 rats, and then PCOS rats treated with the L-thyroxine, they were also 6, and PCO rats received a combination of T4 and melatonin injection. Next group, which were non-PCOs, were given only T4, and 1 has been only given the melatonin. The concentration of the thyroxine given to them was 25 microgram per 100 gram body weight, and the melatonin was 200 microgram per 100 gram body weight. This is the parameter and the methods which were done to achieve the objective, the hormonal analysis of melatonin, gonadotropin, estrogen, progesterone, testosterone, TSH, T3 and T4, and expression pattern of MT1, MT2, TR alpha, ER alpha, DIO2 by doing QRT-PCR. So first we have done the first part, that is the induction of the PCOS by observing their histological micrograph. This letrozole has been given for 21 days to achieve the induction process, and the confirmation has been done. As we can see in the histomicrograph, there was in the PCO group, there were 1, 2, 3, 4 cysts were present. So that gives a confirmation. And after that, we have started the analysis and further experiment. Now coming to the result part, first of all, the thyroid tissue weight. The PCOS significantly increases the thyroid tissue weight, and the injection of the L-thyroxine as well as the combination of the L-thyroxine and melatonin recovered. However, it was not a significant one, but they have covered as comparable to the control one. This increase in the weight could be related with the increase in the volume of the thyroid follicle. In the histology, we can see the restoration in the thyroid follicles and their colloids when the combination treatment has been given to the rats, PCO rats. Now, coming to the plasma melatonin and FSH LH concentration, circulatory concentration, very evidently it can see that during the PCOS, the melatonin level was very low. It was significantly low, and the therapy, the combination treatment or the T4 treatment to the PCOS, they have restored the circulatory level of melatonin up to the control level of the rat. The FSH and LH both has been shown to have altered during the in the PCO rats. As we know that the ratio of these two should be approximately 1:1, but here we can see very well that the ratio has been disturbed during the PCOS condition. But when the co-administration has been given, as well as in the thyroxine administration during, especially in the case of the LH, the restoration could be seen, but it was most evident during the co-administration of T4 and melatonin to the PCOS rat. Similar trend has been noted for testosterone and progesterone and estradiol regarding the co-administration. But one thing which was confirming once again regarding the retrodome criteria, that hyperandrogenism was noted because testosterone level was significantly high in the PCOS rat. However, the progesterone and estrogen both were noted very low in the PCOS rats. So the T3, T4 and TSH level in letrozole-induced condition, the decreased free T3 and T4 level as well as the TSH level was noted. The TSH level was noted, it was not significantly low, but the co-administration, we can see here that is as the earlier result were there, the co-administration of T4 and melatonin to the PCOS rats were restored the concentration level of these hormone up to the control level and comparable to the control rats circulatory hormonal level. This is the summary of all the hormones and thyroid weight as I discussed earlier. Now coming to the receptor expression, first of all, the MT1 receptor expression in the thyroid and ovary. The left panel is the thyroid expression. We can see that the MT1 expression in the during the PCOS condition showed higher in the thyroid, on the thyroid tissue, and the T4 treated group rat showed increase in MT1 receptor expression as compared to the control. Again, the MT1 receptor expression on thyroid tissue of PCO rats receiving co-administration of melatonin and thyroxine showed restoration and recovery to the level shown by the PCOS rat only, not with the control rat. So there could one thing is very clear, that there are the alteration in the expression of the melatonin receptor. However, when we come to the ovarian MT1 receptor, contrary to this, the ovarian receptor during the PCOS condition, they shows a down regulation in the MT1 receptor expression, and the treatment, the combination treatment, however, was even in the combination of the treatment, it was maintained, it was down-regulated to the control as well as the PCOS1. So the correlation can be established with this result, that there is something going on at the receptor level when this such kind of endocrinopathy has been there inside any organism, especially in the mammals. Now, the MT2 receptor expression. When looking into thyroid tissue of the PCOS, they show down regulation in the MT2 receptor expression in comparison to the control group. However, the PCO rats receiving the exogenous treatment of T4 exhibited significant upregulation as compared to the hypothyroidic and PCOS rat, and combination treatment of melatonin and T4 to the PCOS led to the significant decrease in the thyroid MT2 receptor when compared with the hyperthyroidic rat as alone, not with the control one. Coming then the ovarian MT2 receptors expression, the PCOS rat showed insignificant upregulation. So the MT1 was down, but when we talk about the MT2, it is upregulated during the PCOS condition. T4 treated shows significantly decrease. However, the co-administration of melatonin and T4 resulted in the decrease of the MT2 receptor expression as compared to the hyperthyroidic as well as to the control group of rats. ER alpha, the thyroid tissue of PCOS rat shows the significantly higher relative expression of the ER alpha in this letrozole treated PCOS group, and the T4 treatment to the PCOS group as well as the co-administration, no one has restored up to the control level. However, the T4 treatment was effective in the case of the ER alpha. Finally, the 2 receptor DIO2 and TR alpha, the ovarian tissue of PCO group, hyperthyroidic group, as well as the PCOS rat treated with the T4 and the PCO rats received the combined treatment of melatonin and thyroxine showed significantly higher expression of DI2 receptor when compared to the control and the PCOS without any intervention.
Sorry to interrupt, Dr. Rai, you have to conclude.
Sir, give me 1 minute. Only one slide has been left.
Sure, thank you.
Next to the TR alpha, the combination of the treatment T4 and melatonin has down-regulated significantly even to that of the control group of animals. So with all these findings, we may conclude that effect of various metabolic and reproductive modulator on melatonin production defines a bidirectional circuit between hypothalamic pituitary thyroid and hypothalamic pituitary ovarian axis via corresponding receptor present on thyroid, ER alpha, MT1, MT2, and ovarian tissue, TR alpha, DIO2, MT1, and MT2. This study provides new evidence suggesting that melatonin is acting like a buffering molecule, regulating not only the hyperthyroidism, but equally the reproductive endocrinopathies such as the PCOS. The result further establishes that such buffering action of melatonin might be mediated via its 2 membrane receptor, and the melatonin action were noted synergistically in thyroid and ovary tissue with T4 via autocrine, paracrine, and endocrine modes. So these in turn might be influencing the ER alpha receptor of thyroid and DIO2 as well as the TR alpha ovarian tissue to maintain the metabolic and reproductive homeostasis, thus keeping the animal reproductively healthy. In conclusion, this study objective was to understand the PCOS effect on thyroid, pineal, and gonadal function, and to identify the comparative hormonal profile, the expression of the hormonal receptor, and the histopathology across the different group to understand the physiopathology of the PCOS. So the result provide novel finding and add new knowledge in the field of reproductive and metabolic endocrinology. Further, there are the other parameters which has been done. They actually indicate that PCO is associated with the multi-endocrine alterations. And finally, I would like to acknowledge the different funding agency to accomplish this work. And this work has been mainly done by one of the PhD student, Hindol Ghosh. And these are the list of my PhD student. I would like to thank to all for the kind patience to hear me. Thank you.
Thank you, Dr. Seema, for your nice presentation and very good talk. And I participated in one or two, maximum, because we are already over the time. So, please, if there are any quick questions, please, Dr. Seema will be able to answer. So, if no questions, I must invite the next speaker. Dr. Sharma, can you introduce the next?
Sure.
Yeah. Thank you.
So, the next speaker is Dr. Sharangoda J. Patil. He is the Associate Professor at Department of Zoology, NMKRB College for Women at Bengaluru. His research interest is contraception, antioxidants, pharmacology, medicinal plant. And he
Excuse me, sir. Excuse me. Sorry to interrupt you, but I think Dr. Patil is not here today.
Oh, Dr. Patil is not here?
Yeah, we have been trying to contact him, but could not. So please conclude the session.
I think if he's not there, then I must thanks on behalf of my coach, Dr. John, all the speakers for very excellent talks they delivered on very important topic, and also to the audience for very interactive session. Thank you very much.
Ma'am, actually one speaker is not there, ma'am. So they have started the Oration 2 now.
Yeah.
Yes.
Yes, ma'am.
Thank you, sir.
They have given half an hour.
Thank you very much for your excellent company for sharing this session.
I will call.
Thank you, Dr. John. Thank you.
Yeah. Thank you, sir. Looking forward.
Yeah, thank you, Dr. Sharma and Dr. John. It was great for you to chair the session. Now we'll move ahead with our second oration, Dr. Mrs. Mridula Kamboj Memorial Oration. And for that, our chairpersons are Dr. VP Kamboj, whom we have met in the inaugural session. And the other chairperson is Dr. Kiran Curtoch, who is Distinguished Scientist Chair of ICMR at the Indian Institute of Health Management Research, IIHMR University, Jaipur. And her research interest is general medicine and therapeutics. I would like to request both the chairperson to please chair this session. Thank you, sir and ma'am, and over to you, please.
Thank you very much.
Thanks.
Kiranji, shall I tell something
sir,
about
please,
my...
please, sir, please.
Then you can start the session, isn't it?
Okay, sir.
Okay. Dr. Mridula Kamboj did her MBBS, MS in obstetric gynecology from Agra Medical College. And then she came to Lucknow and joined a Christian missionary hospital here, that is Fatima Hospital. That was a 20-bedded hospital at that particular time, and under her guidance, that hospital first in obstetric gynecology went up to 200-bedded hospital in 80, and then it became a general hospital, and she became its medical superintendent in 1991, from senior gynecologist and medical superintendent. And she was the medical superintendent of the hospital till her death, that is in January 2015. She was a well-known gynecologist, not only in Lucknow, but right up to Nepal, Gurpur and Nepal. After her death, actually, Mridula Kamboj was a dedicated gynecologist, a superb wife, and caring mother for her daughters, and she guided them both to the medical side. And the daughters, in her memory, initiated this award. Thank you very much. Now I'll request Kiranji to introduce the speaker and proceed ahead.
Thank you, Dr. Kamboj. It is my privilege to be actually co-chairing this session of oration of Madam Kamboj. I just add 2 sentences to what Dr. Kamboj has said. She was not only a very good clinician and a very good organizer, she was also a very kind-hearted person. Because she has done it from Agra, I know a little bit about her, and she was a very kind-hearted person, and she was a mother to all her patients. This was just, I wanted to add this, sir. I hope you don't mind it, sir.
No, thank you very much. Thank you very much.
Okay, sir. Then I would like to introduce this, the speaker today is Dr. Taruna Madan. She is actually, she's B Pharma and M Pharma PhD and head of a department of innate immunity at ICMR National Institute for Research in Reproductive and Child Health, that we have already heard a couple of lectures from that same institute of ICMR. Her main interests is they have pioneered the biomedical research on human collections and a family of immunoregulatory patterns of recognizing proteins and deciphered the relevance in the host defense. She has claimed many awards. These are the list is there on this, including the INSA Young Scientist Award and the others, GP Talwar Oration Award from the NIA, from the Department of Biotechnology. And I request her to please present her lecture. And her lecture, she will be presenting on soluble pattern recognizing collections are integral to the human reproductive health. I welcome Dr. Taruna Madan, please.
Thank you, Kiran ma'am. Hope I am audible.
Yeah, you are absolutely audible.
And thank you, Kamboj sir. It is definitely an honor. I thank from the bottom of my heart for selecting me for this prestigious award, and this motivates my team to contribute even more. And I would start sharing my screen. Ma'am, is my first slide
no, it's visible? Not. No, ma'am.
Okay. I'll do it again.
It's visible. I was seeing it.
Okay, sir.
You have to double click it.
Yes.
It was visible, but not in slideshow.
Yes, sir. I will put it in the slideshow mode, sir. Thank you, sir. Sir, is it in the slideshow now?
Yes, ma'am.
Madam, now it is visible to you. Dr. Mrs. Curtoch, madam. Dr. Mrs. Curtoch, madam, you are there?
I think this is visible to the audience. It's visible to us.
Yeah, but
visible, very much visible, Dr. Sharma.
Yeah, it's visible, but I'm not able to hear Dr. Curtoch, Mrs. Curtoch's view, because she is the chair. Dr.
Kamboj also we can confirm from sir.
Yeah. Dr. Kamboj, can you hear me, sir?
Yes, I can hear you.
Yeah. So, because we are not able to connect, I think, to Dr. Mrs. Curtoch. So, with your permission, I think, please grant the permission to Dr. Madan to start the talk.
Yeah, please do. But Dr. Curtoch was there. She did speak.
Yeah.
And she welcomed Taruna ji. Taruna ji can start the lecture.
Please.
Thank you, Dr. Sharma, and thank you, Dr. Kamboj. So as Kiran ma'am introduced, the topic for the day, soluble pattern recognizing collectins are integral to human reproductive health. So this is, I would be giving glimpses of the research that we have carried out since 2007, when I had set up the Department of Innate Immunity. Welcome back, ma'am. So we initiated the Department of Innate Immunity at ICMR NIRH, Mumbai, in 2007. So to this esteemed audience, I don't think so I need to explain the relevance of immune regulation in the performance of testis, spermatogenesis, and also during pregnancy, gravid uterus. Both these sites are called immune privileged sites. Now, there is a shift in the paradigm that initially people used to think that the testis and the gravid uterus, they are suppressing the immune system. But now we understand very well that the mechanisms are towards regulation of the immune response rather than immune suppression. And there is a balance between promoting the, promoting the progression of pregnancy and promoting the progression of spermatogenesis, along with the immune system being prepared for fighting the infectious pathogens. So there is a balance that is maintained in these organs, and that is why we call them as immune privileged. So I'll move to the next slide. So here we show that the immune privilege of the testis and immune privilege of the gravid uterus, the end result is same. You have fetal protection from maternal immune attack, and you have germ cell protection from immune attack, but the mechanisms that both the organs utilize are quite different. Molecular mechanisms, there are some molecular mechanisms that are similar, but most of them are quite different. And but the important thing is that you have immune regulation happening in both these reproductive sites. We do understand that the female reproductive tract as well as the male reproductive tract, the genital organs, are exposed to the genital pathogens, and therefore the epithelial cells that are lining these mucosas, genital mucosas, are fully equipped with the with a paraphernalia of immune soldiers, including the antibodies, including the antimicrobial peptides, which are, some of them are like defensins, and definitely the microbiota also plays a key role in regulation of the sterility of the genital mucosa. And what we understand is that there are immune regulatory molecules which are C-type lectins. Now, why we call them as C-type lectins is because they have affinity towards calcium, and presence of calcium ion is very, very important to maintain their structure, three-dimensional structure, as well as it is enabling them to recognize both the pathogen as well as the host cellular receptors. And there are various families of C-type lectin receptors and C-type lectin molecules. So you have soluble C-type lectins as well as membrane receptors, which are C-type lectins. And we chose to work on soluble receptors and surfactant protein D, as the arrow shows you, the one of the largest molecule on the slide. And that is the protein that we started working on. And the collectins are, I'll just move to the next slide. So the collectin structure, as you can see, you have an N-terminal region, the middle part is collagen region, and then you have a carbohydrate recognition domain where your calcium ions are required. And they are lectins, so they are going to identify the glycosylated structures on the pathogens as well as on the host. So the C-terminal domain is having the CRD domain or carbohydrate recognition domain or the lectin domain, and the N-terminal is required for oligomerization. So the more the oligomerized the molecules are, the better is the function. So here I'm showing you the three structures of the three family members of collectin family, and all three of them are soluble: mannan-binding lectin, surfactant protein A, and surfactant protein D. Now, mannan-binding lectin, as the name suggests, it is recognizing the highly manosylated structures. The SPD and SPA both were first discovered in the lung, as they were part of the lung surfactant, and it is known that they have a role to play in the maintenance of surface tension at the alveolar air exchange membranes. And these molecules then were later discovered that all the epithelial mucosal cells, mucosal epithelial cells are able to secrete surfactant protein A and surfactant protein D. I'll just take you back to the discovery of these molecules. So Dr. Bhattacharya first published in 1975 the two hydroxyproline containing glycoproteins from the normal animal lung lavage and lamellar bodies. In 1978, they published another landmark paper where they identified these glycoproteins from the amniotic fluid, and that was the first indicator that they could be relevant for reproductive health. And then in 1989, there was a discovery of surfactant protein D from Department of Pathology at Washington, St. Louis. And this was a discovery of surfactant protein D. And then Professor KBM Reid, Fellow of Royal Society and former Director, MRC Immunochemistry Unit, Oxford, their group purified, characterized, and cloned the gene for lung surfactant protein D in 1992. And same way, mannan-binding lectin was discovered from the human serum, which is actually coming into circulation from the liver cells. So liver cells secrete mannan-binding lectin, or MBL, and it has all the three molecules have a great role to play against infections. The mean normal serum concentrations of SPD, they range between 48.7 and 109 nanograms. For SPA, it is 25 nanograms per ml. And for MBL, it ranges from 2.5 to 880 nanograms per ml. So in MBL, you do have three groups, like they have, low MBL levels, middle level of MBL, and then high MBL levels. And all these three categories of individuals, they will show different responses to the for the immune regulation, as well as for the defense against the pathogens. So the low MBL level individuals are definitely more susceptible to infections. And there is an institute in Denmark who actually purify MBL and then use this MBL for therapy in the patients who are having recurrent infections. So as we were talking about surfactant protein D, the largest collectin, and as you see, the more the oligomerization, better the function. And you can see here the monomers form trimers, and then you have formation of hexamers, and then dodecamers, and then a fuzzy ball structure, which is even many times more potent. So the pattern, as I said, they have a dual role. On one hand, they are identifying the pathogens through their carbohydrate recognition domain. It inhibits an array of pathogenic bacteria, fungi, and viruses. On the other hand, it has receptors on the cells, and a very interesting and intelligent molecule, we must say. In 2003, Gadai et al. Had published a Cell paper suggesting that these molecules, when their CRD domain is not occupied by a pathogen, the CRD domain binds to inhibitory receptors on the macrophages. They had basically focused their research on macrophages, alveolar macrophages, and the CRD domain binds to an inhibitory receptor called CRP-alpha on the macrophages and puts them into rest. So these macrophages are put into rest, suggesting that there is no pathogen around. Now you can take rest. And then when the pathogen is around and the CRD domain is occupying occupied with the pathogen, the collagen region will bind to C1q receptor, the first complement molecule C1q receptor, and the CD91 and associated receptor, and then they are signaling for the activation of macrophages. They enhance the oxidative burst in the neutrophils, and they enhance the phagocytosis. So to summarize, it inhibits T cell responses, restores quiescence in activated lymphocytes and monocytes when pathogens are not around, enhances phagocytotic clearance, not only of the pathogenic bacteria, fungi, and viruses, but also the cells that are infected with them, the cells that are undergoing apoptosis, the cells that are undergoing necrosis. So a wide variety of roles these molecules are playing, and therefore very aptly their systemic and tissue expression is altered in infection, inflammation, as well as cancer, suggesting that they have a role to play in infections, regulation of inflammation, as well as immune surveillance of cancer. So here I show you the paraphernalia of receptors that these molecules work through. So as we know, the toll-like receptors, TLR2 and TLR4, these are LPS binding moieties, and these molecules, once they recognize LPS, they signal for NF-kappa B entry into the nucleus, and then signaling for various pro-inflammatory cytokines. Now, these surfactant protein A and surfactant protein D are known to bind to TLR2 and TLR4, and thereby stop the signaling that is coming through the LPS. So LPS is not allowed to bind to these molecules, and then the signaling of NF-kappa B is under regulation. So this is just one of the mechanisms I shared with you. And so just to give you an overview of what we have worked with respect to collectins. So when I joined NIRH in 2007, we put up an aim before us that we would like to decipher the role of these collectins in immunoregulation during HIV/AIDS, because we had some publications that were suggesting that SPD has an interaction with GP120 and therefore could be relevant for the protection against HIV/AIDS. With respect to pregnancy and fertility, we worked on preeclampsia, endometriosis, preimplantation loss, male infertility. And with respect to cancer, we worked on prostate cancer and breast cancer. And the aim was to see whether they can be useful for diagnostic and therapeutic applications. So I'll just give you very brief glimpses of the work, some of the work that we have done. So this was the information available to us from Let Larsen group at Denmark in 2004. They published SPD is membrane associated in epithelial cells of the cervical glands, as you can see in the first left-hand side figure. Then you have in 2B, you can see the squamous epithelium heavily stained with surfactant protein D, and the glandular epithelial cells are also stained. So they are expressing surfactant protein D in quite a large amount. Then we can see here the fallopian tube mucosa, mucosal epithelial cells. And then here we see the ovarian follicles. And here we see that ovarian follicles, the granulosa cells are negative for SPD towards the lumen, whereas the theca interna cells are positive for SPD. And then theca lutein and granulosa cells of the corpus luteum are positive for SPD. So the idea to work on, to discover the role, we also interacted with Dr. Uday Kishore, who was the person who has, for the first time, synthesized a recombinant fragment of human surfactant protein D. So what he did was that, as you can see here, the red box. So here the part of the collagen domain is taken, the neck domain for the trimerization of the molecule, and the complete C-type lectin domain. So this fragment was, recombinantly expressed by Dr. Kishore, and he did this work way back in 1996 when I visited Oxford University, and that's how I'm carrying this legacy forward, working on these molecules. So the first experiment that we did was in silico, along with Dr. Susan Thomas, who is our bioinformatics person. She is a scientist E and head Biomedical Informatics Center at our institute and an IIT alumni. And she helped us in molecular docking of surfactant protein D recombinant, as well as the CD4 and GP120 of HIV. So we knew that GP120 and recombinant fragment of SPD are interacting, but whether the site is going to have an impact on GP120 and CD4 interaction, because this interaction is the one that facilitates the viral entry. So we saw that there are common site of GP120 that interacts with CD4 as well as with recombinant fragment of human SPD. So this discovery led us to further experimentation, and Dr. Rishikesh Pandit, who is now a postdoctoral fellow at NCI US, was working for his PhD program with us, and he established the potent and broad spectrum anti-HIV activity of a recombinant fragment of human SPD, along with the most important thing, reversal of the virus-induced cytokine storm. So here I show you the microarray data. So we analyzed that when we are comparing the gene expression, when you are treating the cells with HIV, vaginal epithelial cells with HIV, and when you are not treating them, there is thousands of genes that are upregulated, and there are some which are downregulated. But when we treated RFHSPD and HIV together, pre-incubated them, and then exposed them to the cells together, we saw that most of the genes that were, pro-inflammatory and were induced by HIV were downregulated. So for example, you can see MyD88, here it is upregulated, here it is downregulated, MMP9, again, a pro-inflammatory thing which is upregulated by SPD, but it is downregulated here. So CD44, and similarly, so you can see what all is green on the left-hand side is red on the right-hand side, suggesting that SPD has ability to reverse the gene expression induced by HIV, a very important finding. And then the most important thing was that we wanted to develop a microbicide, a vaginal microbicide using RFHSPD. So we prepared a formulation, we evaluated its safety and efficacy in vitro as well as in vivo in the rabbit model of vaginal irritation, which is advised by the pharmacopoeia as a mandatory thing to perform. And we patented this formulation with ICMR's help. Another important experiment Rishi could do, at the fag end of his PhD, that we took help from Dr. Raina Fichurova, who had developed these human vaginal epithelial cell lines. So we approached her lab, and Rishi visited her lab to perform an experiment where we developed an ex vivo model of HIV transmission. Now, we wanted to develop this model because working on the primate model is very expensive and very time consuming. And so it was important to have some preclinical studies with respect to efficacy of the formulation in the ex vivo model. So here what we did was we took epivaginal tissue in the upper chamber, and the lower chamber we had put activated PBMCs, and on the top of the epivaginal tissues, the human reconstructed epivaginal tissues were either untreated, treated with HIV-1, and recombinant fragment of human SPD, and HIV-1 plus MALP2, which is a TLR agonist. We used MALP2 as a positive control, which will enhance the HIV transmission. So in this case, we evaluated these activated PBMCs for HIV-1 P24 antigen ELISA to know how much virus is transmitted, as well as we did epivaginal tissues for gene expression studies, which you can see the gene expression highlighted here. So this work clearly showed that yes, this formulation is effective, and we were able to inhibit almost 90% of the viral transmission from the epivaginal tissue to the activated PBMCs. And this work was published in Frontiers in Immunology. We worked with Dr. Ajit Yadav, who again joined us for his PhD program. We evaluated the expression of surfactant protein D, surfactant protein A, and MBL in early placenta as well as in term placenta. And most importantly, his work showed us that when there is inflammation, so when there is parturition happening, so what we did was we compared the placental tissues at the term and at the spontaneous labor. So we had C-section and the spontaneous labor cases, and we compared their placental tissues for expression. We found that the expression of these collectins were many-fold enhanced during spontaneous labor. And the same thing was observed when we compared the MTP cases, placenta from the MTP cases to the placenta from the spontaneous abortion cases, suggesting that these molecules have an important role to regulate the inflammation at the feto-maternal interface. We were getting some information from the other groups that SPA alters LPS-induced inflammatory response in the fetal membranes in the preterm birth mice model, and SPA secreted by mouse fetal lung signals the initiation of parturition. Then SPD is having high third trimester SPD levels in IUGR cases, high levels in the preterm infants, and then elevated levels in term human placenta of spontaneous labor and abortion. That the work I shared with you, Dr. Ajit's work. And then with MBL also, similar informations were available. Some important evidences came from the gene deficient mice. SP and SPD gene deficient mice show delayed parturition. And we also know that the levels of these three collectins increased in the human amniotic fluid at term. So carrying on the legacy of those findings from our predecessors, we tried to understand the expression of SPD in the murine uterus, and we wanted to understand the regulation. So we knew that surfactant proteins, both A and D, they have steroid receptor elements in them, and that is how when at the when the preterm babies are born, the mothers who are delivering these preterm babies, they are given corticosteroids so that the lung maturity can happen. So that shows that the surfactant protein expression in the lung goes up when the corticosteroids are there. So people had shown that in the promoter region of these molecules, the genes, you have steroid receptor elements. Now, estrogen and progesterone are also steroidal hormones, and therefore we had presumed that we may have a cross reactivity. But what we found was that these molecules have very specific estradiol receptor elements, progesterone receptor elements, as well as they have testosterone receptor elements. And I will come back to the male fertility in few slides. So here what Sharon has shown, that when you are in the estrus phase of the estrus cycle, you have increased expression, many-fold increased expression of surfactant protein D. And when we ovariectomized the mice and then treated them with different hormones, so estradiol definitely positively enhanced the expression. And when we went for E2 plus P4, the normal physiological way of hormone expression during the estrus cycle, we found there was a many-fold increase in the SPD expression. So this suggests that, you know, it must be playing a very important role, and that is why it is regulated. So we thank Professor Jeffrey Whitsett, who is chief neonatologist at Cincinnati. So he generated the SPD knockout mice, and we were kind recipients of the gift that he gave this to us. So we tried to study the estrocycling, the litter sizes, the pre-implantation and post-implantation loss of the SPD knockout female mice. And here you can see there is a prolonged metastress in these mice, and therefore conception of these mice was delayed, and also these mice showed reduced litter sizes. So a very difficult mice to breed, but we were fortunate that we could work with some numbers of mice to understand. So here you can see the litter sizes are reduced. And to understand why the litter size is reduced, we evaluated the pre-implantation loss and post-implantation loss by doing the serial sectioning of the ovary and counting the number of corporal lutea and counting the number of implantation sites that we see in the uterus. And here you see that almost 50% of the implantation sites were lost. And this was the reason for reduced litter sizes. And this definitely showed us that surfactant protein D has a very important role in the maintenance of pregnancy. So we went ahead with a human cohort, prospective cohort of pregnant women, 922 women to be precise, that we recruited with ethical clearance from our own institute, from Wadia Hospital, from D.Y. Patil Hospital, and Dr. Kavita Kale had joined us on this program for her PhD, and she clearly showed that there are surfactant protein A and surfactant protein D levels are low in the serum of women who are having early onset of preeclampsia, and most importantly, this was associated with the progesterone to estradiol ratio. And most importantly, when we classified the early onset preeclampsia patients into severe and mild, we found that severe patients had even more lower levels or even severe reduction of SPD, and suggesting that definitely this stands a merit to validate it as a biomarker for the early onset of preeclampsia. The idea here is that if we can predict early on the early onset preeclampsia, then we can give preventive management treatment, aspirin, low-dose aspirin treatment to these pregnant women who are at high risk for developing preeclampsia, so that you can prevent happening of preeclampsia in these women. And we are moving forward in this direction. She had also established this kind of result in the animal model of SPD knockout mice, where we had mated both SPD deficient male and female, and we saw that not only the litter sizes, the placental sizes were small, and the spiral artery remodeling that happens that contributes to preeclampsia is also inhibited in these mice. Let us come back to the expression of SPD, but in the male reproductive tract. And Sushma, who recently got married, Dr. Sushma Rokde, had worked with us on this project. And what she showed was, as Sharon had shown, that the progesterone and estradiol regulate SPD expression. She showed that developmental regulation is happening for SPD, and you see that the germ cells were actually positive for SPD expression. And we also showed that when the testosterone was suppressed using acylene flutamide treatment, the SPD expression went down. So it is very, very specific regulation of SPD expression in the testis by the testosterone. Most important thing, what we saw was when we worked with the knockout mice, that the testis size was smaller, epididymis was smaller, and we also found that when we are challenging these mice with lipopolysaccharide, I'll show you that in the next slide. So we had 10-week-old wild-type mice, which we injected intraperitoneally with 5 milligram per kg body weight of LPS. And you can see here by the yellow arrowheads in the middle, when the LPS treatment happens, the testicular immune privilege is breached. So the barrier that is created by the Sertoli cells is breached here, and you can see the gaps there from where the immune response can come inside the seminiferous tubules and can hamper the spermatogenesis process. And the important finding was that when we treated intraperitoneally these mice, which were LPS challenged, and their spermatogenesis was compromised with RFH-SPD, we were able to restore within 48 hours the spermatogenesis treatment, spermatogenesis process. So that shows that these molecules definitely have some potential to be exploited for their utility in therapy. We further worked, as we worked with the pregnant women cohort, we worked with the Indian fertile and infertile men cohort with genital infections and inflammation. We tried to evaluate their levels of SPD in serum and seminal plasma. And we found that definitely the group 2 and group 3, who are people with, who are men with infection and inflammation, whether fertile or infertile, they have very low levels of surfactant protein D, again suggesting that this molecule could be relevant for maintenance of male fertility. Most important thing, we wanted to know why these levels are low. We are seeing low levels in preeclamptic women. We are seeing low levels in women who are having silent miscarriages. The data I have not shown here, but they were also low in the infertile and fertile men with infection. So here we got a clue that in the infection and inflammation, the neutrophil numbers go very high in the reproductive tract of men, and these neutrophils secrete neutrophil elastase. And from the previous studies from the lung, people had shown that elastase is able to degrade SPD, and we found the similar thing, as you can see on the right panel with the yellow box. The first 2 lanes are the control, and rest of them are from the infected the men with genital tract infections and inflammation. And this clearly showed that the protein is getting degraded, and that's how you find low levels of these proteins in these men. And provide probably, if we can restore these levels either directly or indirectly by the steroid treatments, probably the infertility of these men could be reversed. As I mentioned in my first introductory slide, that surfactant proteins do have a role in cancer immune surveillance, and that was a discovery in serendipity. We were working, when I was at CSIR IGIB, I was working with pulmonary infections, and we were working with their interaction of surfactant protein D with the eosinophils, and we worked with an eosinophilic leukemic cell line, which was put into apoptosis by SPD treatment. And that was a discovery for us. And then we treated various cell lines, cancer cell lines, with SPD. But mind it that this concentration that we use for inducing apoptosis in these cancer cells is quite high than the physiological concentration that is present in the reproductive tract. And we found that say if the, so it is almost like 10 to 20-fold higher concentration that induces apoptosis in the cancer cells. So Gargi, who has submitted her thesis and is awaiting her viva voce, treated the prostate cancer cell lines PC3 as well as the prostate epithelial cells, primary cells, which were isolated from the prostate tumor explants that we got from Tata Memorial Hospital. And we showed that there is a molecule called GRP78 that is overexpressed in the prostate tumor cells, and SPD goes and binds to this GRP78, and therefore the GRP78 signaling is inhibited. Also, we know that several growth factors, for example, we have epidermal growth factor or EGFR. So the receptor of that binds to RFHSPD, and RFHSPD blocks the EGF-EGFR interaction and thereby stops the proliferation of the tumor cells and puts them into apoptosis. So I would end on a philosophical note. The more I live, the more I learn, the more I learn, the more I realize, less I know. Just to thank all my collaborators who have been part and parcel of this research journey. And also I would like to express my deep gratitude to all the patients who gave us their precious samples with which this work was possible. And definitely when you are putting your best of your efforts, you make impacts and your work is recognized. And we co-edited a special issue in Frontiers in Immunology, Odyssey of Surfactant Protein A and D, and I was one of the co-editors with Dr. Kishore, my mentor. And at this moment, we are co-editing a special issue of American Journal of Reproductive Immunology, which is entitled Clinical Reproductive Immunology, Indian Perspective, and that is putting India on the map of reproductive immunology. And this is my team, and I want to thank you all. The lower picture is from Immunocon 2019 that we had organized, and you can see Dr. Kishore and Dr. Chiplunkar and Satyajit Rath, who and who of the immunology field. Thank you so much.
Kiranji to say the final words.
Yeah, this was an excellent talk and a real journey through science and how from aspergillosis you went into reproductive medicine. Thank you very much, ma'am. Thank you. And all the best for future.
Thank you so much, Kiran ma'am. Thank you.
Congratulations, Tarunaji, for the award.
Thank you, sir. It's an honor.
Thank you. Yes.
Congratulations, Dr. Taruna.
Thank you so much, Dr. Rosanne.
Congratulations, Dr. Taruna Madan. Congratulations.
Thank you, sir. Thank you, Sharma, sir. Thank you.
Many congratulations,
May you get many more awards.
Thank you so much, ma'am. Thank you. The awards would come from if we are able to put up these products into the market, and that's what is now my last part of the journey, the last decade. I would like to dedicate to that we bring out these products.
Very good. Very good.
Last, say that is the beginning of the journey, not the last.
It could be the beginning of the journey for many people. Yeah.
Thank you so much, sir. That is real motivation. Thank you.
Many congratulations, ma'am. It was a great talk. It was a great and very informative talk. Just one request, ma'am. Can you just stop sharing your screen so that we can share your award?
Yes.
Thank you. Thank you, ma'am.
Thank you.
Yes.
Wonderful.
Excellent.
Thank you so much, sir. Thank you.
Thank you, ma'am. And I would like to thank our chairpersons for wonderfully chairing this oration. Now we'll move ahead with our next session, which is session 3 on endocrinology. And the chairpersons for this session is Dr. Kamal Bakshi, who is at Fortis Hospital, Greater Kailash. And Dr. Bakshi is the first Indian gynecologist to develop and standardize in utero fetal skin biopsy technique. Welcome, ma'am. And the other chairperson for this session is Dr. Roya Rosati, who is Medical and Research Director, Medical Health Research Trust, Hyderabad, and whom we have heard, she gave a wonderful talk in the session first. So we welcome both the chairpersons, and I would request you to kindly chair this session. Over to you, ma'am, please.
Thank you. I would request Dr. Roya to introduce. Please, Dr. Roya.
Yes, ma'am. My pleasure. My pleasure, ma'am. My pleasure. And we have the first speaker,
Dr. Deepa Bhatia from NIH, Mumbai, and her talk is on shift in paradigm of our basic understanding of FSH and FSH receptor biology, which is very, very important in reproductive biology. And she is one of the very great scientists of the country. She's been recipient of many, many awards, and we have been hearing in all day, ISSRF in many, in the last decade, I have been hearing her, and she is one of the best scientists. Dr. Deepa, please.
Yeah. हमारे ग्लास बियर भी थे, उन्होंने सिर्फ 4-5 ग्लास बियर हमें लिख के दिए व्हाट्सएप की, सिर्फ आपकी यही है, तो फिर मैंने वही रखवाए, बाकी फिर उधर रखवा दिए फार्मेसी के, लेकिन थे हमारी मैडम को फिर फोन किया मैंने 6:30 बजे।
Please mute your official things, please.
Hello, good evening, everybody. Can you hear me?
Yes.
Okay, and you can see my slides also. Thank you, madam, for a kind introduction. And basically, I retired from NIRH last year in July, and then I have joined the company Epigeneres with the aim that whatever research I have done, I should bring it to the clinics. And you may be surprised by the title that I have today, I'll be presenting. But the reason for this is because a lot of confusion exists because so many extragonadal tissues are reported to express FSHR, which is coming as a surprise. So as we all know, FSH, everybody knows it is produced by the anterior pituitary. And basically we understand that it acts on the granulosa cells in the ovary and on the Sertoli cells in the testis. It facilitates ovarian follicular growth and spermatogenesis, and therefore FSH is known to be central role in mammalian reproduction. Early follicular development, like the primordial follicles, are thought to be gonadotropin independent. FSH exerts an indirect effect on the germ cells by stimulating the granulosa cells or Sertoli cells to secrete growth factors, etc., which are needed for germ cell maturation. Besides this basic biology, we also know that FSH has a therapeutic potential and has been used for so many years to stimulate the ovaries in the assisted reproduction clinics, and also in men with the hypogonadotropic hypogonadism to treat infertility. However, recent times, if you will go through the recent literature, it is being suggested that all men with idiopathic infertility will also benefit by treatment of FSH, and that we should develop protocols to treat idiopathic infertility in men also with FSH.
Ma'am, sorry to interrupt. Can you increase your slide? It's not that visible. Maybe can you do it full screen? Yes.
Sorry, just enlarge it.
Yeah. Thank you, ma'am.
This is fine?
Yes, ma'am.
Yes.
Okay. So basically FSH was reported in 1930 and is central to mammalian reproduction. And despite we have been working on FSH for the last 90 years, there is so much more to learn about FSH, FSHR biology. The first slide here shows how the FSH docks with the FSH receptor. Blue is the receptor, and this is the FSH alpha and beta. And the main receptor, FSH receptor, which everybody has worked on, is the canonical FSH receptor. It has 10 exons. And this is the way it is present. It has a huge extracellular domain, transmembrane domain, and a small intracellular domain. People have all focused on this exon, canonical FSH receptor, but few groups have reported that the FSH receptor actually exists in 4 different alternatively spliced isoforms, which are shown here. This is the canonical FSH receptor, FSHR2, R3, R4. So the biological functions are known for FSHR1 and R3. R1 is the canonical one. It activates the cyclic AMP pathway, whereas FSHR3 is the growth factor type 1 receptor, and it activates the MAP kinase pathway. Now, what other 2 isoforms don't have biological functions right now known, but what I like to point out that if we work with primers and probes designed from exon 8, 9, and 10, we will only come to know what is happening to the isoform R1, and the others will be missed out. If R3 has any role, it will be missed out. So it is more ideal that we design primers and probes from the early part of the receptors so that all the isoforms get studied. However, I'll share now. So this is what confusion has occurred. So the questions that I'm asking on this slide is, does FSH affect both the granulosa and Sertoli cells only? Are primordial follicles really independent of FSH? So basically, I went through the literature very carefully, and it was in 1990s that papers were published showing that initial primordial follicle growth is gonadotropin independent. So this work was done on sheep and human ovaries. They isolated different types of follicles, and they looked for FSH receptor expression by RT-PCR. And they found no FSH receptor in the primordial follicles, etc. But the reason why they did not find is because they were making their primers and probes from exons 8, 9, and 10. At the same time, at the very same time, there were people who were reporting FSH receptors on the ovary surface epithelium and also in the oocytes. The only difference between the two groups was that this group was having primers and probes from exon 1 to 5, whereas these from, so it is the choice of primers from where you design has resulted in this misconcept that initial follicular growth is independent of FSH. There was another study which was reported by Roy and LB in 2008, which showed that primordial follicle formation in hamsters FSH is very crucial for this. So what they did, if you treat pregnant hamsters with anti-FSH specific antibody, number of follicles reduced from 25% to almost 2.4%. So FSH has a very important role in primordial follicle formation. And this suppression of these follicles was reversed by treating with FSH. They were completely able to reverse the process. And primordial follicles assembled very quickly when PMSG was given to these animals. Similar to the ovaries, even in the testis, there are reports available that besides Sertoli cells, even the germ cells are expressing FSH receptors. So I've listed those studies here. This also includes an article by Dr. Shade's group from NIRRH, who had reported FSH receptors on the spermatogonia of immature marmosets. So what does this tell us? That FSH may act directly on the male germ cells and also on it is expressed on the oocytes also. So this, I suggest that the role of FSH in ovarian and testicular biology is still not fully understood. So this is the work that we have been doing since 2012. So the first paper that we had published, I was really surprised to see that when we treat the mice with PMSG, look at this expansion of epithelial cells on the ovary surface epithelium. I was really surprised to see this. We published this, and there were so many follicles inside this cluster here, this one. Then we published, after that we took mice, we chemoablated them, treated with busulfan and cyclophosphamide. So all the follicles are gone. But this chemoablated mice, if we treat with FSH, the ovary surface epithelium becomes prominent. At higher magnification, you can see so much of proliferation in the surface epithelium, and the cells were taking a BRDU also. So this was surprising to us. We have published this work also, and then we learned how to isolate these stem cells from the ovary surface epithelium, and we found that these stem cells were expressing FSH receptor along with a stem cell marker, SSCA4. So the co-expression of SSCA4 and FSHR was really intriguing, that the stem cells co-express FSH receptor. Then my student, he had took an ovary surface epithelium smear and did FSH receptor expression study. And what he found was that the epithelial cells are totally negative, but the stem cells are positive for FSHR. He also did, besides looking at the protein, he also looked at the mRNA by in situ hybridization, and he found FSHR mRNA in the stem cells. And if we take these stem cells in culture and treat with FSH, we find a lot of stimulation of the stem cells by FSH, whereas without FSH, this kind of germ cell nests, etc., were not observed. So what we concluded was that FSH receptors are there in the stem cells, and that these stem cells, FSH receptors are functional. Similarly, in the testis, Hiren found that the stem cells which are expressing SCA1 stem cell antigen co-express FSH receptor. This is the co-expression data. He also found FSH receptor by in situ hybridization. And if we treat the mice with PMSG, look at the increase in the number of germ cells, and they all are expressing increased amount of pCNA when you treat with FSH. He performed Western blotting. So when he took only the Sertoli cells, he found this particular, we got this band, which is canonical FSHR. But when he took the intact testis, he could see these additional isoforms of FSHR. The antibody he used was against the common region, extracellular domain. So he was able to pick up all the isoforms by using that particular antibody. And this is one, and also when he did RT-PCR, he found that R3 is much more increased compared to R1. So it is R3 which is actually involved in stem cell biology and FSH. And this is my another student's data which is recently published. What she found was that if we treat mice with diethylstilbestrol, the neonatal life, they develop testicular tumors when they are 100 days old. And in these testes, the DES treated testes, FSH-R3 is almost 40-fold upregulated. R1 is not there, and these have testicular cancer-like changes. So what does this show? That FSH receptors are expressed on gonadal stem cells in addition to granulosa and Sertoli cells. There is a co-expression of stem cell markers along with FSHR, and that this FSHR are functional, and more important receptor isoform is the FSHR3. So I found this one paper published in Journal of Clinical Investigation. And this particular data was published, actually it was included in the supplement. But if you look at this testicular immunolocalization, by this is in situ hybridization. This is very interesting. If you see, these are dot-like cells which are producing mRNA here. And if you look at the red dots, red arrows, these look like small cells which are dividing here also. So these are actually the stem cells that we research on. But when this group looked at the protein expression, the protein was found in the Sertoli cells, see? So here, this group has not, just put this data in the supplement, they did not highlight this, but it seems that the mRNA for FSH receptor is expressed by the stem cells, whereas the protein continues to be expressed and is lately largely observed in Sertoli cells, etc. This was very interesting. So this is the work because there was a lot of disbelief that FSH are on stem cells. So Hiren used 2 different antibodies. We also brought a peptide from the company which supplied us the antibody, and pre-incubating the cells, the FSHR antibody with the peptide totally negated FSHR expression. So FSHR expression is definitely there. This is the co-expression of FSHR with OCT4, which is a pluripotent stem cell marker. And Hiren also studied increased FSHR expressing cells after FSH treatment, which were in the size range of 2 to 6 micron. So the stem cells are activated by FSH treatment, and they are increased in numbers, and they express FSH receptors. Please look at this slide very carefully. I found this and I want to share with you. This is a single seminiferous tubule of a mouse testis, which was treated with FSH, and it is now here showing expression of pCNA. See the large number of these small size stem cells. They are showing nuclear pCNA. Cells at the base are showing nuclear pCNA, but as these cells undergo several rounds of divisions, etc., the pCNA shifts to the cytoplasm. And eventually it is totally lost, because as these cells differentiate further, pCNA is no longer observed. Now, the confusion started coming when the people published that besides the ovary and testis, FSH receptors are also expressed in human endometrium, cervix, myometrium, placenta, etc. They did immunolocalization. The problem happened because when they wanted to do RT-PCR, they did not find FSHR. So that is where the whole doubt came on the extragonadal expression of FSH receptor. And so many papers were written. Is it really real? Whatever I'm showing you in this slide. So they all doubted this. They said, is the extragonadal FSH receptor really real? Is it? They did not believe it. And why they did not believe it? Because wherever they showed extragonadal FSHR expression, they said Western blotting data is not there. Antibodies are not good. They are not validated. Whether these receptors are really functional or not. And even if you are seeing at protein level, you are not able to see it by RNA level. So that really confused people and they don't believe in it. But if we compare this data in normal tissues with endometriotic tissue, this is something very interesting. In endometriotic tissue, FSH receptors are expressed. They are reported by mRNA and protein, both are present. Here you don't get RT-PCR, but here you get. And also they have reported that FSH receptors are functional. Why is it? Why this endometriotic tissue is showing mRNA and functional receptors? Because endometriosis basically is a stem cell disease. There are stem cells are much more increased in numbers. Therefore, they are picking it up by RT-PCR, etc. And the cells remain much more immature. Therefore, you see everything in these cells. Whereas in normal tissues, you will not pick up by RT-PCR or in situ hybridization. So what I have also thought a lot about this, but if these are the stem cells, they express FSHR mRNA, and then they expand and differentiate into adult cell types. Here the protein persists, but these cells don't require the mRNA any longer, so they don't show in situ hybridization results for FSHR because these cells will be continuously replaced by the stem cells. So FSH is basically having an effect on the stem cells, so this is what I wanted to share with all of you. Now, besides reproductive tissues, FSH receptor has been expressed on all the different kinds of cancers. FSH receptor mRNA, this is showing mRNA for FSH receptor in all different types of cancers in the body. But here again, there is a lot of disbelief because what people say, what the reproductive biologists suggest, they mention, they discuss that in normal cancer, normal liver, normal stomach, normal lung, you don't see FSHR. Why you start seeing FSHR only when there is cancer? And also, even if you see in the cancerous tissue, the adjacent normal tissue is not expressing FSHR. So that is why there are special issues written on extragonadal FSH action and they don't believe in it. But the reason why only this region is expressing the tumor tissue expresses FSHR at both mRNA and protein level, and normal tissues do not, because normal tissues contain very few stem cells. And cancer is a stem cell disease. So where the cancer stem cells increase in numbers, if you do a biopsy, you will get lots of FSHR expression, both at the mRNA and protein level. And even the adjacent tissue here will not show FSHR. So this needs to be understood and appreciated. Our group has also worked on the uterus. We have shown that the stem cells in the uterus also express FSH receptors. And we show different isoforms by Western blotting in the uterine stem cell fraction, which was enriched. We did RT-PCR and we found only FSHR3. R1 doesn't get amplified in the uterine tissue. And this is very interesting data that we took a mice, it was overrectomized, and we treated it with FSH. And this FSH treatment resulted in increased OCT4 expression. OCT4 is a marker for stem cells. So the stem cells were increased in numbers and OCT4 expression was increased as a result of FSH treatment. So we have also been working on mice where we expose mice neonatally to estradiol on days 5 to 7. One of the mice resulted in tumor-like uterus by 7 months. So my student Pushpa, she collected this uterus, the fluid also, and the uterine tissue, and extracted RNA from both of these. Look at the results here. Both the uterine tumor tissue showed increased amount of FSHR3, FSHR1 is not there, and this fluid, when she centrifuged, processed, nicely extracted RNA, showed almost more than 1,000-fold increase in FSHR. R1 is not seen there again. So this is what increased FSHR transcripts suggest increased number of stem cells. And these stem cells, because they are so much increased in this fluid, must be involved in resulting in metastasis, etc. And it is FSHR3 which is implicated in endometrial cancer, because cancer is a stem cell disease. Now, I have explained testis, ovary, uterus, but please, these stem cells are existing in all the organs in the body. And we have worked on the bone marrow also. In the bone marrow also, FSH receptors are there on the stem cells. R3 is more regulated by FSH treatment. And there is so much of activation of the stem cells in the bone marrow when we treat with FSH. This is a study we had done where mice were treated with 5-fluorouracil. This is normal bone marrow. This is after 5-fluorouracil treatment, total loss of blood cells in the marrow. Now, if I treat these mice with FSH, FSH enhanced hematopoietic recovery by 3 days, 72 hours. So this is with FSH, this is without FSH. So FSH was really helpful in enhancing hematopoietic recovery by almost 3 days. We have published this work also. So it is not only our group which is showing this kind of data. There are other groups also. There was a paper written, why are hematopoietic stem cells so sexy? Meaning that why hematopoietic stem cells are expressing hormones of reproduction. So very interesting. So they also found that when a woman is being stimulated with FSH for the ovaries, the number of stem cells get mobilized into circulation, etc. Effective mobilization of stem cells when the women are treated with FSH therapy. So I will like to summarize my results, which I've shared with you, that FSH acts on tissue-resident stem cells and progenitors in multiple organs. It acts on the stem cells besides granulosa and Sertoli cells in the gonads. It also has a direct effect on the uterus, on the uterine stem cells. And more important is FSHR3 rather than the canonical FSHR1. So these results, I think, call for a complete shift in our understanding of FSH action. And we need to pay more attention to alternatively spliced FSHR3. Okay. So basically, I have published whatever I have shared with all of you today. I have published as 2 reviews last year. And that is why Dr. Lohia got interested and wanted me to present this work today here. I'm really thankful to the organizers for inviting me for this. And to Hiren, it is most of his work at my lab. And what is the message of my talk is that we need to think of FSH, FSHR, and stem cells together. Thank you so much.
Dr. Deepa, I have a question. If there are no questions, because both of us, we are clinicians, yes. We need to know what is a clinical application of such a huge research you have done, and what is the role of these FSHR to the resistant ovarian syndrome in POF, in the other daily clinical problem and dilemmas. What is the thing which we can help the patients? Is there any way your research can be of any use to the society? What is the future plan?
That's a wonderful question, madam. I have to think more on this, but definitely it has a lot of direct relevance, clinical relevance, because when the doctors are treating women for hyperstimulation of the ovaries, you will be surprised they're actually stimulating the stem cells. Because many times a woman you treat with FSH, there are no follicles. She goes home, she comes back with beautiful follicles. Where did they come from? They have come from the stem cells which got stimulated by the FSH. And maybe this answer I'll think more and think about it and get back to you. But definitely it has a lot of relevance, especially because I've connected it with cancer, endometrial cancer, etc. So I'll get back to you on this answer.
Thank you.
Thank you, madam.
Dr. Roya, if I may comment on this, though I have a slide in my presentation also to answer this question of yours, that what is the commercial relevance of the work which Dr. Deepa Bhartia has done.
I did not say commercial. I did not say that.
I mean, which can be used for the society and the patients. That's what I mean, that whether it the product can come out.
There is a paucity of time. Rita, please listen to me. Madam Professor Kamal Bakshi is waiting to introduce the next speaker because we are running short of time. Ma'am, are you introducing Dr. Deer Singh? Ma'am, can you unmute?
She may unmute, please. We cannot listen here.
Unmute yourself.
Can you hear me now?
Yes,
First of all, I would like to congratulate and thank Professor N.K. Lohia, Dr. Radhesham, Dr. Ansari, and as well as Barkha. I would say today has been the most wonderful day because it is the science that has brought so many facts, and if science doesn't exist, the knowledge doesn't come, and we are not able to treat the patient and help the society. So long live the research. And it was so nice that even the younger and the seniors brought the unique and a novel research from a country which is rich. So may God bless all the scientists so that we form a team, the researchers and the clinician. And Roya asked a very relevant question. And Doctor, I would say, Deepa, you raised certain or tinkled my brain cells, but we will discuss later. Thank you. It was a wonderful talk.
Thank you. Thank you, madam.
Now I would like to introduce Dr. Dheer Singh, and I would say his research interests are varied, identification and analysis of the genes, application of microRNA as the interference tools for livestock, and functional genomics, epigenetic regulation, gene expression. He has so much of research interest, and he is the fellow of the National Academy and the fellow of the National Academy of Agricultural Sciences of India. He has been awarded the fellowship from the Department of Science and Technology. Wow, Ministry of Science and Technology. He has published many papers which are reviewed, book chapters, and has been given many awards. But we are interested, sir, in the great research that you are going to give to us and highlight, He will be speaking on delivery upregulation, proximal promoter, IT-driven gene expression, and increases estrogen production through estrogen receptors for granulosa cells. Floor is yours, Dr. Dheer Singh. We are looking forward to your wisdom and share to help our patients.
Thank you, madam. Can you see my slides?
Yes, sir.
Okay, thank you very much. Thank you, Chairperson. And as outset, I would like to thank the ISSRF organizer and particularly the Professor Lohia and his team for giving me the opportunity to share actually some of my results. Well, as the chairperson has already highlighted, I have a diverse research, but sometimes the research is coming from some of the shoot off. So basically, I'm actually working on some genetic and epigenetic regulation of some fertility gene. And our gene of interest actually, as far as ISSRF member is knowing that's a CYP19, which regulates the estrogen. So estrogen, if you look actually, the estrogen is a very wonderful and outstanding, because the estrogen regulation is a female fertility is concerned. This is the most important, actually, the hormone, which is very good also, and sometimes it is very bad also as far as the hormone. So our interest actually 20 years back actually come into the understanding how the gene is regulated. And I came to know not only this particular estrogen, because I belong to the livestock and our model is a buffalo. So here actually, if you look, the estrogen abnormality is going to be lead to the several actually the diseases and the abnormality into the female reproductive system as far as buffalo is concerned. And similarly, I think because these not only the problem into the animals, but that's also into the human, because the uterine infection is very common. And I have several actually the paper we publish actually how the estrogen actually regulated under uterine infection. Late maturity, placental retention, delayed heat, and apoption. This is one sort of a different name, but it is also regulated the human, because CYP19 is a very, very common, and this estrogen is regulated by the CYP19 gene. And CYP19 is a very wonderful actually the enzyme gene, because this is one of the gene which is regulated at the transcriptional level. I think it is a nature make it, because most of the actually the gene or the protein or enzyme which is secreted and absorbed. But here is the most of actually this is regulated at the transcription level. So if the protein is formed, it should be normal, and that's why for the fertility should be restored. But the transcription regulation is the different promoter. So I will share it here. And to understand this, actually, whatever we understand actually CYP19 gene regulation is concerned, we have developed a couple of model actually 10 years back actually developed model. And our granulosa cell model is exactly copied to the in vivo system. And why I'm talking about the in vivo system, because this model I developed along with the Stanford actually the laboratory. They have actually the gene regulation at every hour actually after the PMSG and LS given. So they have a 60-hour data. So if you give the PMSG, what the granulosa cell dynamic is going to be happen, and when from estrogen to the progesterone is shifting, what are the gene is going to be up and down, so that the estrogen actually regime can be shifted into the progesterone regime. So we have actually validated into our granulosa cell model, because bovine granulosa cell model is mostly actually the human, whatever the research is came, it is based on actually bovine granulosa cell model, because we have to bring from slaughterhouse. So we develop a model, and that model actually give rise to exactly what happened in vivo system after the FSH and LSR. And that's why this model has been curated by NIH for the gene and chemical interaction. So based on this model, and we have also knowing that how the CYP19 is regulated in buffalo in our system model. What I was talking about, this is regulated at transcription level, means the different transcription is going to be regulated and different. For example, in ovary, where in granulosa cell, because the in ovary granulosa cell is the only cell which synthesizes the aromatase, express the aromatase, and estrogen is synthesized with the substrate from the endotheca cell. So here the promoter 2 is going to be expressed. But in case of the, as far as luteal tissue is concerned, the promoter 2 is going to be shifted into the promoter 1.1, which is a very proximal promoter. So these all paper is published, but interesting is that if the granulosa promoter 2 is going to be upregulated, this could be a problem, because the upregulation of estrogen in breast cell as through promoter 2 is going to be a cancer. So I was, after the basic understanding, I was looking for the different actually, like I have already explained that under uterine infection, under cystic condition, how the estrogen is regulated and what could be actually the molecular mechanism we have elucidated at this, and this is also we know. So here, what the piece of work, what I'm going to present it here, because nowadays, just like in morning actually under the, this inaugural, our honorable Dr. Kamboj has said that our population is going to down. So we should not worry about the population, but the fertility down is going to be happen. And that's because of several problems. I think it is well established. One of them is endocrinology, endocrine disruptor. And as far as, but because our institute is a dairy institute, and we are also know that milk is also very, people have know that a lot of contamination is concerned. Because in case of our, because whatever we do actually in toward the nation actually as far. So you might be knowing as far as the green revolution is concerned, a lot of pesticide has been used, but that was a need for that time. But today we need actually as far as the clean milk is concerned, and our theme is the clean milk. And we have shown, and it has been observed, and that is a concern of FSSI, that there is a lot of actually pesticide is coming, because most of the pesticide, they are lipophilic in nature. They can cross the membrane. So in milk, actually, that is one of the pesticide, the organochloride pesticide is very common nowadays. I'm not going to share actually what is the status of national, but what I'm saying is that in the milk, it is present, and through milk, it goes to baby, not only to us in adult. And it has been also reported that in follicular fluid, organochloride is present. So once it is exposed to the granulosa cell, what is going to happen? So I'm going to be share, because my purpose was actually, our purpose was to granulosa cell has the receptor, and they are working through estrogen receptor alpha. And that's why I was trying to develop actually the sensor, biosensor granulosa cell we have, so that, because this is, if you look at granulosa cell, of course, if you go further analyze this, actually it's tested by the HPLC, another method, they are going to be tested by one. But if you look the cells, so you see the granulosa cell, only 200 pesticide can be sensed by one cell. So if we are trying to develop, but I'm not going to share. Reason is that because that mechanism is going to be utilized into the development. So actually the pesticide shows the estrogenic and protein also through estrogen receptor. These are well-established mechanisms, but in the granulosa cell, so we in this present study, what a small piece what I'm going to be share, that how the CYP19 gene is going to be through receptor dieldrin. Dieldrin is a very common and active ingredient of the pesticide, which has been reported into the milk, and through milk it is going to be in circulation. Because of lipophilic nature, it can be lead to any including the follicular fluid. So in this actually we develop, we have already granulosa cell, we assess actually, let me share it here, what the dose actually I have used. This is the MRL value. MRL means in the FSI actually declares what the level is toxic or not. So we have tested at actually MRL level, and beyond that actually it should not be. So this level actually is toxic, the MRL level to be sensitive, because beyond that is already actually established. So if we estimate actually beyond level, then milk is not good for that. But most of the method is that because there is no toxicity study as far as one country is concerned in terms of the milk. So we have actually taken the level of MRL, and at that MRL level, how we can sense actually that if there is a presented to fair milk. That part I'm not, but I'm actually sharing the mechanism. And so we look actually the dieldrin at 6 and 12 hour, 24 hour. The purpose of here is not toxicity study, but purpose is to sense the granulosa cell of the actually MRL level, which is a permissible limit into the milk, so that we can develop a biosensor. So we test, and most important thing that the test should become also very fast. So that's why 6 and 12 hours should be I have taken, because at this level, the toxicity level is also not showing, means the viability cells retain. But interestingly, if you look that level also, because that level has been shown that is not toxic, but the granulosa cell shows the estrogen through estrogen receptor alpha, and this CYP19 is going to be upregulated. That's upregulation of CYP19 is my concern. So we tested actually how, because the people, it's very difficult to show that the MRL level is not toxic, but the CYP19 gene is going to be upregulated. So we thought actually what the molecular mechanism by CYP1, because I'm trying to develop a sensor based on the LAMP, because if gene is going to be upregulated, now the technology available by which you can sense actually the gene expression also at the field level. So if we can find out the exposure of actually the milk and to the cells and some way, that's I'm trying to develop it. And expose the cells, then gene is going to be upregulated. We are trying to, so that's we go for the NGS. But that part I'm not, again, I'm sharing. CYP19 is interesting for fertility. That's I thought, why not share actually into this ISSRF, so that those who are looking for the endocrine receptor can be conscious and some regulation toward as far as this toxicity or endocrine receptor. So CYP19, we have all actually as far as the model is concerned, I have already shared that the CYP19 is expressed based on the transcription of regulation. So we looked actually about CYP19 in different level, and it has been shown the permissible limit is 150. At the 250 is toxic, but 150 is toxic means at this level, if it is present, the regulatory authority is not going to be removed, not going to be take this because this is a level permissible limit. But at the level of the CYP19 is going to be upregulated because there is no sensor available so far as at this level, because this is based on the codex actually based on the different countries what the level is present. So at the 150, the 6 hour and also responding, and the cells is responding, and CYP9 is going to be. So we look actually what because if the promoter is different and if it is not going to be expressed what is in ovary granulosa cell, so we tested both the promoter, and that's anyone can share with my paper the CYP19 1.1 promoter. That's called 1.1 promoter. We give this 1 point because it's coming from distal part. That's 120 kb far from the actually the gene from where the transcriptionally starts. So this promoter is not very going to be changed by this, but the promoter 2, which is a prominently expressed actually estrogen in the ovary, as particularly granulosa cell, is going to be upregulated. So this is the gene expression data, and then we try to understand whether this gene expression data is really going to at the cellular level, because it's known the organocolor, right? They can be worked by the both genomic as well and non-genomic basis. So if it is going to be genomic basis, it has to be bind with the promoter. So we tested actually the promoter through CHIP assay of the because we have all model. And interestingly, we found our CHIP data also says CHIP data is a very for the students that what the promoter is going to be, any gene is going to be upregulated differently. RNA polymerase is going to be bind. So we tested actually RNA polymerase as a antibody against antibody, because the gene. So in control and treated, the binding of RNA polymerase is going to be very many-fold increase as compared to control. This clearly shows, as far molecular biology is concerned, if any gene, because none of the gene like that, RNA polymerase is not going to be bind. RNA polymerase is going to be bind, but how tight is going to be bind, it's basically molecular. Those who know the molecular, as far as the gene transcription is concerned, it is a very much known that the transcription always subtle mechanism there, RNA polymerase goes bind, but when the gene is going to be, cell is going to be cells for regulation of any particular gene, it is going to be bind by the RNA polymerase. And we found actually our result that under the treatment of the dialed rate, at the actually permissible limit at the MRL level, it is going to be upregulated. And the promoter, the RNA polymerase, is going to be tightly bind with the RNA polymerase. That's confirmed actually as far as the gene is going to be regulated by this. And similarly, the estrogen is also going to be down. So the purpose is here because the estrogen, and this is already known, but we are going, what we are showing that is going to be regulated to promoter 2. And promoter 2 regulation, because that's the most important part, is that gene is present in every tissue. So this particular tissue promoter is also present in breast, but breast normally does not express or do not express actually aromatase promoter. But if the dieldrin is a lipophilic nature is leads to the breast cell, if it is going to be bind with the gene promoter, it can be upregulated. So it could be cancerogenic. So that's interesting because promoter, if you get any ligand, get the promoter, it is going to be expressed. So we express actually the mechanism through a genomic mechanism by which the dieldrin can bind to a CYP19 gene expression through aromatase, and that through promoter 2. And this promoter is a well established. And recently we the same, this particular piece of work, we able to publish as per organonucleotide pesticide dieldrin upregulate into the reproductive toxic is also a very good as far as reproductive toxicity is concerned. So that actually what the message is that, home take message is that the dieldrin above the permissible limit is really, it's going to be toxic self itself, but at the level of actually the permissible limit, if it is goes to reach to the follicular fluid, it could be a, because the estrogen is upregulated also is not good, because the vessel level, if it is going to upregulate, it's create a problem. That's why some of the actually estrogen inhibitor is also used now. And that's I'm not going to be shared here is basically clinician is there and they know how the estrogen inhibitor are being used for the different type of fertility. So that's actually, this is shows that this mechanism, the endocrine receptor, particularly through milk, That's a most important part, is the novelty of this. So with this, actually, once again, I really thanks our team. That's this particular, and we have a different funding as far as this expression is concerned. So thank you very much. Thanks for giving me and for persons listening. That's this is one piece that's where we have to be very careful as far as this toxicity through milk is concerned. Thank you very much.
Yes. Thank you, Dr. Dhir Singh. I would say it is a very, very nice talk, though the time is short, but I would say if you could just kindly, as far as this is concerned, it goes into the milk. The small children who have the further life to get. A bold message. See, we keep on seeing on the TV lots of other things, but what is important is for humanity. So such good research work is that the times have gone that when we needed that. Now the time has come, it is more than 40 years that we are just taking pesticide, And not only it has been seen, because one of our students 20 years ago did a study, from the forensic department, and we found that the patients had increased infertility, higher incidence of blood pressure. But I'll stop short, but I would say a bold message from the conference must go to the newspaper, both in various languages, that the time for the community to wake up and for the people who take the action, And thank you for a wonderful. Now, Dr. Roya for the next speaker.
Ma'am, if you give permission, I would like to ask Dr. Dhir Singh one question.
Sure.
CYP
But a very brief
19. Yes, ma'am. Yes, ma'am. CYP is supposed to be one of the detoxification enzyme. And I think it was maybe about a decade ago where one of the students did on patients with endometriosis and the prevalence, and the polymorphism of gene also could be one of the factors in pathogenesis of the endometriosis. I know that your institute is basically all animal model, but what is your experience in this?
Yeah, you are very much right. The CYP19 has also in detoxification, but the CYP19, one of the very large group of the gene actually. What we are talking about the CYP19A1, which is not actually detoxification, it is especially involved into the aromatization of the androgen. So that's into the only express into the CYP19 into the estrogen, and that's a different hormone. That CYP19A1 and CYP19A2 is a different actually, madam, because a very large group. So here, this is a very specific for the aromatization.
Okay. We'll move to the next speaker, Dr. Rita Singh, who is professor of University of Delhi. She is being visiting research award from Elie Stadium Scientific Council, France, and she is also the recipient of the Professor L.S. Ramaswamy Memorial Award, Academic Visitor Award, International Grants for her research project, and DBT International Award, and DBT Indo-US Award, DST Young Scientist Award. Her talk is on impact of the high LH level on glucose uptake and the storage in the granulosa cell, implication in the pathogenesis of PCO ovarian syndrome, which is very, very important topic. Dr. Rita.
Good afternoon, everyone.
So, madam,
can
what
you
next?
see my?
Okay. आराम से. Yes, yes. आराम से जाइए. Yes. No, क्या मैडम? आप भी हैं उसमें. मेंबर. हम बताएंगे आपको, अभी तो मुझे ही नहीं पता है यहाँ का.
Professor Yatindra Kumar's mic is on, madam.
अभी पता नहीं, उसका पता लग.
Happy to be here and see that Dr. Bakshi is chairing the session where I'm going to speak, and Dr. Roya, thank you very much. And I want to personally congratulate Dr. Madan for his great award, Padma Shri, and all the encouragement he's been giving us on this platform. I always feel so privileged to see him, and Dr. Kamboj and Dr. Curtoch, Madam Curtoch. I remember the Vaishno Devi conference. All my memories and happiness. I start my presentation because my chairpersons won't give me more time. So today I'm going to share something, the concept, the way Dr. Deepa Bhartiya also talked about. FSH is becoming very, very important, hormone in not only fertility, but also in the overall health. So, so we all know that in polycystic ovary syndrome, one of the major defect which has been ignored several years when it comes to the therapeutics is the high LH levels. And these high LH levels, they are known to increase androgens, and that is being thought to be the major cause of all the problems, whether it was hirsutism or whether it was, follicular maturation arrest. But then over the years, several years of our work and attention to this aspect has brought very interesting observations, which I have shared with all of you on other platforms. But today I'm going to again discuss some very interesting aspect which is coming and which is also bringing in some therapeutic, possibilities. So when we look at PCOS, it has major repercussions that these women are more prone to develop the metabolic disorders like insulin resistance, obesity, hypertension, cardiovascular diseases, and all those things. So this high LH, when it caught our attention, we started working on it. And meanwhile, in 2015, another paper came which showed that high level of HCG can attenuate insulin sensitivity. And then there was another paper in 2013 which showed that LH can decrease FSH sensitivity. And these things combined together can absolutely stop the fertility of a woman in the reproductive age. So we did a lot of work which I've shared many times, but now today we discuss on this aspect that we discovered that FSH and LH, they modulate glucose metabolism, but when they are alone in the experimental system, they behave in a certain way, but when they are together in the in vivo system, they will have a crosstalk and their outcome will be different. So here you can see in this graph that they both increase glucose uptake, but when you increase the concentration of HCG in the presence of constant FSH, where you get this glucose uptake at the very good level, HCG inhibits the glucose uptake which is stimulated by FSH. And when we looked at this particular response in various PCOS patients, we found that many of the PCOS non-IR, I mean the patients which do not have insulin resistance, they responded to FSH in terms of glucose uptake, whereas insulin resistant patients, their granulosa cells had this defect in glucose uptake. And when we did an experimental model where granulosa cells were incubated with individual hormones as well as both hormones, we saw that glucose uptake was inhibited by LH. And this was very interesting to find when, in FSH stimulated cells, there was IRS2 expression, which was coming. I will show you some data, but before I go on to that data, I will show you one very interesting aspect, which you must remember when you, see the other data, which I'm going to show you on insulin receptor and substrates modulation by FSH. So here, if you see in PCOS women, there is high LH. So now the estrogen levels in these women are not impacted by that. You see PCOS have high LH. So there is no correlation of the levels of LH and estrogen or insulin levels and LH levels in these patients. So there is something more happening here. So something which I'm going to show you is based on the concept that when there are more than one hormone, then their receptors may interact differently when there is only one single hormone which binds to its receptor. So these things have brought in the concept that these GPCRs make either the homodimers or the heterodimers. So these presence of these heterodimers I will discuss in our physiological system, which we have shown for the first time. Till now they were experimental systems, but so what we did was we expressed the FSHR and LHR in HEK29 cells, and then we, exposed them to these hormone combinations. It was so interesting that with the increase in the HCG, there was increase in the BRET signal. What does it mean? The increase in BRET signal means that there is closeness of these two receptors happening when both the hormones are there. And with increasing concentration of HCG. But when we gave individual hormones, there was no change in the BRET signal, means that those receptors were not interacting. But then there was one interesting thing. When there was very low level of picomolar level of HCG, it was stimulating. It was helping FSH in this glucose uptake. So there was increase in glucose uptake significantly. But as soon as there was increase in the concentration of HCG, it was inhibiting the glucose uptake stimulated by FSH in these cells. So both the responses were meeting the same ends, that alone they were increasing the glucose uptake in the experimental system. And all this enormous amount of data we have published in 2021. You can see this paper, it's a lot of data in this. But the important point is during this folliculogenesis and the exposure of these growing follicles with FSH and LH at different combinations may lead to different responses by the FSH receptor. And this is something which is bringing out the concept of biased signaling. Means there is differential signaling by the same receptor depending on how it is interacting with the other receptors or other hormones. So in our system, basically what is happening is FSHR is stimulating IRS2 expression in response to FSH, but in PCOS, it is not able to do it because there is more of LHR stimulation there. But interesting point is that number one, high LH levels, they are not able to affect the steroidogenesis in these cells by FSH, that is the estrogen synthesis. And in addition, the IRS1 expression, these are both substrates of insulin receptor. IRS1 is not affected by LH, whereas IRS2 expression through FSH receptor is inhibited. So what is happening is there is a selective inhibition of FSH responses by high LH. And that's why when we culture the granulosa cells from PCOS patient in culture, they grow much more healthy and they divide much more healthier. So IRS1 expression is increased, whereas IRS2 expression is inhibited. So there is a defect in metabolism in these cells, and these metabolic defect leads to depletion in FSH stimulated glucose stores. We have shown that FSH increases glycogen synthesis independent of the insulin receptor stimulation. It is not transactivation of insulin receptor, but it is the direct response of FSH, which is like insulin response of glucose uptake and glycogen synthesis. So this we had already shown that IRS2 expression is increased by FSH stimulation, and beautifully this is concentration dose dependent. We have done all the promoter studies, everything is published, and this process is stimulated by cyclic AMP pathway. But the important point is, can this whole process inhibition can lead to the inhibition of insulin sensitivity? So that is the important point which is coming in, that decrease in FSH sensitivity by high LH can also lead to the decrease in insulin sensitivity, because FSH stimulation increases insulin receptor substrate which are not provided. So all these things are very important, which certain other papers have also shown that IRS2 knockout mice are infertile and they are not able to respond to gonadotropins. So IRS2 is one of the key signaling molecules which happens to be important for granulosa cell or the folliculogenesis or follicular growth. So these things we have shown in the PCOS patients, that how IRS2 expression is lower in these patients. So the main important point which comes in is that FSH is also regulating GLUT4. The way Dr. Haldar showed that in PCOS patients there are mutations in the GLUT4 gene as well. So these things are becoming very, very important. And I will go on to the important slide where, so this pathway which is being affected due to high LH is also influencing the responses of IGF-1R and insulin receptor. So now how these things are affected? Number one, there is selective impairment of FSHR responses. So how it is happening, whether LH is causing certain epigenetic changes, that is very plausible. But are there certain genetic defects in FSHR sequence and FSHR sequence which may lead to isoforms of FSH and FSHR in PCOS patients, which can lead to defective responses or the selective defects in their responses? Because, and then of course, high LH is causing the metabolic attenuation of responses. Now another point comes. Now there is another response of high LH, which is the direct response, that it increases glycolytic pathway. So when you give high LH to immature rats, you will see that their follicles or their ovaries become very big. High LH or LH itself, it causes more glycolysis. So that is why there is more use of nutrient by LH. And that may be one cause that PCOS patients do not have high glucose when they have insulin resistance. So the role of LH in maintaining blood glucose is another important thing which we have started to do, that whether it can have such a huge response on glucose levels. And obviously then there is a selective or the secondary insulin resistance because of IRS2, which these patients are susceptible. But the important point which I want to make is that these things, various responses of FSH are happening at various stages of folliculogenesis. Like Dr. Deepa talked about in the primordial cells. Even we have seen that there is particular kinase being inhibited by FSH, and that is causing the primordial cells to initiate to grow. So we are in a process of building that data and completing that work. So there are different kind of responses being generated in the follicles depending on how FSH is initiating its responses. So this is the aspect which has been, emphasized by many people these days, because FSHR is not only gonadal, it is also non-gonadal protein now, which is bringing it, as Dr. Bharti has already explained. But then there is two important cases which she has not touched. One is the post-menopausal women. Now, if you see post-menopausal women, they have very elevated FSH as well as LH also increases. So when you see these women, there will be various kind of metabolic defects which start building in. If they also, so we are proposing that high LH can cause various kind of metabolic processes to get attenuated. So that is what you see in postmenopausal women, where suddenly they start developing the metabolic issues. So this is something which is very important. So all these things are also coming in picture for the expression of FSHR in the pregnant women. There have been studies where they have shown that if you knock down FSHR in mice, then in the uterine endometrium, then what happens is that the pregnancy doesn't happen. So these are the results which lead to the role of FSH beyond fertility. And the value of this, which the question my chairperson asked, there are now many labs which are working on small molecules, small peptides or the nanobodies, and various forms of glycosylated gonadotropin fragments. Now these selective stimulations or inhibitions which are being, in fact, my France award, which I'm going to take up in this summer, is also for checking some of these small molecules and nanobodies on this defective pathway which we have discovered in PCOS women, and whether we can find any of these small molecules or peptides which can modulate FSH receptor and restart this RS2 expression. So these are the important, in fact, there are a lot of companies which are in fact taking forward these steps of making these various molecules, and in India also we should start this kind of work. But then we don't have this live cell imaging, certain very advanced equipment for which they were very interested, and we collaborated, and we are doing this very important work so that live cells and live service movements, you can see by these very beautiful technologies that how these molecules are interacting with the receptor and how they are modulating their structure. So we are modeling their structures, and we are trying to find how we can, it's kind of a micro repair mechanisms which we are working on. And these are very, very important for patient treatments, not only the PCOS, but also the postmenopausal women, as well as many pregnancy related issues which are coming up. And that's what I feel, that this is a very important science. And I'm very happy to see Dr. Jb Talwar sir. Namaskar sir, and really happy to see you here. And I'm very thankful to all my students and my collaborators. I had put their photos where I had actually shown the work which I used that advanced machine. So I just want to show you my. So this is Dr. Pascal, who is there in France, University of Indra and University of Tours. And Francesco is the one who helped me with this machine where we study the breath and other live cell, signaling mechanisms. So thank you very much for all your attention and giving me this opportunity. I'm very, very thankful to Dr. Loya and Dr. Ansari for doing so much of hard work and maintaining beautiful newsletters. And I would also like to sometime do the newsletter on FSH, FSHR and its mechanisms. So sometime when you plan to do on these gonadotropins, please give me the opportunity. Thank you very much, sir.
Great, you are most welcome.
Great. Thank you, Dr. Rita, for giving an excellent talk on the LH and the PCO. Now the next speaker is Dr. Prabir Mukhopadhyay, PhD, Presidency University, Kolkata, and he is being recipient of many awards, including the French Visiting Research Award, Professor Ramaswamy Memorial Award, Academic Visitor Award, recipient of the International Grants and DBT International Award, DBT Indo-US CRH Award, and DST Young Scientist Award. His topic and his title of talk is on high casein and P-enriched diet can effectively attenuate arsenic-mediated reproductive disorder in adult male rats. Professor Prabir.
Good afternoon, madam. Thank you for your introduction. But I would like to add one thing that, this is not my CV. There might be some mistake. Okay.
I see. They have sent this to me on my
Yeah, this is not mine. Anyway, I'm a professor of Presidency University. I'm a HOD of Department of Life Sciences, and I have the teaching experience of 33 years. Okay, so I start my presentation. My topic is high casein and pH diet can effectively attenuate arsenic-mediated reproductive disorders in adult male rats. At the outset, I also thank the organizers for providing this platform to share my research work on the environmental toxicity management, particularly the reprotoxicity, in front of all of you. Firstly, I'm showing you some rising trend, where I can say that the human sperm count, is being gradually decreased. Pardon? Pardon?
Is not visible, sir.
Your slides are not visible.
They're not visible.
Able to see the slides.
Just a minute. So this is visible.
No, we are not able to see. Can you share your slides?
Share it.
I think
just a minute. I want just 1 minute. It is not.
No, I think Dr. Nidhi has to remove this slide. The ISSRF-2022 there, so they have to remove the slide.
Remove that, sir.
Yeah, it has to remove. That's why.
Yeah, still it is there.
Yeah, that's why probably.
Yeah.
Sir, we have removed it. Can you reach? Now I think we can
now
see.
It is.
Okay.
Yes.
Yeah.
Yes. Now.
Yes. Just this is the first slide. So.
Yes.
Just a minute. Not working. So this is the rising trend. Human sperm count gradually decreases with the progress of the year, and this is the data taken from Lancet 2021. It clearly shows that the sperm concentration, total sperm count, sperm motility, sperm viability, sperm morphology, these all are going down gradually in different manner. But it's true. And obviously, this may lead to the infertility. Definitely, I don't know after some time. So, under this condition, my aim is to emphasis on the heavy metal toxicity, and our concern is arsenic. Arsenic toxicity is causing distress to about 150 million of people. As per WHO, the permissible limit is 10 microgram per liter, that is the 10 ppb, but before that it was a 50 microgram per liter. And the most interesting thing is that the most worst affected people are from low socioeconomic background, who are getting poor nutrition. Next slide, please. This is the scenario of India. The 6 states, Uttar Pradesh, Bihar, Jharkhand, then Chhattisgarh, West Bengal, Assam, Manipur, these are heavily affected. And more particularly, the Ganga, Meghna, Brahmaputra basin, the people who are staying over here, they are more affected. And this is the condition of our state, where the 14 districts are affected, and the maximum level is in one district is 3.2 milligram per liter. That is a 3.4 ppm. Next slide, please. This is some newspaper report. They are providing us the data. That is how it is increasing in heavy fast rate. The people are being affected, and this is the thing that is a potent relation. This are the papers. Potent relation exists that the people belonging to the lower strata of the society are suffering from malnutrition as they don't get enough micronutrients and proteins from their food. So they are prone to arsenicosis. Next slide. So arsenic causes the disorders of so many systems: arsenicosis, cancer of skin, lung, bladder, cardiovascular, developmental defects, lung and liver and lung fibrosis, peripheral vascular disease, diabetes mellitus, hematological disorder, neurological disorder. But our concern is the reproductive system disorders, and these are proved. And there are so many epidemiological data, and has been on different rodent model. It has been proved that it causes the dysfunction of at the level of gametogenic, at the level of the steroidogenic. Next slide, please. This is the arsenic metabolism. It is excreted as a MMA and DMA, and for that it needs the methionine, and the methyl group is given from the methionine, is taken from the methionine. So the methyl pool is depleted. It produces so many free radicals: H2O2, hydroxyl, NO, superoxide, and more particularly dimethyl arsine peroxy radical and the dimethyl arsenic radicals. And thus it generates ROS, damaged DNA, lipid, and proteins, leading to the degeneration. And this is that model that the ROS causing, but it is at the general level.
She will reduce hair.
So our aim is our supplementary diet, the diet which we have chosen, whether that can mitigate. And for that, we have selected the studies like the histological and histomorphometry, some biochemical assessment, some spermatozoal parameters, its potentials assessment, and to check the steroidogenic status and the DNA damage study. Next slide, please. Next slide, please. So this is the animal model. We have chosen 24 adult male rats, and it has been repeated several times as per the ethical clearance. So this group was given the normal diet. This group was given the arsenic trioxide at a dose of 3 mg per kg body weight per day. And this group was given the arsenic along with the high protein diet. I'm coming later on in the next slide. The treatment was continued for 13 days. Next slide, please. Here I'm showing you this is the diet, normal diet, and this is the diet where the simple casein amount has been increased and the pea protein has been added, pisum sativum, and the calorific value of both the diet is remaining same. What we got? We have seen that the weight of the testes and the accessory reproductive organs all have decreased, and here it has been gained. Gain means that it has been restored. There is no change or very little change. Next slide, please. The histology, from the histology, we can see that the disorganization of the germ cell, increased luminal area, and the decreased spermatozoal concentration. Almost all the lumens are vacant. And from the histomorph, we done the histomorphometry. I'm not presenting here. We have checked we have measured the diameter, luminal diameter, germinal epithelium height, Leydig cell number, Sertoli cell number, Leydig cell nuclear area. In all cases, we have seen that there are the remarkable changes, and here those are not seen. Next slide, please. This is the scanning electron microscopic analysis, photomicrograph. We have seen the luminal area has been increased and disorganized germinal epithelium. This much I can say from this photograph, and here it is not seen. This is from the third group of animal. Next slide, please. So from this we can say that the casein and this high protein diet can maintain the reproductive organ weight in spite of arsenic exposure, and the structural integrity of the testes is recovered upon supplementation. Next slide, please. Yes, now we are moving to the epididymal level. That is a very vital organ, as we all know. There are some changes, the important changes are being taking place, and here the spermatozoa stay for 10 to 14 days. So we see that we have counted the changes, the structural anomalies at the level of, this is the rudimentary tail, this is the rudimentary tail, this is the headless tail, this is the bent tail, this is the coil tail, this is the carved tail, this is carved mid piece, this is the bent mid piece. And everywhere we have seen the changes in case of arsenic treatment. And these are the scanning electron microscopy view. And more particularly, I like to add that in case of rudimentary tail, the number is less compared to this defect. And these defects occur at the level of epididymis, not at the level of testes. Only the rudimentary tail is formed in the testes. But much attention should be given on this. So why this thing happens? The defect at the level of epididymis is much more. Next slide, please. And this is the scanning electron micrograph of spermatozoa. And this is the normal spermatozoa depicting the acrosome, postnuclear clamp, plasma membrane, nucleus, perforatium. And here we can see the soiled perforatium. We can see the dorsoventrally constriction. We can see the serration of the connective piece. This is, and the distorted postnuclear cap. This is, here it is a distortion, but almost these are absent, and we have counted this. So this is the result, that with this treatment we got this, and while supplementation, so though some difference is there, but we got better result. We got the protection with this supplementation. Next slide, please. This is the motility. As you know, this is very important, potential of that spermatozoa in terms of its fertilizing ability. So here also we see at all level, total motile spermatozoa, progressive motile spermatozoa, or in all aspects. And this is the non-motile spermatozoa, and this is non-progressive motile, non-motile. And in all case, we see there is a decrease due to arsenic treatment. And here we see that there is a significant restoration. Next slide. So this effect at the level of epididymis is there at the level of by this supplementation, and the spermatozoal features are restored. And one thing I have forgotten to mention, the previous slide. This is the host positive, that is, which gives the idea that how much the sperm plasma membrane integrity is intact. So here also that it is seen that the plasma membrane integrity loss by the arsenic treatment is minimized, is mitigated up to a certain extent. Next slide, please. This is the just diagrammatic, for, as you all know, that for the spermatogenesis, we used to check these two enzyme activity, 17 beta HSD and the 3 beta HSD. So we have, next slide, we have measured the activity, delta 5 3 beta HSD, 17 beta HSD, and we have seen it is the protected. We have seen the protection, and we have measured the testosterone level. So we have seen by ELISA, we have seen there is the protection. And the data which are not being included, we have just done this. We have seen that LH and FSH level is being restored. The LH FSH levels are also restored. And that the decrement of LH level and FSH level has already been established. So many scientists have stated quite a long time back. But this diet can maintain that level up to a normal level. So next slide, please. So normal steroidogenesis was sustained, and testosterone level was also maintained upon supplementation, we can say. Next slide, please. Here, so what are the causative factors? Arsenic causes the increased production of ROS. So definitely the oxidative stress parameters are to be studied. Accordingly, we have studied the non-enzymatic, that is the MDA, protein carbonyl, GSH, and here the enzymatic SOD catalase, GST, GPX, GR. In all cases, there are the changes, there are the reduction of the activity, and these are the protected. And here also the rise of MDA, PC, and the GSH, decrease of GSH. So these are restored. So it means that this diet can protect the oxidative stress mediated by the reactive oxygen species. Next slide, please. So we have measured the Ross generation. We have taken the spermatozoa out from the chorda and we have taken the Ross generation by fax. And here also it gives that clearly shows that the generated Ross level is seen to be decreased at the level of spermatozoa. So before that, what I have shown you, that is at the level of testicular tissue. Here it is at the level of spermatozoa. So this protein diet can mitigate can decrease the reactive oxygen species level at the level of spermatozoa and definitely at the level of testicular tissue. And we know that this increased Ross can lead to firstly the oxidative enzymes are in action, and then the increased level can cause the decrease of can cause the DNA damage. So accordingly, we tried to see. We have with the isolated spermatozoa, we did the anilyl blue experimentation. This is the AB positive spermatozoa, and this is the AB negative. That means here no staining. Here the DNA remaining intact, and here the DNA, normally it is converted to histone to protamine conversion is taking place, and it hampers in the conversion. So it binds with the histone, and then it becomes a colored. It takes the deep blue color. And so we have counted these, and we have seen that the AB positive number of the AB positive number are increased, and again decrease. We've seen decrease, but definitely not at the level of the normal level. And this is the AO, actinic orange staining. We know that this is the metachromatic shifting is seen when there is a DNA damage, and it gives the red fluorescence, and the red fluorescence has increased. So it shows that the metachromatic shifting, or that means the DNA damage, DNA stand break. So, and here we see there is a significant difference between these two. So the damages are protected. So that means damages at the level of spermatozoa DNA damage is protected with the addition of this dietary supplement. Next slide. This is COMET study. This further proves that by means of assessing the comet tail length, we can show that this damage, DNA damage, is protected. So next slide, please. From this, so high casein diet, it can mitigate DNA, it can it can protect the DNA damage at the level of testes, at the level of spermatozoa. Here I am not showing the DNA damage at the level of testes by means of DNA extraction and its ladder experiment. That is not being shown, but that has been done. I'm just waiting for repeat experiment. So the protection is there. Next slide, please. So the next obvious thing is the apoptosis. So for the apoptosis, we are concentrating to assess at the level of RNA, at the level of protein, to assess the BAX, BCL2, caspase 9 and caspase 3. And to say that we are getting very promising result, but yes, it is being protected, but some repeat result is needed. And we are also trying with the spermatozoa to check the spermatozoa apoptosis by annexin 5 by flow cytometry analysis. Here also that has been done, but again, some repeat experiments need to be done. So we are, but for this, next slide, please. Without this, so we can say that the DNA damage can be protected. And if there is a DNA damage, then the next obvious thing is the apoptosis. So whether this diet can protect this apoptosis or not. So for that, I am taking the next slide, please.
Professor,
so
if it is possible,
just 30 seconds, madam, 30 seconds. I'm showing you my earlier paper where I have supplemented the female rats, the adult female rats with this supplement. And there I have seen that this is the BAX anti-apoptotic, this is the pro-apoptotic protein. So this is being increased. And with this diet that has been minimized, that you see, this is, And here also the procaspase-3 and the cleavcaspase-3, these are increased. So it indicates that the arsenic mediated, it has done at the level of uterus. Arsenic mediates the apoptosis, and that is protected. That is protected with this dietary supplement. So with this, I can, next slide please. I can conclude that the study concludes that the high casein and P in this diet was successfully mitigating the toxic effects of arsenic on male reproductive system. High casein and P diet was able to mitigate the ROS-mediated testicular DNA damage. And this finding inferred that the diet successfully attenuated several deleterious effects of arsenic on epithelial spermatozoal populations also. And these are the potential of P and casein to minimize lipid peroxidation, radical scavenging, and metal chelation perhaps remain to be the chief mechanism that prevent arsenic-mediated oxidative damage and redox imbalance. And furthermore, the methyl transferring potential of P is main contributing factor, and the clinical trial with the strategical modulated diet may be undertaken to extrapolating these findings on human victim to find an easy and effective strategy to combat this arsenic-induced toxicity. And these are the findings. These are the findings. Next slide. And these are my group. No. These are my present groups. This Shagnik Bishas, who has done this work, and these are the other fellows of my laboratory. Thank you for your presence.
Thank you.
Thank you, Dr. Mukhopadhyay.
Okay, thank you, madam.
Yes, thank you.
It
No,
has
ma'am.
been a-
Ma'am,
Yes.
I would just interrupt for a minute. We, on the basis of ISSRF team, we really apologize to Dr. Mukhopadhyay. I think there was some mix-up of the slide. So this is his correct introduction slide.
Right.
Sorry about that, sir. Sorry about that. So, and we really thank you for your talk. It was really a great talk. Thank you, sir.
So, excellent talk. Since the time is short, we are not asking you any questions,
Actually, it's a
but
very-
we will do by the email. But it would be
Exactly.
nice to know why you chose only P. If can other diet be seen? Is P has that better advantage over the other? We will discuss over the email.
Thank you, madam. And
Yes.
as because it has the methionine, so that's why, and it has the antioxidant property. So it's a long work. It's a work of a thesis.
Ma'am, can you introduce Dr. Suranty Sharma, ma'am? Professor.
Thank you,
okay,
madam.
okay.
Thank
Thank
you,
you.
madam.
Thank you.
Thank you, madam. Thank you.
Now we have, thank you, sir. Now we have the last speaker, but she is very, very important because she's going to talk on a very important topic. Dr. Suranty Sharma, now who is a PhD and is working in Germany. Now she is going to talk on developing in vitro organ culture and microfluid-based testes on chip model to study in vitro primate spermatogenesis and endocrine dynamics. Her research interests are large and on various, and she has a large number of publications and more than 17 peer-reviewed. But as you all see, they speak so beautifully. Their research is so wonderful. So we will rather have time to listen from you, Suranty. We are looking forward for your genius presentation. Yes, Suranty.
Yes. Make. She has to unmute.
So I'd like to thank the chairs for the kind introduction. And I apologize for any background noise as I'm in India currently and there's election season going on. So welcome to my presentation on my postdoctoral work, which was a collaborative project between the Schlatt Lab at the University of Münster in Germany and the Legat Lab at the University of Twente in the Netherlands. The title of my talk today is Developing Testes on a Chip Model to Study in Vitro Primate Spermatogenesis and Endocrine Dynamics. So before I jump on to the experimental details, I'd like to give you an overview of the rationale for this work. So the rationale came from my PhD project. I can't move the slide for some reason. Yeah. So before I jump onto the experimental details, I'd like to give you a brief background. The rationale came from my PhD program, which was part of the Marie Curie International Training Network called Grow Sperm. It was a research consortium including 10 leading European research laboratories working on male fertility, and it was funded by the European Union Marie Curie FP7 funding agency. So the network included 9 PhD projects and 2 postdoc projects, and all the projects had a strong collaboration, and we all were working on different model systems. And the overall rationale was to devise fertility preservation strategies for infertile men. So as there are no therapeutic options for infertile patients to produce sperm, we all know there's a need to develop alternate strategies for ex vivo sperm development, especially for prepubertal cancer survivors. And there is a very limited understanding of primate spermatogonial stem cell biology, right from stem cells to functional sperm stage, which is a major research gap. As you see in this illustration here, the Grow Sperm program focused on investigating different aspects and stages of male germline development. So this involves a series of sequential mitotic and meiotic divisions for maturation of primordial germ cells into spermatozoa. The expression of SRY protein initiates a cascade leading to male-specific gonadal differentiation, the appearance of Sertoli cells, their aggregation and formation of testicular cords are the initial cellular features of male sex differentiation. Testicular hormones like AMH and testosterone released from Sertoli and Leydig cells evoke subsequent sex-specific differentiation of the organism. So in male-specific germ cell developmental pathways, multipotent primordial germ cells migrate and colonize the gonadal ridges and further differentiate into unipotent gonocytes in seminiferous tubules. These gonocytes then undergo sequential cell divisions, differentiating into spermatogonia, spermatocytes, spermatids, and spermatozoa, thereby completing the cycle of spermatogenesis. So my research goal was to evaluate testicular differentiation aspect and to establish strategies for germ cell development, and especially for prepubertal cancer survivors who are at the risk of losing their fertility due to chemotherapeutic treatment. Clinics are cryobanking testicular tissue from such patients with a hope to use this tissue to develop sperm in the future, but we don't have any strategies yet. So as there was limited human testicular tissue available, we used testes from non-human primate models, marmosets and macaques, as they share close developmental pattern as observed in human testicular development. So we all know testis is a bifunctional organ serving both as a gland and a gonad for producing gametes and androgens. The steroidogenic and spermatogenic function of the testis are controlled by the GNRH pulse generator via 2 different feedback systems. The LH testosterone excess controls androgenization, and the FSH inhibin excess controls sperm production. So besides complex endocrine regulation, testis has a subcompartmental physiology. The tubular compartment we know is the site of spermatogenesis, while the interstitial compartment is the site of androgen production. The interstitium contains steroidogenically active Leydig cells, which are testis-specific somatic cell type producing androgens, stimulating the release of androgens, which induce or maintain a male phenotype and stimulate sexual organs and androgen controlled actions in the periphery. So this synergistic interaction between the steroid and gamete producing entities of the testis at endocrine, molecular, and cellular level enable it to function as a gland and as a gonad. However, it is very challenging to recapitulate this in vivo synergy of various subcompartments of a testis in vitro systems. Therefore, several cell suspension-based and testicular tissue-based experimental strategies are being developed. And most of the findings from my own research studies performed using either testicular xenografting, spermatogonial stem cell isolation culture, and testicular organ culture approach show limited spermatogenic progression with conventional ex vivo culture strategies. Different labs are working on different techniques ranging from iPS cell differentiation or 2D or 3D monolayer cultures, or transplantation approaches. But still we are facing this challenge, and we do not have a strategy for achieving fertility preservation clinically. So most of these techniques have certain drawbacks. However, we noticed that using intact testicular tissue had several advantages. As you can preserve the structural integrity of the tissue, the cellular topology and the somatic microenvironment of the tissue is maintained. This led me to focus on strategies and attempts using intact testicular tissue. So when I tried to dig deeper into this, this was a century-long journey which started from 1920 with the first attempt by Cempi, and then Trowell's method was developed in the 1950s, and then there were several attempts reported in the literature using various modified versions of Trowell method and organ culture strategies, where tissue explants were used to culture either on metal or agarose blocks floating in the culture media. So during the final year of my PhD, in 2018, I came across work from Ugawa Group in Japan, as you see some of these publications here. They employed a microfluidic-based model to culture intact seminiferous tubules and demonstrate maintenance of mouse spermatogenesis for over 6 months. And they were able to produce progeny using the sperm, which they developed, which was developed using this strategy. So I wanted to develop similar system for primate spermatogenesis to culture intact primate seminiferous tubules. So with this goal, the aim was to create a controlled in vitro environment mimicking in vivo physiological and biological conditions in a primate testis, develop a testis on a chip culture system for maintenance of seminiferous tubules, which are isolated from primate testis, and to evaluate the cellular and endocrine responses by exposing this tissue and seminiferous tubules under stimulatory and non-stimulatory culture conditions. So what is microfluidics? For people who are new to this technique, it is a combination of both science and technology of systems that processes or manipulates small amounts of fluids using microchannels, and miniaturized devices are fabricated. You can control the microenvironment as it allows high level of integration, and the laminar character of the flow allows creation of gradients. The high surface to volume ratio allows efficient heat and gas exchange. And you can confine your samples, which is very close and which closely mimics the in vivo conditions within a biological system. So this was a device design which I developed together with our collaborators, the bioengineers from University of Twente. And these devices were made up of PDMS. The seminiferous tubule samples were loaded to the sample loading port into the culture chamber. The loading port was closed using PDMS lid. The culture media was perfused to the inlet port into the culture chamber, and it reached the sample by diffusion through parallel slit channels, as you see here. This is the culture chamber where you can place your seminiferous tubule samples, and the culture media is diffused through these parallel channels. And perfused media is collected through the efflux port into the efflux collection tube. So on the right, you see enlarged view of this device with slit dimensions. So this platform was further characterized using modeling software, and we had to develop different iterations of this model to get the most perfect device so that we don't damage our samples, because seminiferous tubules are very fragile. And when you load the samples, there is a risk, very high risk of damaging the samples. So we had to modify the design and dimensions several times to come up with this design, which was also published. We published it 2 years ago. So this platform was then characterized by 3D COMSOL modeling of the fluid flow. We wanted to check whether shear stress generated by the flow would be harmful to this tissue. So it was evaluated numerically. Tissue was modeled as a rigid wall and is represented by these orange color graphics here on the left. In cases 1 to 6, different conditions were considered. And case 1 here represents positive control. No separation channels were implemented here. Case 2 and case 3 represent tissue in the center of the chamber. Case 4 represents gap present at the tip of the culture chamber.
while case 5 represents tissue filling only a small portion of the culture chamber. And case 6 represents a circular defect present randomly in the middle of the chamber. So the shear stress was calculated to be lower than 0.25 dyne per centimeter square in all the samples, and highest was found in case 1 and case 5, where no protective barrier was implemented or less tissue was loaded. And these values were significantly below the physiological values reported for endothelial barriers, which are typically 0.1 dyne per centimeter square, and they become permeable when exposed to considerably high shear rates. So on the right, you see a plot showing the effect of flow rate on shear stress. We observed that the least flow rate exerted least shear stress for all the samples representing different culture conditions. So we continued with 100 microliter per hour for the on-chip culture samples and use this flow rate for our experimental conditions. So here you see the experimental scheme. We used marmoset monkey testis. Seminiferous tubule samples were loaded. These were prepubertal marmosets. And we loaded these samples onto these PDMS chips, which we developed. The inlet port of the device were connected to the media-loaded syringes fitted to a perfusion pump for continuous perfusion of media during the culture. So as you see, the samples were cultured in 3 different culture conditions, with control media, which contained no gonadotropin and serum supplementation. Low-dose media had low dose of gonadotropins in serum, and high-dose media contained 10-fold high dose of gonadotropins in serum. These cultures were maintained for 9 days, and they were later analyzed for different endpoints. As you see, morphological changes were analyzed by imaging and later determined histologically. Efflux samples were analyzed for testosterone and estradiol levels. So here you can see the morphological observations from day 0 and day 9 culture samples in different culture conditions. We do not observe any significant changes in the tubular morphology during the 9-day culture period. When we look at the live imaging observations of the samples cultured in the high-dose stimulation group, we see active cellular subpopulations in the basal and the tubular compartments. If you have a very close look, we could observe active areas throughout the tubular samples. And they were not, in contrast, in the low-dose group, we did not see so much active cells. We wanted to corroborate it further with other analysis, which we decided to continue. So when we looked at the endocrine parameters, here is a plot for the control group. The data presented for the testosterone efflux sample in the 3 different culture groups at 26 different collection time points. So as you see here, these different time points are plotted on the x-axis. On the y-axis, you can see testosterone levels in nanomole per liter, while each color-coded sample plotted here represents sample from independent chip. The testosterone levels in the control groups ranged between 1.5 to 2.5 nanomole per liter. In plot B, for the low-dose group, we observed comparable testosterone levels were recorded, and until day 4, we were able to record comparable testosterone levels, and values rose up to 6 nanomole per liter thereafter. Here are the samples from high-dose group presented with different scale bars. So C1 and C2 represent the testosterone levels for high-dose group. Here again, we recorded high testosterone levels after day 4. Values rose up to 32 nanomole per liter. When we looked at the estradiol levels, we observed similar situation, similar condition. And in control and low dose group, most of the samples, we were unable to detect estradiol. In contrast, in high dose groups, especially at later time points, we observed higher estradiol levels compared to the earlier time points. When we looked at the histological level, these semi-thin resin embedded sections were created, and we performed PAS stainings, and we observed more apoptotic-like cells in day 0, day 9 control, and day 9 low dose groups. In contrast, in high dose group, we observed more surviving cells and more germ cells and less amount of dying cells were reported in these samples. So finally, we were able to conclude that the prepubertal primate tissue corresponded to the dose-dependent stimulatory culture conditions in the testis-on-chip platform. Irrespective of the culture media used, tubular maintenance and survival were observed during the 9-day culture period. This system allowed better diffusion and can potentially be used for running long-term primate testicular cultures and for exploring spermatogenic and steroidogenic function of the testis. As a future outlook, we believe that this system can be used for various applications. We started off with a goal to achieve in vitro spermatogenesis, but that's a very far-fetched goal. We still have to work on this technique, modulate a lot of experimental conditions for that. But for now, with the results we have so far, we can say that we can use it for periodic pulsatile stimulation, for exploring spermatogenic and endocrine function of the testis. We could use it as an ex vivo reprotox tool for testing drugs, toxins, and physical factors. And we could also use it as a replacement for animal testing. So finally, I would like to thank you all for your attention, and I would like to acknowledge all the people who were involved in this work. I would like to acknowledge my mentor, Professor Schlatt, and collaborators from the Legat Lab at the University of Twente. And I would like to thank the organizers for giving me the opportunity to present my work here today. And if for any questions or any queries, or any opportunities, please contact me on my email address. And if there are any questions, I'll be happy to answer right away. Thank you very much.
Thank you, Dr. Swati, for giving an excellent talk and very, very innovative, very new, and basically, which is, I think, is the future of the genomic. It is all omics and metabolomics, where it is very, very new topic. And excellent hard working also you have done. I must congratulate you for giving such an excellent presentation. Any question? I think because there is a paucity of time, we are running short of time. So we will just close this session. And I must say, hearty congratulations to Professor Luhia and the team for being, having organized this excellent international meet, ISSRF 2022.
There is a question in the chat box. There is room, for Q&A. Thank you so much.
There's a question chat box. I think do we have enough time? There is a oration now. I think on the email.
Thank you. I really like to thank our chairperson for chairing this session and all the invited speakers and oral presentations for a wonderful talk. Now we'll move ahead to our oration 3, which is Professor L.S. Ramaswamy Memorial Oration. And for this session, I would invite our chairperson, Dr. S.K. Gupta, who is a scientist at National Institute of Immunology, New Delhi. And his research interest is reproductive biology, immunology, immunodiagnostics, and vaccine development. And the other chairperson is Professor Channa Haldar, who is former director of Institute of Science, Banaras Hindu University. And her interest is melatonin regulation of reproduction and immunity. So I would request the chairpersons to please chair this session. Over to you, sir and ma'am. Thank you.
Thank you very much. I think I would like to request Dr. S.K. Gupta to say a few words about Professor L.S. Ramaswamy before we start the lecture.
Ma'am, I think Dr. Gupta is not present today.
Okay.
Yeah.
Then I take a privilege to say a few words.
Definitely, ma'am. Definitely.
Honorable Professor R.L. Ramaswamy was DSc in that early time in 1938 from the University of Madras. He was a director of Primate Project Institute for Research in Reproduction and known for the primate research in initiation of primate research in India. He did a lot of studies on the primate model and extended it to the human research. And I think it is enough word to explain the magnanimous characters of Professor Iss Ramaswamy. With these few words, I now would like to request our Oration awardee for the Professor Iss Ramaswamy, Arun Chogle, and would like to introduce him in short. Dr. Chogle's interest is radiobiology, biochemical tumor markers, radiation hazards and protection, and radiotherapy. He is a recipient of lots of awards. A few of them is the AFOMP Member Excellent Presentation Award, Outstanding Faculty Awards, Dr. Farooq Abdullah Sher Kashmir Awards, and so on and so forth. Today, the Oration 3 by Dr. Chogle is entitled Recent Advances in Radiotherapy Towards Organ and Fertility Preservation During Cancer Treatment. So Professor Arun Chogle, now it is your time to enlighten us.
Thank you, madam, for your kind introduction. I hope my slides are visible and I am audible.
Yes, sir. Yes.
Chair madam has already introduced Professor Iss Ramaswamy, and when I was seeing her biodata, she is also recipient of the Professor Iss Ramaswamy Oration, and he was the founder head.
Privileged, yes.
Yeah. So he was a founder head of the Department of Zoology, University of Rajasthan, and being a founder head, he has established the zoology department. And as the chairperson has told about, he was a distinguished reproductive biologist, not only in animals but humans. And this award for his great contribution was started by the zoology department, carried forward by ISSRF since 1990. And the first awardee is Professor N.R. Modgal. He is a professor of biochemistry. And the last year's Iss Ramaswamy Memorial Orator was Professor Balaram Bhargav, who is Secretary General, Director General of the ICMR. So it's a proud privilege. And the coincidence is that on my 63rd birthday today, I am receiving this very prestigious Professor Iss Ramaswamy Award. Thanks to ISSRF and especially to Dr. Lohia, who has paid hugely for the growth of the science, and with whom I have almost more than 2 decades association. I will be talking on recent advances in radiotherapy towards organ preservation and preservation of fertility while treating patients with cancer. I will confine to the radiotherapy because I see medical oncologist Dr. Hemant Malhotra, very renowned medical oncologist. He will be speaking about the fertility in the young cancer survivors, and he's a medical oncologist. He will cover a lot of those medical oncology and other aspects. So my talk is divided into 3 parts. One is the cancer incidence across the world and India, recent advances in radiotherapy technology, and the challenges toward providing organ preservation and preservation of fertility while treating patients with cancer by radiotherapy. And some of my PhD students and other students are working on the estimation of the doses, the second cancer, the prevention of radiation doses to the organs, organ at risk, especially the genital organs. So just in brief, cancer incidence, on 4th of February, we have celebrated World Cancer Day, and the logo was Create a Future Without Cancer, the time to act now. close the gaps, because there is a lot of disparity in cancer treatment across the globe, also in our country, from east to west and north to south, huge disparity in availability of the treatment reaching to the. So one size doesn't fit all. Every challenges demand a different solution, and that is how therapy. And as you know, cancer is a second killing disease, and incidence of the cancer in brief, almost about now 20 million new cancer cases everywhere across the world. But if you talk about the Asia and Oceania, 50% of the cancer in this region, and most of the countries in this region are either underdeveloped or developing with a lack of treatment cancer facilities. Patient comes in advanced stages, and however, cancer is, as I told you, 70% of death from the cancer occur in low and middle income countries. Around 1/3 of the deaths from cancer are due to the 5 leading behavioral dietary risks: high body mass index, low fruit vegetable intake, tobacco, and all these things. Tobacco itself has 22% of the cancer death. And in India, it is estimated that 12% rise in cancer by 2025, NCRP report 2020 says, and almost about 12 lakh new cancer patients every year in India. So in cancer treatment, huge advancements have taken place. And because of that thing, the survival of the cancer patient after treatment has increased a lot. And even in the younger cancer patient, the survival has increased. And therefore, the organ preservation and fertility preservation is very, very important. That also in young patient who are going to survive for a longer duration and would like to have their children, family things. Cancer treatment option, what we have got is a surgery, surgical oncology, very, very well developed. Chemotherapy or medical oncology, much more developed, and which Dr. Hemant Malhotra hopefully will be speaking about. And radiotherapy. In radiotherapy, we have a brachytherapy and teletherapy. It has 125 years of experience now in radiotherapy. And along with that, on 8th November 1895, almost about 127 years ago, discovery of X-ray has revolutionized not only the diagnosis, but the therapy and treatment. And the earlier concept before the X-ray availability, diagnosis was open and see invasively, and now first see non-invasively with imaging devices, see it. So now we have range of equipment for diagnosis and also for therapy, which has not only increased the tumor control probability, but the radiation risk and comorbidity has been brought down. Survival has increased, and the tumor control probability from 20 to 80%, it has escalated. However, ionizing radiation in the form of X-rays or gamma rays or photon, all those things, is a dual-edged sword. It can cure the cancer, at the same time it can cause the cancer. It is very harmful because it produces the somatic as well as the genetic effects, and few gray doses to the gonads can create infertility into that thing. And therefore, the primary aim of radiotherapy, because of the dual-edged nature of the radiotherapy, is deliver a lethal dose to the tumor. We want to kill all the cells, cancer cells into the tumor. At the same time, we want to spare the normal tissue, OAR, organ at risk, and that is a comorbidity we want to keep at a minimal, at a manageable level. How to achieve this thing? It's an art, it's a science, it's a technology, it's a knowledge, it's a skill. And therefore, in 125 years, the radiotherapy has come in a big, big way. And it is a technology driven and image guided and many other things I will talk during this course. So evidence-based and tailor-made treatment, we have moved from the one treatment for all to a tailor-made specific to the the patient, patient site, organ, age, and all those things. And this was the history. 29th March 1896, within just 3.5 months of the discovery of the X-ray, Emile Grube treated a breast cancer patient, and that's how radiotherapy started. And during that time also, he used the lead shielding to protect the normal tissue. That is what the knowledge that time also grown. So external beam therapy and brachytherapy, these are the two modalities where external beam therapy, as the gamma rays, electron beams, X-rays, or particle radiation we use, called as the EBRT, where radioactive source is away from the patient body. In brachytherapy, we have radioactive sources close to the tumor or into the cavity. So it can be surface mold, it can be intracavitary, it can be interstitial, removable seeds or permanent seeds, and these are the technique. However, just within 10 years of the discovery of X-rays, and after that, 1 year in 1896, the radioactivity, radium and polonium. 1906, Burgoni and Tribundu irradiated the scrotum of the ship by X-ray, and they found that the cells which are rapidly dividing, cells which are undifferentiated, are highly radiosensitive. Therefore, the sensitivity of the organ, sensitivity of the tissue is not same throughout the body, and therefore lymphocytes and spermatogonia oocytes are highly radiosensitive because of the nature of their highly dividing, undifferentiated. And then this is the osteoblasts, spermatids, muscle cells are medium, nerve cells which are differentiated radiosensitive. So this is a radiobiology which drives the radiotherapy or cancer treatment by radiation. And today, what we call is not a physical dose, it's a biological dose, and it is theragonistic. By knowing the biology of the cancer, we make the radiation doses, and I will show how high-tech radiotherapy can spare the organ, reduce the doses to OAR. So we started with superficial X-ray therapy, just 50 to 100 kV. Then 1940s we have deep therapy. Then 1950 we got a cobalt therapy, that is a cobalt 60 with radioisotope. Now we have linear accelerators of variable energies and also electron. We have CyberKnife, 6-degree movement, the 6 MV linear accelerator with a very fine 1 mm beam also, and you can have what is called as a stereotactic body radiotherapy. That is, beam can be angulated, beam can be tilted and treated, sparing the normal tissue and concentrating the dose into the tumor, and the dose gradient fall is a very, very sharp. Then we have a brachytherapy, and now we have what is called as a LDR to MDR to HDR, high dose rate brachytherapy. Earlier days, cancer cervix, 7 days treatment, patient with a radioactive source into a ward, isolated for 7 days. Now it is 7 minutes treatment. Patient can come, take the treatment for the cancer cervix, cancer breast, and this thing, go home. This is a minutes of the treatment. So this is what the thing, and this is the interstitial treatments we can do with a multiple applicator, multiple doses. At the same time, you are doing the imaging, and imaging guiding you where to give the dose, where the dose should not reach, is it reaching there, and all those things by image guidance you can do. And by brachytherapy, all kinds of tumor cancers, vagina, breast, esophagus, cervix, nasopharynx, prostate can be treated. And the evolution has taken place in radiotherapy after just 1895 discovery of the X-ray. As I told you, initial period was of the deep therapy. 1950s cobalt therapy has come up. 1960s we have got a linear accelerator, variable energy, this thing. Then we have moved computerized treatment planning, 2D to 3D to this thing. Then we had 1970 CT scanner, that is you have got a 3-dimensional imaging. Then 3D planning has come, accurate dosimetry has come. 1990s, now we have MRI, PET CT, that is not only the functional image, molecular imaging, you can say functional imaging we have got. the multi-leap colorimetry I will show you. With that, how you can confirm the radiation dose to exactly to the tumor, no spillage of the dose outside the tumor, optimization, IMRT, intensity, modulated radiotherapy, image guided radiotherapy, and these all involvements. You can see 1935, all these beams. This is the what we call as a dose bath, huge, all area, even the organ at risk and other things used to get. And my dose, I could go only to 40 gray. Thereby, I will not be able to achieve the complete tumor control. But with the cobalt 60, I could raise the dose to 60 gray to the tumor. At the same time, spillage of the dose outside decrease. With linear accelerator, escalated dose, and you can see the development, LINAC 3DRT, protons, and now with the carbon ion. As you like the shape of the tumor, sparing normal tissue outside this thing OAR, and the dose from 40 gray to 100 gray, tumor control probability has hugely increased. At the same time, spillage of the dose outside the tumor volume is not there, the entrant beam and all those things. So the integral dose, normal tissue dose has tremendously reduced, tumor dose has increased hugely. Even another example of the brain tumor, 1980s, a parallel opposing field, and then pituitary, pineal gland, everything was irradiated, affecting the sex hormones and this thing. Now you can see with the 3D vertex field, then 3D conformal, IMRT, and proton. Now exactly the tumor, no spillage of the dose, no other part of the brain, the pituitary, the glands doesn't get any kind of dose outside this thing. That is how accurately able to deliver, spare the dose, spare the organ and things. And this is what IMRT is: minimal toxicity, decreasing the dose in the OAR and increasing the dose in the tumor, maximum local control. That is what was happened. Again, you can see how beautifully we can, this planned tumor volume in this thing, you can give a radiation dose, organ at risk, absolutely no dose sparing. As the tumor is there, you can sculpt your radiation ISO doses. And not only that thing, if you want to have tumor stem cells and where hyperactivity tumor is there, you can increase the dose, and that is what you can do, create a hotspot and maximize the dose and cure and spare the normal tissues and all. So IMRT, any kind of, with the image guided, you can have sculpting of dose, sparing the normal tissue, normal organ at risk. So IMRT allows higher doses of radiation to be delivered to the tumor while sparing more healthy surrounding tissues, organ at risk. And this is what an image guided onboard imaging on the linear accelerator. We have now MR LINAC. In one machine, we have magnetic resonance imaging, and the same table on the same thing, linear accelerator. All those things are increasing. Also, real-time tumor tracking system for gated radiotherapy. Here, breathing in, breathing out, movement of the tumor is being tracked with this camera. And accordingly, when the tumor is in the site, the beam will be on. When tumor is outside or any organ, critical organ has come into the beam site, the beam stops so that sparing of the dose takes place. Now, in 125 years, we have moved from a population-based radiotherapy to a personalized radiotherapy, individualized plan, image-guided treatment, plan adaptation as required. And that is how, here you can see again, you are during the treatment, you are monitoring and you are adapting to this thing. Is the image guided technique, that is a position management, how you can do with this thing. So this 1906 imaging simulation, CT sim, MRI, PET CT, 4D CT, 4D PET CT, PET MRI, and this all guidance imaging, biological imaging, and functional imaging has helped us to delineate the tumor and the tumor activity as well as the normal tissue and all. And this is how a precision radiotherapy has come up. We do the imaging, and imaging is a part and parcel of radiotherapy. And as I told you, modern imaging gadgets not only give you the anatomical imaging, but the functional molecular imaging. And then you do the planning on the computer 3D, 4D, and precise. This is the multi-leap collimator. They go on moving and they just adjust to the tumor, and then we deliver after stimulation. So it was a non-digital conventional radiotherapy analogous to carpet bombing about 30-40 years ago. And now it is like a pinpoint laser bombing. A cell or a pixel is targeted. IGRT is analogous to precise targeted bombing, and the collateral damage is reduced by much below 5%, escalating the dose, preserving this thing. So these are the multi-leap collimator, how you can then shape your doses into the tumor. And we have a tomotherapy machine where you have got in the same machine, you have a CT scan and the linear accelerator. So you scan on the same table, plan and treat there and there itself. So there is no movement, no disorientation of the patient. And you can see with the tomotherapy, different sites can be treated. There is no tube bath, just this spinal cord malignancy or secondary, you can treat in a different way by sparing. So you have again the cyber knife 6-arm, we have a gamma knife for the brain tumor where 192 radioactive sources focusing the radiation dose into just a small area and ablating the tumor by giving very high doses. I talked about the MR Linac. In a same machine, we have got a MR on-board imaging. At the same time, we have the linear accelerator in the same room, same setup. We have intraoperative radiotherapy. We have minimal surgery. That is just taken tumor, and then earlier you used to close it, then come the patient after wound healing, 3-4 weeks, again radiotherapy, intra, there and there's a theater. You can treat this, give this patient, close it. Preservation of the organ also takes place. We have now what do you call it as a mammocyte. So just a small drill you do, put this applicator, connect to the brachytherapy machine, give the radiation source, and then implant it, deplete it, take it out. So no loss of the tissue, no loss of the organ, no disfigurement. You can preserve by this mammocyte. Intraoperative radiotherapy also in many other areas we are able to do where you can. Then we have now proton or particle therapy, and in India already one center at the Apollo Cancer Hospital is functioning. A second center this month itself will be initiated in Tata Memorial Hospital, where proton has a biggest advantage is that the cobalt or linear X-rays, they go on depositing the energy and they exit. So this is the way the entrance and exit, even tumor is here, entrance and exit doses. But the proton, you can see you have a Bragg peak. They will not deposit while traveling in one place. They will just go and deposit completely. So you can use a huge doses with the proton therapy. And here you can see with the proton therapy, this complete the brain tumor, highly precision allows complete sparing of the brain stem, no dose to the other part, maximizing dose into exactly the tumor things. Again, you can see with photons, this is a dose given, protons only to the cord spinal, no underneath and over doses. Again, you can see it here image here with the protons or the particles, very nice confirmation of radiation doses. Another example of the protons, you can see it here. When I'm going to treat with the photons and IMRT, there is spillage of the dose outside the tumor volume, but with the protons, exactly. And one example, you can see that standard photons, 101% dose to the cochlea, 33% with the IMRT, and just 2% with the proton. So you can spare the pituitary pineal glands and these things by appropriately. Again, now we have a flash radiotherapy where high intensity radiation doses, 40 gray per seconds are given. In a second, in a fraction of second, the radiotherapy is given, and thereby this has shown radiobiologically remarkable sparing of the normal tissue and ablation of the tumor. So flash therapy is also going to help you to spare the normal tissue, organ at risk, and ablation. Now, what are the challenges to providing organ preservation and preservation of fertility while treating patients with cancer radiotherapy? This is my last part of the talk. The biological community faces 4 main challenges related to the preservation of the fertility in the people with cancer. The improvement of the patient-specific life-preserving treatments, the identification and reduction of the threat that cancer treatment poses to fertility, the expansion of safe and effective options for fertility treatment, creation of symptoms management plans for patients who lose endocrine function from the gonads as consequence of cancer treatment. And as I told you, when we are going to give a pelvic irradiation, and then now with the high-tech radiotherapy technologies, earlier it was a box technique or flat pelvis used to get a uniform dose. Now RT is non-uniform. The use of conformal radiotherapy and IMRT have sculpted the RT to minimize the exposure to surrounding organs, and therefore even pelvic RT usually associated with the decreased sexual function to 23 to 56% to the dose of 22 24 gray. Now RT is able to spare one ovary, or even you can do a transplantation of the one ovary to the lateral abdomen and treat cancer cervix or rectum or the pelvic cancer without giving the dose to the ovaries, which will cause the sterility. And that is what 2 to 5 gray can decrease fertility, lead to early menopause, but you can keep these doses less than 2 to 5 gray into this thing by just, you can see it here. This is the tumor where we are able to give more than 40 to 50 gray, but this will receive less than 2 gray where your ovaries will be spared. Or also you can see it here how dose you can make it, plan it that this area only gets less than 2 gray. And this is how planning you can do, you can simulate, and you can do. So fertility challenges in men with cancer are less complex than those women because of the relative ease of the sperm banking. However, several factors can negatively affect male fertility, including disruption of the hypothalamic pituitary gonadal axis, damage to germinal epithelium, and depression related to diagnosis of the cancer. They can be there. A permanent sterility or infertility may occur at the testicular doses which are more than 6 gray, with lower doses leading to temporary azoospermia lasting for months to year. And these are the doses. If less than 1 gray dose, it is 9 to 8 month recovery will be there, 2 to 3 gray, 30 months, and around 4 gray, in 5 years, this will be there. But now we have a testicular shielding also. These are high atomic number testicular shielding. You can do this thing, treat here, so that testes do not get the radiation more than 1 gray and producing the sterility. So we have these gadgets available. So the direct toxic effect of chemotherapy and radiation exposure on the gonad is generally dose dependent. I will not speak about the chemotherapy. I'm not expert of that thing. Dr. Hemant will be talking about that thing. Efforts to tailor standard therapeutic regimens to the specific disease and fertility sparing therapies and fertility preserving are available, can be planned more towards children with cancer who are growing, who are. A lot of publications are there regarding this. Can you exclude the pelvis in the childhood survivor, how it can be done? The publications are there. I will not go into details of this because of the time paucity. I know already we are running 1 hour late. And these are some of the review papers which have come, and they have shown that with the modern radiotherapy technology, the preservation techniques, hormone replacement therapies, we can preserve. Organ preservation is excellent in radiotherapy. And then again, female onco-fertility, current understanding, therapeutic approaches, contrast in future. Also, that review article talks about with the recent knowledge, recent expertise, we plan and we do those things. So a lot of publications. So this is recent advances are tailored, personalized approaches, reviewing radiation dose with advances in external beam radiotherapy, including IMRT, the stereotactic radiotherapy, stereotactic radiosurgery, stereotactic body radiotherapy, image-guided radiotherapy, and brachytherapy and particle therapy spares organ better while selectively targeting tumor-containing regions. So optimal patient positioning and organ tumor motion tracking, onboard tracking we can do, management, also optical to minimize the therapeutic ratio of the high precision RT, organ preservation and fertility. It is doable, it is possible and being done with this thing. So first and foremost thing is a safety first. With having that approach, we can tailor-made our treatment specific to the patient, specific to the need, and we can achieve. Once again, I thank the organizers for giving me the opportunity and for this prestigious award of Professor L.S. Ramaswamy Oration. Thank you very much.
Thank you, Channa ma'am, for chairing this session. And thank you, Dr. Chogle sir. It was really a great talk, and we wish you a very happy birthday from all of us. Now, we are moving ahead to the last session of today, which is on reproductive tract cancers. And for this session, our chairpersons are Dr. Pradeep Kumar Ji, who is a scientist G at Rajiv Gandhi Center for Biotechnology, RGCB, Trivandrum. Dr. Pradeep's research interests are development and differentiation of testicular germline stem cells, factors regulating control of cell division through meiosis, and the structural organization of spermatozoa, making it capable to fuse with the oocyte. Welcome, sir. And the other chairperson is Dr. Rita Singh, Professor, University of Delhi, and whom we have just heard, and she gave a great talk in session 3. So we welcome both the chairpersons, and I request both of you to please chair this session. Thank you, sir, and thank you, ma'am. Over to you, please.
Okay. So am I audible?
Yes, yes.
Oh, okay, great. So
yeah.
All right. Yes, Dr. Professor Rita. So, so let us get started. We have in this session 1 keynote lecture and 3 invited talks. We met Dr. Suranty Sharma earlier today. It was midnight over there. He received the Lifetime Achievement Award. So I'm not going to read out all his biodata because already people know him. But we kept him awake the whole night. So that he receives the Lifetime Achievement Award, and we made him get up early in the morning over there. And Rhode Island is a nice place because that is this just Caprio, and I love Cotton Providence, and I think Dr. Sharma also must be watching. And it was fun having Dr. Sharma in Trivandrum 10 years ago, and he has not aged, and he's a great stage performer, and we are missing it because it's online. So with this, let me invite Dr. Sharma to deliver his keynote lecture on programming of autism in the mother's womb. And we know about Abhimanyu's story, so the kids, the fetus starts learning in the mother's womb. It has nothing to do with reproductive tract cancer anyway. But yes, Professor Sharma, we are keen to listen to you.
Can you see my slides, please?
Yes, Professor Sharma, yes. All set.
Okay. Thank you very much, Dr. Pradeep Kumar and Dr. Rita Singh. I hope she's there. Let me start with my thanks to Professor Loya, Dr. R.S. Sharma, and the organizing committee for giving me an opportunity here in this important meeting to present our recent work. Yesterday, some of us were awarded Lifetime Achievement, were honored with Lifetime Achievement Award. So I would also like to congratulate at this time my co-awardees. So let me get back to my talk because we are running out of time. We are a bit late. So I'm going to talk to you today for next 25 minutes or so on programming of autism in the mother's womb. I was giving a talk a few months back, similar talk, and some people in the audience thought it was a very strange thing of telling them that autism can be programmed in the mother's womb. The reason is that we know genetic factors, environmental factors, but besides that, 60% of the cases still remain unexplained. So we need to really figure out what is happening. So I was giving this talk and there was a very tricky question that was asked by one of the audience asking me, can the fetus be educated in the mother's womb? So I paused for a second and then I remembered something that we have heard and read since childhood. And the story is Dr. Kumar said Abhimanyu. So Abhimanyu came to my mind, and you all know the story about Abhimanyu, that he learned how to get into the war zone in his mother's womb, but he did not hear rest of the talk by his father to how to get out of there. So this is the example that I gave them, and I don't know whether they were convinced or not, but this is the example that I gave them, and I strongly believe that yes, it's possible to educate the fetus in the mother's womb. So coming back to autism spectrum disorders, so it's not a monogenic disorder, it's a compilation of different disorders, and they may share etiology, but we will just go through some of the aspects as I go forward. So children and adults with ASD might repeat certain debilitating behaviors and succumb to unscheduled serious health risk and mortality. The problem is no cure exists for ASD and there's no one-size-fits-all treatment. So that's the issue that we need to really understand. And the reason we don't have any cure because we don't have any models, animal models. We cannot suit drugs in humans. So you need to come up with animal models to really figure out what pathways to target and what to do. And so although a number of etiological factors have been described with no well-defined association or experimental evidence, now sometimes we need to have public awareness about a disease and we need to have a reference. So this is the movie some of you might have seen, Rain Man. It was a beautiful movie and they depicted the characteristics of an autistic adult, how they behave, how they get into these repeated behavioral traits that if they lie down in the middle of the street, that's what they will do. They will not get up from there because that's what they want to do. So those are some of the things, and then they develop severe neurodevelopmental issues and they succumb to mortality. So ASD, as I pointed out, is a heterogeneous disease. And what is really important here, the recent CDC data here, Center for Disease Control, suggests that 1 out of 54, all 54 children, 1 out of 54 children will develop autism-like features. And it's what is important here, it's 4 times more in boys than girls. And it's ever increasing phenomenon. Every year we see huge increase in the cases of autism, particularly in developed countries. I don't know what is the situation in India because we do not have a national registry there. How many kids succumb to autistic features. So I can't tell you, but in the US and in some developed countries, we have these national registries. In the US, we have now data 1 out of every 54 children develop autism. So it's a very high rate of this disease. Now, how did we get into this business of autism? Number of people in my lab, they were studying maternal infections with pregnancy complications. Dr. Hanna was a clinical fellow, and now he's chief of pediatrics at New York University. And then Professor Bloy was a visiting professor for a couple of years. She worked on the similar project, and Jess Thaxton and Tanya Nevers were PhD students. So what they did, they found out that if they challenged the pregnant mice with viral mimics or inactivated viruses, that could lead to either fetal loss or preterm birth, depending on when these things were administered during pregnancy. And recently, we are also working on COVID-19 disease in pregnant women, whether do they have effect on some features like autism. So let me take you through why we started it. So these are the experiments that we had already done, that we had injected post-implantation, and that lead to, this is a viral mimic poly-IC double-stranded RNA. So that led to fetal resorption. And if we injected that on day 15 of pregnancy, the mouse pregnancy is 20 days, that resulted in preterm birth. And this is exactly what happens in humans also. Viral infections in first trimester can lead to fetal loss, and later on, they can lead to preterm birth. Now, but what is important for us, what really happens here if we inject same mimic around day 12.5 of gestation? But what is so significant about day 12.5? This is where neurogenesis starts. So you have implantation, placentation, neurogenesis, blood-brain barrier establishment, and of course, that's the time of parturition, birth. So question was, what will happen if we challenge pregnant mice with viral mimics or bacterial products or live bacteria? What will happen to the fetal growth, fetal brain development? So this is exactly what we have done. Now, before we started our work, we also looked in the literature. Was there any evidence for maternal infections during pregnancy and the incidence of autism? And there were a number of papers popped up. I have just listed 3 here. And these are all clinical observations that yes, there's association, temporal association of infections during pregnancy and incidence of autism. Furthermore, there have been recent studies in humans and in mice, which suggest that immune angle in the pathogenesis of autism, particularly focus has been on this cytokine called interleukin 17 and interleukin 23, and these are human studies. And these cytokines are produced by a particular T cell type, which is Th17 cells. And in mouse studies, they have demonstrated that IL-17 is really a consequential for the onset of autism-like features. So these are the references that we had access to. So we decided the problem here is that there's no IL-17 producing T cells during normal pregnancy. The question is, where are they coming from? So, as I said, they are not present at the maternal fetal interface. So what is the source of IL-17 in the placental microenvironment? And that's when Dr. Sukanta just joined my group. And his concept that we have developed recently have been very hot ticket, and we have been requested by a number of high impact journals to really describe our concept. And these are very recent articles that we have published. Now, the questions that Sukanta asked, okay, what is the source of IL-17? Where are these Th17 cells coming from? And these are present in human brain at the maternal fetal interface. And some people have also demonstrated that they are also present in the brain tissue, particularly the cytokine IL-17. Now, if they are not present, what is really contributing to the propagation and amplification of these cells at the maternal fetal interface? So our concept was, do uterine regulatory T cells? We have tried peripheral blood regulatory T cells. We cannot convert them to TS17, by the way. So this is the uterine regulatory T cells transdifferentiate into TS17 cells at the maternal fetal interface. Are these cells, the transdifferentiated regulatory T cells, are they associated with pathology associated with autism? And can depletion of these Tregs or recruitment of normal Tregs inhibit the onset of fetal neurodevelopmental defects that we are describing here as autism? So this is experimental design, different times, the injection of poly-IC. I'm just taking an example, poly-IC, but we have injected a number of things to see, and at different times, and we have collected tissues. So this is a very simple experiment, which is a FACS analysis. You basically do these experiments to type the immune cells. And this is a peripheral blood, splenic population. And we were looking for TS17 cells, which are recognized by this transcription factor called ROR gamma T plus. And FOXP3 is a hallmark marker for regulatory T cells. So this is the splenic T cells. We did not find any propagation amplification of these hybrid Treg and FOXP3 cells. However, when we looked in the uterus in response to poly-IC, there are no cells in saline treated mice, but in poly-IC treated mice, we saw a huge increase in these cell types, which are double positive FOXP3 and ROR gamma T. And this is what's surprising to us, that in periphery, we do not see any increase, but in the uterus, we are seeing a huge increase in these cell types. So that was the first indication that yes, it is possible that you can generate TS17 cells at the maternal fetal interface in response to viral infections. Now, does it lead to IL-17 production? So this is injection of poly-IC at early stage of pregnancy, day 6.5. Only thing you produce is the TNF-alpha producing regulatory T cells. However, when we inject at 12.5, all of a sudden you don't have TNF-alpha producing regulatory T cells, but you have now IL-17 producing T cells. So it's a very temporal window of infection that would impact the production of TS17 cells and production of IL-17 cell cytokine. And this is again further. So we have done it day 6.5, 12.5, and 15.5, and this is IL-17A. And you can see this only comes up when we inject poly-IC day 12.5. Day 15.5 is mainly TNF-alpha, which leads to preterm birth. And here is also TNF-alpha that leads to fetal loss. So the question was, is uterine Treg TS17 transdifferentiation associated with cortical architectural changes and transcriptional alteration in the cortex? So before I show you our data, I want to draw your attention to this wonderful article, seminal article, published in New England Journal of Medicine in 2014. On what these authors, this is a post-mortem tissue from autistic children who died. So these investigators have taken stacks of brain tissue and created an image, and what they found that there was a patch-like structure in the neocortex here, and they called it invagination. And so this is a cortex disorganization in autistic kids in a post-mortem tissue that they analyzed. So we tried to look whether in our case, the mouse cortex tissue would show the same kind of disorganization in response to poly-IC. And this is what Sukanta did here. These are different layers of neurons and cortical layers. And these are immature neurons, intermediate neurons, and these are mature neurons here. This is about ascending migration of neurons to the neocortex. And this is a saline cortical tissue on day 14.5. And same tissue, when he looked in poly-IC treated mothers, the fetal brain development showed us the same kind of disorganization like what they had seen in humans. This is a patch-like structure. We see the same thing here. Now, he did something very important, Sukanta, that he looked at, these are the genes identified in human brain tissues. Some of them are associated with development of cortical interneurons or axonal guidance or neuronal polarity and synaptic plasticity. So he looked, and what the people have shown in humans, that these genes are downregulated in autistic children. And this is exactly what we found in our modern mouse cortical tissue, that many of the genes were downregulated. Some of them were not affected, but many of the genes that have been looked at in human tissues are also undergoing downregulation in our mouse model. So Sukanta also did, this is a immunohistochemical analysis for some of the proteins that I discussed in the previous slide. So you can see this is a saline. So you can see homogeneous staining for this protein TBR1. And these are the intermediate neuron proteins. And you can see the SATB2. And that's only expressed in the top part of the neurons. These are the mature neurons. And this is a merged picture of it. So you can see here the TBR1, and then this is the SATB2 here that is expressed. But in poly-IC treated animals, first you see down regulation of this protein. You don't see the similar kind of expression or profile. And SATB2 was severely down regulated. And you can see the merged picture again does not look like what we see in saline treated. And I'm not going to waste my time. This is the actually structure of the cortex here. And we see that this is the reason this is affected by poly-IC treatment. So question is, if that is the case, if Tregs are the culprit, how can we use this information for therapeutic intervention? And so question would be, can we deplete regulatory T cells at the time of neurogenesis and restore normal brain development? Also, can we replace bad regulatory T cells with good regulatory T cells? And here we use our information. We had published a paper in 2011, and we suggested that human chorionic gonadotropin, if the Professor Talwar is sitting there, he would really like this piece of data here, that HCG can also recruit good regulatory T cells to the pregnant endometrium. So we try to use this also approach, whether we can replace bad Tregs with good Tregs. So, oh, it's frozen. Okay. So this is what we have done, Sukanta has done. He depleted these cells in pregnant mice. And this is a poly-IC. So you can see there's a huge increase in the TS17 cells. But when we depletes regulatory T cells in the system, this cell number goes down, and that also applies to the production of IL-17 cytokine. So this is exactly what we expected, and this is what we have found, and these experiments have been repeated multiple times. And this is the cortex structure. In saline, there's no problem. In poly-IC, you see patch-like structures. But when we deplete animals of regulatory T cells, these patch-like structures disappear. There's one or two still remaining. And we have done these experiments. We have injected antibody multiple times, and we have gotten rid of these patches also. So this was quite satisfactory to us that yes, we could play around these games and deplete these cells and restore normal brain development. And this is again the staining with these proteins. So in the IgG, isotype match IgG treated animals, you still had patch-like structures, and expression of TBR1 SATB2 was compromised. Whereas when we depleted these regulatory T cells using CD25 antibody, we could restore normal cortex architecture. So this was quite surprising to us and quite satisfying that yes, we can really target regulatory T cells for therapeutic intervention. Now, so this is in humans. How do you really look at, because autism is associated with severe behavioral problems. So how do you really go about checking whether what you have done really correlates with behavior in autistic children. So in humans in the clinic, we use we use a technique called ultrasonic vocalization in the clinic. So these autistic children have a totally different pitch than normal babies, and cry also. Their cries are different because, so we use this apparatus. We bought it for $15,000 from Germany, and we subjected our pups to this device, and this is exactly this. So this is the kind of pitch, you heterogeneous pitch that you observe in normal pups, but in pups from poly-IC treated moms, that pattern is totally disturbed. But when we look at this thing in pups from CD25 or regulated T cell depleted moms, we can restore the same kind of acoustic pitch that we saw in saline treated. And this is a quantification of the data where in number of pups that we have done, that poly-IC compromises this ultrasonic vocalization, whereas we can restore it by removing regulatory T cells from the system. This is a couple of other behavioral techniques that we have used here. This is the what is called in clinic, we do that, a marble burying index. So this is a normal pattern. The normal babies would not disturb these marbles. They would not really take them to the bottom. Whereas the autistic children, because they have repeated behavior, they will just dig the marbles deep. And what this really shows here, that this is a male-centric phenomenon. In female pups, we did not see that. And this is what I told you, that autism is fourfold higher in male children compared to female children. So this is what we see here. And this is the Neslet shredding index. So autistic kids, because of their repeated behavior, they keep tearing the piece of paper in 1,000 pieces, whereas the normal children would not do that. And you can see again, it's a male-centric phenomenon where we see much higher activity in male pups compared to female pups. So the behavior-wise also, our approach is working out. Now, this is again, we have done, so question is, can CD25 antibody treatment or depletion of regulatory T cells can restore these behavioral changes? And you can see here male, because we did not do these experiments in female pups, but in male pups, you can see saline does not make any difference, whereas poly-IC increases the marble burying index, whereas the depletion of regulatory T cells can restore normal activity. And that is again for the shredding. So it's the same thing, that we can really restore normal behavior by depleting regulatory T cells. Now, clinically, depletion of regulatory T cells won't be feasible, because that's regulatory T cells play a number of roles in different systems. So we try to do the SCG treatment. I told you that we had published papers where we demonstrated that SCG can recruit good-looking regulatory T cells to the maternal fetal interface. And you can see here, and we have done the same series of experiments. I don't have time to take you through all the experiments that we have done, but similar, in parallel, we have done similar experiments. But this is important here, that this is a shredding and marble burying techniques. And you can see with SCG marble burying, you can really restore normal behavior. And by the way, I'm not showing you the data, but SCG also blocks transdifferentiation of regulatory T cells into TS17 cells. And same thing for shredding index, that SCG can normalize shredding index also. I'm getting some linings on my screen, so I don't know what they are. I have no idea. Anyway, so question was that why do we have the higher incidence of autistic features in male children versus female? So we looked at the IL-17 and IL-6 production, and you can see in poly-IC treatment, IL-17 receptor induction in brain tissue is much lower in female pups compared to male pups. Similarly, IL-6 production after 6 hours of treatment poly-IC in female pups is much lower compared to male. So these are some of the same thing. This is the ROR gamma T expression in response to poly-IC. In female pups, we do not see similar level of propagation of TS17 cells. So some of these features explain why female, and now I don't have time to tell you why IL-17 is not expressed at that high level in female placenta compared to male placenta. We have huge set of data explaining that. But this is exactly the information we got, that the reason why male pups or children have higher incidence of autism is because what is happening at the placenta level. Now we have also done transcriptomic analysis, whether male differ from females, and this is a volcano plots here. So this is a brain tissue, cortex tissue, brain tissue on day 15 of pups. And this is the male pups here. You see a huge number of genes, are almost 404 genes are significantly affected, and they are upregulated. And now if you look at the female brain tissue, only few genes are upregulated in response to poly-IC. So this is, again, at the transcriptome level, we are seeing differences in male and female brain tissue. Now, this is very important. So we have completed this set of studies, and you can see here, this is a principal component analysis, bioinformatics analysis, where whole cortex transcriptional signature between indicated groups. So you can see this is saline right here. Poly-IC has huge day and night difference in the transcriptome profile, whereas CD25 antibody or SCG treatment can normalize it and bring it back very close to the saline treated animals. So this approach is working out very well, that we can affect this even at the transcriptome level. And again, we have done male and female, and males are the ones that really saw this huge difference. Now, this led to something very intriguing, and we did not really expect it. So when we did analysis of genes here, we found out that this particular gene, ADAR1, which is the adenosine deaminase acting on RNA enzyme, this is involved in RNA editing, is hugely affected in response to poly-IC. So we did not really know. We did not know much about this gene or protein. But when we did literature survey, we came across this article in Nature Neuroscience, that widespread RNA editing dysregulation in brains from autistic individuals. And this really has taken us to a different avenue now of research, where we are focusing a lot on ADAR1 and targeting this pathway for therapeutic intervention. So let me just show you a slide or two of this data. This is ADAR1 expression in brain tissue, cortex tissue. This is a poly-IC. You can see compromised expression of this protein ADAR1 in brain tissue in pups from poly-IC treated moms. If we deplete regulatory T cells or treat these mice with the SCG, we can restore normal ADAR1 expression and normal cortex structure. So these are some really high impact observations, where now we can target this pathway, ADAR1 pathway, or regulatory T cell pathway with SCG or depletion of regulatory T cells. So the problem is, initial problem is regulatory T cells that are transdifferentiate into inflammatory TS17 cells, and they produce IL-17, and IL-17 affects brain development, and that leads to also higher IL-17 receptor A expression in the cortex in the brain. So these are some of the important avenues that we are now pursuing at this point. So we have huge amount of paper is in revision in Nature right now. We submitted this work to Nature, so they have some questions, which I expected. So hopefully it will work out in our favor. So this is the conclusions and future studies. Now, as I told you, we do not have any cure for autism at this point. The reason is we do not have any well-defined animal models. We feel that our mouse model that we have developed mimics all the behavioral cortex, architectural, and transcriptomic features of human autism. And this is the scheme, the mechanism that we think that something goes wrong at the placenta level in response to infections. And this is a temporal infection. Particularly, we have data from influenza during pregnancy, and particularly women who have very high temperature having influenza. In those moms, there's a high incidence of autism, children born to them, and particularly male offspring that experiences autism-like features. So this is what is really happening, and we are very high on SCG because people have used SCG in different ways. It's a pleiotropic hormone. It's a pregnancy hormone, but it also is expressed in different cell types. But we are very high on SCG because depleting regulatory T cells would not be clinically feasible. So we are trying to work out, and our patent has been filed and it has been revised. So we think that in utero, during pregnancy, if someone has influenza or high fever or viral infections, if we can treat them with SCG, that would really help the brain development, and we can reduce the incidence of autism. So we are now doing RNA sequencing of individual male and female placental units, RNA editing targeted RNA sequence of cortex, where I talked about ADAR1, and then we are really focusing on SCG for its therapeutic significance, at least in the model that we have developed. Nobody would let us shoot SCG for autism in humans at this point. So we are trying to convince people that yes, this will work, but we need to first show in animal models that yes, it works. And so this is working out very well. So these are the people. This is the very important slide because I'm just giving you a talk, but these are the people who really did the work. So Kanta Jas is responsible for most of the work with the help from other people in the lab. And these were clinical fellows who were working in the lab. Some of them have left, some of them are still working. This is a clinical core staff they collect. So we have started a new, for humans, we are detecting IL-17 and TS17 cells in cord blood at the time of delivery. So if we collect cord blood and if we can detect IL-17 and TS17 cells in cord blood, that would be a predictive assay for us, for we can make a statement that yes, these offspring are likely to develop autism-like features. And of course, we have some collaborators, local collaborators, and this work won't be possible without the funding sources that I have listed here. Before I let you ask me questions, I just want to make a pitch for my journal. I edit this journal, so please submit your A category work to this journal. It has a very respectable impact factor. And I'm looking forward to having some articles from India. Actually, we are coming out with a clinical reproductive immunology in India special issue where Dr. Jb Sarma and a few others are Dr. Iss Rf and Dr. Tarunamadan, Deepak Modi, and Mohan Raut from Bombay. They are the guest editors. So please submit your A category work to this journal, and we'll make sure that it gets the proper review. Thank you very much.
Thank you very much, Professor Sharma. Though we are running late, I think we will allow one question in case somebody has any question. Otherwise, you might feel very bad. Is there any question from somebody?
I have a question.
Okay, go ahead.
Professor Sharma, first of all, a very wonderful talk. And I was just wondering, so during infection, is it that the testosterone level rises or HCG level goes down?
Well, if you remember, I said that this is the second trimester. Second trimester SCG levels are intrinsically very low. They are very high during first trimester, but second trimester they are very low. So that really does not apply here. What we are seeing here, if SCG was there, that's what we are proposing, that SCG would block transdifferentiation of regulatory T cells to TS17 cells. So that's why we think we are very high on SCG, because it goes down in second trimester. So to replenish that, we are trying to work on SCG. We can use that as a therapeutic option.
So in these babies who develop autism, so has anybody looked at, there is a connection with testosterone, I understand, but has anybody thought of looking at dihydrotestosterone?
Well, testosterone story has been refuted.
Okay.
That didn't pan out. That didn't pan out. So you are absolutely correct. Some people from England had published on testosterone connection with the autism, and particularly, and that their idea was that since it happens only in male offspring, testosterone must be playing some role, but that has been refuted.
Thank you.
Okay, so thank you, Professor Sharma, for the lovely talk. More questions are there, then we will communicate over email to you. So, Professor Rita,
just one question, Pradeep.
You really want to ask a question right now? Okay.
Just quick one.
Very quick, very quick.
Dr. Sharma, it was wonderful, wonderful journey of your research ending up. My straightforward question is: have you ever seen or got a chance to see a correlation of C-reactive protein in early second trimester of pregnancy with the kind of outcome that you are seeing?
So C-reactive protein is part of this milieu that people always look for inflammation. So C-reactive protein is there, but we cannot really, so that is just part of overall inflammatory milieu. So we cannot figure out what is really inducing it, and it's not a homogeneous finding that in some people, some cases you can see CRP, but others you may not see the same level. So that's why people are not pursuing that line of investigation. That's my understanding. I may be wrong.
Okay, so
I will ask in the email then.
Yeah. Okay. So, Professor Rita, are you there?
Yes, of course.
Okay, so will you please introduce the next speaker?
Okay, thank you. Can go on now. We all know Professor Suri very well, and he's one of the eminent professors and scientists in our country as well as in the world. And we all know that he is a great translational cancer researcher who has various laurels to his credit. And if I keep reading, it will take many pages, but some of the few, as you know, he's a fellow of the very prestigious National Academy of Medical Sciences and National Academy of Sciences. And
Rita,
then
we can skip that. Rita, we can skip it. That's fine.
Okay, yeah, everyone knows you very well, and
no, it's okay. Yeah.
So, and we all know his seminal work on SPAG9 and all the other achievements. So I would invite him to give the talk, and let's have it from him.
Thank you. First of all, I would like to congratulate all those who have got the awards and achievements of their long journey to reach where they could put their awards in form of the platform which ISSRF offers. And I'm going to talk about the reproductive health for all, which is actually a Sustainable Development Goal 3, which defines as a good health and well-being for all. And primarily in this, before I go on for my general journey of the last 32 years, I would like to first of all acknowledge the team of dedicated oncologists, surgeons, pathologists, and gynecologists from the various hospitals, various collaborators who have been working with me. And of course, Dr. Nirmala has been all through this journey with me and a pillar standing behind whatever we have achieved so far. And of course, the funding that we got, generous funding for all kinds of work that we have been involved in. And I think I have been blessed or lucky to have a continuous funding for cancer research program for the last 15 years from Department of Biotechnology. No other project gets so long sustainable support to actually take your further work down. And then DST, ICMR has supported me in a big way for ovarian cancer trials. And of course, there is ICR that we work on celiac disease where we are trying to characterize gluten's actually allergies in India. And having said that, our program is basically focused on developing kind of a milieu and
Dr.
bridging the
Suri,
gap.
excuse me. Could you please put the slideshow on?
I have put the slideshow on.
Okay, we can see the speakers also. So I thought maybe we are seeing your full screen.
Yeah, I'm also seeing the full screen. Yeah, it's still in the slide sorter mode. Anyway, that's okay. Yeah.
No, it is in the correct mode. I can see
Okay,
it properly.
now it's okay. Now it's okay.
It's fine.
Now it's okay. Sorry.
Is it fine?
Slow.
Yeah, now it's fine.
So eventually we have to actually bridge the gap between the clinicians and the scientists, and that way working together can translate some of the new scientific discoveries that could eventually help in improving the care for the cancer patients. So basically the forte is towards the cancer patients, which is a major actually disease burden as far as a non-communicable disease is concerned, that is defined in SDGs Goal 3. And we also know that there is a lot of input that we can have from a biotechnology point of view, which India wants to have a kind of sector which is recognized as key driver for contributing to India's 5 trillion economy by 2024. So having said that, I will just skip the slide. So having said that, this is just eye-opener kind of thing, and everywhere in every talk that I always emphasize that this is some kind of disease that we see causes so many deaths. For example, cervical cancer death is basically one patient is dying every 8 minutes. Every 13 minutes in India, one woman dies because of breast cancer. So every 50 out of 50 women, one woman dies because of ovarian cancer. And of course, there are 2,500 people die every day because of tobacco-related disease or because of lung or oral cavity cancer. That's a huge burden. And to address that, now my slides are getting. So to address that, before I get into, I just want you to focus on something that always actually bothers me is that cancer kills globally more people as compared to if you total up number of people dying because of AIDS, hepatitis, tuberculosis, and malaria combined. And that's because of the fact that in our developing countries, especially in India, we are 70% odd living in a rural population where there is less awareness and there is a limited medical infrastructures available. So this actually contributes to the whole burden that we are seeing as far as India is concerned. So how to go about it? And well, these are the certain slogans, but then one has to put it in the practice becomes a very difficult option. So prevention, there are so many environmental factors, there are lifestyle factors, there are dietary factors. So these are the factors which can actually be prevented, but then the things keep going. But so then detection and diagnosis. Again, in India, we don't have any such plans or missions where we can make people come for early detection diagnosis unless and until a patient has a clinical nuisance value. Patient doesn't walk into the clinic. And then treatment options, modalities actually are left with a limited, options with the clinicians where they always feel that how to treat the patient, and they are at the fag end of their life. So early detection saves the lives. Answer is yes. But how do you control the cancer? We normally, you have heard about and everybody knows about that there are 3 arms that are existing right now, and there is the 4th arm which is being added of late. And in cancer patients, the surgery, radiotherapy, and chemotherapy are the 3 major options, which actually every cancer patient, if it is curable or surgery can be performed, then these 3 options are given to the patient. But if a patient is walking in the later stages, stage 4 or advanced stage, who fails the 2 line of systemic treatment, there is hardly any treatment options left for them. So that's another big challenge. So our forte has been a bedside to bedside translation outcome with the bedside to bedside reverse translation. I like this kind of slide because the whole concept of my lab 30 years ago, when we started, it was very difficult for us to convince in a milieu where, in an ecosystem where most of us were involved in doing some kind of basic research. And basic research doesn't mean that you can't do basic research with the patient tissue samples. You can do basic research with the patient samples as well. But then you can have some innovation coming out of that basic research from the patient itself, and that could be translated into some kind of treatment options would be wonderful. So that was the thing that I always wanted to have. So this cartoon just depicts a kind of reverse translation and translational outcome from bed to bedside to bedside to the bedside. And in this, in initial part of my beginning, I always called patients were like my patient passport. Like in the passport, we have the name, date of birth, and place of birth, all the details. Similarly, patients were having a kind of various clinical pathological features, various grades and histotypes, molecular profiling data, and disease progression stage. So this basically defined the patient passport. And why do we need to study this in the lab? We wanted to study if we can have a stages, early spread, nodal involvement, lymphovascular invasion, metastasis, grades. In grades, we look for a well-differentiated, moderately differentiated, or poorly differentiated. And then you can have a different histotypes, then progression of disease, prediction of patient's response to any chemotherapy, any kind of treatment, rapid detection of the tumor response, endpoints of the disease monitoring, and the multi-step nature of cancer patients treatment. Basically, all this was actually directed towards another platform that we actually opened up, was the biomarker platform. The biomarker is nothing but something from the fluid or from the tissue that we can detect or we can pull out from the cancer patient tissues or cancer patient blood, so that we can define and we use that as indicator for any of such various pathological features or grades or research types or disease progression that could be helpful. So there, the biomarkers are important because it can have a non-invasive screening for early stages. We can have a detection and localization. We can have a disease classification and prognosis as well. And response to therapy, as I said, and screening for the disease recurrence. Now, all these, actually, this is very, very old slide of mine and very close to my heart. And believe me that all this that you see from non-invasive screening to the bottom, every 3 words have been translated into one complete project. So we have 4 projects, 5 projects on where we are looking for detection, localization, disease classification and prognosis, responses to the therapy, and then screening for disease recurrence. So this is quite helpful that when I started. Then we do have patient tissue bank with us. It's not like defined tissue banks that every institute is trying to make, but we have a good amount of tissues from various indications, various types of solid tumors. And we do have patient cell line derived that we established in our lab to work, because I guess some of the cell lines which are actually sold in the market or available with the ATCC, they have been gone through rigorous all kinds of treatments. So sometimes they can give you false, profiling as far as your studies are concerned. So patient passport is very important. So coming to the lab, now this is reverse translation going from bedside to the bench side. Coming from the bedside, the things were coming back in the bench. And then we found out a molecule which actually Dr. Rita just mentioned about the SPAG9. This is a way back in 1998 that we discovered. And at that time, this was pulled out from the subtraction hybridization library from the cancer tissues and the cDNA library from the testes. And that's how the name actually designated as sperm-associated antigen 9. Although this is a molecule which is actually a scaffolding protein involved in the MAP kinase pathway, which actually supports the cellular proliferation of the cells via JNK CJUN involvement. So SPAG9, basically we looked its expression in various types of solid tumors. For example, cervical carcinoma, ovarian carcinoma, breast carcinoma. These three top ones are the leaders. You know, they are the ones who actually are most abundant in India and elsewhere in the world. And then prostate cancer is another one. Then we have bladder carcinoma, then renal cell carcinoma is a kidney, salivary gland, thyroid, and colorectal carcinoma. So some of the representative pictorial that I kept here. So we validated the presence of SPAG9 protein, which was actually facilitating the cellular growth, which we did. I'm not going to show the whole data, but yes, it was supporting the cellular proliferation in all the cells that we used from a variety of indications of the tumors. And if you can kill the presence of SPAG9 within the cells, I mean, the cell death was happening. So it's just a brief. And also in the animal model, we could show that if you can kill the SPAG9 in the tumor, it can actually reduce the tumor size. And similarly, if you kill by gene silencing, the migration actually reduces. This is just a pictorial to show how the cells are migrating. This is through the gel. And then you put a matrix, you can show for a cell invasion. So primarily, SPAG9 was involved in cellular proliferation, migration of the cancer cells, and invasion again. And that's how it correlates. Now, when we talk about this migration, invasion, and cellular proliferation, you have EMT coming in. So EMT actually makes the cells to migrate, and then from EMT epithelial mesenchymal transition to the mesenchymal epithelial transition. That's how the niches actually go all over the body, and that's how they develop the METs, the metastasis. In the meantime, we also started 16 years back, 17 years back, the phase 1 clinical trial in Cancer Institute, Adyar, where we wanted to use the tumor lysate expressing SPAG9 protein. And it was 14 patients at that time. And I'm just showing you one of the patients representative X-ray, chest X-ray. This patient was actually given DCs along with the chemotherapy, which is actually a line of course of treatment. And what you see here is she had a small METs, which we can see in the lungs. It grew in another year's time, and it grew another year's time, so big. And then with the treatment, the interventions that we had along with the chemotherapy, this was totally cleared off. And this patient is still alive till today. So that gave, kickstart to the whole medical oncologist team in Cancer Institute Adyar to amplify this whole program into a number of bigger patient sample size, and then to use recombinant SPAG9 protein. And we actually outsourced the SPAG9 protein to the Bicons engine. And at the same time, we also have now trademarked this protein, which is SPAG9. SPAG9 is a molecule, SPAG9 of NII. So primarily, this is involved in dendritic cell-based vaccine, where we isolate peripheral blood mononucleotides from the cancer patients itself, can culture in presence of cytokines and pulse with human SPAG9 and a DC culture for 3 days maturation, and then you harvest the mature DCs, and then the preparation of the DCs injection. So we have a 10 dose immunization schedule, having each dose having around 1 million cells. So they come on 0 day, 7 day, 14 day, and then 28 day, and then later on a monthly cycle. So it's a kind of a 6 to 9 month schedule where they actually get this intradermal injection. Not that it has any side effects, except that the side effect is at the site of injection there is some sort of irritation or inflammation. This is just the essays to show how immature is getting into mature, and then we put it back. So we want to expand this to all the hospitals where we have a basic facilities of apheresis. We can isolate PBMCs. We have also duplicated now this facility in Jaipur, in Mahatma Gandhi Hospital, where we are expanding this I just talked about, where we are expanding now with the ovarian cancer trials in 75 patients. Now this is actually increased. We have, because of the COVID, we lost 1.5 years, 2 years time. So we have asked to drop the tumor lysate arm and now accept tumor lysate arm. We had to increase the number of patients. So it will be 120 patients. Otherwise, we were trying to compare if we can compare with the tumor lysate. And at the same time, we also have now been approved for breast cancer trial. This is a effect of metronomy chemotherapy effect on dendritic cell-based vaccine for triple negative breast cancer patients. Again, DBT is going to fund this for another 5 years, and the money is going to come probably in another 2 to 3 months. So that's where we stand. We are waiting for cervical cancer trials codes to be broken in another 2 to 3 times. However, intermediate 3-year studies every 3 months, they're all CT scans, PET scans, CT PET were done for looking for their remission or response as a readout endpoints, and we have very encouraging results. That is how we jumped onto ovarian cancer patients, this medical oncologist team, and again jumped onto breast cancer trials. I'll skip that. This have duplicated there. That is what I was talking about. And we have also got now around 8 monoclonal antibodies generated against SPAG9, because this is surface localized in the tumor cells. And we are in the pipeline where we are trying to do the targeted immunotherapy or localizing the disease or the tumor itself. So we are conjugating this monoclonal antibody with fluorophore or with the warhead with the chemotherapeutic agent, so that less toxic drug can be delivered to the patient in such kind of cases where we need to give the chemotherapy. I think I'm going to end here by saying that I started with my acknowledgement with Nirmala. And Nirmala again got Women Scientist Award for 2019 for Pharmaceuticals Producers of India on June 1st, 2019 from our health minister now, Mr. Mansukh El Mandviya. And I thank you for your kind attention. And we just happened to publish this Cancer Cell International, where we're trying to show from 4 to 5 patients that we could actually get some samples with their consent, where we have done all immunological parameters of looking at CD4, CD8, and activated NK cells. I thank all the organizers, and I thank all the team who has been involved, and my apologies in not joining this morning because of the commitment I could not avoid. I thank you all, and congratulations to each and everyone who has got award. And big congratulations to Professor, my boss, my supervisor, PhD supervisor, being conferred with the Padma Shri Award. Thank you very much.
A very nice talk indeed, and we know all the efforts Dr. Nirmala has put in in your lab. I have several times met her, very hardworking and very, very elegant lady. And your work is very, very interesting. I would ask if anyone has any question. I think we can have one question.
Well, I think, thank you, Dr. Rita, if I'm allowed. I don't have any question. Thank you so much, Dr. Shuri, for sharing such good, wonderful data, and really best wishes what you're looking forward with the upcoming DBT grant, and it's laudable. Thank you very much for sharing the data.
Congratulations, Dr. Taru, taking this responsibility of Department of Biotechnology Institute. Glad to see you, and
Thank
congratulations.
you. In your boat now. Thank you very much.
Welcome to the gang.
Hearty congratulations, Anilji. Whenever
Thank
we
you.
listen to you, I tell you, we are full of the pride that we have a great scientist who has done a really innovative research, and that also from India. God bless you, and all the patients will send best wishes.
So sweet, so sweet. You're close to my heart. Actually, this is where my heart goes. I want to be in the hospital. I want to be, you believe me, I see those patients, my goosebumps, my hairs will stand. And I just think I want to see them all alive. And cross your fingers, all those cervical cancer patients are doing very well. I didn't show the pictures of, I happened to give a talk 2-3 weeks back here in Nasi chapter. Professor Loya had invited me, where I had shown the patients actually photographs at that time. But thank you very much. You're very close to my heart.
I wish to join others in complimenting Professor Suri for his wonderful research outcome. And all of us look forward for translational implications and utility and outcome of your research for benefit of the human race. And we hope that your all ongoing clinical trials will go fast, complete phase 3 clinical trials at the earliest possible, and you get approval from all drug regulatory agencies at the earliest possible, and your research is turned into products, and it comes in the form of vaccines, and people who are suffering with different types, cervical cancer, the breast cancer, ovarian cancer, they are getting big relief out of your research outcome. Thank you so much, and compliments and congratulations for your work.
Thank you, Professor Loya. But in cancer trials, phase 3 is not required. This is efficacy, and then after that, definitely it will spread out to the multi-institutional, multi-hospital. Yeah.
Very great.
My boss is there. I see my boss.
I wish you year 2 to be the eureka for you.
So sweet. So sweet. Thank you. You're very close to my heart, sir. You've been. I said, and I told you, I spoke with you that night that you have taught. This is all your. You see your fruit. And I remember, and I quote. I should have quoted. Always when I was your PhD student, you always had one sentence that actually has been implanted in my head: never ever make soap bubbles. So I'm not making soap bubbles, sir. And I'm so happy that a third party, clinicians, medical oncologists actually are validating what we have done. It's not in the lab that we are doing. So it's so self-satisfying.
This year, 2 should be that way when we'll go forward. It will be the day when you will put in the market. Things will be in the market.
Yes, sir. Yes, sir.
So Pradeep did not allow us to talk to Dr. Sharma, but thank you very much, Dr. Sharma, for the talk which you started with Abhimanyu's story. And I like the discussion which you did with Dr. Malini. Sorry for chairs. I just wanted to take 1 minute. Thank you very much. An intruder, of course. Thank you.
Go ahead.
I also want to intrude and want everybody to join in congratulating Dr. Taru Sharma becoming the NIAB director. So I think we should all congratulate him.
I think they did it. Thank you, Malini. Thank you so much. Thank you very much.
That's a big responsibility, actually, though. I bet, and I'm sure she will deliver.
Thank you.
And we feel pride. We take proud that she's ours. She's a member of ISSRF. So this is.
I thought you will tell me that you are very close to my heart, like you were telling to others. Anyway, thank you very much.
He will tell next time.
You are close to my. You are a member of IC. You are close to my heart. All members, all.
I think we are going to have so many celebrations. In the morning, I said Professor Madan has committed to celebrate his Padma Award. And now Dr. Taru has also committed. She said that after becoming director of such a prestigious institution of the country, so she has also committed. And we look forward to Dr. Suri also. By the next year, he comes out with something which would make us to be proud and a occasion of great celebration.
We need an offline celebration, sir. Offline.
Right now, we can accept all the Swiggy vouchers if that's the way you want it.
Professor Suri, this is Suranda. I have a couple of questions which I will email, but you are focusing too much on heart. दिल तो बच्चा है जी, तो वो कहीं भाग जाता है इधर उधर.
Ah, it's so sweet. You are very close to my heart.
So, Dr. Sharma, this is
ये एक्चुअली क्या है कि आपके तार जुड़े. आप जयपुर में जयपुर की शाम जब यहाँ डॉक्टर प्रोफेसर लोहिया ने इवनिंग गेट टुगेदर किया था. But you know, your work, I mean, I'm seeing, I wish, we had more time for discussion at kind of profile.
So. I joined
you had, started with the infection, you went into immunology, you went into transcriptome, you are silencing gene, you are using monoclonal antibody to silence. You're using SCG now. I mean, come on. And this work will go through nature, I bet, believe me. And that will be another celebration, Professor Lohia. And we have a chief editor of AGR.
T-Rex to HCG correlation. Wonderful, Dr. Sharma, really.
I'm just saying, I mean, there is no delay, no time. I mean, this kind of lecture should have, it is something so refreshing. I mean,
both of you with the monoclones, both of you with maps, Dr. Surya.
Actually, we have now a last presentation by Professor Hemant Malhotra sir. And you all agree.
We'll keep quiet, Professor Loya. Now we understood.
Yeah. Boss coming.
So, Sarita, we go forward.
Yeah, please.
Okay, so can Dr. Sudi
please introduce him.
I will.
Yeah, and share the screen, and can you please?
Yes, sir. I'm connected, and as soon as you permit me to share my screen, I'll do so, sir.
Just a second.
Meanwhile, Madan sir, we need to come to which place for such a big celebration?
Welcome.
Professor Loya, offline कर दीजिए, अगली बार मत online करिए, please. Dr. Iss Sharma, don't keep it offline. Don't keep it online.
Dr. Surya, please stop sharing.
I am.
No, your screen is still visible. Okay. So the next speaker is Professor Dr. Hemant Malhotra. So he is going to talk on reproductive and fertility issues in young cancer patients. His biodata is there. I'm not going to read it out because of lack of time. So Professor Hemant, can you get going?
Thank you very much, sir. A pleasure and honor to be here. And my apologies for juggling the program a little bit. I had another talk, just finished that. So if I sound a little hoarse, please pardon that. So I'm going to talk about reproductive and fertility issues in the young cancer patient. And I know that we have a very, what should I say, a very varied and vast audience, but I hope I will be able to convey some knowledge and some message to them. So my greetings to you from the Shri Ram Cancer Center at the Mahatma Gandhi Medical College Hospital in Jaipur. And I have no personal disclosures related to this presentation. So this is what I intend to do, like a couple of slides on the introduction and background. Talk about the gonadal toxicities of chemotherapy and radiotherapy, the two main modalities used in cancer treatment. Some basics of fertility preservation, which I'll be the first to admit, I probably know a lot less than some of the listeners. Talk about a fertility preservation procedures in the males and in the females, what specific things we do in the gynae cancers and the breast cancers for fertility preservation, and then some take-home messages where I'm sure that I'll finish between 15 and 30 minutes from now. So ladies and gentlemen, more and more young patients are being diagnosed with cancer, and with the advent of new drugs and other advances in treatment of cancers, more and more cancer patients are surviving. Issues related to fertility and pregnancy are now becoming common and becoming very important. One study estimated that fertility and pregnancy related issues are one of the top 3 priorities for young women with cancer. So this is right next to the anxiety which they have about survival. There have been several major advances in the techniques and applications of fertility preservation. This again, a lot of you will know more than I do. And now today it has become mandatory to talk to the young cancer patient about fertility related issues. It has become mandatory to document this in the patient's file. Otherwise you are liable to land up into significant legal trouble several years down the line. So this was a survey which we did with a colleague of mine from Tata Hospital, physician knowledge and attitude towards fertility preservation in cancer patients. And with regards to fertility guidelines, almost 2/3 of the physicians surveyed had suboptimal knowledge. And with regards to fertility preservation strategies, almost half of the physicians surveyed did not have adequate knowledge. Now, indications for fertility preservation are a lot. And today we are going to talk only about issues related to oncofertility, how we can reduce gonadal toxicity of cancer and non-cancer treatments, how we can reduce gonadal toxicity of radiation therapy. So these are the list of agents, chemotherapeutic agents, which produce gonadal toxicity. And you can see that the patients which have high risk of producing gonadal toxicity are the alkylating agents, which include busulfan, chlorambucil. Cyclophosphamide is a very, very commonly used agent in most chemotherapy regimes. The platinum agents and the taxanes, which are again very frequently used, as are the anthracyclines, they have intermediate risk with regards to gonadal toxicity. And antimetabolites like vincristine, methotrexate, mitomycin, 6-mercaptopurine, they have low risk. There is a big list of drugs, particularly the monoclonal antibodies, in which we are not sure what is the risk of gonadal toxicities with these agents. Now, how does radiotherapy and chemotherapy affect female fertility? The sterilizing dose for the ovaries is actually different for different ages. And you can see very clearly here that the prepubertal ovary is a lot more resistant to the sterilizing effect of chemotherapy and radiotherapy as compared to the mature ovary. And what does radiotherapy do? Again, the toxic effects of radiotherapy are dependent on age, drug, and dose. If you talk about the uterus, it produces fibrosis of the uterine muscles. It reduces the elasticity. It produces vascular, microvascular damage. it produces endometrial atrophy. And if there is a prolonged exposure, and after 20 years of the exposure, it leads to decrease in uterine size. And all these lead to decreased fertilization, decreased implantation, and increased adverse obstetric outcomes. What about ovaries? Radiotherapy damages the follicles at all stages. It produces vascular damage, and this leads to infertility, premature menopause, amenorrhea, transient or permanent. What about chemotherapy? Again, the effects are age, drug, and dose dependent. It leads to apoptosis of growing follicles, primordial follicle activation, and subsequent apoptosis of burnout effect, vascular damage, and oocyte disintegration and increased mutations in the oocytes. And this all leads to increased destruction of the growing follicles. What about males? How does radiotherapy and chemotherapy affect male fertility? It damages the spermatogonial stem cells, and this can lead to permanent azoospermia. It leads to damage of the differentiating germ cells, and this leads to transient azoospermia. Damage to the supporting cells, Sertoli cells, can lead to permanent azoospermia or transient azoospermia, and damage to the Leydig cells, particularly after high dose radiotherapy. It can lead to testosterone deficiency. So when do you start thinking of referring a patient for fertility preservation? Once the oncological diagnosis is confirmed, once we decide that this is a patient who can be cured of, he or she can be cured of a cancer, and a treatment plan is defined. And then you assess this treatment plan. How likely is this treatment plan? How likely is it to induce infertility? If there is a significant chance of the treatment plan inducing infertility, and there is a good chance that the patient will survive several years, then fertility counseling is mandatory. The patient's interest and the family's interest in fertility preservation is very carefully evaluated. And then after all these things have been ensured at the oncology level, the patient should and is referred to the reproductive medicine or the family or the fertility preservation expert. And these are the patient evaluations which should be done before offering fertility preservation. There should be a strong desire. The oncological outcomes, the diagnosis stage, survival rate should be in place. Risk assessment as to disease, chemotherapeutic or radiotherapeutic protocol, age of the patient needs to be kept in mind. The patient's medical status and general health has to be kept in mind. Time available for conducting fertility procedures to be kept in mind. Suppose you have a patient with acute myeloid leukemia in which life-saving chemotherapy has to be started within days, then this goes into the background. On the other hand, if you have a patient of Hodgkin's lymphoma, which is a slowly growing malignancy, then you can have a 2 or 3 weeks delay before initiation of the oncology treatment to conduct the fertility preservation procedures. Age of the patient, pubertal status is important. Ovarian reserves is ultimately important for the success of the fertility preservation procedure or the oocyte storage. For cancers in which there is risk of ovarian involvement, this again is an important factor. Gynecological assessment for any other adverse conditions which could be counterproductive to pregnancy have to be ruled out. And of course, the partner has to be evaluated. There are certain barriers to fertility preservation world over and in our country. There is lack of referral pathways. So there is lack of collaboration between cancer and fertility doctors. There is lack of adequate trained staff to deliver these services. There is the huge and quite significant cost of these procedures. There is a lack of social support for decision making. And there is this big perception in the patient's mind, and particularly in the patient's family's mind, that cancer is a terminal, non-curable illness, and the need for immediate treatment is much more overriding than the then the thought process and the procedures for fertility preservation, institutional policies, processes, and resources are lacking in most institutions. There is also a lack of consensus about the best way to deliver these informations to the patient. So what are the fertility preservation options in the males? For the post-pubertal male, these are the established options. The simplest is semen cryopreservation, and testicular tissue cryopreservation is also can be thought of if semen sample is unavailable. Experimental techniques include whole testes cryopreservation. For the prepubertal testes, nothing is established as yet, but testicular tissue cryopreservation, germ cell suspension, all these are experimental protocols. For the fertility preservation techniques in females, you have a whole bunch of them. Again, if the patient is prepubertal, you could think of gonadal shielding when you're giving radiotherapy. You could think of ovarian tissue cryopreservation. For the pubertal patient and the pubertal ovaries, fertility protection in terms of gonadal shielding, ovarian transposition, and GnRH agonists, which protect the ovaries from the effect of chemotherapy, can be thought of. For fertility preservation, if there is sufficient time before starting specific anti-cancer treatment and sperms are available, embryo cryopreservation can be thought of. If sperms are not available, oocyte cryopreservation can be thought of. And there are certain experimental techniques which are also being tried and tested. These are the various risks and benefits of the different fertility preservation techniques in the female cancer patient. And I think this is a matter of the experts, but all the techniques which we have, they have some pluses and some minuses, and which need to be considered in choosing any one of these techniques. Some criteria have been suggested for ovarian tissue banking in the world and in India. The age has to be less than 40. Ovarian functions have to be confirmed. The risk of treatment-induced loss of ovarian function and significant reduction in ovarian reserve have to be considered. Viral serology have to be negative. The patient should not have disseminated disease. There should be no contraindication to surgery or anesthesia. And of course, you need the informed consent. If the patient is less than 18, then you need consent of the parent guardian. Then a realistic assessment of the long-term survival should be known. And in the case of hormone-sensitive malignancies, the oocyte retrievals are contraindicated, or time is not enough, and we have to start chemotherapy urgently, as in cases of acute myeloid leukemia and rapidly progressing cancers. Fertility-sparing surgeries in gynecological malignancies can be thought of for the patient desirous of pregnancy, the young patients, the patients who have very early tumors, and the patients who have need for adjuvant treatment. Very early cervical cancers, can be treated with less aggressive surgery like conization or trachelectomy or radical trachelectomy. Excuse me. Similarly, endometrial cancers in the very early stage, they can be managed with sometimes medical therapy and minimal surgical resection. Ovarian cancers in the very early stage, germ cell tumors in stage 1, you could think of adjuvant chemotherapy or unilateral salpingo-oophorectomy, and not the panhysterectomy, which is usually advised and done. For vulval and vaginal cancers, wide excision in the early cancers can be thought of. Fallopian tube cancers, fertility preservation is usually not advocated. What about breast cancer? These are some of the common chemotherapeutic regimes which we use. If you use a regime like CMF or CEF or TSC, primarily because of the C, the alkylating agent over there. If you use 6 cycles, particularly for women around 40 years of age, then the chance of producing premature ovarian insufficiency are more than 80%. And the determinants of this premature ovarian insufficiency are age, the chemo regime which you use, and the cumulative chemotherapy dose administered. Excuse me. Some fertility preservation options which we use in breast cancer include, of course, the whole counseling gamut, but we have some drugs also, GnRH agonists, which can protect the ovary when the chemotherapy is being given. And this is the ovarian suppression which the GnRH analogs produce, and which protects the ovary from the effect of chemotherapy. And this benefit has been proven. This was a randomized trial. The premature ovarian insufficiency rates in patients who received GnRH agonist were 14%, and this was almost half of what was seen in the control group, which did not receive this agent. Similarly, pregnancy rates were also double in the group which received the GnRH agonist as compared to the control group. And this was statistically significant. And this medical intervention can be considered in young women of breast cancer receiving chemotherapy. And there were no adverse outcomes of the use of GnRH agonists. And you can see that the survival, disease-free survival, and the overall survival in both the groups of patients who were estrogen receptor positive and estrogen receptor positive, whether they received the hormone ovarian protection, oncological outcomes were not affected. This is usually thought of in patients who desire ovarian function preservation and in patients who desire fertility preservation. There have been some very excellent guidelines presented and published by the Fertility Preservation Society of India, and I've taken quite a lot of my material from these guidelines. And for those of you who are interested, you refer to look at this document and update your knowledge. So if I were to summarize now, this is my last slide. There has been an increase in number of cancer survivors with good prognosis due to progress in the diagnosis and treatment of cancers. Oncologists should now discuss the risk of infertility resulting from cancer treatment with patients at their earliest opportunity and provide initial information regarding availability of fertility preservation. Patients with a good oncological prognosis at risk of infertility and interested in preservation of fertility should be referred to the appropriate expert. Details of the procedure, including costs, risks, chances of success, should be explained in details. Genetic and social counseling should accompany all this, and limiting factors, particularly in our country, should be addressed. And these primarily are lack of awareness, costs, lack of insurance coverage, and experimental nature of some of these procedures. So, ladies and gentlemen, an integrated approach between the patient, the patient's family, the treating oncologists, and the reproductive medicine specialists need to be in place for good outcomes. With that, I'd like to thank you very much for this opportunity, and I hope you enjoyed the talk as much as I enjoyed preparing it. Thank you very much.
Thank you, Professor Malhotra, for briefing us about the possibilities available for preserving fertility in cancer patients. Professor Hemant would be happy to take one question in case somebody has a question.
I just want to.
Yeah, Anil, go ahead.
Yes, Dr. Suri.
I just want to add here. Dr. Hemant is the one actually, as a part from Sri Ram Cancer Center, working in the kind of work that we are involved with in ovarian cancer trials. But you saw, going through the presentation, let me share with rest of you all. Under his belly, already he's doing 3 different trials with more than 30 centers within this country. And not only in this country, those trials are involved pan globe, pan world. And I think we are lucky to have a speaker who had actually given such kind of stimulating talk. My question, Dr. Hemant, was the radiotherapy that you said in the slide, in young women, it is less effective as compared to old. What is the basically cause for that? Is there any?
So Dr. Suri, the prepubertal ovary and the prepubertal testes, they are resistant or relatively resistant to the effects of both radiotherapy and chemotherapy. This is because being prepubertal, they don't have the mature germ cells to damage. If it is pubertal and postpubertal, the germ cells are already there, and they can be damaged by your cancer-directed treatment. If they are prepubertal, then you don't have those mature germ cells to damage and to destroy. So that's why they are more radio and chemo resistant, the prepubertal ovaries and the testes.
Okay. Yes, Dr. Rita, would you please introduce the next speaker?
Okay. Meanwhile, I can ask one small question to Dr. Malhotra, which is the best way to preserve female ovarian, germ cells, whether it is follicles which are being practiced, or is it the whole ovary?
So, madam, I am, working and practicing oncologist. I am not a reproductive medicine expert. I'm not a fertility expert. So I don't think I'm qualified to answer that. But if you do look at the slides, there are some pros and cons of all the procedures and pros and cons of both the procedures which you're wanting to know. And they have to be individualized and decided for an individual patient, keeping into factor the patient's age, the patient's ability to, the amount of expense they can make, the type of therapy which they have received. So all these factors would kind of then tell you as to which is the ideal procedure for an individual patient.
Thank you very much, Dr. Malhotra. I think it was a very, very informative and very interesting lecture. I got a lot of, information on these various chemotherapies. So now we welcome Dr. Alpna Sharma, whom I know for several years. And she's a very, very eminent professor, and she has been working on these aspects of cervical cancer and has many laurels. You can see she's a fellow of, she has PAMS and PAPS and so many other excellence research awards. I welcome her for delivering her lecture.
Ma'am, you have to unmute yourself.
Yes.
Yeah, thank you, ma'am.
My slides are not visible so far, or it's there?
No, ma'am. Not yet.
Please share your screen and.
Yes. Now, can you see?
Yeah, you can put the presentation mode, please.
Yes, yes.
Yeah, perfect.
So, good evening, everybody. I know today this is the last lecture. Everybody must be tired. So, first of all, I would like to thank the organizers, Professor Lohia, Professor Iss Sharma, and other members of the organization, and the chairperson for the nice introduction. So, it's showing very big, my screen. I'm not able to see this side. So, on the right side, I cannot see my slide. So, organizers, can you do something for this?
I think it's on your own screen only. Please just, you'll have to move things.
I'm moving.
Can you go to the right top? There is a thing called view.
Right, more.
Oh, there is something called view on the right side top of your screen.
Oh, I'm not able to see. Anyway.
So there is a cursor, you know, in between your picture and the slide. So just move that cursor to right. So your most of it will move to the right and slide will open up.
So I do not know, it is not happening. Anyway.
No, Dr. Sharma, in the center, there is a view option, and then you open it, you can reduce your zoom option. So I think you will
because we can see it very well. We can clearly see your slide.
You can see my slides, eh? Okay.
Yeah, we can see.
We can see your slides.
So it is not going back. So I will start again. Is it fine? Can you see? Okay. So the title of my talk is Diagnostic Potential of Circulatory HMGB1 in the Liquid Biopsy of Indian Population of Cervical Cancer. So already discussed quite a lot about its incidence and the causes. Cervical cancer is the commonest cancer in Indian females and second amongst worldwide. About 15% on human cancer can be attributed to viral infection, and viruses are second to tobacco as a risk factor for cancer. Human papilloma virus is the main cause of cervical cancer. We know we have high risk and low risk HPVs. 16 and 18 are high risk HPVs which are involved in the cervical cancer. It occurs most often in women over age of 30. So the major cause of cervical cancer is HPV. We know we have in the cell cycle G2 checkpoint and G1 checkpoint. So when HPV is not present in normal condition, during the G2 checkpoint, a functional P53 protein detects a DNA mutation, and those cervix cells with mutated DNA do not divide, and hence the cervix is healthy. But if HPV infection is there, then this HPV, it produces the P53 inhibitors, and P53 we all know it's a tumor suppressor protein, and if P53 inhibitor is there, then this protein is deactivated, and hence with P53 deactivated, cervix cells with mutated DNA successfully divide and can cause cancer. So if we see the diagnostic tests for cervical cancer, there are many, like bimanual pelvic examination, which is to some extent inaccurate, Pap test, which has low sensitivity and lot of false negativity and false positivity, then colposcopy, biopsy, insufficient tissue volume, infection or bleeding, CT scan which cannot discriminate between normal cervix and stroma and cancerous tissue, and exposure to the hazardous radiation. Then PET scan, which actually early stages poorly diagnosed. It is very expensive, and we know that it has a short decay of radioactive substance, which cause patient to be immediately scanned. So why we require liquid biopsy? When we compare tissue biopsy versus liquid biopsy, the tissue biopsy is a localized sampling bias, time intensive, it is invasive, painful, and difficult to obtain. Whereas if we see liquid biopsy, it has comprehensive profiling, immediate sampling can be done, it is non-invasive, less painful, and easily obtained. And again, we all know that these cancer cells, there are hallmarks of cancer, where sustaining proliferative signaling, evading growth suppressor like insulin growth factors and others, avoiding immune destruction, like we have CD8 T cells which are cytotoxic in nature, which can kill tumor cells, but either their number is less or they are non-functional, enabling replicative immortality, that is telomerase activity, activating invasion and metastasis, like migration is there, then it can go to the different organs and can cause metastasis, inducing angiogenesis. It requires the tumor cells, they require good supply of blood, and then resisting cell death, deregulating cellular energetics like oxidative stress, mitochondrial membrane potential, and many other phenomena. So we have touched upon earlier two other factors. So initially we have done some cytokine way back in 2007 cytokine profiling in Indian women with cervical intraepithelial neoplasia and cancer cervix. And then we have also seen these cytokines Th1 and Th2 in patients with advanced cervical cancer undergoing neoadjuvant chemoradiation. And their correlation with treatment response was seen. Some of 1 or 2 slides of results I have put it here, which says that yes, Th1 cytokines are lowered, whereas Th2 cytokines IL-4 and IL-10 are increased in SIN as well as in the cancer patients. Then we correlated this data with grade of SIN and the clinical stages of cancer cervix. And there we have found that except interferon gamma, other 3 are correlated with the grade of SIN, and all 4 are correlated with the clinical stages of the cervical cancer. So this result studies, I put very few results here. If pronounced shift from type 1 to type 2 cytokine production is associated with more severe disease. Then telomerase activity was also seen in SIN and the cervical cancer, which has also correlated with the different grades of the SIN, as well as the early cancer has lower telomerase activity and the advanced cancer has higher telomerase activity. And there we have also correlated with the HPV data, and the ROC curve showed that yes, the area under the curve was not very good, but yes, still the telomerase activation correlated with the grade of cervical lesions and HPV status and clinical staging, suggesting it as a possible target for detection of cancer cervix, although telomerase activity increases in other cancers too. Then molecular circulatory expression of insulin growth factors and in Indian females with advanced cervical cancer, modulatory effects of chemoradiation and angiogenesis and laminin in cervical cancer. So some of the data for IGF-1 and IGF-2 is here, where patients have shown higher values, and IGF-BP3, which is a negative regulator, has decreased levels in these patients, which has further corroborated with the mRNA expression as well as with the Western blot. So IGF system might possess a prognostic ability in cervical cancer, and then this angiogenic factor and laminin, it has also correlated with the pre-treatment and post-treatment data, where the levels have decreased after the post-treatment, and we have compared with the relapse-free survival as the disease-free survival in these patients. So today my talk is on autophagy, that is the resisting cell death in these patients. So what is autophagy? Autophagy is a Greek word where auto means self and phagin means to eat. It is a pathway in which cells degrade and recycle their own component. The mechanism where some of the inhibitory factors are there, some of the stimulatory factors are there, PI3 kinase and some AMP kinase are the activators, where mTOR is the negative regulator. So initially the double membrane, it engulfs the cytoplasmic contents and then form a vesicle, closed vesicle, which is known as autophagosome. When this autophagosome, it fuses with the lysosome, it forms the autolysosome, where we all know that these lysosomes, they have acid hydrolases and other enzymes which can cleave this cellular component, and then it can come out with the permeases and can have these small molecules which can be used for the energy purposes in the body. So again, in 2016, Japanese cell biologist Oshiniro Sumi won the Nobel Prize in Physiology and Medicine for his research on how cells recycle and renew their content, and the process called autophagy. So high mobility group BOX1 is a nuclear protein. So in normal cells, its expression is very low, while in some of the cancer, the expression has shown higher. HMGB1, it has many biological functions. It acts as an extracellular signaling molecule during migration, inflammation, cell differentiation, metastasis, promotes cell autophagy, and induces death. One of the receptors of HMGB1 is a RAGE, which is a receptor of advanced glycation end products, which is a signal transduction receptor. Some studies say that HMGB1 and P53 regulates apoptosis and autophagy in colon cancer cells after treatment with DNA damaging anti-cancer drugs. DNA damage promotes interaction between HMGB1 and P53 in the nucleus and cytoplasm, and loss of P53 increases cytosolic HMGB1. Although it's a nuclear protein, but if there is a loss of P53, it increases cytosolic HMGB1, leading increased binding to a molecule which is known as backlin 1, thereby promoting autophagy and decreases apoptosis. In contrast, loss of HMGB1 increases cytosolic and HMGB1 and apoptosis and decreases autophagy. So apart from HMGB1 and P53, another important hallmark of autophagy is the nucleoporin 62, that is P62, which recognizes toxic cellular waste, and it is scavenged by autophagy. So we pose a question, is there any alteration in autophagy related molecules in cervical cancer? Do they have some potential as non-invasive diagnostic marker in this cancer? So in this study, the expression of HMGB1 and its associated molecule RAGE, P53 and P62 were investigated. Further, the diagnostic significance of circulating HMGB1 is evaluated. So in this, we have recruited 20 patients of age this, and two types of controls we have included. One for the healthy sample control for the blood, and second control for the blood as well as for the tissues. And these C2 has the total hysterectomies for benign indications such as fibroid and prolapse, where tissue and blood both were collected. So first of all, we have seen the HMGB1 concentration in the circulation in patient in both the control groups, where we have found that yes, in patient, the HMGB1 concentration was significantly higher than both the controls. Then we have seen the relative mRNA expression of HMGB1 and related molecules like RAGE, which is its receptor, P53, so tumor suppressor, and P62, that's the nucleoprotein. We have found that yes, the levels of mRNA expression of HMGB1 was higher in the patient, same its receptor RAGE, but P53 and P62 mRNA expression was lower as compared to control. So initial experiment, when we have seen the Western blot analysis of this HMGB1, we could not find any band. We were surprised why it is not there. Then we have gone back. Yes, when P53 is low, then naturally cytoplasmic fraction of HMGB1 might be higher. So we did the Western blotting both in nuclear fraction as well as in the cytoplasmic fraction. So as compared to non-tumor tissue, the nuclear fraction has shown lower levels of HMGB1, where cytoplasmic fraction has shown higher expression of HMGB1. And this is just the relative fold intensity, which says that yes, HMGB1 in cytoplasm was 3.5 times fold higher than the nuclear fraction. Then we have seen the Western blot analysis of other molecules like RAGE, P53 and P62, where we have seen in tumor tissue, the RAGE was higher as compared to non-tumor tissue, and P53 was lower as well as P62 was lower than this. This is again the relative fold intensity of the same bands. Then we have correlated, the SPMR correlation analysis was done for HMGB1 levels in serum and the mRNA level as well as the Western blot, the protein expression. Here we have seen that yes, it is significantly correlated, but at protein expression level, it was not correlated. Maybe the number of samples was just 10, so we could not find significance here. Then we have plotted the ROC curve for the circulatory levels of HMGB1, which has shown area under the curve 0.8625 and sensitivity and specificity 80 and 85 respectively, which is reasonable, not very good, but yes, still better. So then we thought that we should confirm by in vitro study also of this HMGB1 expression. So we did the siRNA approach was used. So first we have download the siRNA approach was used, and this first picture is just the bright field image, and second B is the GFP image showing transfection efficiency. And then we have seen this approach. We have used the siRNA targeting different regions of HMGB1 transcript, which was used and procured from the Sigma, which is the cocktail of sequences synthesized commercially. So here we have seen that yes, the HMGB1 expression was lowered, and this 50 nanomole have shown good efficiency, and this has approximately from 1 to 0.4% decrease in the expression of this silencing. Then after this, we have seen the expression of the molecules like RAGE, P53 and P62. And there we have found that in mock as compared to mock, the test has shown HMGB1 level low because there we have silenced this. And then RAGE has shown lower level. P53 has increased and P62 also increased, which we could not appreciate here. But yes, when we have done the relative intensity, we have found that yes, the HMGB1 level significantly lower, same the RAGE, but P53 and P62, they were elevated. So with this, we could say, yes, HMGB1 is important in this disease. We have also done some, I am going to summarize here, but I want to say that, yes, when we have gone to the literature, we have seen that in endometrial cancer, HMGB1 level was not increased circulatory levels. But in ovarian cancer, it was higher, but not as higher as in the cancer cervix. So here I would like to summarize the circulatory levels of HMGB1 were significantly higher in patient as compared to controls. The molecular expression for HMGB1 and RAGE significantly higher, while P53 and P62 significantly downregulated. Correlation analysis showed significant correlation between circulatory and tissue levels of HMGB1. ROC curve analysis showed better sensitivity and specificity for HMGB1 for non-invasive diagnosis of cancer cervix in liquid biopsy. And siRNA mediated silencing of HMGB1 leads to the alteration of other autophagy molecules, that is RAGE, P53 and P62, and in inhibition of the process of involved in the inhibition of the process of autophagy. So with this, I would like to acknowledge Professor Manoj Sharma for providing us the sample, and Professor Jb Sarma from AIIMS. And these are my students. The main work has been done on this Rehan and Nidhi and Ashu. Thank you very much for patient hearing. I would be happy to take questions if there are some.
So now we can take a question. Any question?
Alpana.
Yes, Dr. Suri, yes, please.
Wonderful work. Actually, though, because I know a couple of obs-gynae, associate professor, assistant professor are trying to approach for liquid biopsy,
yes.
Or diagnostic for small scale trials as well. I was wondering, when you are trying to correlate this biomarker, around 50% of the CS cervix or other tumors have P53 mutated. So how do you correlate, like down regulation of P53 and then it increases your molecule? Down regulation of P53 and your molecule has actually increased, right?
Yes, no, P53 is, yes, P53 is low. That's why HMGB1 is in higher, because they have inverse correlation.
Right. So in mutated, you will find very high, right?
Yes. Like in cancer cervix.
Right.
Patient.
So in 55, around,
very high.
So my question is, around 50 to 55% only the P53 are mutated, not every tumor.
Yes. So I'm telling, when HPV is there, that time P53 has major role. HPV positivity, this high risk HPV positive patient, which are Most of the time, 80% cancer service patients are positive with HPV. We know.
Yeah, no, so you're right. CS cervix is 95% actually is HPV positive,
Yes.
positive
So the
agent,
low risk or high
causing the
risk,
cancer.
yes.
Now, I don't know, we haven't actually analyzed with Dr. T. Raj Kumar, oncologist
Yes,
at Cancer
yes.
Institute.
From Cancer
So,
Institute.
like 1,200 patients that we had to recruit, we had to had inclusion criteria where the first criteria was HPV positivity should be there, second was stag-9. But we haven't seen P53 mutated patient, because on an average, he was saying around 50 to 55% patient will have P53 mutated.
So maybe,
Not all.
sir, this is a small patient's cohort, very small number. So maybe if we go for the higher number, we can get more positivity or less positivity. That will depend upon the number of patients. This is just 20 patients. We have initiated this study, yes.
Oh,
Very
that's good.
small
That's
study
good.
going on, sir.
Yeah, that's wonderful. That has a direct translation into the clinics.
Yes.
So anything
Still
would
going
be,
on, sir.
yeah, wonderful. I'm very glad.
Thank you. Thank you very much.
Thank you, Dr. Sharma. Very nice talk and very interesting data. So now we can, she was the last speaker of this session. So Dr. Pradeep, would you like to consolidate or shall I do it?
Yeah. Let us wrap up. So we had a very interesting session. It started with Dr. Sharma talking about learning disability at the fetus level. So autism can possibly happen even at the fetus level. And thereafter, Dr. Suri talked about his bench to bedside, back to bench, back to bed, back to bench, with his PAG9 in the center stage. Dr. Hemant talked about possibilities of fertility preservation that's available in young cancer patients. And lastly, Dr. Alpana Sharma talked about her HMGB protein and its correlation with the cancer diagnosis. So yes, that was a good session. Session 1 late, but doesn't really matter. So with that, we conclude this session. Back to the organizers.
I think we had a very long day and almost going to complete 11 hours, started at 10 o'clock, going to be 9. So no words to convey big thanks to all the chairpersons, all the speakers, and it has been a wonderful science whole day. So with this, we close today's proceedings and look forward meeting all of you tomorrow morning around 9:45 or so.
Big applaud to Professor Loya, you and your whole team in keeping
Thank you.
us
Thank
all.
you very much, everybody.
Thank you, sir.
Bye-bye. Bye-bye.
Thank you, Dr. Sri, Dr. Pradeep.
Thank you, Dr.
Bye-bye.
Loya.
Bye-bye.
Thank
See you
Dr.
tomorrow.
Marathon session, sir.
All wonderful, wonderful.
Wonderful session.